Hey, we are back. Let me move back into place and get the reactive up. There you are, Clockwork. There you
I'm here. I'm
are.
here. We're living.
Hello.
Oh, I thought I had my hard remote for a second. Okay, I'm going to minimize over here. We're good. We're good. Check, check. Okay, good. Yeah.
All right,
Uh,
sick. Clockwork, you want to introduce yourself while I make sure that the right YouTube chat is active?
Perfect. Perfect. Hey, everyone. My name is Clockwork. I'm also a uh, life sciences YouTuber here. My reactive PNG here is actually from my very first stream with Phi. Oh, from way back in the middle, very beginning, almost of 2024. Um, I hadn't really like had my immunology up as much as I could have back then. And so this is a costume I was wearing for like the Phi universe. And I was like, yeah, I'll just be a CD4 T cell because there's helper in the name. And it's like, yeah, I'm helping. However, now that I a little bit better versed in immunology, it's kind of weird because this kind of implies that I'm Phi's boss in universe, which is not what I was going for. Like if I could go back, I'd probably be more something like a mast cell,
The
which is
big
more like a
boss.
friendly, yeah, yeah, more like a friendly rival as opposed to uh, the weird overlord that CD4s are to a lot of neutrophils because they tend to do a lot of like recruiting and saying, "Hey man, can you go kill this guy for me?" style stuff like the mob boss in the T cell family.
Big head
Nice.
honcho.
Yeah.
So let me explain a little bit as to what we are going to be doing today. We are going to be ranking every Nobel Prize from the birth of molecular biology in the 50s to the recombinant revolution in the 90s. And we've got opinions. Okay, we've got opinions. Um, I'm going to have to lean heavily on Clockwork. My voice is going. So we're going to teach you a little bit about the Nobel Prizes, but I'm gonna be honest, I am not that familiar with the majority of these. So in some cases, I won't have any additional insight other than what the official Nobel Prize blog has. Like their website has a little blurb for everybody. So that's what we're going off of. And I want to be very clear. This tier list is a vibes tier list, okay? Every Nobel Prize is important. Every Nobel Prize is cool to somebody. This is a taste kind of thing. Do I particularly like the science? Does it make me feel things? Okay, this is a personal ranking. You can have your personal ranking. These are going to be mine or Clockworks, and we're going to have a little bit of time to talk about those. So, are you ready, chat, to get judgmental over scientists that are much more accomplished than either one of us?
Yeah. There's just something so uniquely cursed about two science YouTubers actually having the brazenness to rank the Nobel
Yeah, who the
Prizes. Like it
hell are
feels,
we?
yeah, it's like who like Z tier us.
Yeah, like we're just two schmucks, okay? But I'm
Yeah.
I'm like even schmucks can have tastes, right? So this is
Yeah.
more about taste. So let's go ahead and
And there's a lot to learn from like ranking them too, because you have to kind of like figure out your priorities. So I think
yeah.
y'all will at least learn a lot from our about our perspective.
All
So
right.
I'm very excited to get through it too. I've
So
put maybe a little too much thought into it.
we've got that up. Clockwork, let me share my screen with you so you don't have to deal with YouTube delay. Okay, so that's just for you. So
Yeah.
this is our setup. Okay, this is our setup. So Clockwork, maybe if you want to do like the first half, and then maybe I could do
Of
the
course.
last half, so we can share the load. I just need a little bit of a rest from all the craziness. So, what is our first Nobel Prize that we will be talking about here?
Right, so chat, just so you know, we are covering the Nobel Prizes for physiology or medicine. Occasionally in molecular biology, the Nobel Prizes kind of lapse into chemistry as well, but trying to add in chemistry would bring in a lot of physical chem and kind of go way outside of our wheelhouse. And we are starting off with a bang. We're focusing on kind of like this mid period where molecular biology was really finding its stride. And we're going to start with 1953 with, pretty much the only one that actually deserves S tier. And I'm sorry. In 1953, Hans Krebs and Prince Albert, and Albert Lippmann too, won the Nobel Prize for their work in elucidating the citric acid cycle, also known as the Krebs cycle. Which, if you're in sort of the biology space, it's the bane of your existence while you're an undergrad. But then you kind of learn to appreciate it as you go through. So just to give you a background, folks, nutrients are broken down in our cells to release energy for the construction of cells. And so after Albert Georgi, a Hungarian biologist who actually got the Nobel Prize, I think, in 1937, identified several important reactions in these metabolic processes. In 1937, Hans Krebs was able to present a complete picture of an important part of metabolism, which is the Krebs cycle, which is all the reactions we use to squeeze as much electron carriers out of sugar and metabolites as possible inside your mitochondria. In this process, which is cyclical and has several steps, nutrients are converted to other molecules with a large amount of chemical energy. The latter are ultimately converted into adenosine triphosphate, which provides chemical energy to facilitate other biochemical processes in the cell via one of the coolest things in life itself, ATP synthase. It's and just the work that had to go into this was absolutely incredible. And also Hans Krebs had to abandon his lab in the middle of doing this and go to England to complete his research.
Oh,
For
England.
first of all, England. Ugh. Second of all, geopolitical reasons. So
Ah.
I mean, easy S tier, right? Like it is the foundation. It is
Dude,
the
easy
beating
S
heart of it.
tier.
Yeah,
Easy S tier. I'll
yeah.
say a few words as to why I think this is S tier. When you learn, as Clockwork beautifully explained, the first time you read about the Krebs cycle, it sucks, dude. It's complicated. But when you do more biology, you know it doesn't matter what you're studying, tumor biology, neuron biology, whatever biology. Eventually, you come across, oh, that's the citric acid cycle. That's important here too. Yes, the citric acid cycle is so central to everything, and its function is so beautiful and elegant. While you're learning, it sucks because it's complicated. But when you take a step back, it's like, oh, this one central metabolic pathway. Like when you see the pathway of metabolism in a cell, the citric acid cycle is literally the center of it. It is that important. It is the heart of metabolism in the cell. So many things feed into it, and you get so much out of it. Um, and I love it. I don't remember every step, but I do love it. Any other words, Clockwork, for this?
The other thing too is that if you really take the time to truly memorize it down in your heart, basically the back of your hand, everything else in sort of pharmacology, signaling becomes so much easier because the Krebs cycle touches so much, so many weird things too. Like if things get out of hand, you'll see it even in transduction and stuff. So it's one of those things where it's that first moment, if you really master your understanding of the citric acid cycle, you will kind of unlock this master key that helps you contextualize other biological information as you go through either a medical school degree or just trying to understand how molecular biology works. So
Yeah,
there's
it's
a
a
reason it's foundational here, and it's a reason that it's more important than maybe another foundational Nobel Prize that we will visit momentarily, maybe in 10 years. I don't know, boys.
hot takes. But yeah, I think unproblematic S tier. Also, thank you so much for Davik for joining membership. Thank you so much. All right, next Nobel Prize.
Yeah, so after that, something that at the time was honestly a miracle, but has kind of gone out of fashion. We've got in 1954, John F. Enders, Thomas Weller, and Frederick C. Robbins. They cultured viruses. I mean, I want you to understand that many infectious diseases are caused by viruses, and which are very small, very hard to study. Like we didn't have images of them until electron microscopy. It was invented much later, like in the 20th century. A virus lacks metabolism and of its own, and it cannot multiply without infecting a living cell. And for a long time, the prevailing opinion was that viruses could not be cultured in a laboratory. However, in 1949, John Enders, Frederick Robbins, and Thomas Weller succeeded in culturing the virus that causes polio in human muscle and other tissues in a laboratory setting, which is just wild. The fact that you could actually get it to grow and be studyable. This became an important step on the road towards a vaccine against polio and just the science of virology in general. But it's still tough because it's one of those things where it's kind of gotten lost in the sauce as virology became a more advanced science. So this one's hard to place. This is an easy A or B tier, but I don't want to dilute the value of other A tier things and
Yeah,
throwing this immediately into A
I'm
tier. I
actually
mean,
going with BC. So maybe we'll. Oh, by the way, this is an important part of this stream. If you want to change the position of any of these, you can donate $10 and move any of these to any kind of tier, and we're just gonna have to stick with that, right? So if you want to get up in. Oh wait, the freaking thing is. Oh yeah, no, it's still up there. That's somewhere, right? Oh, is that not? Oh, it might not have updated, but hold on. Let me put in a little bit of extra text that just says $10 and we'll move anything anywhere.
I mean,
10.
within reason, within reason. If you come in defense of a certain hot take that we're going to have, actually
Some
pretty
fighting
early in
words.
this, we might have to resist their fi. I might have to like
I don't know, for
donate 20
charity?
to. Yeah, I
For
don't know.
charity, dude?
Turn it into a bidding war.
Yeah, for a
How
bidding war.
principled are
Yeah,
you?
yeah, yeah. A bidding war. I'm
Yeah.
okay with the bidding war.
I got you. I
Where can I
got you.
put this that isn't obnoxious? Let me change the color on this so it's a little more easy to
Yeah,
see.
highlight it. You got to let people know.
Yeah.
Maybe we should just be like scoundrels. We should just like make everything F tier. Like, you love enzymology?
Yeah.
How much do you love it, bud? It's gonna be F tier until you fix it, buddy. I'm sorry.
All right. Yeah, I'm going with B/C. Since we both agree on B, I think a fair place to put this is actually, yeah, like B tier. It's kind of cool.
Immediately revealing the entire issue with this. Like I feel awful doing that because again, it was so it's polio, man. They practically eradicated it a
I
decade later thanks to this
I know,
discovery.
but I
So
want
I
I
know.
want you to get in touch with your feelings on it because right, because this is what I mean. Feelings, emotions are an important decision-making tool, and if you do not viscerally feel emotion when you think about culturally viruses, it cannot be an S tier, right?
I completely understand. Yeah,
Yeah, this
exactly.
isn't about
Although
the impact. Yeah, polio is important. Like nobody is arguing that these aren't important. What we are talking about is just our personal taste. What makes us go, that's tasty biology, right? Culturing viruses, like it's important. I get it, but it doesn't activate my neurons. So just put it that way.
totally get it. This which makes the next one maybe kind of like controversial because I have
Ooh,
strong emotions about
spicy.
this, just because like
Oh, sorry. I
looking at
think
molecular.
sorry. I think this website actually. I ordered this last night in the terms of year, but it feels like it scrambled it again. So we might have to. I know the one that you're talking about, but I'm actually going to
Yeah,
go in order so that we keep track of where we are. It's important for the
gotcha.
Word document I send you so that you don't have to go all over the place to find our
No, I hear
place.
you. I hear you. No, I mean, yeah,
Uh,
we're in 55, right? Yeah.
yeah,
I mean, unless
55.
yeah,
Yeah.
yeah, exactly. So while you do that file, just kind of give us the next description. So
Yeah,
it's
yeah, yeah. Go ahead.
1955, and this is really where we start like establishing enzymology as a science with Hugo Thorell, whose name, yes, I am butchering, and I am so sorry. I want you to understand, catalysts are substances that speed up chemical reactions without influencing the end products. Enzymes are catalysts active in biological processes, which was groundbreaking information back in the day. Like it's amazing like some of the sort of like half-baked like half theories folks had for like how like biochemistry happened as biochemistry started emerging from like the 1920s until like the mid 50s, early 60s. So what Hugo Thorell and colleagues did, they investigated how enzymes that promote oxidation reactions are constructed and function. And a breakthrough came in their laboratory in 1935 when Thorell demonstrated how a yellow-colored enzyme in yeast had two parts, both of which were crucial to its function. So they split it apart, demonstrated how it was split apart in the lab, and demonstrated how they both contributed to the function in oxidation reactions specifically. Um, Both of which, and so he also explained how iron atoms in many enzymes have an important function in transporting electrons. And this is kind of like the foundation of what powers the Krebs cycle, because we are powered by oxidation and reduction reactions. Like the whole business of being alive is moving electrons across the mitochondrial membrane so we can turn that kind of like almost electric current into ATP via ATP synthase. And because like eventually this information led us to elucidating like NADPH oxidase. Like for me, it's an A or B, but at the same time, the rest of of enzymology kind of immediately left this behind and kind of forgot about this foundational discovery. So I think
Yeah.
I'm being overly defensive by saying, but we didn't even know about oxidation in biology until then, man. Like, come
Yeah.
on.
I'm kind of a stickler. I would say B. I like enzymes, so I can appreciate them, but it doesn't get me excited. But I
I hear
think
you. I hear you.
enzymes are kind of cool. I think yeah, B is a pretty comfortable place for
But
for
not just enzymes, right? But like the enzymes, the
yeah,
ones
yeah.
that are like in the mitochondrial membrane. Like a lot of these early prizes are just oops, all mitochondria, which for me is going to have like a bit more of what is it called? Like
Oomph.
a good place, yeah, oomph factor, more of that emotional factor.
I
Um,
think that's fair. But I think we can
all
agree
right,
on at least B.
yeah, I think so too. I think we'll
Ooh.
keep it in B. And now we're going to get
Sorry to
to
cut you off.
yeah, go for it.
But we got a donation from number one virology fan. Culturing viruses in S tier, let's go.
You have no choice.
Yeah,
It's
we
over.
have no
It's over.
choice. We have no
I mean,
choice.
You're gonna really pollute Krebs like that, dude? You're
We're
gonna make
moving that
Krebs's
up.
legacy as
Uh,
at the same level as polio?
and on the
Like,
YouTube side, host cell go pop. I feel much emotion about culturing viruses. All right. I mean, both of y'all, you're getting it up there.
to be
We
fair,
can't argue. It's for charity. It's for charity. We're just gonna have to swallow that.
exactly. Also, like, imagine biochemistry at this time. Like, you're wandering, like you're in a black room. You can't see anything, and the amount of stuff these scientists managed to figure out with nothing, like not even the right kinds of scopes. What somebody in the comments was like, with the fact they did this without NMR, blows my mind entirely, and I completely agree. Like, I can barely do it with the tools we have now. Imagine doing it with less, like, popsicle sticks and hope, right?
Yeah,
Like,
that's a
it's
lot of
really
early
amazing.
biology is popsicle sticks and hope. And that's what makes
Yeah,
classic molecular biology so beautiful. If you ever
yeah.
end up in a situation where you're reading papers from the 50s, oh, they are terrible to read. But when you understand what they did, it's like, wow, you really just had like, I don't know, Notepad, like the equivalent of a programming language like written in Notepad. Like technically you could code in Notepad if you
A
really
little
wanted
bit.
to. You could.
You had punch cards by now, right?
Yeah.
Like, you're working with it a little bit, and not to and to sort of like tease, hot takes incoming, but sometimes you do just follow ass backwards into the right answer. And do you deserve a Nobel Prize for that? I don't think so, but we'll find out together.
All
More on
right.
that later,
More on
though.
that later. Let's head
Um,
into that next Nobel Prize.
All right, so here's where, we see that, the Nobel Prize in Physiology or Medicine used to be just for like straight up medicine. So in 1956, Andre F. Cournand, Werner Forssmann, and Dickinson W. Richards got the Nobel Prize for something that's still used today, and that's cardiac catheterization. So even though one of the principal investigators here, Forssmann, succeeded in inserting a catheter into his own heart in 1929, badass, there was great hesitance in the medical community about continuing this type of research. Nonetheless, beginning in 1941, Andre Cournand and Dickinson Richards published a series of studies that established the use of cardiac catheterization, among other things, in order to introduce contrast fluid for X-ray images and to measure pressure and oxygen content inside the heart itself. Like, for a long time, it was really hard to know what was going on inside the heart with lots of cardiac issues. Because it was possible to reach the upper chambers of the heart, blood pressure and the blood's oxygen content could be measured on the way from the heart to the lungs, which was impossible before. And people still do this to this day. Like, you take a small little tube, you put it in like one of your like leg arteries and veins, sorry, and you just shove it on up in there, and it's still used as a diagnostic tool for, like, figuring out cardiac issues. So given that a lot of the real big medical breakthroughs of the 20th century are involved with the science behind cardiology. We're going to get into cholesterol a little bit later too. I think this might be like an A tier, just because, like, imagine getting the Nobel Prize in the 50s for a protocol that's still widely used today, even though it's kind of outside of our wheelhouse of molecular
That's
biology. I
so
don't know how you.
interesting. That's so interesting. Your placement of this is so interesting, dude, because
I
I think
know.
what this is saying is that we do have different tastes. That's why, even though Clockwork and I both do molecular biology content, I always come back to this. We're not actually competitors because we like care about it. I mean, there's a commonality to what we care about, but there's a lot that I think are mutually exclusive. I don't care.
You know what? Fuck
I'm
heart. You hate hearts. How dare you?
I'm putting this in C. The reason why
Oh wow.
I'm putting this in C, and we'll average out to a B, is because like I just find like molecules and cells to be more interesting than this is physiology, right? Cardiac catheterization. This is a medicine thing, and shout out to my medicine bros. Love what you do. I was not fit for your science. It just doesn't excite me. I acknowledge that it's cool to like a certain degree, but like I think a good way to put it is, how likely am I going to do a Find the Neutrophil video on cardiac catheterization? Is there something about that topic that pulls me to make content? Do I love it that much? And the answer is not really. Whereas enzymology, culturing viruses even, there's something there that I could make into Phi content. But cardiac
No, I
stuff, not for me, really.
totally hear you. And I think, audience, if you want to really understand the distinction there, you're going to give that a C tier because you wouldn't make a video about that, whereas I would give it a B, A tier, because I would watch the bejesus out of just like midlife crisis explaining cardiac catheterization. Like, it's just one of those things where, I want to understand why you like how and why you think about that. But then again, my ideal YouTube content is old men describing how they carve wood. Like, I do not watch the kinds of videos I make at all,
Yeah,
actually.
same.
I'm
I
in a
don't
completely
watch
different.
my content.
Oh yeah. I mean, obviously, like, some of the S tier stuff. Like, I watch every Kurzgesagt video. I try to keep up with
Yeah.
Veritasium. I obviously watch all of your videos, Fi. That's not
Yeah,
even a
thank you.
question. Um, but at the same time, like, outside of that, like, my YouTube diet is completely different from when it was when I was first inspired to make these kinds of videos, which is super weird when I think about it.
I watch your videos to do artistic studies. I'll put it that way. And I'm being honest, right? I like watching your stuff to analyze them. And we've reached the milestone that will allow me to analyze your videos on stream if you would allow me to.
Of course you
Um,
can. Yeah, and I'll like for me, the most important thing is to be in service of the science, right? So, my entire channel exists because the protein data bank is CC0, so I can just take those structures and make them wiggle
yeah,
and make you cry about them,
goes
maybe.
hard.
Yeah, so I need to make sure that I have that same kind of openness on the other side too, and not only analyze, but I hope analyzing my videos leads to a couple of Blender streams as well, so we can get you up to speed and comfortable in the molecular wheelhouse.
Oh, that would be
Let's
interesting.
go. Yeah. Right.
Yeah, if any
All right.
doctor wants to spend $10 to make me move cardiac catheterization up because you're a doctor and you care about the way the body functions as opposed to me, I care morally about cells. Feel free to drop that 10.
Yeah.
Just saying. All
And
right,
then if you
next
want to
up.
agree with the Phi dogma, what's gonna happen if like you have multiple people who just keep donating to like bump something up and down? Like, what if cardiac catheterization just gets completely kicked
Bidding
around?
war, dude. The last
Bit-
person in gets the position.
Amazing. All right. So I'm actually very based on the criteria we've set, I am super interested how we're going to handle 1957. All
Okay,
right.
go
So
for
check
it.
this out. 1957, Daniel Bouvet gets the Nobel Prize for discovering antihistamines. Obviously, hormones and signals and other signal substances are chemical materials that convey signals governing different parts of the body. These substances include histamines, which are involved in allergic reactions, and it can be difficult to moderate these effects. Like learning how antihistamines work is basically kind of like managing certain kinds of the immune response without killing you, which is actually very difficult. Um, but then Daniel Bouvet looked for substances that block histamines, and in 1937 he found the first antihistamine, which later led to other antihistamine formulations to relieve allergies. Fun side fact, that first antihistamine he discovered was extraordinarily toxic. So he's like, cool, I can turn off this part of the immune system and kill a guy. So obviously, the science of antihistamine pharmacology got a little bit better afterwards. Um, but Bouvet also produced additional substances that block signal substances in other ways and have a numbing effect, among other things. Like, dude basically helped establish pharmacology. Like he was part of like the pharmacological revolution that happened in the early to mid 20th century. Like he went to the Amazon, hung out with a bunch of indigenous folks with like the blow darts that like knock their like they hunt with. He developed a muscle relaxant that was used as an anesthetic for like 50 years, based off of that sort of like on the ground research. So like, dude really gave himself entirely. First, he moved from Switzerland to Italy to get his professorship in pharmacology, and then he just like went out in the world and found like substances he could try to synthesize in the lab.
Him.
So again, like I kind of like in an A tier situation, but then again, the thing that he got the Nobel Prize for, super deadly. So it's like, are you giving the prize to the science or to the guy? Like, so I understand why this might get a little bit lower given where else we're going to go, but at the
Yeah.
same time, A tier is suspiciously empty at this time. So I'm also like trying to even out the geometry of our
Uh,
of our of our tiers here.
I could be swayed to an A. It feels more at home at B for me because yeah, I don't know anything about the scientists for most of these. I didn't really do that much research. It's mostly about the finding and its context in my biological training. Like histamines and allergic reactions in general, I do have a high cognitive appreciation for because they are so weird. And I'm afraid of them because it doesn't make any sense to me. I know you did a video on allergies, right?
Briefly, I did a video on one new thing we discovered about certain kinds of allergens that I clickbaited a little bit too close to the sun with. And I feel very fortunate that it didn't blow up because I would have felt really bad if that was something that got a little bit too far out there. There's better
I know
ways to
that
talk about
feeling.
immunology.
I
Yeah.
know that feeling. But the idea is allergies, like, growing up, the immune cell kills bacteria, blah, blah, blah. Okay, when the immune system freaks out over pollen, that's when I think the logic that I understand of the immune system starts to go away. So I pulled everything down. I think I'll let you pull me up here. We'll put antihistamines in A tier. Not where I
Let's
expected them to
go.
go, but
Let's go.
I
'Cause
accept
your
this.
secret, yeah. it's secretly you're giving an A tier to pharmacology, the concept, because there's a lot of like really cool pharmacological stuff that's not going to make it to the
I
Nobel
mean,
Prize
I don't
kettle.
know. Isn't there a Nobel Prize coming up later that is more like the pharmacology Nobel Prize?
Yes. However, I mean, there should have been like there should have been a handful of others in the intervening years, but we'll get there when we get
Hmm.
there. So it's just good that like old school pharmacology got this, and also antihistamines were like a crazy idea back in the day. Like you can do this, like. Anyway, we'll get there when we get there. Why
Hmm.
isn't there the steroid Nobel Prize? Anyway, we're ramping up towards a big moment early hit here in the stream, folks, because we're getting to 1958, which means we got George Beadle, Edward Tatum, which they're going to get the Nobel Prize for like elucidating the beginnings of the central
Yeah,
dogma.
it should be
And this is a
beginning. Yeah. This is not
yeah,
like they figured everything out, but the beginning of the central dogma. Yep.
exactly. We're going to really establish it in a couple of minutes, actually. And then it's a joint prize also given to Joshua Lederberg because of the work that he did figuring out bacterial transduction at the Howard Hughes Institute when he was 22. And I just want to give this
Oh my
like an
God.
immediate F tier for try hard behavior.
That's so
Like
try hard, dude. 22. I can't imagine.
22, 22 nanorooms is 22. How dare
No,
you? Like, what?
Nanoroos is so young. What the hell?
It's so much better than us at this whole thing. How
God,
dare you?
such an aged
Um,
neutrophil, so crusty. Young people are
yeah.
really talented, aren't they?
Oh, I'm just a big bag of reactive oxygen species at this point.
All right,
Anyway,
you know
let's give
what?
the context here. Yeah. So
Salt,
organisms
we're putting it in F tier for salt. That
solve.
someone
No, you
so
should,
young can get a Nobel Prize.
again. I mean, Lederberg also went on to do some really awesome stuff, and that
All
kind of
right.
helped put the Howard Hughes Institute on the map. So if you wanted a more even-handed approach, like give it a C tier, just because it's unfocused. Like why are we giving out joint prizes? What are you doing? Why are certain other people not getting joint Nobel
We just got
prizes?
a donation from Surprise Pants, not to interrupt you, for to swap our positions, which sure. That's fine. We'll do that. But yes, continue your rant.
Oh, you mean like within like the YouTube stream itself? Like, would you have to take over the Clockwork channel, and I'd have to like learn how to
Oh,
draw
oh, I
monster?
don't want that, dude. That would require doing Blender, and I do not want that.
I mean, it's possible. That should be
No,
like an
no,
April
no,
Fool's
no,
joke,
no.
really. We'll jump off that bridge when we get to it. Anyway, so yeah, let me
Uh,
give you some context. So, again, organisms' metabolism, the chemical processes within its cells, are regulated by substances called enzymes. So, George Beadle and Edward Tatum proved in 1941 that our genetic code, our genes, govern the formation of enzymes. They exposed a type of mold to X-rays, causing mutations like the DNA changed. And afterwards, they later succeeded in proving that this led to definite changes in enzyme formation. So, like F tier for try hard behavior for Lederberg, but A tier for wow, that's experimental design, dog. Like really isolate your variables, and this is how you start going from like the dark room of molecular biology to like, oh, this is how it do be working, right? So
I I
the fact that you knew
agree.
you would change a gene, the fact you knew you had changed a gene, and then stuff inside the enzyme that you could measure changed in 1941. It's World War II. And you're like, nah, man, I figured out enzymology actually in New York of all places.
I will say,
Um,
I'm actually going to drag you up here. I want to put this in S tier. Is that okay?
I'll go with it. Yeah, exactly. Because like I think, as y- an experimental design person, I think you can really appreciate like a well isolated variable.
Beautiful. This
Yeah.
is. I
And
think I read this paper in graduate school.
no way, dude.
I read a couple of these actually, as we'll get into. Just again, they're not easy to read. They are essentially Shakespearean because they were written so long ago. So the language they use is not meant for an easy read. But when you take a look at what they did, oh man, a human brain came up with that. Because now we
Yeah.
have fancy tools and you can really push what you can do with fancy tools. But again, they were rubbing two sticks together, right? They had nothing. And to be able to determine to crack the code based on like using, I don't know, Play-Doh and safety scissor. Like it's amazing. Remember, I don't remember the details of the paper, but I remember being amazed by it. So that's
Like
gotta
what
be
is
an
the
S tier.
and it also like quick shout out if you are a certified 5 the neutrophil head, you are well aware and very respectful of just mouse models in general. A lot of 5 papers have a lot of mouse models. You see mice
Hmm.
as a really strong model organism. Shout out to the OG eukaryotic model organism, the one that really did all the good work. And that's I can't believe I literally forgot the scientific name. Just yeast in general, like baker's yeast.
Saccharomyces
Cerevisiae.
cerevisiae.
Oh my
Or
God,
that's our
oh my
guy,
God. So
dude.
I've
Saccharomyces.
never blanked. That's maybe the most embarrassing blank out moment I've ever had in public in my life, and I've had some truly truly legendary ones. But yeah, let's not even mention Liederberg bacterial transduction when you're 22. Get a real job,
Yeah.
dude. Actually, you
Get
did,
a
and it
real
was really,
job.
and you did a really good. It was a really good one too.
Yeah.
So bitter.
So young and so
Anyway,
successful.
anyway,
And
all
meanwhile,
right,
us two old heads, relatively.
exactly. Okay, I don't know if this is going to be controversial, but
Hmm.
this one's very important to me.
Okay.
As this is the one thing I ever did in the laboratory, basically.
Okay.
So 1959, Severo
Oh, sorry. Are
Ochoa
we
and
putting
Arthur.
bacterial trans? So it's two different ones. Central
Oh, it is
dogma
two different.
and bacterial
Oh,
transduction. They both got it.
you split it up. I thought you kept them together the way the Nobel Committee did. No, keep him, and you can decide between F and C tier.
F tier for being a tryhard.
How dare you? But also, this is actually a very important
Yeah, it is
scientific
important and it's
discovery,
cool. But also,
and
he
it's
was
also
22, so I'm mad.
yeah, and also it's like, oh, you used E. coli as your model organism. Good. Wow, I wonder how creative. Even though I think he might have been part of establishing the protocols for E. coli.
Salt.
Anyway, yeah,
It's not
so
a real
silly
F
again.
tier, but it is our spiritual
Exactly.
F tier.
Yeah, it's a bait F tier. Like
Hmm.
if you appreciate prokaryotes as model organisms for how we understand life on Earth, you gotta pay us to put that one up, boys.
Yeah.
I'm sorry, forgive me. Um, but you do not have to pay me to throw this one into an extremely high tier because this is when we discovered one of the goats, boys,
Oh, here we go.
fellows.
Let's go.
1959. So, Ochoa and Kornberg isolated for the first time the prokaryotic DNA polymerase 1,
I'm
the big
snapping.
boy
I'm
enzymes.
snapping.
The substances known as DNA and RNA bear organisms' genetic code and also determine their vital processes. Arthur Kornberg took an interest in the way DNA and RNA are formed and which enzymes steer this process. And again, we're talking like the early 20th century. This stuff's hard to figure out. Um, Enzymes are substances that speed up chemical processes inside organism cells without being consumed. And by studying bacteria, Kornberg succeeded in isolating DNA polymerase 1. I forget what the code for it, in 1956, an enzyme that is active in the formation of DNA. Using a DNA molecule as a blueprint, the enzyme builds a copy of the DNA molecule from nucleotides, which are the building blocks of DNA, which is going to set up um, just so much. And the fact that we like
The
have this,
recombinant revolution, baby.
it's
Recombinant
also
technology.
like the eukaryotic DNA polymerase is, massive, but even the prokaryotic ones are very big. It's a beefy fella. So the fact you can isolate a very complex, very big, very volatile protein like this, again, with popsicle sticks and dreams, is a miracle. And of course, by isolating DNA polymerase, it's super easy to study from that moment onwards. And so we have the PCR, the polymerase chain reaction machinery, because of the foundations laid here. And just because that powered the whole genetic revolution and everything else, I mean, that's an S
This
tier
is
for
this is an
me.
S
Like
tier. This is
this is
instantly S
foundational
tier, dude.
instant foundational S tier,
Instant
and it's
S
like,
tier.
and I mean,
Uncontroversial.
I just want you to hold up, get your right hand, audience, and just make a little crab claw. That's your DNA polymerase right there. It's like so easy to imagine, and it's just gonna like use that little crab claw hand and glue DNAs to like
Hey, Clockwork,
polymerize DNAs
can you
together. Yeah.
recommend me a good video on DNA polymerase?
I can recommend you a half decent video
Come on.
on the eukaryotic DNA polymerase, which is very different, not nearly as elegant looking. You can kind of see the glove in eukaryotic polymerase, but not so much. And also, the fact that it organizes into this gigantic replosome slash primosome is the whole thing. Whereas the prokaryotic instrument, it's way more elegant. Like you
Yeah.
understand why people back in the day
What
were
is
like,
the
hey,
video, Clockwork?
yeah, but if you want to go to, youtube.com/at clockwork bio, I mean, you'll find some interesting stuff there
Yeah.
about the eukaryotic central dogma. Um, all right, and now
Oh, sorry.
but
Just
let's keep
to
moving. Go
acknowledge.
for it.
Thank you, Anonymous, for the donation. Thank you so much.
It's awesome. And forgive us for just kind of rushing through this, but we do
We
have
have a lot.
30 years and 2 hours, and we are not even
Yeah,
25% the
we're
way
not making
through.
time. Some of these will have to go much
We're
quicker.
we're gonna have to like snap through. Speaking of which, this is going to be tough because I know you're passionate about this one, Fi.
I
It
mean, do
gets into
you? I just
60.
made a thumbnail and I wasn't really thinking about it. So, yeah,
Yeah,
go.
I got you. I got you.
Read it off.
1960, we get immune tolerance. So our immune system protects us against attacks by microorganisms, and rejects foreign tissue. Part of our immunity has a hereditary basis, though, but part of it is acquired and it's not present in the fetus. So in 1949, McFarlane Burnett theorized the ability to distinguish between one's own and foreign tissue, and how that might not be hereditary, but is something that is acquired while you're in utero, while you're a fetus. The theory was substantiated when Peter Medawar succeeded in performing transplants of tissue between different mouse fetuses. And the results had significant implications for establishing the science for organ transplantation. So this is where we start really kind of like understanding, oh, we can just kind of swap parts out too. But it's all about sort of beginning sort of the phases of understanding how the
Yeah.
immune system
And
works.
I'll just say, just so that we can get back on track, I think this is B. I put an S in the
Oh,
thumbnail for clickbait reasons, but I don't
you baited me. I
know.
feel baited now.
Yeah,
I was
yeah.
like,
I'm
I'm
not
ready
that
to fight over
invested.
this one. I got you. Yeah, because it's also like it's kind of like ghosts of the old school. Because it's like
Mhm.
we're doing eldritch horrors to mouse fetuses in this one, but it is very important sort
It
of
is very
foundational
important.
science. But it's not DNA polymerase. Get out of my
Yeah,
house.
it's not like, oh my god, the
I'm
GOAT DNA polymerase. It's
I'm not
important
putting
for
you next
sure.
to DNA polymerase as a molecular biology YouTuber this early in the game, especially when we're about to see some really cool molecular aspects
So
of
so where would you
immunology.
put immune tolerance?
I think we're gonna keep it in B,
Okay, B tier.
just for contrast, maybe for drama. I understand um, why you might feel passionate about that, but I haven't even started annoying you yet, fellas. Speaking of which, 1961, C tier is for cochlea. Uh,
I agree.
and don't even describe it. Dude did a lot of cool experiments elucidating how the cochlea works, but then we're gonna see a little bit later, other folks did a better job understanding how like the sort of neurology of hearing works. So good for you, Mr. Cochlea, but that's an easy. I
Yeah,
mean,
I think if
that's a
we were to
stretch
put
too.
something in C, cochlea, pretty reasonable. I don't really care. If you're an ear nerd, that's probably more meaningful for you, but I'm not an ear nerd, so I don't really care.
As somebody who made like my very last 2D video was the molecular biology of hearing and like signal transduction therein. So if you want to see like back when I was bad at doing videos, you can find that.
Oh, wow.
Um,
Okay, so that's coming from someone who did do a video on the ear, and you don't even care about the cochlea.
it's a cool it's cool, but having drawn the cochlea, having had to do like a 2D animation of like all those bones and stuff, it's like I'm
Sorry,
over it.
we
But at the
should at
same
least
time,
explain the cochlea is an inner ear structure
all right,
that transducts signals from and converts it into something that your brain can recognize. I don't know a whole lot about
yes,
it, but it's never really interested me to begin with.
totally get it. So you got a little snail shell in your ears, connected to three little bones, and as you hear the sound of my voice, I'm shaking your eardrum. Your eardrum is shaking those three little bones, and it's wiggling some jiggly fluid inside this little snail shell guy in your ear, and lining that little snail shell is a bunch of little hairs that are attached to cadherin molecules. Like your body makes little anchors to them, and as I vibrate that, I am letting calcium ions into your ear neurons, and that gets transformed by your brain into the wacky little sound of my voice. And as you get older, those hair cells break, and you will hear that little ringing in your ears as you go from high pitches to low pitches. That's just cadherin breaking, and your body's like, I don't feel like putting it back together. And so that's why you gradually lose frequencies that you hear as you go through. And again, the science here is actually incredible because we're using old school science. Like the person who did this cochlear research, like used a lot of really interesting like electro experiments to show
Yeah, but this
how
is about
signals are
taste. So I will
transduced.
cut you off there
Yeah,
so we can get to the
yeah,
spicier Nobel Prize. Like,
exactly. Yeah, I'm also like fundamentally a coward, so I'm trying to delay my hot takes as much as possible. But we're getting to a very big
this
moment
is
in
a
the
spicy
history of the
one.
Phi the neutral. Yeah, we're getting to a big moment here, audience, because you might be
Okay.
learning some things about Phi the neutrophil
And
that you
clockwork.
find, yeah, and me too, honestly. And I was so happy when I mentioned I was going to be toxic about this one, because Phi then was like, yeah, for obvious reasons, it's like, oh my God, thank God, somebody
Okay.
gets it.
So what's your take on
Anyway,
this? 1962.
so
Who wins this?
it's fellas, it is the true and genuine foundations of molecular biology. We are in 1962 when Quick, Francis Crick, James Watson, and Maurice Wilkins win the Nobel Prize for locking in a model that is the correct structure of deoxyribonucleic acid, or DNA. There's a name missing there if you know ball, and
Uh, well,
I'm gonna
let's
stay
let's
salty
let's
about it.
uh let's let's
Okay.
see if the audience can figure out who's missing from that list. Can you repeat?
If you don't know, audience, I will feel physical pain.
Yeah. Surely our audience knows who's missing from this list. Francis Crick, James Watson, Maurice Wilkins. Who is not there? Who is not there? There's somebody suspiciously missing from this.
I just want to
Hmm.
say somebody posted the correct answer.
Yeah.
However, it is the incorrect answer because you forgot the individual's title. And it's very personally important to me because these people, very purposefully demeaned this person a lot by calling them by a nickname instead. So you're missing the thing that comes before the name. That's so very important, audience. There we go. Thank you.
There we
Sorry, I'm
go.
good now. Chad has corrected. Right. So yeah. So Dr. Rosalind Franklin is missing from this Nobel Prize, and we'll get into that in a second. But let's explain this Nobel Prize real fast so we can get into the drama. Let's like turn our emotions off for a hot
Mhm.
second. So yeah. Where am I right? Yeah. So let's give some context. In 1944, Oswald Avery proved that DNA is the bearer of the organism's genetic code, and further explanation was provided when James Watson and Francis Crick, quote unquote, determined. I'm sorry, they did actually determine the structure of the
Spicy.
DNA molecule. They kind of lucked into it, like, and we'll explain later, like the data wasn't 100% there, and they're like, it's got to be this guy's, right? And it just happened, yes, it was. So this structure, DNA, a long double helix contains a long row of pairs of four different nitrogen bases, which allow the molecule to function like a code. The molecule structure also explains how it is able to copy itself, which is, once they figured out the structure, they're like, oh yeah, copying's obvious. So it really launched like molecular biology as a discipline. So the nitrogen bases always pair in the same configurations, so that if the molecule is split, its halves can be a supplement to be a template for copying it. So during the 1930s, a number of laboratories are racing to figure this out. There's a very famous American biologist who would later go insane because of his obsession with vitamin C, who almost figured it out, quote unquote, almost, and quote unquote, figured it out.
Mhm.
But people were figuring out using X-ray crystallography, which was a very, very new science and also very dangerous. I'll get into that in a second. What am I doing here? So in London, no, sorry, in England, and Maurice Wilkins and Rosalind Franklin worked to determine the structure of DNA using X-ray crystallography, famously with photograph 51. That was in King's College, and yes, in London. So they did not succeed in mapping the structure. They didn't have time really, because again, there was a race on, and also that wasn't really their job. Their job was just taking good X-rays of stuff. Um, but then Maurice Wilkins like brought that data to Watson and Crick and was like, "Hey, can you guys look at this and see if you could come up with a model?" Watson and Crick come up with a model. They get all the credit for establishing DNA, the structure of DNA, and that is one of the most famous like original moments in molecular biology. Um, so it came down to the data itself. Like photograph 51 is a an astounding achievement in engineering as well as science, because doing X-ray crystallography is just so hard, and it's also super dangerous. And the reason Rosalind Franklin wasn't included in this Nobel Prize, at least according to the committee, was because it's 1958 and she had recently died of ovarian cancer. Which is why putting this in F tier makes sense, because later on the Nobel committee is like, oh, we'll give the prize posthumously to other folks. But without Dr. Rosalind Franklin, there is no Photograph 51, and no one's figuring out what the DNA double helix is. Like the American scientist who went little cuckoo bananas for vitamin C, whose name I keep forgetting, thought DNA was like a reverse triple helix with the phosphates on the inside and the nucleotides on the
I love
outside. So
that. I love those
yeah,
models with the phosphates on the inside. It's so
I
goofy.
know it's so amazing, and that I wish I could remember that guy's name. I'm really embarrassed that I'm
I
blanking
don't
on it. But
remember
yeah,
either.
either way, yeah. So yeah, it's Dr. Rosalind Franklin, and for a really long time, Watson specifically of Watson and Crick just called her Rosie. Like in everything he did, he made this whole memoir about how he's the coolest guy ever. Oh, and Rosie was there too, boys, or whatever. And it's like Watson also is very problematic because
Oh, he
he
is a character for sure.
is Watson the guy who established Cold Spring Harbor, or did Crick establish the Cold Spring
Oh,
Harbor
I don't
laboratory?
remember. I don't remember
Neither
who
do
was
I.
Cold Spring Harbor.
Either way, like Crick, cool guy, pretty chill. Kind of didn't appreciate Watson's whatever. Watson kind of got a little high on his own supply. Assumed he
Not
was a
fondly
genius.
remembered. Not fondly
Not fondly
remembered.
remembered. Very, very much was like, okay, well, if DNA is correct, therefore racism's a good idea, boys.
Yeah. So
So
we're gonna
Watson
put
was not the
this
guy. Yeah,
as is. This is an S tier discovery, but an F tier Nobel Prize, I think, is fine to
yeah,
say.
exactly. And again, it's mostly because if you include Dr. Franklin, we're cool. I'm good with it because effectively, Dr. Franklin died of ovarian cancer, probably because uh, she was so not obsessed, but like focused on getting really good X-ray images, and that meant that she was next to a radiation source for up to like 40 hours at a time, just like fine-tuning the dials to make sure that her images were great. And also, frankly, the Immune Committee could have given her the Nobel Prize for her next work. Like, she immediately leaves London in the middle of all this drama. She goes to another lab. She does some amazing work crystallizing the structures for the tobacco mosaic
She's
virus, which are also
genius.
beautiful. Yeah, like pure genius, huge loss to science. Basically sacrificed herself at the altar of radiation to elucidate one of the most important foundational discoveries that have unlocked the whole history of molecular biology. So I think we managed to get through like the hot take toxic aspect of that pretty
Yeah.
good.
And I think I
There's
just
a
want to add one more thing to the appreciation of Dr. Rosalind Franklin. Crystallizing DNA, horrible. In order to get a crystal, you need the biomolecule to act in a very regular way. And DNA does have regularity to it, but it isn't something like is locked into place like certain enzymes where you can get it to crystallize very nicely. The actual fact that Dr. Rosalind Franklin was the one to do it meant that she had to been a very talented X-ray crystallographer at that. She didn't luck into it. She was skilled, extremely so.
yeah. And just to be a pedant. The other aspect of this is that there was actually the person who was doing the crystallization of DNA samples. Dr. Franklin wasn't generating like the actual DNA. Like one of her friends in Europe was like making these really
Mm-hmm.
sick DNA crystals, and she was taking individual samples from them and getting the crystallography X-ray data. So there's
Mm-hmm.
also like a third tier of, well, why do we care about that person who did these brilliant,
That's
beautiful
insane. I didn't know that.
isolations of purifications of DNA as well? Yeah, there's it's like where does the credit need to be given, right? Like the fact that Franklin was even able to get photograph 51 is a lot to do in part to this like endless infinite treasure trove of like perfect DNA samples, perfectly purified DNA she had. But that's neither here nor there. Again,
That's not
we could
pedantic.
talk
That's
all
just
day.
me genuinely being wrong. I thought she did the crystallization herself. I didn't realize she was
No.
on the machine, and that was her only. Not to diminish what she did, right? Like taking an image of this is also non-trivial, right? She was a very talented person in her field. I just don't understand structural biology because I'm a freaking noob.
No, I get it. Because also back in the day, it's like you got to have the person who's getting the pure, like it's
Yeah.
purification versus crystallization,
Yes. Yes.
like
Yes.
getting that pure stuff. Anyway, like that's why you don't go into molecular biology because you'll spend 5 years of your life purifying something just for the electron microscope to, literally obliterate it, and then you have to start over and try again, and hopefully you get a good cryo EM tomographical sample of it. So
Biology's hard, folks.
it's very painful. It's very painful. And then what's your reward for getting this beautiful structure? Some doofus like me will go and like make videos out of it because
Yeah.
it's CC0, baby.
Yeah. Let's
Everyone
go.
owns a protein data bank.
Okay. Let's move to the next one pretty quickly. Synaptic transmission.
All
Uh,
right, yes.
so,
Okay.
yeah. Go on. Real
This is a
quick.
super group of like Britishness. And so this is a vibes high tier and also just like experimental design again. So we are getting away from our hot takes. In 1963, we've got John Eccles, or I think it's Eccles, Alan Hodgkin, which is going to be the Hodgkin's lymphoma guy's great great nephew, and Andrew Huxley, Aldous Huxley's half brother. Like super group of super Britishness. They figure out synaptic transmission using big old neurons from octopuses. Which I mean, there's just so much going on there. This is one of those things. This should be a movie. So again, to give you some context, audience, the nervous system in people and animals consists of many different cells, and in cells, signals are conveyed by small electrical currents and by chemical substances. And so, using big old neurons, by measuring small variations in electrical charge at contact surfaces between nerve cells or synapses in the early 1950s, John Eccles, I really hope I'm getting his name right, showed how nerve impulses are conveyed from one cell to another. The synapses are different types, which either has a stimulating or inhibiting effect. And so, this key insight is what kind of establishes like neurology as a science as well, because we now can see how certain connections like activate things, whereas turn them off. And so a nerve cell receives signals from many different synapses, and the effect is determined by which type prevails. Um, yeah, and then it just kind of duplicates from there. But again, this is A tier for big ol' octopus neurons in the
A
first
tier for
place.
me.
Like
I'm a
yeah,
neurobiologist. A tier makes sense. All
Did you
right.
did you have to look at did you look into this actual paper because of grad school and stuff?
No, I did not. We did not
Okay,
have to,
gotcha.
because this isn't a part of my canonical education. This is like, I don't need to read this paper to do neurobiology because it is foundational, but for a formal training, you have to like learn the basic stuff too. So,
The question then becomes, wait,
yeah,
do you have to are big ol' octopus neurons still a valid model organism in contemporary
dude, we can do neuron
neuroscience?
stuff and flies, man.
Okay, yeah,
We
never
got a
mind.
lot better working with neurons over the course of these several decades, right? We could
I just
do
want to keep
fly
saying the phrase,
neurons,
just making sure. Yeah, it's okay. I guess
but
yeah,
it
with
started in
cell
the
culture and yeah,
octopus. It
yeah.
started
But
in the octopus.
let's just get back to big ol' octopus neurons, though. I just want to say that phrase one more time just to get it out of my head. I need
It's
that
good. It's a good phrase. Okay,
yeah.
fatty acid
All right.
metabolism.
All right, here we go. Now we're getting into molecular biology, folks. We're getting into the stuff that tormented you in school. So it's 1964, and Konrad Bloch and Fyodor Lenin are going to get their Nobel Prize for fatty acid metabolism. This is one of many cholesterol Nobel
So
Prizes.
many cholesterol Nobel prizes.
Yeah, there was somebody in chat, I think, yeah, yesterday. Was yesterday the day? Was
I think
yesterday
yesterday in
Friday?
the Discord, I think,
Yeah,
or in the chat.
pointed
Yeah,
out
you're right.
somebody's biochem professor has a whole bit where they point out that cholesterol is the molecule that's won the most Nobel Prizes, and I was like, oh wow, yeah, it did. That's so crazy. Anyway, so cholesterol is an important component in your body's cells, specifically in the membranes to provide stability and stuff, and it plays a major role in several biochemical processes outside of structure. So Fyodor Lenin and Konrad Bloch and their respective colleagues investigated how cholesterol and fatty acids are formed and converted in the body. Among other things, they showed that acetic acid is an important component in cholesterol, and how the formation occurs in reactions with many steps. So this is one of those things you definitely learn pretty much right after the Krebs cycle. If you're in an average third year biochem course, you've been tormented by OChem and now you're going to get tormented by pathways. If you're there, keep going. I'm proud of you, and I know you can make it. But this knowledge is important for an understanding of heart disease and other illnesses, and then just in general, again, it touches so much across the whole biological universe. But we'll get into that maybe a little bit later.
All right. So what do
So
you think
again,
about fatty acids? So I'm like timekeeping to make sure that we end on time.
no, I get it. So
Uh,
it's one of those things where the two opposing forces are, wow, this is so foundational for our understanding, but this really, really sucks to learn in third year biochem. I don't know what your experience was like, but
I put this in
this,
C. I don't care.
yeah. Okay, C is for cholesterol. Can we just put all the cholesterol
Yeah.
papers in C as a bit? Awesome.
Not even as a
Um,
bit. I genuinely think, for my taste, I just don't care. I'm sorry. I don't care about cholesterol. Maybe one day I
I,
could change my mind, but
I love how
me right now, I don't
neurobiologists are
care.
like neurosurgeons and neurologists in general, where you just feel like if it's outside of your field, it's beneath you. I love
Oh,
that.
it's
I
not
love how that
beneath
carries over to
me.
I
It's
know.
not beneath me. I just don't. I can't find the story. That's what it
I
is.
understand.
Is that it is a
Oh,
skill issue. I can't find it. Maybe somebody else better than me can find the story, but it's not obvious to me what the story is.
it's just the problem is that it touches so much that it feels like there is no story because it's just everywhere. It's one of those background characters that if you focused on it would be hilarious. It's the Rosencrantz and Guildenstern are dead of biology, which is the nichest possible reference. No one's gonna get that.
I
Anyway,
get it.
let's keep running. Okay, let's keep running.
All right,
We got
next
this. We
up.
got this. We got this. Okay, it's 1965, and we're starting to get the real heavy hitters in. And
Okay,
Francis
let's
Jacob,
go.
Andrew Wilf,
Monod, Jacob,
and
let's
Jacques
go.
Monod, when they get their Nobel Prize for figuring out it's mRNA, folks, the
It's
biochemical
mRNA,
processes,
baby.
S tier, maybe
S
A if we
tier.
have to
No,
have
S
it
tier.
done. Yeah, they figured out mRNA carry you can argue a B tier because their research made people think that mRNA is DNA's little assistant. Oh yes, Mr. DNA, I'll send that message for you. When it's mRNA's show, and DNA is just a hard drive for the real boss of the cell. But that's neither here nor there. Let's rush through. We got this.
Yeah, I
All
had to
right.
read this paper too. I don't remember a lot about it, but I did have to read this paper. It was
Like I
beautiful. I
read.
do not remember it.
Yeah, this is why I shouldn't have contextualized the first ones because now we're missing out on a lot of the bangers. But again, some of this context is going to start getting real complicated because now we're
Okay.
in the phase where molecular biology is a real science. Moving on, 1966.
This
Peyton
is an
Roux.
important one for this stream.
This is oncoviruses, right? Yes.
Mhm.
So so this is where we're getting our first cancer situation. In cancer cells grow and multiply beyond the normal limits. And in 1910, Peyton Roux extracted material from a cancer tumor in a hen and injected that into a healthy chicken. And the wild thing is that chicken developed cancer. And he concluded that cells from the hen's tumor contained an infectious substance, maybe a virus or something, that transmits cancer. However, the study could not be replicated in mammals and was long overlooked. When research showed that viruses can operate by affecting the genetic material of normal germ cells, interest in Ruse's discovery was reignited. So in cancer, cells grow in multiple. Oh, weird. That's just a copy. Awesome. So yeah, like this is that first thing where it's like, oh, um, not some cancers are like literally transmissible. Like you could a virus can give you basically cancer, and we're going to get into that more as we get into viral oncogenes and stuff.
Yeah,
So
so the,
I
the
feel like
next
this might.
paragraph is actually not redundant because it continues on to explain the full
Oh, I
context
missed
of
it.
that Nobel Prize, which is that there are certain cancers that are can be treated by hormone therapy, right? So the next part of that is that prostate cancer attacks a gland that is the male sex organ. It is one of the most common forms of cancer, and Charles Huggins showed that the course of the disease can be affected by hormones. So very much in line and relevant to today's topic. I'm just going to save you the trouble, Clockwork. This is a cancer stream. We got to put the cancer ones in S tier. I don't
Here's the
make the
thing, though.
rules, but that's what they are.
This is how I know you're an ineffective marketer, Fi, because everyone's here for cancer reasons. Therefore, the true blue business decision is putting this in C tier just to annoy somebody enough
True.
that they donate to put that into S tier. So if you are
No.
a stone cold killer in the audience, pay money to drop this down to C
No,
tier
dude,
once
I'm
we
gonna
put in
get.
more
If I put
comments.
it in C tier, I'm getting Google Doc eat. I'm getting canceled. I gotta save my hide. This is
I understand it.
self-preservation.
Yeah, that's why I'm asking your audience to do it for you. I'm not doing it. I'm doing enough here. But yeah, it's one of those things. All right. Okay, so this one next up, we're just going to keep going through. We're getting into a little more cell biology as well.
Mhm.
And again, this is close to me because I loved making a video about these kinds of cells. But 1967, Ragnar Granit, Keffer Hartline, and George Wald, they got their Nobel Prize for photoreceptors, the cone cell specifically. So our vision works by the light around us being captured by a large number of light sensitive cells located in the retinas in the back of our eyes. After a series of nerve switches and conversions of chemical and electrical signals, which is, by the way, an analog to digital signal transition that happens inside your eyeballs, which is crazy. Um, all that results in you seeing things, getting visual impressions using very sophisticated electrodes. And again, this is an experimental design Nobel Prize. Um, Ragnar Granit was able to study the electrical impulses from the retina cells, and in so doing, in those studies conducted from the 1930s to the 1950s, he demonstrated that there are different types of cones, different kinds of visual detector cells, basically, and that these are sensitive to light in three different wavelengths, which we eventually proved by looking at how those actual proteins are organized. That's called rhodopsin, and some of them hold on to a key pigment in a certain way that makes them more sensitive to blue light, red light, or green light. But the fact you did this with electricity alone in the 1950s and 60s is just pro gamer banger science move. So again, it's one of those things where, as a science appreciator, I feel like you got to put this high up, even though like we will get into the molecular biology of signal transduction a little bit later, though.
I'm okay with putting this in A tier.
Yeah,
How's
A
that
up is
sound to
good.
you?
Yeah.
A tier.
It's cones though, so I feel like C is for cones too. Like we keep that bit going, but it's too painful for me. I don't know if I
No,
can
no,
do
no.
it.
Cones are cool, dude. Cones are cool, dude. That's totally fine.
Yeah,
And as
this
a
is the exception that
neurobiologist,
proves the
right,
rule.
I had to learn a little bit about cones during my graduate time, so I do have a soft spot for them. So I think cones in A makes perfect sense to me.
Did you have to learn about like the various like glia cells that like change the signal around and like make it make sense? Because that sounds like pure torture to me. This is
Dude,
why I never could get in. Yeah.
at some point, those papers were on my desk. To be honest, I don't remember them at all. It didn't have as big of an impression on me. Like the S tier stuff, the basic core, beautiful, classic molecular biology left an impression. The cone stuff, I remember finding them cool. I remember finding them really hard to understand, but it didn't leave that like my life is now different kind of feeling that you get when you read certain papers. So I'll
No, I
say
get that. I
I
get
did
that.
enjoy the cone stuff. I don't remember a lot of it, but it I remember it being cool.
I love it. I love it. The chat bailed us out because it's A for vitamin A, and
Perfect.
retinol, the pigment, is actually a vitamin A derivative. So that's
I love
sick.
that.
Great, sick. All right. So audience, are you mad about us putting Watson and Crick in F tier? I want you to stay mad because this is one of the main reasons why. Because next up in 1968 is the discovery that actually deserves the Nobel
Oh, let's
Prize
go.
for this thing.
Let's
Because
freaking go.
I'm going to provide some more Watson and Crick slander. So they did that whole thing with their protein model. They didn't fall ass backwards into it, but it kind of, you could say that they did. And one thing that they talked about afterwards was like, I have no idea how this code works, guys. Crick very famously said, it doesn't make any sense. Like, how do you take four letters and make all those proteins out of it? It doesn't make any sense to me. Um, and it wasn't, this is a very rare Crick L, like Watson, all L's. Crick, the occasional too hot of a take. But 1968, the Dream Team. It's Robert Holly, Gobin Korana, and Marshall W. Nierenberg figure out exactly how protein translation happens. So we know that DNA is a code, but how do you translate that into protein? With letters, dude. And you turn the letters into words, and so there's a math to four protein letters, four DNA letters leads to 21 amino acids. It's very beautiful that I meant to memorize and forgot, but let me just give you the description, folks. In the 1950s, it was established that the genetic information is transferred from DNA to RNA to protein. A sequence of three nucleotides in DNA, known as a codon, corresponds to a particular amino acid in protein. The proteins are formed in what are known as ribosomes, which lie outside the cell nucleus. The transportation of amino acids to these ribosomes takes place with the help of a particular kind of RNA called transfer RNA, goat RNA, by the way, or just tRNA, actually. There exists a special tRNA molecule for each codon. Robert Holly was the first person to successfully isolate tRNA, and in 1964 was able to map its structure and show how we kind of like assemble proteins word by word effectively. So we translated the code, we figured out that it's like a physical thing. This is also a critical moment in RNA science where we realize, oh, dude, RNA is like information, but since it's so floppy, it can have a 3D structure and therefore perform catalysis itself. No way, dude. How does that make sense? Big RNA head here, by the way. Love me some
Who
good
isn't?
RNA
Who
science.
isn't?
Yeah, right? Right. Where's the ribonuclease P Nobel Prize, by the way? Huge omission. Sorry. Anyway, but yeah, anything to add there? It's S all the way.
so
This is
so
yeah.
this Nobel Prize, I've been saying, oh, I remember reading this paper and it left a deep impression, but I don't remember this paper. I remember this paper. I remember having to learn this, and the reason why I remember is this paper was so important for me as my development as a scientific thinker. I wish I could show it to you, but it is copyrighted by Science.
Wait, it's from 70 years ago and it's still under copyright by the Elsevier Mafia? You're kidding me.
Uh, Last time I mean, maybe there's some fair use thing that makes the copyright expire. I don't think it is in public domain yet. I think when I checked, I got the feeling that it was still okay. Well, here's the thing: if it is free to read and republish, I will do a journal club based on this. Because here's the thing: this paper is nasty. It is really difficult. It is maybe one of the hardest papers I've ever read in graduate school. It's also like I'm getting like teared up thinking about it now. It is also the most beautiful text I have ever read. Like when I read it the first time, I didn't understand it at all. And then when we went over it in class, the way the ocean waves that come over your soul when you start to piece together, these dudes were geniuses. Like they with sticks, with little sticks that you find out the side of the road, they were able to decode the connection between the DNA code and actual protein. Like as you said, this is an indisputable S. I would put this is again, it is my favorite paper. It was so important for me that after this, I went to my undergraduate lab instructor and I was like, could I do a special session for the students where I try to teach them this paper? Because I'm not going to remember this paper unless I try to teach it myself. And I would like to get some teaching experience. And if you want an afternoon off, I would be happy to come in. And they said yes. They let me come back to my undergraduate institution and give a lecture on this very paper. So this paper has a very special place in my heart. I love it so much. It is like the Euler equation, right? The mathematical ideal of beauty, where all of your mathematical constants happen to align in some funny kind of way. I get the same cosmic sense of holy cow, humans can be so clever and also so
But
complicated, right? Cells are so complicated, and humans figured that out. That's
and
so difficult. S
not only
tier.
that, but humans going against the grain too, because again, like by this time, like by the time they were doing this kind of research, like Watson and Crick had basically been deified, and so everything they said kind of like drove what research was going to get done or not. And they had lamented, "Oh, it's an unsolvable problem. I don't know." Again, this is me drastically oversimplifying. It wasn't just them. It was a lot of very smart people who tried to decode it using math and whatever. Like people did not think this was possible. It didn't make any sense because nobody thought about these molecules in this way. And these guys who's come in and are like, yo, what if it's 3D though? That's not the key insight, but like it is so transformational to our understanding. This is where the real magic starts coming in. So you're gonna progressively watch me and Fi get more and more excited because you're about to see some of the stuff that we were, that like was more discussed while we were maybe an undergrad and impressionable, and the thing that made us really fall in love with like the just sheer wonder that is both molecular biology itself and the science of understanding how it be doing that molecular biology works, if that makes sense.
Yeah,
Again,
I'm
I'm
abusing my
crushing our
power as
time.
the person who's actually this is my stream. I'm putting it at the tippy top of S.
I would
It's
I would I would
crazy.
literally make a double S tier. I don't know if that's even
Okay,
possible. I'll
no, no,
just
no.
put it
Let's
up
do it.
there.
Double S tier. Like
Double
I
S.
think
Okay.
categorically this one means more to me than the other papers do
Yeah, I
at
would
a
pay a
roll
lot of
above.
money for the Phi goes through this paper stream just because I remember um being assigned this paper. Here's a critical divergence in our educational journey, Phi. I remember being assigned this paper, and I remember skimming it and just kind of letting it bounce off of me, and I didn't like really come back to this science until way later. So like this is a huge undergraduate clockwork L. Um,
Oh well,
that's
you
all
read
I'm
it in
saying. Yeah,
undergraduate, dude. I read it in graduate school.
it was
So
assigned to me in undergraduate as like a bonus thing. Like if you know ball in molecular biology, like if you're a like a mole bio professor, you are assigning this in class basically. And I
mhm.
had one of those very passionate professors, and I was like, I don't know, man,
It took
it's
my
not for me
graduate
today.
student brain to figure it out. So I wouldn't beat yourself
Yeah,
up too bad. It was really
yeah.
hard to figure out, but when you do, it is the most beautiful thing in the world.
Yeah, but it's sad because it's like I was somebody opened the door to the promised land to me, and I was like, I don't want to walk through that. So yeah,
But
one of
I
those
think you
things.
need help, right? I couldn't do it alone. When I
Yeah,
read the paper for
yeah.
class, dude, I bombed. When the professor
Oh,
started to help decode it for me, it was only then when I started to put it
oh.
together in my brain, right? Because when I read this myself, I was like, there's no way, dude, this paper's so freaking complicated. But and this is why, again, this is something earlier from stream. If you want to do higher level biology, you need an environment where people can challenge you and help you and tutor you and mentor you. You can't do it just from books. It's too hard. Um,
Not even just from papers too, by the way,
yeah.
audience. Like, it's one of those things where this is one of those places where, since molecular biology is so young, like remember, it's we've been doing this for 70 years, right? But that's still a very, very small time period in the history of science.
Mhm.
Um, like the Socratic method kind of does work because there's just so much noise to guide yourself through, even especially now. Like I don't even know how you study bioinformatics without like, a whole council of like graduate advisors because there's just so much information now to understand on your own. So,
Right, yeah. Okay, we
no
do
need to
have to
vamp
move
about
on
that.
though for our
Yeah, I
love of
get you. I
this,
get
but
you.
it deserves all the time. But we do have to move on. This
All
will be
right.
the last one you read, and then I'll take over from here. So we can split
Sick.
the load.
Awesome. Sick. But yeah, so 1969, we're talking about bacteriophages. So bacteriophages are viruses that attach themselves to bacteria. Go figure. And they infect bacteria by emptying their genetic material into them, which leads to the rapid spawning of new phages inside the bacteria. Phage, not phages. That's a grammatical L on my part. But and by applying genetic concepts and developing statistical approaches in their studies of bacteriophages, Max Delbruck, Salvador Luria, and Alfred Hershey, like Pennsylvania Hershey, were able to shed new light on a range of unanswered questions within genetics. For example, in 1943, Delbruck and Luria proved through statistical investigations that bacteria, like more complex organisms, develop via mutations. So there's like a lot of little things in here. It's a very, very complicated Nobel Prize because it's actually covering a lot of ground, right?
Yeah. I'll say I know we're filling up the S tier, but phage were really important in genetics.
And
They're really important, dude.
still kind of, and they really genuinely still are. Very much yes, especially just yes, pure yes, no more context.
Like, yeah, just one more sentence about this. Before the age of current technology, phage were one of the few things that you could actually use to study genetics. Like, this is a tool and a discovery, right? And this is one of the sticks that they use to probe how genetics worked. It's very, very important. S tier,
Yeah,
it's got to be.
because again, like we knew that there was a code, we knew how it moved around, but there's like a lot of like not there's a lot of blank spaces that get filled in across the back half of the 20th century once we have those foundational discoveries, and they're all critically important for how we understand like the puzzle that is how life be doing what it do.
Yeah.
But we got to keep going though. So this is a big audience is a big Phi lore moment. We're going to see where Phi puts this one.
Yeah,
Because
so
as a
we're
neurobiologist, you're gonna read this one.
Yeah, I'm
too.
going to read this one. So this is my turn. So this goes to Sir Bernard Katz, Ulf von Euler, and Julius Axelrod for motor signaling. The nervous systems of people and animals consist of many nerve cells with long extensions or nerve fibers. Signals are conveyed between the cells by small electrical currents and by special substances known as signal substances. The transfer occurs via contacts or synapses. In the 1950s, Bernard Katz studied how impulses in motor neurons activate muscular activity by measuring variations in electrical charges. For example, he showed how signal substances acetylcholine in synapses are released in certain amounts. Controversial, you might not expect this coming from a neurobiologist. A B. This is a B tier. I don't really care about the peripheral nervous system. I'm a brain kind of guy.
See, that's why it's fascinating, because it helps us elucidate sort of the picture of the Phi lore.
Yeah,
I'll
I have
take
very
that B.
particular taste. So where would you put this?
I was gonna like, for me, it's just like, we're gonna get a lot more interesting molecular neuron stuff later on. So I want to make sure we save it. I would have given it A for acetylcholine because they did talk a lot about acetylcholine in this discovery. But I'm cool with B tier just
Yeah,
because
let's
it's
go
like one of
B.
those things where it's foundational, but then it doesn't get eclipsed. But there's way cooler stuff later on.
Yeah,
So
it
why are we
doesn't
talking about it?
excite me. I acknowledge it's pretty cool.
This is one of those papers that genuinely reveals the weakness of the tier list system, because it is a bit of a slap in the face to put this in B tier. But somebody's got to be there.
I'm even
Someone's
sure.
gotta get the
Like,
insult.
dude, it's our taste. I'm not saying it's not important. I just don't find it very exciting because I'm more of a central nervous system kind of guy.
And I'm more like, I thought those were the same kind of guys. They all have they have very similar molecules at the end of the day.
Yeah, yeah. But I like motor transduction. It's all right.
It has its moments.
It's had its moments.
For me, equally incomprehensible. Just neural circuits.
Hit me up with the hippocampus or the cortex any day before anything to do with motor neurons. That's my jam. Okay.
Something Purkinje cells. Let's go.
1971, Earl W. Sutherland Jr. for hormones. Signals between different parts of the body are conveyed by small electrical impulses and by chemical substances, hormones, and signal substances. Communication also takes place between different cell parts. Earl Sutherland investigated how hormones, especially adrenaline, work. He showed how signals from one cell to another are conveyed by the messenger, the hormone, and how signals within the cell are then conveyed by other messengers. Around 1960, he showed how cyclic adenosine monophosphate CAMP serves as the secondary messenger within the cell. This is complicated for me. I'm between B and C. Where are you with this, Clockwork?
Because of CAMP in particular. By the way, in your molecular shorthand, do you ever think of cyclic adenosine monophosphate as CAMP, or is it always just CAMP?
I say it CAMP in my head, yeah.
Okay. I heard one person say CAMP, and I thought I was going insane once. I just wanted to make sure that was just, like, it's CAMP. I just wanted to make super sure.
Anyway, I think it's CAMP.
Yeah, first of all, adrenaline was a big part of this, and adenosine monophosphate too. So for me, just for the lyricism of it, A tier. And because of this discovery unlocked a lot of discoveries like within signaling, because of how important CAMP is in a lot of different signals.
The reason why I say B is because of CAMP. The adrenaline, don't really care about adrenaline. CAMP though, CAMP deserves a higher tier. So if you're A and I'm BC, I'm okay with letting this in B tier.
Okay, well, we'll let it into B then. We'll commit alliteration crimes. That's fine. That shouldn't be a criteria. I understand.
All right, 1972, antibody structure by. Can I give this DOI? I don't have a DOI on hand, but I'm sure you could Google what the paper is if you're talking about like the big Nobel Prize papers. Yeah, there's no way that I could get all the DOIs in time and also do the rest of the cancer prep.
No, exactly. Oh, you mean for this exact paper, like for me, or what do we?
Yeah, I don't know. But in terms of like, I have all the cancer papers in the channel description, but like the Nobel, I couldn't make that happen.
Well, for a lot of them, like it's really hard to find the papers.
It is pretty hard.
Yeah.
Toxo says, fully cyclic AMP in my head, even if CAMP. That is fair. I think the tier list of what I find to be acceptable here, CAMP number 1, cyclic AMP number 2, CAMP is wild style.
Yeah.
That is wild style. I'll post in the Discord if someone reminds me maybe on Tuesday, because I'm sure Sunday and Monday are going to be awful for me. But on Tuesday, if someone wants to remind me to try to find this paper, I can try to see it if I can. It's like the very important text. Okay, 1972 antibody structure. Gerald M. Edelman and Rodney R. Porter. Our immune system protects us from attacks by microorganisms and poisons in part to the formation of antibodies in the blood that neutralize foreign substances. Rodney Porter and Gerald Edelman separately researched the chemical composition of antibodies. Using various chemical methods, they split up antibodies into smaller parts and mapped their biological effects. In 1959, this allowed them to determine the structure of antibodies. Antibodies make up two smaller and two larger polypeptide chains that form a Y-shaped structure. Very classic for molecular biology. I want to start off this time. Clockwork, where do you put this? You're a structure kind of lad. Where do you put antibodies on this?
Here's the thing. We're going to get into other structure moments which are way, way more impactful. Knowing the structure of antibodies is huge, but this is like the general structure, and the money maker of antibodies is what's going on in those two short chains up top,
Yeah,
which
for
actually
sure.
do the binding. And those are actually very hard to determine, which is why there's now like half a dozen companies getting billions of dollars in funding because thanks to AlphaFold and other models, we're finally able to sort of like generate um, like actually bioeffective and synthesizable antibodies. Like it's super hard. So this is one of those things where like, yeah, it's cool, but your foundational science is only getting like locked in, say, 70 years later. So honestly, this is a BC for me.
This is
Like,
B for me. I think that's fair. It's interesting. Yeah, it's kind of cool. Antibodies are cool,
I,
period, just in
yeah,
general. But
period. Like again, as a mold bio person, like I this is incredible science, incredibly foundational.
yeah,
But if we have to do the tier list, then like
but
there's gonna be
taste
more
wise,
impactful.
doesn't taste
Exactly.
very good.
Yeah, it's literally vanilla. It's
It's
VT.
vanilla. Yeah. That's so true. Actually, it's vanilla. 1973. This is a bit of an oddball one. This one goes to Karl von Fritz, Konrad Lorenz, and Nicholas Tinbergen. I'm butchering everything, but everyone's from around the world. I'm not going to be able to say everybody's name. So, some animal and human patterns of behavior are innate. Examples of such behavioral patterns in animals can be seen in how they convey information to one another, how they behave when mating, and how they care for their young. Karl von Fritz, whatever, Konrad Lorenz, and Nicholas made pioneering contributions within ethology by studying animal behavior. Nicholas Tinbergen used dummies in his experiments. One of his discoveries at the end of the 1930s was that birds preferred to brood eggs with exaggerating markings in the form of size, spots, and color. One of these Nobel Prizes is not like the others. I wonder which
It's
that
like
one could
it's one of those
be.
things where like I guess is this the year they ran out of ideas? Like why are we not getting like there's so many amazing experiments that didn't get Nobel Prizes, like establishing how we know the ratio for DNA replication, for instance. Like no Nobel Prize there, but
I do
you give
feel
this one
bad
out.
because, like, where do you even put this discovery? Like, does it belong in psychology?
It's got to be psychology. It's almost a sociology
Like,
one
it
too, a
should be
little
its
bit
own
almost.
category. Like, animal behavior is kind of in an awkward middle spot between psychology and biology, where I think none of us wants to adopt animal behavior into our own category. So it's just kind of awkward. I do find it kind of cool, but as a molecular biologist, I just, I would never make a 5 video on that.
Yeah, where's the behavior molecule? Show me the
Yeah,
signal
yeah,
transduction.
exactly.
Yeah,
The
it's
the
like there's
signaling. That's what our slice is. So I'm gonna put this in C.
yeah, because we can't be mean and put it in F tier. I think
No,
this
no,
is
no. I don't think
this
it's
this
bad.
this this one argues for the existence of D tier because it's like I am less than not interested in this one actually, but it's not actively bad.
It's not bad. It's just kind of weird. Amongst all of this very classic molecular biology Nobel Prizes, we get like a weird hybrid psychology biology paper in the form of animal behavior, which again, I don't I'm not saying it's bad. I'm not saying it's bad science. It's just we're talking about the code for life, and then we get birds and
Birds be doing things, yes.
yeah, it's a little
No, I
different. It's
mean,
just not for me. That's all.
but again, this is if you are just a pure experimental design sicko, this might be a B or A, just because like how do you even make coherent data out of such a fuzzy
That is
subset? So
fair.
that's where the prize comes from. Like it's
Yeah.
actually very, very cool that like, hey, I figured out a way to like quantify animal
Behavior.
behaviors
Hmm.
without like, the sort of like data tools we have now, which is cool. It's also kind of established statistics as a thing in certain accents of biological science. But again, not going to be too defensive here. C makes sense,
Yep.
especially since it's a molecular biology stream.
Mhm.
However, if you are somebody who cares deeply about the legacy of these scientists, and we have you're just here listening to us just completely destroy this legacy. I really appreciate your patience, and I am so sorry. Oh my God, I am so sorry.
We're not
Especially at the very
behavioralists, right? This is not
yeah,
our thing. I think people will
and
give us a pass. We're
it's
molecular guys. Like, come on.
it's also here for contrast because we are about to spend the next like 7 prizes going ballistic.
Yeah, these next ones are
It's
spicy.
it's 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 a very big it's oops all S rank decade. What a great time!
S
Imagine being in college during this.
So for
I
RBJAR, what's the ranking metric? It's how much we like these. Okay, it's a Nobel
yeah,
Prize tier list. They're all important. I want to emphasize
deeply
this is
important.
all amazing science, but this is what we find to be exciting and fun.
Yes, exactly. Although there is a reason why we started in the 50s and not even a moment earlier, because there is some genuinely weird stuff.
Yeah, there's some weird stuff before the 50s, let me tell you.
Folks, did you
Okay.
know that the guy who invented the lobotomy
Oh,
got a
yeah.
Nobel Prize for
Lobotomy
the
gets a Nobel
lobotomy?
Prize.
And not Dr. Rosalind
A
Franklin?
lobotomy guy gets one and not and Dr. Rosalind Franklin. Wild.
Forgive me, I'm
Okay.
saying.
1974, Albert Claude, Christian de Duve, George E. Pollard, whatever, their names are hard. They got this for microscopy and organelles. So our bodies are made up of cells that contain organelles, components with various functions. Around 1945, Albert Claude conducted a number of trailblazing studies of cellular components. He made use of the newly developed electron microscope, very fancy, which enabled him to capture images with a level of detail not previously available. Claude also developed methods for separating the various parts of pulverized cells with a centrifuge so that they could be better studied. This also became a breakthrough for cell biology. S tier, do you have any questions, Clockwork? Any disagreements?
It's S tier.
It's
Also,
S
just
tier.
like a bit of clockwork lore. This is about the time where my mom, who ended up being a lab tech, started getting like into biology, and she like saved photocopies of some of these electron microscope things. And so when she was like trying to explain like what she was interested in, she would show me these pictures and be like, "Look, there's like little guys inside the little guys, little clockwork." And I was like, "Dang!" So this literal this person's actual work is like foundational for like my interest in molecular
And
biology,
again,
but
like
whatever.
S tier is for topics that when we think about them, we get a little emotional. So it makes sense. You have
Yeah,
a personal connection through your mom to
yeah,
this particular topic. I find that beautiful because again, we're people, right? We're not tastemakers, right? We're not like you should also agree with us, but it is a chance for us to talk directly to you, the audience, a little bit about who we are and why we find this to be compelling.
there should
So
be
you
a
could totally
double S
disagree.
double S tier, and it just should the title should be mitochondria is the powerhouse of the cell.
It's so true.
Anyway, but yeah, let's not
Okay.
get too obnoxious with it.
1975, reverse transcriptase. Oh, okay.
Okay, here we go.
David Baltimore,
It's
Renato Dilbecco, Howard M. Temin for reverse transcription. In 1910, Peyton Roux demonstrated that certain viruses can cause cancer, although it would be some time before the processes behind this were understood. Renato Dilbecco and his colleagues proved during the 1950s that genetic materials from the virus are incorporated into the host organism's genes. The genes of the infected cells, which were partly constructed by the virus, could cause abnormal growth within the organism. Dilbecco's studies concentrated on tumor viruses with DNA as the genetic material, but ultimately also proved to be fundamental to the discovery of related to viruses with RNA as the genetic material, aka reverse transcription, the process by which a virus infection causes your very DNA to change because that virus has injected its genome into you. S tier, right? Are we
because
happy
not
with
only
S
is it
tier?
S tier because like wow, viruses are evil, but also like we're gonna steal that and we're gonna use it for our own nefarious purposes later on. More on that later.
Recombinant science. I think
Yeah, let's
that's,
go.
and this one, I don't even need to read this next one, y'all. Any questions? S tier. Any questions? Prions,
Just the
dude.
fact, just the fact that you were like you're able to like isolate prions in again the mid 20th century, crazy. So it's cool that we know about it. But we don't, we figure out what prions are, dude.
That's nuts. Imagine
Use
like you
rec-
thought you had everything figured out. Funguses can cause disease, viruses, bacteria. Oh, surely there can't be another thing. Proteins? What the heck? Amazing.
like not just proteins, but like the laws of physics themselves are
Yeah.
against you basically with prion disease because it's just like eventually like the structure can just collapse. Dang, dude.
I
Anyway,
love
we can talk about it
that.
all day.
I know. And I have in my video on prions, which if you're watching this, you've probably watched it. You know how deeply I love the topic of prions.
Don't let Phi fool you, audience. Do not let Phi fool you. He is not just talking about prions in the
Thank
prions
you,
video.
Toxo.
Secretly, every disease is a prion disease if you look hard
Secretly.
enough in the Phi videos.
Yeah. And
And like
my
I'm
collab with
doing a
Clockwork
collab
in the
with
flu.
yeah. Yeah, I was like, wait a minute,
Mhm.
the flu is a prion disease? No, like but actually, it's more of like It's funny how that all worked out because doing my video on allergies made me realize I kind of got a little bit wrong the structure of how the flu messes up mitochondria. But that's another thing. It makes a pore. It doesn't blow up the membrane. It just pokes
Mm.
holes in it anyway. Whatever. So
Prions or
we can talk about
prions depends on what country you're from. They're both fine.
yeah, and if you're like me and you grew up in the South and are currently in the Mid-Atlantic and you just have a completely broken accent, you just mispronounce everything and you just kind of live with it. Don't ask me how I say the word monks. It's awful.
I love that. Oh, we got a $25 donation from VSAN. Thank you so much. Oh, that looks like a joke, and then I'm going to have to tell you at the end of stream because they redeemed the joke. But thank you so much for the donation, VSAN. Thank you so much. Yeah, prions, I'm not even going to read it. You all know prions are great. To save time, let's just move on to 1977 with Roger Guillemin, Andrew V. Shaley for peptide hormones. Another hormone Nobel Prize. Hormones are substances that convey signals between the different parts of the body and regulate their functions. Around the pituitary in the brain, releasing hormones serve to release other hormones and are formed around the pituitary gland. The releasing hormones occur in very small amounts, from large quantities of pig brains and lamb brains, respectively. Roger Guillemin and Andrew Shaley extracted a sufficient amount of releasing hormone to determine its structure in 1969. They subsequently were able to produce it with chemical methods. Not interested. This is a C tier for me. Any comments?
For me, it's contrasted hormones with the next one, honestly. But yeah, it's one of those things where I would give it a B because it does help us learn a lot more about cell signaling later on. But let's
okay, I'll be generous. You can drag it up to B. That's fine with me.
Awesome. However, this is where it gets maybe a little controversial. This is where you have to realize I was raised by a lab tech. The next one has to be A tier because it's a killer protocol. Again, it seems very esoteric, but we learned so much based off of building this protocol.
Okay, I'll read this through because I didn't read these. I just copied these, so there are some that I don't know a lot about, and I see a word in this paragraph that I really love right now.
Yes, and you like this too because you know this protocol. I think I feel like you might have actually done this lab work.
No, anyway. I didn't do this myself, but I have read papers where this was the thing. So, without further ado, Rosalind Yalow won the Nobel Prize here for work for diabetes. So Rosalind Yalow was a nuclear physicist. She developed radioimmunoassay, RIA, together with the Dr. Solomon Berson. So RIA is used to measure small concentrations of substances in the body, such as hormones in the blood. Yalow and Berson tracked insulin by injecting radioactive iodine into the patient's blood. Because the method is so precise, they were able to prove that type 2 diabetes is caused by the body's inefficient use of insulin. Previously, it was thought that this disease was caused by a lack of insulin. So this is a story that does get told in my canonical education, at least. And radio labeling, dude, chef's kiss. Chef's kiss, dude. Do you want to nerd out about radio labeling, considering you have more of a personal connection with this?
No, that's basically it, right? Like, I mean, the description writes itself because it's one of those things where you can get so much information once you build this tool, and that's what what Rosalind Yalow did here was make a protocol so that it's eventually a reproducible thing that you can do for other kinds of hormone tagging. So first of all, we were going completely in the wrong way trying to solve type 2 diabetes back in the day, and having this information let us understand like the actual thing that's going on in type 2 diabetes, which is gigantic. And then more importantly, we now have a really sick lab protocol we can use to track all sorts of little guys throughout the body. And so instead of this like opaque, like impossible to figure out thing, which is human metabolism, all of a sudden everything's on the table. And we just need to chip away at every single hormone for 200 years, and we will have a complete understanding of how it is to be alive. So for me, it's all about the lab stuff. Like you can't get the science unless you have the protocol in place. But again, that's my bias. I was basically raised in a histocompatibility lab. Like if
we got to put that in A tier. And also in terms of like personal connection, I have a family history of diabetes. It's probably coming for me at some point.
I mean, not anymore with the advancements in small molecule GLPs. You
That's
might
true. GLPs, I
get
might
the
I might be on them at some point.
might get that preventative GLP situation. Let's
But
go.
as a kid, I remember like thinking I was going to be a doctor and being like, I was like 12 at the time and thinking, my dad has diabetes. It would be cool if I grew up to be a part of the cure for that one day. So,
Yeah,
so for little phi, I think I agree with putting this in A as well. Not just for radio labeling, but diabetes is an interesting condition as well. Okay, next
yeah, I
up,
get you.
ooh, we got another fun one. 1978, Werner Arbor, Daniel Nathans, Hamilton O. Smith for restriction enzymes. So, an organism's genome is stored in the form of long rows of building blocks known as nucleotides, which form DNA molecules. In the late 1960s, Werner Arbor discovered substances known as restriction enzymes, which cut DNA molecules at sites where a certain sequence of nucleotides occur. You can think of them as precise molecular scissors for your DNA. The discovery of restriction enzymes opened the way to explaining in which other genes exist on the in which order other genes exist on the chromosome, to investigating genes' chemical composition, and to putting DNA together in new combinations. A clockwork. To me, I gotta put restriction enzymes in S tier.
Exactly. And here's the thing: I
S
would only
tier.
put it in A tier because of all of the lore I received as a younger person, of all of the like folks in like the scientific community who are so mad about with restriction enzymes. This is like the first genetic engineering scare. Like people tried to ban the use of restriction enzymes for like changing DNA around, like in the late 70s, early 80s, and it like really messed with like MIT's dominance for a while there. But that's a whole other thing. So that's the only reason I do it. Just real quick lore drop, by the way. One other reason why the previous one for radio immunoassays gets a high level. The person who got this Nobel Prize, their student also found this component in Gia monster venom that became the foundation of GLP like science. Just a fun fact there. Like he used the technique to figure out that you could like mess with the GLP-1 receptor with Gia monster venom. Sorry, my bad. It's something I wanted to add in, but
No
didn't mean
problem.
to it.
Yeah, I
Didn't mean
mean,
to go back. Glad
again,
I did.
it's showing
We're
a piece of
super
our soul and our connection to biology, and I think
yeah.
it's very
So
valid. So restriction enzymes in S. I do want to address the peptide thing. As a biologist, the type of biologist that I am, I'm always more interested in curing disease than I am improving people's bodies. So exogenous peptide consumption is like not a thing that I care all that much about, which is fine. If you find it interesting, that's totally valid. But if you want to move that item for $10, I can move the peptide hormone stuff to whatever tier you want.
we finally figured out how to use the rage bait here.
Yeah,
So it's just having your own
yeah.
personal connection.
It's just I don't really care because I'm more focused on curing diseases because that's just my pet interests. But I acknowledge that they could be cool for somebody. It's just not for me. But yeah, for $10, I'll eat my words.
It's a measly $10. You can make me say anything for 10 whole
That's
dollars. Like
true.
I am
I'll
not
dance like a monkey up
scrupulous.
here.
Yeah, for
Uh,
real.
okay,
Anyway,
next one.
I wonder if this is
Oh,
controversial. I wonder if this is a divergent moment for us because I just want to see where as a molecular biologist you
stand
place
on
your
this
tool
next one.
set. Yeah.
Okay, 1979. Alan M. Cormack, Godfrey N. Hausenfield for the CAT scan or CT scan. Sorry. The discovery of X-rays and the possibility of obtaining images of the body's interior quickly led to medical applications. The possibilities of X-ray technology were further expanded with computed tomography (CT). If X-rays are sent through the body from different angles and registered when they have passed the body, images of different cross-sections are created through advanced computer calculations. Around 1957, Alan Cormack developed the necessary methods of calculation. In addition to cross-sections of the body, computed tomography also provides a basis for three-dimensional images. It's very valuable, of course. Medicine. You
You
love
can't study what
CT
you can't
scan.
see. You can't study what you can't see. This
It's
is the thing
true.
that made you see so many little things. I mean,
It's true.
actually, very large things. So it's one of those things where like becomes way better once we can add things that allow us to get down to the molecular level there. But I mean,
But I
it
don't
would
care.
try.
The thing is, I personally don't care. I put this in C tier. What about you?
C is for cat. I knew it. It hurts.
C
It
is
hurts me
for C is for
to
CT
let that
scan.
happen.
Do you
It's
agree?
it's it's an SA for me just because like you can't study what you can't see.
All right.
That's the
Okay.
thing.
You
But
can
this is
drag
the
it back
thing.
up to B if you feel that strongly. But for me, because it's not a molecular method, I just don't really care.
Don't I mean
Arguably.
some molecular studies rely on like stains inside CAT scans, right? Anyway, don't listen to me. Never mind.
I
Go
mean,
on.
maybe, but not the type of biology that I typically find myself looking at. Like, it's not a central toolkit in the papers that I find myself reading. I'll put it that way.
Also, there are some aspects of the science of tomography that eventually make cryo electron microscopy so cool.
I would
So
put
it's one
that
of
in
those
S
things where
tier.
like,
Cryo
yeah,
EM, I would
exactly.
put
Like,
in S tier.
yeah, exactly. We don't
CT,
get there without,
me, I don't care.
I get you, I get you. But that's physics, and that's also like let's just have the cryo EM paper go to S tier. Even though I don't think we're not gonna get
We're
to
not
that.
going to get to it
But
today.
either way, yeah.
Okay, next up. 1980. This is going to immune reactions. So this is going to be a nightmare. Barouge, Benna, Seraf, Jean Dowsett, George L. Snell for immune reactions. Our immune system rejects damaged or abnormal cells, allowing our body to function properly. During the 1960s, Barouge showed that through studies of guinea pigs that the immune system's reactions to certain substances is determined by genes that exist in certain areas in a certain chromosome. George Snell and Jean Dowsett had previously shown that genes that govern rejection of foreign cells exist in similar areas in mice and people. Ben Seref's results have shed light on the interplay amongst different parts of the immune system. You might think I would put this in A, but in S, but I don't feel the emotion coming from this. But I do want to put this in A. It's
I think
immunity.
it's a strong A.
I think it's a strong A.
Really yeah, this is where the science of immunology really starts taking off. Like, there's some really cool foundational stuff before this, but this is where it's like, oh yeah, now we're cooking. Like, this is way more foundational to the science of specifically, what is it, organ transplantation as well. So
Yeah.
I'm right there with you. Like, yeah, you gotta start like understanding where the impactful stuff comes in, and this is why we put an earlier immune situation much lower. This is why it's up. It's lower because like people stand on the shoulders of giants. Like this tier list should be very
Yeah.
1970s and 80s heavy.
I do want to point out we've just been raided by Nep. Thank you so much for the raid, Nep, all the way over from Twitch. I learned taxonomy and I'm still alive. Thank you so much for bringing your audience to this stream. For those of you who are unfamiliar with me, I am Phi the Neutrophil, your white blood cell VTuber extraordinaire. I make a bunch of videos on the immune system and other biology. My friend here is Clockwork, another very talented molecular biology animator. Would you describe yourself as such, molecular biology animator?
Everything but the word talented, obviously.
Come on. Who does a 3D model better than Clockwork? Show of hands. Anybody know a better animator on YouTube? I don't.
The Wahi Institute does incredible work using their own engine that Blueberry has been developing for 30 years. He's an actual genius, and I wish he was my real dad. But that's neither here nor there.
But thank you for the VTheorem. Yes, Nep is a part of VTheorem, our resident horticulturalist. For the sake of the raid, to keep that tight.
The raid is a good thing. I'm sorry, I'm a YouTube main. Forgive me.
YouTube can raid, but it sucks on YouTube. So Nep had to bring her audience from Twitch, actually, which I super duper appreciate.
That's brutal.
So it's
that's
awesome.
You as a streamer on Twitch just press a button and people whoosh, just go over to wherever you send them.
YouTube sucks. There's a bunch of menus you have to open before you can even think about raiding somebody, and even when you get to that menu, you don't know who to raid. Because Twitch, you could do like, oh, who's gaming right now? I'm gonna raid into this gamer. In YouTube, it's like, you've been watching these channels. Do you want to raid into them? By the way, they don't live stream. It's stupid.
Anyway, forgive me for being so decrepit so beyond the stream parlance.
So what we are doing right now is we are doing a tier list on the Nobel Prizes for physiology and medicine from the classic era of molecular biology all the way up to the recombinant revolution. And we are more than halfway done. And we are now talking about the 1980 Nobel Prize for the immune reaction.
20 years and 10 minutes.
For immune
Let's
reactions.
go, team.
Let's go.
Wait,
10
years, 10 minutes. We got this. Yeah.
We got this. Immune reactions A. I think that's fair.
Easy, yeah.
1981.
Point,
I talked about this on stream. Visual processing. Roger W. Oh no, wrong one. I skipped ahead. There are two different awards. One is for the cerebral hemispheres. Sorry, this is Roger W. Sperry. So both humans and animal brains have two halves with somewhat different functions. One way Roger Sperry studied these functions was by examining patients whose hemispheres connecting nerves has been severed to alleviate serious epilepsy. By the 1960s, Sperry could reveal that the left hemisphere is more geared towards abstract and analytical thought, calculation, and linguistic ability, while the right hemisphere is more important for comprehending spatial patterns. Complex sounds like music. Wasn't this debunked? Am I crazy?
It's not debunked. It's just that the science, evolved past this point. It's more complicated than left brain, right brain. Like there is some level of localization, but we don't have to get into that right now. This is an easy one. There's two C's in corpus callosum. We know where this one
Okay,
belongs.
C for corpus callosum. That's very fair. Even though I'm a brain guy, I don't really care
Yeah.
about this one.
There's
There's better
better
brain
brain
ones.
devices. And
Yeah.
audience, just to be over explaining here, the corpus callosum is the bundle of nerve fibers that connects the two hemispheres of the brain. And if you cut that, it can sometimes make epilepsy super more manageable, but it can also create competition between your two halves of your brain, where like you start doing things you don't realize you're doing.
Oh,
So
also, I just want to point out I have very bad rate etiquette because I live on YouTube. Uh, Thank you, Nick, for joining. And it looks like you just finished up a stream on Twitch to learn about taxonomy, which is very fun because, as I mentioned earlier, taxonomy is the bane of my existence. I hate taxonomy so much. So I do admire that you were able to spend some time with your audience to learn about a topic that I hate. But I think it's important. I think it's important. But I admire the fact that you can tackle that because I would not, you could not, I mean, maybe you could pay me enough to do taxonomy. I don't know. If you want to donate $100 right now, I'll do a taxonomy stream too. But um, so that's awesome. Great job on the taxonomy. Okay. Okay. 1981 visual processing. Oh, 9 minutes to do the rest of these. Hubel and Wiesel. We already did this on a stream. Visual processing. They used cats. Our vision works.
Purkinje cells. Put it in A tier and move on.
Burn in A2. Yeah, I'm not going to read too much into it. You can watch that stream where we talked about neurons and the optic nerve and the connection between the eyes and the brain. So cool. That's got to be A tier for me. Prostaglandin, 1982. This is for Soon K. Bergstrom, Bengt L. Samuelson, John R. Vane for prostaglandins. So prostaglandins are hormone-like substances that control several important processes in the body. They're also active when the body is attacked. In the 1950s, Soon Bergenstrom succeeded in producing pure prostaglandins and determining the chemical structures of two important examples, PGE and PGF. He showed that these are formed through the conversion of unsaturated fatty acids. Prostaglandins are used in medicines, for example, to trigger contractions during childbirth, induce abortions, or reduce the risk of gastric ulcers during treatment using other pharmaceuticals. I think I'm not a huge prostaglandin fan. I put this in C. What about you?
No one should be a prostaglandin defender. The correct thing is C tier. It's very important information, but a lot of the reasons that certain kinds of inflammation super suck are because of prostaglandins, and you can actually reduce prostaglandin sort of expression and synthesis with your various anti-inflammatories like acetaminophen and the other one, Tylenol. No, Motrin. My bad. I just want to say.
Yeah,
Anyway, yeah. So that's
I
the
think
thing.
prostaglandins to me are like a side character. Yeah, like a one
I
that you don't
mean,
really care about, but he's there.
they're there, and they're the reason that if you're experiencing an uncomfortable situation, they might be there making you more uncomfortable. So why would we honor that? Besides, we got a abs. We have one of the top banger papers. Let's go.
Let's freaking go. 1983 goes to Barbara McClintock. Let's go for mobile genetic elements. So this one,
Transposons,
oh,
folks.
this
And
one's
where did we
crazy.
find them? We found them in corn.
Many
If you
characteristics of
you can change the world
organisms.
if you look at corn hard enough.
Oh yeah,
Right?
the corn story is crazy.
Yeah.
Yeah, many characteristics of organisms are determined by heredity, that is, by their genes, which are stored in the chromosomes inside their cells' nuclei. Barbara McClintock studied corn's hereditary characteristics, for example, the different colors of its kernels. She studied how these characteristics are passed down through generations and linked to changes in the plant's chromosomes. Appropriate that Nep rated into this. During the 1940s and 1950s, McClintock proved that genetic elements can sometimes change position on a chromosome, and this causes nearby genes to become active or inactive. Oh my god! So much of your DNA right now sitting inside your chromosomes can move. They can just leave and come back into a random place, and that's crazy. Your cells are really good at preventing that from becoming cancer, but it happens to a lot more of your chromosome than you might think. Your chromosome is very flexible, and Barbara McClintock, the GOAT, she is a hero of science, right? If you, her story is insane. As a woman in science in the 80s, getting this Nobel Prize is incredibly meaningful to show that Darwin was right. Women can do science too. She's the GOAT, dude.
And this is also one of those papers, like if you're thinking about like being a scientist, where you start to see how biology becomes this science where just find every tool in the toolbox and just start like drawing connections, like make the crazy connections map, and you will find some brilliant things. Like Barbara McClintock knew she had all this information that she could rely on based on how mature the science of genetics with model organisms and botany had become, and she relied on that to help sort of like build this incredible picture. And we've used those methods to find other crazy things, like how, what is it, 40% of our DNA is viral retrotransposons, or
It's
at least
nuts.
modified
It's
viral
crazy.
retrotransposons. Like over the course of evolution, we have let some viral fellas stay with us, and they've been used for everything from building myelin to potentially like build the structures of how we store memories and stuff. It gets crazy.
And
Things move around.
it's
Thank God we have
nuts.
a spliceosome to keep things like under control.
And
Anyway,
there's a question, can we get a video sometime? Yes. At some point, yes. Do you think this belongs in triple S tier?
yeah, because it's Barbara
Triple S tier?
McClintock.
Yeah. Barbara
Barbara-
McClintock. Dude, the GOAT. Dude, she's the GOAT. I read this paper
Quadruple-
too. Right, by the way, for my education.
A tier above triple S that's quadruple B. It's Barbara McClintock.
It's Barbara McClintock, dude. You gotta.
I would say something here. Okay, clockwork lore drop again. My mom's a scientist who grew up during this era, so looked up to people. I would say something smart. Like if I said anything like remotely clever as a kid, my mom would say, "What? Who do you think you are, Barbara McClintock?" Like, just one of those things where like that would like held up as like a role model even in my younger years. So like I was amazed when Fi's like, "Yeah, Barbara McClintock." I was like, "You too, bro? No way."
Yeah, dude. Because I read her paper as a part of my canonical education, I had to work out what she did. And again, grad school was a while ago, so I don't remember the paper off the top of my head, but I remember just being like, oh my God, this is genius. And the
Yeah,
consequences, the science, the finding. Whereas prostaglandins, I'm like, meh, I mean, maybe somebody finds that cool. The fact that your genes can just jump, everybody has to find that cool. It is Phi law.
Like,
You have to find that cool.
Exactly. Not only that, because it's probably due to like maybe potentially competition between nuclear and mitochondrial DNA in the first like establishing moments of becoming eukaryotes. Like that's a whole thing
It's
too. Don't wanna get
awesome.
into it.
Oh, it's awesome. We don't have the time, but if I ever make a video, it'll
that
be
part
there.
of the science is very, very fresh and new, and it's almost irresponsible to make videos about stuff that's not fully understood. Just because like I don't want you to add the hot takes to like.
But you can just always append. This is the current research. Could be wrong in a couple of years. I'm not scared of covering new stuff as long as I quantify it.
No, I get you.
Okay,
All right,
next
we're
one.
almost there. 3 minutes left,
1984.
3 years left.
Niels K. Georgia, oh God, Kohler, Caesar Milstein for antibodies. The immune system includes cells, lymphocytes, and antibodies that neutralize substances foreign to the body, otherwise known as antigens. In 1955, Niels asserted that all kinds of antibodies already have developed during the fetus stage and that the immune system functions through selection. In 1971, he asserted that lymphocytes teach each other to recognize the body's own substances in the thymus gland. In 1974, network theory is based on the idea that antibodies not only attach themselves to an antigen, but can also become attached to other antibodies. An immunological reaction arises when an antigen disturbs the system's equilibrium. A tier?
A, easy, yeah. And
Easy A.
That's why the structure paper, who cares?
Yeah,
This is
structure,
our
who cares?
mechanism.
Mechanism, that's spicy.
I care. But it's only A tier because like there's still more to learn here on the antibody side of things. But
Yeah,
we'll get there
and
eventually.
like I understand as someone who's doing a lot of immunology content now that it is important, and I appreciate that, but it doesn't make me feel emotions the way that S and triple S do. Oh, another cholesterol paper. Okay, 1985, Michael Brown, Joseph Goldstein for cholesterol metabolism. Cholesterol plays an important role in several different biochemical processes. However, too much cholesterol in the blood can narrow blood vessels and cause problems. In 1973, Michael Brown and Joseph Goldstein discovered a receptor on the cell surface that takes up the LDL, particles in which cholesterol is encapsulated. They then clarified how the cell's own production of cholesterol is balanced, with the cholesterol supplied via the blood and influenced by diet. They also showed how genetic abnormalities affect regulation. C for cholesterol tier. Is that okay?
It's just that
Spicy?
the argument for A or B tier is that this research led to the development of statins as a drug class, which may be perhaps the most astoundingly impactful drug class
You're right. Statins.
in the history of the later S tiers for statins. No, you drag it down to A just to keep it normal. But again, I think I might be getting that wrong.
You know what?
I believe this is what led to statin research.
If this is statin research, I'm comfortable putting it with A. I'm probably going to need a statin at some point, so I can't complain. it can
I mean, everyone
elevate
is. I mean,
to A
audience,
tier.
statins, just so you know, are those drugs you see on the TV a lot that lower your cholesterol. They're very important because the Western diet is designed to kill you eventually. Hot take. That's not true, actually. It's just that eventually you just end up with too much cholesterol. And lowering it has made it so that not everyone dies of a heart attack at the age of 47. It's pretty cool.
Pretty
Lots
cool.
of lots of life expectancy. We got squeezed out of like a little bit of research.
Gotta be
A
fair. I
little
did not
bit,
see
yeah.
the statin connection, but I'm okay with elevating it. Okay,
I
next
think I
up.
might, but here's the thing: that statin connection might be fictional. I might be misremembering.
Okay,
And if
we'll
I am
mediate.
instant
We'll
F
mediate.
tier, and I have to do like a jail stream for you of
Jail
some
stream.
kind.
We'll put it in B. Kind of cool. Okay, 1986 for Stanley Cohen and Rita Levi Montalcini for growth factors. Human beings develop from a single cell that divides to form new cells. These new cells then also further divide and multiply. Little by little, different types of cells with different functions are formed. After Rita Levi Montalcini discovered a substance that promotes growth of the nervous system in the mid-1950s, Stanley Cohen discovered another growth factor that promotes growth of cells in the skins and cornea. The discovery of what are known now as growth factors have produced a deeper understanding of medical problems like deformites, senile dementia, delayed wound healing, and tumor diseases. Dev biology. This particular one for me is probably a B. What do you think?
It's only A tier because like the only like actual lab work I ever did was with VEGF, and this is close enough. So it's one of those things where like I have a that emotional connection, but I could
All
be
right,
argued
we'll
into B
give
just
we'll give you your emotional connection. We'll put it in A tier.
Awesome.
I think it's cool too, but there are cooler things in dev bio, in my opinion.
Shouts out to VEGF though.
Shout out to
Let's make
VTheorem.
some more. Let's make some more vasculature, folks. Anyway.
All right, 1987. Susumu Tonegawa for antibody diversity. The immune system includes antibodies that neutralize substances foreign to the body and microorganisms. Antibodies are formed in a type of white blood cell, B lymphocytes. Antibodies are proteins and their production is governed by genes. There are hundreds of millions of different antibodies, which vastly outnumber the combined total of human genes. In 1976, Susumu Tonegawa showed how this is possible through the redistribution of genes in a cell during its development into an antibody-producing B lymphocyte. VDJ combination. S tier? S tier?
Easy, yeah, because
Easy
it's
S
also,
tier.
this is where we really put immunology like on the map, right? This is some magic right here. So easy S tier.
Easy S tier.
Again,
Your cells are scrambling themselves. Their own DNA are being scrambled. That's insane.
there are more. You are able to produce more protein combinations than there are stars in the possible universe. Just so you can like make sure you fight anything that can come at you. Just the fact that we like go down to the base laws of math itself just to make sure that nobody weird comes and takes over our body is it still just hurts me to think about.
It's
Just, like,
amazing. Also,
yeah.
thank you for the donation, Comey. Thank you.
Yeah, it's also like hardcore spliceosome action as well, which
It's sick.
that's another thing.
It's sick. Your immune system got hands, dude. This is the immune system's got hands Nobel Prize. So
The
easy
immune
S
system
tier.
leaves nothing on the floor. Like it tries every single thing.
Okay. 1988, drug development for Sir James W. Black, Gertrude B. Ellison, George H. Hitchens. Many of the body's processes are controlled by substances known as hormones. These are absorbed by the cell receptors in the cell surface. The hormone adrenaline causes the heart to pump harder and for blood pressure to rise. At the beginning of the 1960s, James Black developed drug propanol, which is a beta blocker that has a calming effect on the heart by blocking the receptor for adrenaline. At the beginning of the 1970s, he discovered the drug, I'm not going to know how to pronounce it, cimetidine, that suppresses the formation of gastric acid and used to fight ulcers. It's so long. I'm not going to finish. But basically, there are a bunch of dudes that developed drugs, not just isolated it from nature, but actually developed them in an intelligent way.
And helped establish like the protocol that eventually became like the pharmacological revolution across the back half of the 20th century, which is
S tier, right?
kind of weak. Yeah, I mean this is S tier, but if you like want to be a real just like not nice person, you could give this a B tier and be like, yeah, we're better at it now though.
I mean, but they
Nice job,
started
old
it.
man, but yeah.
I know that's not like it was a while ago, but they came in with fire, dude. And Nev says, I worked under a plant growth hormone specialist regarding cold tolerance. That's awesome.
Yo.
That's sick.
Wait, how, were you working with Arabidopsis? Was it an Arabidopsis lab? Anyway, I'll ask later. Give me more Arabidopsis facts. I love Arabidopsis. Anyway, moving on.
Drug development, I think.
Arabidopsis is like the model organism in botany, by the way, folks. If you do any molecular biology, just
It's
look up
probably
Arabidopsis
Arabidopsis, yeah.
All the genes are there. Like,
It might not be, but
awesome.
it comes up so often. Arabidopsis is great.
Yeah.
Okay, 1989, J. Michael Bishop and Harold E. Varmus for oncogenes. The growth, division, and death of living cells are regulated by their genes. If these functions are out of balance, tumors can form. One reason for this may be the incorporation of virus genes into the genes of host cells. In the mid 1970s, Michael Bishop and Harold Varmus discovered virus genes that can cause cancer. However, they found that these so-called oncogenes did not originally come from the virus, but from normal cells. That had these been incorporated into the virus, cancer can thereby occur through the activation of the organism's own genes through a mutation, for example. I mean, this is, I gotta put this in S tier, right? It's the oncogene.
Exactly.
It's the whole, we're doing a cancer stream, 24-hour cancer stream. You gotta put the cancer Nobel Prizes in the S tier. I think that's fair.
Yeah, not even a question. S tier. I mean, it leads to some blind alleys and some understandings of how cancer works, at least in like sort of like the popular imagination of cancer. But in terms of like establishing like the entire toolkit that oncologists are working with today, this is foundational. Like you gotta have it.
All right.
Oh my god! Now
Your
everyone's
bonus one,
understanding.
Clockwork. You have
Do we even have time? Do we have actual time for this hot take? Because we're jumping.
I
30
think
years
it's worth
ahead.
it. I think it's worth it. Clockwork
Okay.
specifically asked if he could put 2005 on this list. And I said sure.
I thought we were going to go way further into the 2000s though. So it's one of those things where I have a hot take. I have heard so much talk about this particular science, but at the same time, I have to do something awful. I have to disparage one of my favorite places in the world, and that's Western Australia. All right, so it's 2005, and two scientists at the Royal Perth Hospital, great hospital, and the University of Western Australia, not the best university in Western Australia. It's fine. It's not the one my wife went to though, so it's inferior. Even though it literally is a world-class research institution, it is the better one. Curtin is a liberal arts university, and UWA is like a proper university. Anyway, so here's the thing: these guys are trying to figure out what causes gastritis because nobody could figure it out. And if you get gastritis, which is like swelling of the inner lining of your stomach, first of all, that is not fun. And then you'll get stomach cancer afterwards, probably. And so they thought maybe there was like a pathogen that caused it. And they kind of isolated a bunch and they found this one, H. pylori, which is a kind of wacky bacteria that's an extremophile, I believe. So it makes sense it survives in the acidic environment of your stomach. But here's the thing. Here's why I have a hot take about this because they tried to make the responsible thing of making model organisms and like models to work, and they just couldn't get H. pylori to colonize anything in a model context. And so the one of the people on the team, Barry Walters, Olbaza from UW, he's just like, you know what? I'm gonna get I'm in this hospital anyway. He gets an endoscopy to make sure like his stomach is good, and then he just drinks a vial of H. pylori in broth and like 3 days later instantly gets gastritis, totally wrecks his stuff, and got an ulcer, got gastritis, and proved that effectively that H. pylori causes stomach ulcers and is potentially what also causes this kinds of cancer. And then like now we treat gastritis with antibiotics and it goes away. Neat, fun, awesome. But at the same time, like it's talked about in the popular imagination as like, yeah, if there's a problem, just do the experiment on yourself first. And that's just like in the era of like biotechnology, it's just a terrible thing for the popular imagination. So F tier, don't give yourself gastritis, even though
Don't
you're in a
drink your
hospital
lab material unless your lab material is food.
even though DMEB looks delicious.
DMM, the forbidden Gatorade.
It looks so yummy, despite the carcinogens.
Long
But
time
again,
fans of Fi the Neutrophil, viewers of Fi the Neutrophil, know how much I detest self-experimentation. Yes, it worked out this one time, but you are encouraging people to be lazy. Because you can also imagine, yeah, they tried it, they failed. You know what most scientists do? They try again. Why take this shortcut of drinking this, right? This is a skill issue.
why did this get a Nobel Prize? And again, not Dr. Rosalind Franklin. You're giving these guys, these bogans,
The bogan.
like, how dare you? How dare you?
That's
By
so
the way,
funny.
go Birds. Go West Coast Eagles, the greatest Australian football team known to mankind. And if Brady Johnston is watching, I didn't say any of this. I am unequivocally, everything I say about WA is pro WA. This is clearly the best Nobel Prize ever given. WA, all day, all the way. West Coast, best coast. Westralia, just be independent.
Westralia, let's go.
You would literally rule the east, that entire hemisphere with all your iron reserves.
All right.
Just saying.
Before we end stream, I do have a joke to tell you because there was an incentive. Gosh, go. You can vamp more about H. pylori if you want, but I'm gonna be a little quiet as I try to struggle to come up with a biology joke that relates to you and your channel. So go ahead, and if you have the time, just
So the other part
vamp a
of like
bit.
doing this as a concept is like eventually, you get to, if we had gotten to the later years, we're going to see that like eventually for whatever reason the Nobel Prize in chemistry kind of takes over from molecular biology a little bit too. I believe that CRISPR was a chemistry Nobel Prize and not physiology and medicine, which to me is wild. So we would have missed like all of the cool stuff. We also missed oh no, because the way we stopped, we were about to get to RNA interference, which is another really huge one. So that's one of the weaknesses as well in this as a concept.
Okay, I have my joke.
Good.
Okay, this one's for you, Clockwork. What is the mitochondria's favorite genre of music? Again, I'm not promising that any of these jokes are good, but you've redeemed it, so I'll deliver it.
You got to give me an actual shot here. Okay, I got this. All right. So genres of music that have to do with respiration or powerhouse-based puns. Is it just power rock? No. Is that even a genre of music? No, I just made that up. No, hit me with it. I got nothing.
All right. A mitochondria's favorite genre of music is ATP synth.
I'm in physical pain.
What would you rate that out of 10? ATP synth.
This is a pain that goes beyond the concept of numbers. This belongs in the basement. This is below zero.
God, that's pretty good.
This is straight to jail out of.
Come on.
I mean,
that's pretty good. That's pretty good. We can. Okay, you're gonna do one for free?
No, here's how does a mitochondria say hello? No, this doesn't make any sense.
It's harder than you think, right?
Say hello, an ATP synth wave, which is actually a genre of music, but that doesn't make any sense.
That's pretty funny too.
you have to like make like a what is it? Mitochondria's favorite music / gesture, but it doesn't make any sense.
You liked it? Fine. Thank you, Toxo. Thank you.
That's good.
Thank you.
And again, it's just because like I think I just hate jokes as a concept. Like I love being like I'm supposed like you're telling me you're gonna surprise me with something before I have to be surprised by it. It's like no, that's like a very stressful circumstance.
Anxiety.
Yeah. So no, it's one of those things where like I can see the value in it. Although is synth just the word synth by itself enough to be a genre of music? I think that's where I get hung up on the entry of
yeah, what fair. Okay, Clockwork. It is the end of our collab. Thank you so much for spending some late hours over here on the Fly the Neutral channel to join me here.
There. Exactly.
More than halfway there.
Dead. Catch you at Sad Boy Hours if things.
Sad Boy Hours. Let's go.
And good for you.
Do you have anything to plug before you go? By the way, any exciting upcoming projects that you're excited to share?
So, I mean, nothing like directly. I mean, just check out, youtube.com/clockworkbio to see
link in the description.
Brand of animation. You can also just find the collabs I have done with Phi. Hopefully, my next video comes out next week, which is
very exciting.
Insane. Phi has actually gotten a top secret preview of
it. I got to see it before.
I went mad with power, and I figured I read enough papers where I made a full atomic model of a muscle cell, of the fundamental unit of a muscle cell. There's enough science that we can fully model 1-to-1 atomic level how muscles do be muscling. So
dude, the render that Clockwork's got going on for the muscle fiber is
actually not even that computationally stressful. It looks way more intense than it is because computers can.
It looks insane.
A bunch, yeah. So it's very
like don't give away the sauce. Say you worked really hard on it and people will click on it.
Slayed the Spire.
You slayed the Spire. Let's go.
Actually, I haven't played that game. I don't even know if I can make that reference.
No, I approve. As someone with over 1,000 hours in this game, I approve.
Yeah, I need to really get into the deck building genre. Either way, audience, thank you so much, Phi. Thanks for having me.
Thanks for coming, Clockwork.
Bye. Go solve it. Peace.
Bye. Okay. What's a quick summary of the stream roadmap? From here on out, we're going to be talking. I think the next topic is going to be new ways to study cancer, and also innovative treatments to cancer. I think that's the next topic. No, this is one more basic biology paper. Oh, this paper's so good, dude. This paper's so good. Yeah, so I think this is the last of the basic biology papers, and then we're going to get into how can we study cancer better, and then the rest of the twilight hours will be dedicated to innovative treatments for cancer. So went a little over time with the Nobel Prize stream. That's okay. I think it was worth it. And now we can get back into the science with, oh, this paper is sick, dude. Okay, title: Sensory nerves enhance triple negative breast cancer invasion and metastasis via the axon guidance molecule plexin B3. Chat, did you know, were you aware that tumors are capable of reprogramming nerves for their own nefarious reasons? What the heck does a tumor even want to do with a nerve, right? Angiogenesis makes sense. When a tumor reroutes the blood vessels to feed itself, that makes sense. Cancer wants to grow. It needs those nutrients. What the heck are tumors doing reprogramming neurons? And, of course, we just said in the Nobel Prize stream, I said multiple times, I'm a neurobiologist. Neurons are very, very close and near and dear to my heart. What's cancer got to do with them? What part of the breast cancer story do neurons play? And that's what we're getting into here. Reprogramming nerves, that's terrifying? Exactly. It's terrifying. It's terrifying. The tumor yearns for dopamine hits. I mean, you're not that far off. You're not that far off. So, what the heck are neurons doing here? So I've been obsessed with the idea of nerves driving cancer progression for a while. Unfortunately, most of these papers are copyright blocked, which is, oh, I'm so mad, okay? Some of these papers I don't think I could actually show, but I enjoyed reading through them. Hijacking of the nervous system in cancer, mechanism and therapeutic targets, tumor infiltrating nerves, unraveling the role of cancer neuroscience in tumor genesis, disease progression, and emerging therapies, the role of the nervous system in cancer. Again, we already know that cancers can reroute blood. What's becoming more and more prevalent in our understanding of cancer is that cancer can also reroute neurons, and that disturbs me to a very deep degree. That's freaky. In a way, a neuron might be a tumor's best friend, actually. Neurons, to some degree, are immune privileged. I went over this in the rabies video. They have immune evasive protocols naturally built within them. And that's a good thing, right? You don't want your immune system to treat nerves the way that they treat the rest of your body. Because, if one epithelial cell in your gut dies, acceptable losses, another one will take its place. Neurons don't really grow back. There are certain areas of the brain where you can get neuronal regrowth, but they're not that frequent and they don't do it very well. So you want to really make sure that the immune system is very delicate around these neurons, which means that these are cells that are capable of telling the immune system to chill. And when you think about it, cancer would really love to tell the immune system to chill, because when we learned more than 14 hours ago, part of the reason why you're not stuffed to the brim with cancer right now is because your immune system is good at suppressing cancers. But again, neurons have the toolkit. They have the genetic toolkit. They have the genetic programs available to them to tell the immune system to quiet down. So this natural friendship between these cells, it seems very reasonable, right? It makes sense. Neurons are also signaling experts. They live to communicate, right? They talk to each other. That's what they're famous for. They talk not only to neurons, but they talk to support cells with muscle fibers, etc., etc., etc. They are built to talk to a wide variety of different cell types. And neurons are also capable of signaling growth factors as a part of their role in the development of the nervous system, right? Neurons can encourage other neurons to grow. They have that capability. So even though it didn't make sense to me before what the heck a cancer cell would ever want to do with a neuron, when you realize that neurons kind of have the exact items that a cancer would need to survive, then it becomes a lot more obvious why these two would establish a friendship. Yeah, exactly, Frost. Neurons have immune camouflage. Cancer would love to steal that from them. So I'm going to do a little bit of a background for this paper, and I'm pulling from this paper, Hijacking of the Nervous System in Cancer, Mechanism and Therapeutic Targets. So remember, cancer cells are not behaving like a normal cell would behave. In some ways, you can think of cancer as a cell that has lost its strict sense of identity. Because all somatic, that just means all the cells that aren't your sperm and eggs, have a full set of your DNA. They're technically capable of doing anything any other cell can do. So because cancer cells have this expanded universe of chemical tools, they can secrete something that they should not be able to secrete, which is bone-derived neurotrophic factor, or BDNF, and neuronal growth factor. So this is not supposed to be true. Neurons are supposed to be making these, and the support cells of the nervous system are supposed to be making these, but because cancer has this stem-like potential, they can pull this trick off as well. So this relationship is bidirectional. Neurons also love cancer because cancer cells give them the factors that they need to grow. They're not supposed to, but they can, and they do. So what does the cancer cell get? Neurons, in turn, are signaling cells that can encourage the growth of the tumor. Again, we already heard neurons are signaling experts, and they can encourage tumorous cell growth. This is usually done under very strict regulatory programs during specific stages of development, or highly localized to a specific subset of cells, right? Your brain grows at a certain time of development, and then it has a program to stop growing, stop making new connections, and then it starts to trim those connections. You are born with more neurons than you die with. And that is on purpose, right? You're born with more neurons than you need. You're born with more synapses than you need. And having the right balance of neurons and synapses in your brain is crucial for, we'll call it a common function, because, you know, there's neurodivergence, etc. I don't want to get into that whole discussion, but from the conventional view, this balance is critical to ensuring that your brain functions appropriately. I think that's a bit better, right? That you can function in this world. So these two get into a relationship with each other. They build each other up, and that is good for the neurons and good for the cancer, and that sucks for you. So a deadly codependent feedback loop then starts to happen. Innervation of tumors is associated with worse outcomes. So tumors that have captured more neurons are typically associated with those cancers being worse. Again, this is correlation. This is not limited to cancers of the nervous system, but many other types of cancer as well. And because this is becoming such an apparent part of cancer biology, this is giving rise to the rapidly progressing field of neuro-oncology and cancer neuroscience. If I had to redo my time in graduate school, I would probably study this, right? Even though I love the immune system, I just can't get over how cool neuron science is. And the only reason why I don't make more neuron videos is because there's a lot more background you have to have with neuroscience that isn't as easy to do for a video. So if you're a young student out there and your university happens to have a neuro-oncology lab, if you want to study something really interesting, that's what I would recommend. Can I have a quick overview? What is a neuron? A neuron is a cell of the nervous system. It propagates signals that generally originate from the brain. So it's basically the baseline cell that controls your thoughts, feelings, emotions, memories, movement, and a whole bunch of other things. Signaling to various organs are controlled by the brain through the action of neurons. So they are the coordinators of the body. So this is crazy, right? This is crazy. This finding blew my mind. And I think Pamela, many, many hours ago, had questioned methods of metastasis. It's not just through the blood. Cancer can metastasize through neurons. Let's take a moment to think about that while I drink. This is horrible. I talked about Fusobacterium a couple of papers ago. The bacterium that originates in your mouth, and when you have gum disease, can infiltrate the bloodstream and make its way to the breast. And that is very typical of metastasis, right? They hitch a ride through the blood and end up in places where they can't or should not end up. The brain has a blood-brain barrier. It's protected from the blood in certain ways. But one way that cancers can attack the brain is by crawling along neuronal tracks. How does that make you feel? It makes me feel bad. That's creepy. They can crawl along the neurons, these cells that are meant to control movement in your body and to control the actions of certain organs. cancer is literally crawling up those neurons. And it's terrible to think about and so fascinating at the same time. RB says similar to rabies. No, well, rabies goes through the neurons, into the neurons and out of the neurons. Cancer cells wrap themselves around the neurons and physically crawl along neuronal tracts. Do we even know it's crawling up there, or is it sending a print request up the neurons? I think they know that it crawls. And that's where we begin this paper. Sensory nerves enhance triple-negative breast cancer invasion and metastasis via the axon guidance molecule plexin B3. So, nervous infiltration of tumors is a very bad thing for breast cancer patients. We know this. Innervation of tumors is correlated with worse disease outcomes. The more nervous tissue is located in that breast, the worse your outcomes typically tend to be. Triple-negative breast cancers are often more heavily innervated than other types of breast cancer. So if it weren't bad enough, the triple-negative breast cancers that I've been talking about, the ones that are the worst case scenario for breast cancer, they happen to have more nervous innervation as well. Is that causative or correlative? We don't know at this point, but it is noteworthy. This research is relatively new. There are actually two types of neurons that can infiltrate tumor tissue: sensory neurons and sympathetic neurons. Sensory neurons are a specific type of neurons that provide a hookup from the extremities, your body, fingers, toes, etc. And they hook to the brain, allowing to actually receive information from those extremities, your fingers, senses, etc., etc. Sympathetic neurons are primarily for fight or flight responses. So these are things that don't directly connect to muscle, but they help activate things like the cortisol response, adrenaline, etc., etc. What is nervous infiltration? That just is referring to the neurons growing into the tumor, or the tumor growing around the neuron. I'm not entirely clear which way it goes. Sensory neurons outnumber sympathetic neurons in breast tissue, but the role they play in cancer progression is relatively understudied. So we know that both of these types of neurons are in breast tissue, but we actually know more about sympathetic neurons and how they interact with the tumor. We don't know a whole lot about sensory neurons. So this paper is focusing on sensory neurons. And I have a love-hate relationship with the nervous system. I know I've gone about how I love studying the nervous system, but boy, is it hard to do so. It is hard to study the nervous system. And why? Well, it's for a couple of reasons. And I just want to give you some context for my feelings on this matter. I was trained as a neurobiologist. Immunology is just a hobby of mine. Part of what makes studying the nervous system so difficult is the fact that neurons are just impossible. They do not mitose. They don't do mitosis. So what you get is what you get. And if you kill your cells by accident when you're working with them, too bad. A lot of cells don't act that way, but neurons do. So because these neurons are postmitotic, they can be a nightmare to actually study. Many studies, and this drives me crazy, there are a lot of neuroscience papers that work with what are called neuron-like cells. Cells that have been treated with certain growth factors to grow into something that looks like a neuron, smells like a neuron, but they're not neurons. These neuron-like cells don't really act like neurons. They don't do the same stuff that neurons do. They don't do action potentials and synaptic transmission, etc., etc. They just look like neurons on their surface. And something that I think is very good about this paper is that they're going to be using actual neurons, which I am so happy about, because if you don't use actual neurons, then you start to question, is whatever effect I'm seeing an effect of the weird cell type that I manipulated into thinking it's a neuron, or is it real biology? So I love this here. Okay. So that is the context. First, we have to examine, and I pull this up because I love microscopy, man. I love looking at these pictures. And they tell you a lot, and it's part of what makes biology so fun, is that you get to look at these pretty images and actually do some science. So on the left here, we have healthy breast tissue, and on the right, we have breast tumors. I'm sorry if you're green-red colorblind. A lot of biologists forget that people are like this. But in green, we have tubulin, which marks all neurons. In red, we have sensory neurons, and DAPI is blue. It's a DNA stain. This is every cell. in the tissue. And chat, I want you to describe to me what you are seeing. What is different about healthy breast tissue and tumor tissue? How much have you raised so far? The progress bar is at the very top of the screen. It looks like it's about, well, it's a little small on my end because OBS makes it a bit small. Where are we? $1,576. Our goal is 2,000. We are close. What do the arrows mean? They're pointing out specific interesting things about this that I want you to ignore for now. Just, what does the breast look like? A very basic, a cursory glance at this. And again, I am helping you practice the art of observation. So Tylenol says a lot more DNA stain. Yeah. And again, we've seen this a couple of times in these papers. It's not that unusual because tumor cells grow more. So it's expected that you see more blue when you're talking about a tumor versus a healthy tissue where growth is controlled. What else can you glean? What else? What else can you say is different about healthy breast tissue and tumor tissue? So Dolphin says, looks like all the cytoskeleton is going all over the place and cells must be dividing more. That's interesting. Sensory neuron marker is super tiny / condensed. Where? In what type of tissue? In healthy breast, it is clumped together, where in tumors, it is farther apart. Oh, interesting. Excellent observations. Neurocells are more distributed in tumor tissue. Yeah, they're a lot more spread out. I love this. These are great observations, chat. Great observations. TRPV1 more condensed in the tumor. Yeah, the sensory neurons appear to be condensed, tight, in a fiber of some kind. Tumor, Drunken Master says, tumor has a lot more neuron tissue that isn't organized. Yeah. Awesome job, guys. Could not have asked for better answers. And that is exactly what the scientists conclude. Sensory neurons look very well organized in healthy tissue, right? They form this little ring, right? Our marker for sensory neurons is red, and they form a structure. I don't know what the structure is, but you can tell that it has organization. It looks like it is serving some sort of function. In cancer, it's lost that organization. It's become this thread, this fiber for some reason. And that might be an important thing. It might not be. But as a scientist, you are looking to examine differences, and it's an important one to keep in mind. Great job, guys. Great job. So, there are signs that there are much more sensory neurons within tumors than in healthy breast tissue. Like, this is very concentrated and this is more diffuse. And so when you take a look and you measure the area in all the samples, what you actually find is different amounts of the marker for sensory neurons in different types of tissue. It looks like if you have a cancer cell, you actually find that there's a lot. It's hard to tell because don't worry too much about the brightness. Quantification in fluorescence microscopy is semi-quantitative, and you're not going to really be able to tell which has more, which has less. So this is a separate experiment determining how much TRPV1 per neuron there actually is. And in the normal healthy breast tissue, this is actually a relatively small part of the entire breast, right? It's basically not even there. But in cancer tissue, you see that TPRV1, the sensory neurons, there's a lot more. There's a lot more by area. Is TRPV1 bad? It just tells you that there are sensory neurons in the breast tissue. At this point, we don't know if it's bad. We're still trying to figure out if it's bad. And we can also see from real patient data, patients with high TRPV1 tend to make up more of the cancer patients, right? I'm going to ignore this. I don't want to think about this lower part right here. Just understand that it looks like there are signs that there are more sensory neurons within the tumors of cancer patients as opposed to regular healthy breast tissue. It's a lot higher in cancer by measurement of mRNA than healthy breast tissue. Okay, Tone warning here. The next piece of data is actually going to examine and show you the relationship between having more sensory neurons in your breast tissue and death. Whoa, Purkinje's going crazy. Hello, Purkinje. No, I'm not going to bed tonight. I'm sorry, Purkinje's here to whine at me. I'm not going to bed tonight. Bai's got work to do. Poor Purkinje. Breaking up the tone warning. So, prepare yourself. There are going to be deaths represented by the data that's coming on this next slide. Yeah, someone donate the $8,888 so I can go to bed with Purkinje, my lovely, lovely cat. Otherwise, I'm going to have to do another 9.5 hours of this. Okay, the next piece of data summarizes patient deaths. Okay, so what do we have going on here? So, this is the data, and this is easy to read, so I will let you do so. So, D focuses specifically on triple negative breast cancer, and all BC is just an aggregate of all breast cancers. And we have a survival curve, which is hard to look at because there are real people being represented in this data. And this is exactly where I broke last year when it came to glioblastoma, thinking about pediatric patients. But it's important. It's important to learn from this, okay? What do you see here? And let me explain things. TRPV1, this is sensory neuron marker. This tells you how much sensory neuron is in a certain tumor. All BC stands for all breast cancers. Low refers to a low amount of sensory marker, right? So in black is all the patients who don't have a lot of sensory neuron marker. In red are patients that do have a lot of sensory neuron marker. What do these graphs indicate about the effect of having high sensory neuron marker in breast tissue for cancer, specifically triple negative? What's going on here, chat? What is going on here? Oh, Purkinje is throwing a fit. Yeah, and I like the way that Tyler Null phrased it. More neuron marker correlates with a lower survival rate. Again, we don't know if this is causative yet, but that is the relationship here. So Adolphin says sensory neuron marker in triple negative breast cancer increases mortality. Again, I want to caution us. At this point, we don't know that, but it is correlated. And we're going to see if that is true later on in this paper. What about all breast cancers? That's kind of weird, isn't it? What's different about figure E versus figure D? What do you make of that? What do you make of that data? What is that data telling you? You are correct that the levels of sensory neuron marker is correlated with decreased survival or increased mortality in triple negative breast cancer. But what about all breast cancers? What about all breast cancers? Anybody have any insight here? Come on, help me out, chat. It's 11:40 where I am. I am so tired. uh, uh, I don't understand what the vertical bar in all BC means. The vertical bar, it just means that after a certain, because like you can imagine, as patients die, you have a lower pool, and I think this is maybe aggregate data. Like I think the drop off after 200 months, that might be a little funny. I would maybe ignore the drop off after 200 months, because it might be a flaw in whatever data they're using. That doesn't look like a real effect. Yeah, the balding and dolphin. Yeah, no apparent correlation. for some reason, and I don't know why, I don't think these researchers know why, but something weird about triple negative breast cancer is that for some reason, when sensory neurons are higher in triple negative breast cancer, that means the patient is worse off. But if you aggregate all breast cancers, it doesn't seem to matter. It doesn't make cancer worse in the aggregate breast cancer, just triple negative, and that's weird. Yeah, Mies, figure E is implying that correlation of death in sensory neurons is triple negative breast cancer specific. And that's bizarre. I mean, it says it's protective, but what they should have done is excluded triple negative in the plot on the right, and it's going to bias it downwards. That's true. I don't really know how much it would have biased it again, because it looks like all BCs is like more than 2000 and the triple negative is like 385. So it's possible, but I'm not gonna lose sleep about. I mean, I agree with you, SP. I agree that it would have been nice to see rest of breast cancer instead of all breast cancers. But what I will say is that the relationship is certainly different. Oof. Oh, but you did notice that the log rank P does indicate that that difference is still significant, even if it is very small. You're right. Yeah, they probably should have manipulated or treated the data a bit differently. I used to do research in this area. That's probably why you need to look at the log rank P. So thank you so much for that, because I'm far from an expert here. It just feels hard to know whether there's a type of common BC that is far from neurons that's messing with the graphs. Yeah, I wouldn't pay too much attention to figure E. It's just kind of weird. And sometimes you can get really invested in some of these weirder findings. But I don't think the authors dwell on this, so I'm not going to dwell on this. Okay. So, conclusion so far. Neurons have a history of helping cancers thrive. Oh, there's a good question. How does one even tell if a cancer is innervated? You can just look for the neurons in there. You can slice the tissue open and put on a stain that will mark sensory neurons in particular, because they make TV, PRN1, or whatever the marker is. So you can stain for that, or label it fluorescently. There's a whole bunch of different things that you can do in tissue. I think weird in graphs is often super useful. I've had 2 publications which explain a bunch of weird things that were ignored in other papers. That's true. For the tired science communicator in me, I'm not going to dwell too much on it. Kinji is going crazy. She is not happy that I'm not in bed. But yeah, I don't have the brain energy to inspect that any further, but I encourage you to, because it is interesting. So sorry, again, conclusions. Neurons have a history of helping cancers thrive and vice versa. We're not sure how sensory neurons innervating breast cancer contributes, but we do know that sensory neurons innervate breast tissue at a higher rate in triple negative breast cancer, and it causes higher mortality in those patients. Or not causes, but it's correlated with higher mortality. Patients with more sensory neuron marker in the breast tissue do not live as long. The question is this correlation or causation? So how are we going to study this? We are going to go to our tried and true friend, a mouse. This one is pretty simple. Inject a mouse under the mammary fat pad with neurons and breast cancer tissue. It's kind of the same approach we took a couple papers back, where we were inoculating a mouse with breast cancer tissue and fusobacteria, right? We want to check whether or not the presence of neurons is doing something. So we take real neurons, we take real breast cancer tissue, and we stuff it into the breast padding of the mouse. So for this experiment, they will be using dorsal root ganglion neurons. I don't know if that matters. I don't know enough neuroscience to know if that matters, but that's what they're using. And of course, since the effect that they found was in triple negative breast cancer, they're going to be using a triple negative breast cancer line. So what do you think of this experiment? Do you like this? Is this weird to you? Any red flags? What do you think of this, chat? No wrong answers, but I want to know how this hits your ear, actually. While I drink, because boy, my voice is tough. Oh. As a soft criticism for scientific language, inoculating a mouse with cancerous tissue sounds so avoidant compared to we gave a mouse cancer. It actually is purposeful, because what I am focusing here, because these are my words actually, what I am focusing here is the method of delivery. It is an inoculation, an injection. Because you can give a mouse cancer in a bunch of different ways, but this is specifically inoculating. This is a cancer that was grown outside of the mouse and then put inside the mouse. So Kangaroo says it could be different in a mouse than in a human. Yeah, there's always a risk of that in a mouse, but do you accept that this could yield something interesting? Is this a potentially good experiment to do? Potentially. Right, I'm encouraging a little bit of nuanced thought. So Kangaroo, you're absolutely right. This is a mouse, this is not a human. But I wanted to get a little bit of the thinky juices going. What do you think? So one thing I would wonder, if you actually are modeling natural in vivo cancer properly, the intracellular matrix matters a lot in triple negative breast cancer. And I would think doubly near cell neurons. And that is something that we are going to talk about. It might be the next paper about modeling the natural quote unquote environment of cancers and just stuffing cancer under the fat pad. Like, I'm not enough of a cancer biologist to know if that's valid, but you do bring up a good point. There are things that are kind of tricky about trusting this kind of data. So I'd say that is a fair, nuanced conversation, right? There are, I want to drive this point home for my audience repeatedly because no experiment is perfect. Everything's got to simplify something at some point. I think this is fine. Feel bad for the mouse, but what are you going to do? We have, if you believe that we should be focusing on human cancers, then sorry. So here's the data. And this is proof, or the authors here are going to try to argue that their system actually works. So they inoculated the mouse with cancerous tissue, and we are going to check if this actually worked. What is the effect here? Oh, thank you so much for the donation. I'm not going to be able to say your name. SP Spishkuros. Can I just call you Kairos? Thank you so much for the $25 donation. And thank you for not redeeming the joke. I'm very tired. But thank you so much. Thank you so much. And thank you for... it's nice to have people who have actually done this research so I don't propagate misinformation. So I'm glad you are keeping me honest. Are they using mouse cancer cells? I think this is a human cancer line. You can just Google 231 breast cancer to see if this is human. I believe this is human. That's a good question, though. That does make a difference. Okay, so beta-3 tubulin in red, this is a neuron marker. Blue is DAPI. This is all cells. 231 is the cancer line injection, and 231 plus DRG is the cancer plus neurons. So let's break this down. 231 only. This is just cancer. And we inject that into a mouse, and what do we find? Compare that to the next slide over on the right, where it's the neuron plus DRG. So what can you tell me about whether or not we have successful inoculation of neurons within these tumors? What is a neuron injection? What is the pink and red? It is a protein that only neurons make, essentially, for the purposes of this. DAPI is DNA, so all the cells make DNA. So blue is total cells, and then the red is neurons only. So looking at this data, would you say that we have successful inoculation of breast cancer tissue when you add in the neurons? Do you see, or I guess another way of asking this is, you injected cancer into a mouse and you injected a different mouse with breast cancer and neurons. Do we see evidence that there are more neurons in the breast cancer tissue that we put neurons in, essentially? Does this look like it worked? Yeah. So, what is chat saying? Yeah, it looks like there's some evidence, right? So Dispel says, for neurons, you should use stain with nu N2. The cells just have tubules. I agree. I think nu N I don't know why they didn't use new N. I don't know if new N is like weird in sensory neurons. This goes beyond my personal knowledge of neurobiology, to be honest. Looks like there's more in the inoculated lines. There's still some neural markers on the top left box there. Exactly right, Dolphin. So it looks like the normal breast cancer tissue when you inject them in the mouse, somehow a little bit of that tubulin gets in. And I think, yeah, other things make beta 3 tubulin too. I think new N, I think, would have made me feel a little more comfortable. But what we can say is that when you actually put the neuron in there, you do see more of it successfully in there. But I am kind of worried because this data makes me it's a little weird for me because there's a lot of signal that is basically zero. I mean, on average, this looks like it worked, but I wonder what the quality of this data actually are, where there are certain areas that don't have any. It could be because by percentage, sensory neurons are never all that prevalent in the tumor, and maybe the difference that we care about is just the absolute difference between these two. But there is a lot of variance in this data, which is something to just keep in mind. Something to keep in mind. Yeah. On average, if we look at the bars, I think it looks pretty good. There are a lot of points that have a lot of tubulin. I'm just wondering why there are so many that have none. And it could be because they're kind of rare to begin with, which is maybe the correct answer, but I don't really know. Oh, okay. I'm getting tired. Mies, you're asking some good questions. I did not have the energy to include those details here, but those are good questions. So, so I'm putting on my grumpy skeptic hat. Apparently, I had one when I was preparing these slides. I would only say more neurons happen to be in the combination injection. Maybe the presence of another cell type encourages native neurons to innervate the tumor. So I think past Phi was thinking, hey, is this because of inoculation or some other weird accident here? But I think it's okay. I think it's fine to say that it worked. Time to get some Zoffy. I can't have Zoffy, man. It's going to ruin me. I would feel so bad if I drank coffee right now. I'm just going to power through. So we have successful inoculation. The next question that I'm sure we're all dying to know is, can we prove definitively, at least in this mouse that's been injected with a tumor, are there tumors worse when there are more sensory neurons present? This is kind of the meat and potatoes of this paper. So, what do you think, chat? This is tumor volume. Green is only the tumor. The only injection was the tumor, and then the red was the tumor plus the neurons. So, chat, read this data and tell me, were they able to see a larger tumor, a worse tumor, aka larger tumor, in the mice that were injected with both neurons and cancer cells? What does this data look like to you? Do we have strong evidence that injecting neurons plus tumors makes the tumor worse so far? What would you say? You gotta trust in your eyes. Trust in your eyes. What is this data telling you? And be careful with interpreting the data too. There is a legend on the left here. There is a legend. Yeah, some nuanced takes in chat, which I'm glad. Yeah, they're labeling that point as not statistically significant. And looking at those error bars, yeah, I don't think that that is statistically significant. But the difference is kind of big, which means that maybe if they did more ends, maybe if those tightened up a little bit, you would see an actual difference. But it's not obvious, right? And again, you don't want to throw more mice into the meat grinder if you don't have to, right? And I use that analogy purposefully because I want to emphasize scientists don't want to sacrifice animals needlessly. You typically do a power calculation beforehand to try to estimate how many mice you need, and then you call your shot. You say, I will need whatever number of mice to have a certain level of statistical confidence, and you don't just do more mice You don't just do that because it's a waste of animal life and we want to be careful with it. So, kind of sucks that you can't call it statistically significant. Although, you know, I am okay with thinking that maybe there's something there and they got unlucky. That's possible. But I'm also okay with saying that there's no difference here. And that's kind of sad, right? This is a bummer. We encountered this before in a different paper about the lipid reprogramming of cancer cells. You have all the logic here. Patients with more neurons in their breast tissue, they die sooner than their counterparts. You can do everything right in science. You have all the right ideas, and then just reality doesn't conform to your expectations. And that sucks. But the researchers don't give up here. Because cancer is not just about tumor volume. Can anybody tell me in chat what is another facet of cancer that they could investigate? So they get to this point, they do their science, they sacrifice these mice. And it's so close. It's so close. Maybe if they do their math differently. Maybe. Who knows? I'm not them. I don't know what math they did. I'm not a good enough statistician to even tell you if what they did is correct or not. Because stats is a little like, it's a little like philosophy in a way. And I'm not going to pretend to be a stats expert. So I'll trust them when they say that data point is not statistically significantly different. But again, tumor volume is not the only factor to consider when it comes to investigating the mechanisms of cancer. What other things can be important about tumors before? You shouldn't trust them. Stats is a common error spot. As a communicator and not a strict teacher, if I were a teacher, I would work a lot harder to do the like double checking of the stats. But as a communicator, my job is to kind of be a cheerleader for this stuff. And to review 15 papers, I'm not going to review them critically. And I said that at the top of the stream too, right? Like I'm here to show off the research, not to criticize it. Because I wouldn't be able to do that 15 times for this 24 hours. Although in my journal club streams where I do one paper at a time, I'm a lot more careful. But for 24 hours of breast cancer research, I'm going to give them the benefit of the doubt here. So, I did see one answer in chat from Miro Knight. Malignancy. Okay, now we're getting somewhere. So cancer is not just about how big the tumor is. Yeah, aggressiveness. Zane is correct. Aggressiveness. So maybe the neurons aren't that involved to a huge degree to how big the tumor gets. Maybe it's something else. And that's interesting. Right? That's interesting. How often do I do journal club streams? I don't plan on doing them very often from here on out because I really need to get back to making videos, right? Like I love doing the charity, but this is charity, right? I could be making a video that can earn me actual money, whereas all of this time is time that I dedicated for this cause, right? I might not be donating a lot of my money today, but I am donating a lot of hours, including the last 24 hours. So I used to do them a lot more frequently. I'm going to tone it down just so that I can crank out some videos and make the YouTube algorithm happy. I hope you understand. I do it probably about once a month going forward until I can get comfortable with the YouTube algorithm. Okay, so yeah, cancers are more than just their size, they're also their aggressiveness. So, I think chat, you've already answered this. We can look at metastasis. We can look at how aggressive this cancer is, how the propensity of this cancer is to spread to other regions of the body. So on the right is lung tissue. We're no longer looking at breast tissue. But we are looking at, so the top is like a histology thing. I don't know how to read histology slides, so I'm not going to say anything about that. But fluorescence, I can read. So the top and bottom are of the same slice of the lung. But the bottom picture has been lit up with the fluorescent signal given off by breast cancer tissue in that same organism, the same mouse. So everywhere you see green, these are all breast cancer cells that have migrated from the breast and ended up on the lung via metastasis. Via metastasis. And now, what do we see, chat? I don't want you to look at the numbers. Use your eyes, because that's a very important part of understanding how to read papers, is to look at the data yourself. Because there are hidden clues sometimes in these. But 231, this is cancer only. This is cancer plus sensory neuron. And what do you see? When you add the sensory neuron, what happens to metastasis? That's scary. I mean, that's wildly more aggressive, but I like to see average values in the symbol. Yeah, scientists will pick something that is representative, or ideally, they should pick something representative of the show. But I think it's really important to actually look at the data for yourself. You might not be able to see all of the pictures, but to understand what this looks like is such a rewarding and important part of the scientific process. So hopefully I do have that data on the next slide. So we'll see. Ooh, Dispel says H&E looks like hyperplasia, at least. Very diffuse nuclear aberrant. Ooh, are you a medical professional? Very cool. Because I don't know how to read these at all. So cool to have these experts show up. Very nice. Ah, there we go. Okay, so this is average data. So 231 all the way here at an average of about 20% of the area of the lung. And then when you add that sensory neuron, you get 10-15% increase, maybe. And the ranges are quite big. It's biology. They're always going to be quite big. But this does look like a statistically significant difference, though. So it might not be as dramatic as what this picture shows, but I do kind of buy that maybe we should be looking at metastasis. Yeah, that's not 10% in the slides. They should have picked something more representative. This is a criticism that is legitimate. You're not supposed to pick the pictures that show off your effect the most. That's kind of, I wouldn't say that that's malpractice, but it's a little dishonest. You should pick something that is more representative. Like I should have picked this lung and maybe that lung to show, because that's a bit more honest. This is a bit tricky. I don't particularly enjoy that. Yeah, so I said they should be representative. In this case, it isn't, and we can criticize them for that. Yeah, this is cherry picking. No. You don't have to cherry pick. I think a 10% difference is fine to show. Let's keep that in mind. So here's a trap that you can fall into. If you sour on the rest of this paper, then you've fallen into the criticism trap. As I said multiple times, it's so easy to tear things down. Kind of hard to build things up, right? So yeah, we can groan. We can say that, okay, it's maybe not so poggers that they picked pictures that aren't totally representative. But we won't hold it too against them. Okay, let's forgive them in the moment. If this starts happening more and more, then we can kind of get mad, but a one-time thing, I think it's okay. I think it's okay. So let's continue. Okay, so how did the breast tissue end up in the lung? Well, we do know that cancer cells like hitching a ride on the backs of neurons, and is the same thing happening here? Because of time, I'm not going to stall here and ask you how you would study this. I'm just going to show you, because this is cool. So they're going to take cancer cells in one condition, and then we're going to expose cancer cells to a media that has been, that neurons have been growing with. So they're trying to figure out, okay, what is causing the metastasis? Is it something that the neurons are spitting out, in which case you should find that in conditioned media? Or is it something that the neurons are doing physically to the cancer cells, like something that you have to have a neuron there for? So they set up this interesting experiment. And they find that when you have cancer cells in a dish, they don't really move around all that much alone, right? You can see these traces. And each line represents the path of a certain cancer cell. And when you only have cancer cells, they kind of wander, they kind of crawl along, but they kind of go in a random direction. And it's not really that interesting to look. And we know that more aggressive cancers that are trying to move, they're trying to move and groove, they will move more. And what the researchers found was that when you add the conditioned media, on average, they don't look like they move all that much. Although, again, this trace looks like maybe they should be higher, but when we look at the actual data, the speed is about the same. And the proliferation is about the same. Now, something interesting happens when you add neurons to the dish. Now, all of a sudden, those cancer cells are moving, dude. They are moving. And it looks like the difference when you add the neurons to the same dish, let's say, it's like that's about double, maybe a little less than double. It's a pretty big difference. The cancer cells that are grown with neurons are moving a lot faster in the dish, which is crazy. That's wild, right? Oh, and Spotty says, this would be an awesome question to explore via live self-fluorescence microscopy. Would make for some cool videos. Interesting. You think that they would make for cool videos? Well, I think those videos are coming. I thought it was going to be this slide, but it's maybe the next slide. So something else that they showed was cancer cells associated with neurons higher than chance, right? So if you plated a dish of neurons and cancer cells and the cancer cells didn't attach to neurons at all, you would kind of expect them to be randomly patterned. But actually, in green, you see the cancer cells, and you see that they're attached to the beta-3 tubulin, these markers of the neurons. And if you expected them to not associate, you'd expect the coincidence of their localization to be random, but they're not. Green in this case is almost always associated with red. They hang around neurons, which is also interesting. And if you take a video, you can actually track the movement of these cancer cells along these neuronal tracks. And you can actually see that when these cells are attached to neurons, they're moving much faster. And when they fall off the neurons, they're a little lost. They don't really travel as fast. So that's also really cool, right? I really love that you can see the story of how the researchers looked at this step by step. It's beautiful. This is a wonderful paper. Yeah, okay, we can roast them a little bit for not picking a representative picture before, but overall, I really like this paper. Oh, do I not have the video here? Maybe I do. Maybe it's coming. I don't know. I have some videos from something. Oh, it's been so long since I prepped these. But they measure this through video tracking. I don't know if they uploaded it to their paper. I hope I have some of it here. But yeah, I just want to also point out that, like, if you pump up the concentration from the conditioned media, then you start seeing movement. But really, it's the contact that's doing the thing. Maybe the neurons are spitting something out that matters, but it's probably not that important. Okay, so how do we find out why this happens? Gene expression stuff. What the heck is going on? What's happening here? I'm not going to go through this. I went over this like 10 hours ago. But in brief, we're checking to see what stuff do these cells make that might be a particular target to look at for understanding what the heck is happening as these cancer cells are crawling up neurons. So which genes are enriched in each condition? This is a bunch of stuff that I don't want to go over. We're also over time, so I'm going to go past this. A lot of things are enriched, like the cells are making stuff that you would expect them to make. ECM organization, that makes sense, right? Adhesion migration, they're making more of that, makes sense. So, this is for heat map nerds, okay? And thank you so much for the donation, Holly Day. Thank you so much. Donating my lunch money for breast cancer research and Biosong. Thank you so much. I will honor that lunch money. And we are that closer to the original biology song, which I started writing because I wanted to see if I could make music. And I think I can. That Blender song, I had a lot of help, but the melody came from my brain. And that means an original song is pretty possible. So again, I'm not going to dwell on this. Immune signaling, surface receptor signaling, cell proliferation, ECM organization, ECM extracellular matrix. It's the stuff that's outside of the cell. If you are interested in studying migration, you're probably going to want to look at stuff that is happening outside of the cell for movement stuff, right? So ECM. So they hone in on ECM as a potential target. Oh god, this paper is long. So they find the top 10 genes that are upregulated when breast cancer is cultured with neurons, and they see a certain kind of pattern. And I'm just going to say it here. There's a couple of reoccurrences of this particular family of genes, PLXBNB1, PLXNB3, PLXNB3, right? These are genes that are important for migration and adhesion. And so you could say that the biggest fold chain is in this FPR1 gene, and you should study that. Oh, excuse me. And I think that would be fine. Here's a lot where like artistic expression can differ. Like there's no obvious reason why they chose PLXB1 from the data here. They could have gone with TMEM8. But they noticed that there was this tempting pattern. This name seems to come up a lot, so that's what they went for here. But fold change doesn't always mean more important, right? You can have a huge fold change that means nothing in biology. It's an interesting thing to keep in mind where maybe you want to follow that lead, but you don't always have to follow fold change. It's just not always the most important thing, because you can have like a really important protein. If it increases that by 10%, you're dead, right? Because that protein is really important to keep in that tight level. So don't always look at the highest fold change. You can, but keep in mind that biology is complicated. Oh, FPR1 was the gene my lab was working on. That's sick. That's awesome. I don't know anything about it, but I'm sure it's very cool. So what are plexins? This group of families, this protein family, this is a group of cell migration proteins. So these proteins, when they are functioning normally in neurons, are instrumental in guiding axon growth. So they teach neurons how to grow appropriately. And so plexins will guide axon growth away from its counterpart semaphorin. So they work to balance each other. So plexins will instruct, hey, axons, don't go towards semaphorin. Grow the other way. So why would a cancer cell make a neuron protein? That's bizarre, right? Because those cancer cells are making a neuronal protein. It's maybe not weird that they do that because there's stem-like cells that can make whatever you can make. But why is it useful? So if neurons are around, maybe neuronal semaphorin may be guiding cancer movement. Oh, isn't that interesting? We just learned in a previous slide that cancer cells migrate a lot faster when they are physically attached to neurons. And we also know that cancer cells make plexin, this guiding protein that tells the cell to grow away from semaphorin. And we know that neurons make semaphorin. What if, chat, cancer cells attach to neurons and they run away from semaphorin, and that's why they move so fast? Isn't that crazy? That's crazy. Here's the model. Cancer cell here, neuron here. There's plexin and then there's semaphorin. And as the cancer cell is migrating, they're saying, oh, there's semaphorin here. I gotta move. I gotta move away from the semaphorin. And so they crawl up along the neuron and migrate along the neuron, trying to get away from that semaphorin. It's a beautiful model. Are cancer cells whipping the neuron horse? It's the other way around. Cancer cells are running up the neuron because they're running away from the semaphorin. That's a beautiful model, isn't it? This entire process of like, why the heck are neurons running along neurons? And they identified a potential model by which this happens. And it is, and again, they haven't tested it yet so far in this paper, but it makes a lot of sense. Cancer cells are making plexin B. We know that neurons make SEMA5. And we know what these proteins do. So it is reasonable to assume, so that we can test this, is that cancer cells are just running up the neuron because they're making a protein that tells them to do so. So how do we test this? For the sake of time, knockouts. And in this particular case, we get to use CRISPR. If you knock out the plexin B in cancer, they should no longer be able to migrate. So what effect do you think that'll have on speed of cancer cells? I go to it in 10 minutes, but this stream is amazing. I have tried to catch as much of it as I can. I hope you hit your milestone. Good night, all. Thank you so much for hanging around, Kangaroo. It means a lot to me. And good night. So, my guess is if you knock out this yellow protein, these cancer cells are going to slow down Finally, 10 hours later, we get to CRISPR. Yeah, it's so true. It's amazing that we haven't gotten to there before. Okay, here are the videos. Okay, so I do have video here. Okay. This is sick. So, on the left here, just cancer cells in green. In the middle here, cancer cells plus the juice that the neurons were cooking in, growing in. And then on the right here, in green, you have the cancer cells, and you can also see neurons. You can see their characteristic cell bodies here as well. What do you think these videos are going to look like? Make a prediction for me, chat. What do you think you're going to see in these videos? What is this going to be? What do you think? Oh, that's good. Come on, chat. I've been at this for 15 hours and my brain is still working. Come on. I know you can do this. You knock out, or rather, you know, this is not knockout. This is before the knockouts, but this is just the baseline state, right? Cancer only, cancer plus neuron juice, and then cancer plus neurons. This is the baseline. No knockouts here. What is this going to look like? So Frost says 2 and 3 are going to look more zappy and will have spiky movements, while 1 is not going to dash as much. Interesting prediction. All right. Only the 1 neurons, you should see cancer cells crawling up the neuron. Maybe some Brownian motion in the right. Oh, yeah. DRG should have a greater speed, increasing speed from left to right. Okay, we have a prediction here. Chat, is your prediction correct? Let's watch the video. Let's do it one more time. What is CM again? It stands for conditioned media. It's the juice that the neurons were growing in before. That's to test whether or not the neurons were spitting something important out. So what do you see? Comparing these. It's not like cancers don't move when there are no neurons around, right? But look at, whoa. But look at what happens when they grow with neurons. Look at how fast this guy is. Keep your eye on this guy. Okay, I'm going to replay the video. He's going to start up all the way over here and watch this segment. Wow, oh my god, that's so fast. Zip, zap. This guy is going crazy, dude. Is it using the neuron like a slide? It is. It's so fast, right? When they are co-cultured, those cells can move. The neuron zipping is wild. They're using neurons as roads. They are. Okay, what about when we mutate? When we mutate the cancer to lose their connection to the neuron, what do we think then? So here we have a control condition. This is cancer plus just the CRISPR stuff, but no actual modification. Right, so you give it the CRISPR tool, but you don't actually make the CRISPR tool useful. And then these two on the right, these are basically the same. These are 2 different knockouts because you want to do this multiple times to make sure you don't accidentally screw something up. This is cancer, but their connection to the neurons have been severed. So what do you predict here? What do you predict here? What do you predict here? I don't know if it's real time, but there is a time marker here. I don't know what time code this is, but, you know. Left, same as cancer plus neurons, Brownian motion in the right too. And are you correct? We'll see. We'll see. So up here is just the green. This is bright field. This shows you the neurons as well. So if you want, so this is kind of messy because neurons are here, but pay attention to the top and we'll play that again. Kind of hard to tell, honestly. This one's not as crazy. Definitely pretty tough. What do the data say when you average this? This one, I would not feel comfortable reaching a conclusion based off of this video. What are the 3 again? So this one is normal condition, just cancer plus neurons. This is knocking out the connector between the cancer cell and the neurons. And this is the same as this one, just using a slightly different single guide RNA. I wonder if they can quantify this. They can do like dot tracking, right? You can measure the individual velocities of these by tracking these over time. There's fancy programs to tell you how to do that. So let's see if the data reflect your predictions, because the video is kind of tough to see. What do we see here? Okay, well, when you don't have neurons, the cancer cells are slow. When you add neurons, the cancer cells are fast. For bars of significance, it's pretty confident that these are different. When you don't have neurons, and when you do have neurons, if you've destroyed the connector, no difference. Similarly with this version, there's also no difference. So yeah, the video wasn't great, but we can see here that if you knock out this protein on the cancer cell, it kind of looks the same as if there were no neurons there to begin with. And that's awesome. It's funny that they label it as non-significant, but it being non-significant is actually marking it as significant. Yeah, the fact that this is NS, that's the important part, right? We just said the model is the cancer cell climbs up the neuron via its connection. It has plexin and the neuron has SEMA5. And when you remove plexin, it's like the neurons aren't even there. That's amazing. It's amazing. 4 bars of significance seems like a lot. I mean, it is because the ends are so high. I think the ends are dots here. I don't love that as a biologist, but I don't know their science to their extent. Like, I would prefer biological replicates. These might be technical, but I'm not going to get too into that because then we're getting to really like heady philosophy of statistics kind of deal. So I think this is awesome. This is beautiful because they had a model. And I think they kind of proved it in vitro, at least on a dish, but I believe that it works in a dish and that's sick. Okay, long paper, went much over than I thought it would. So here's a summary. Neurons are increasingly being implicated in worsening cancer diagnoses. Neurons can provide a positive feedback loop for cancer growth and metastasis. In this study, the authors showed that neurons may play a role in breast cancer metastasis, right? Maybe not tumor volume. It's a little hard to tell in mice with human cell line, whatever. But it's encouraging. I'm not going to say that they were definitely right, but the data look pretty good in a dish. In this study, the authors show that the neurons may play a role in breast cancer metastasis. It looks like the cancers really love climbing on these neurons, using them as lines. Those videos are so cool. So patients with more neurons in their tumors did not survive as long as those without. So again, could be correlation, but now we have a mechanism that might move this closer to causation. Mechanistically, these experiments show that neurons may be regulating the movement of cancer cells via direct contact between plexin and semaphorin, meaning, as chat has said, this might be another bit of surface area for drug designers to exploit, right? You think about neurons when you're fully grown, is you don't really necessarily need to grow new ones as important as you need to avoid having cancer. There is neuronal growth in the brain, certain niches in the hippocampus and the cortex. Yeah, you see a little bit of neuron growth, not a lot though. So maybe it's okay if it means that you don't have metastasis of cancer to your brain to target this. Now, I don't know if that's true or not. Only time will tell. I love this paper. It's so cool. It seems like they proved their point in this model. I don't see why this wouldn't apply in humans mechanistically. I mean, you could say that by using human lines in a mouse cell, the mouse immune system interacting with human cells is going to be a bit different. Maybe the human immune system can prevent metastasis better. It's a little unclear as to how all of that shakes out in a human. You always want to be a little humble when applying mouse stuff to humans. But again, I'm optimistic that they're right. Again, being a good scientific citizen means assigning confidence to your assessments. I never think if something is true or false. I think of it as probabilities. This is probably true. I'm almost confident that this is true, but I always leave a little room for doubt here. I love this paper. It's so good. Very, very cool. I love the videos too. Oh my god, okay. Okay. Wint, the next paper, which I think I'm going to blaze through just because, yeah, my brain is going. And let me take a preview. How long is this segment? Let's see. We're at the Wint section. By the way, slide 177. No, we're past that. We're way past that, actually. What slide are we on? We are slide 268 of 474. How long is this Wint paper? Oh my god, it's long. I'm going to rush through this because I'm tired and I don't want to talk about Wint because Wint makes my brain hurt. This one was a bit of a tough paper for me to get through, so forgive me for going through this a little quick. But I love the neuron paper. I don't love this one so much, not because it isn't good, but because dev bio scares me. This one was also funded by Breast Cancer Research Foundation, so that's also important to take a note of, right? Good research. I'm scared because when I see Wnt, ERK, CD4 and 6, I get sent into a fugue state, right? This is crunchy dev bio, cell bio stuff that we're about to get into. And I'm going to try to simplify it and go through it relatively quickly. What Wnt is one of the coolest signaling pathways? It's cool. I know it's cool. I like Wnt. I just don't remember how it works because it's difficult. There's a lot of stuff. My memory isn't good. You might have missed my rant about like passion and fit. I would not be a good fit for biology because my memory's so bad. You can ask Toxo all about how bad my memory is. I powered through my biology education through sheer guts and determination and passion for the subjects. Stuff like the Wnt pathway. Beautiful when I learned it, terrible when I was tested on it. I'll put it that way. Okay. Okay. Wnt, ERK, CDK4 and 6. For those of you who don't know, this is a very important developmental biology circuit in the cell. Very instrumental. Very important. Beautiful, even. It's great. It's great to learn about Wnt. There's a lot to learn about Wnt. We're not going to go through all of it in this stream, though. But because this is the developmental bio aspect, it is going to dovetail with the cancer story because cancer is a story of growth. So what this paper is looking at is activation of the partial EMT state. My brain just shut down for a second there. The epithelial to mesenchymal state. So like 8 hours ago, I talked about the EMT state. This is a cell program that converts something that should be a flat barrier forming cell into a round cell that is capable of movement, that is more typical of the mesenchyme. And cancers love that because, you know, that's what metastasis is. You form from a well-behaved little guy, and then you start getting your own ideas about moving out of your local neighborhood. Then you form the mesenchymal state, and then you leave. And this paper is trying to argue for the existence of a third state. So there's epithelial and there's mesenchymal. And this paper is saying, whoa, hold on. Biology is a spectrum. There's something in between, the partial EMT state. And it is this partial EMT state that is actually important for cancer. E and M are important, but this middle ground is actually really important too. And I love the conclusions. I think this is a great paper. Oh, it's just like, the stuff is hard to get through. So let's get to it. So what kills when you have cancer? Is it the tumor? Sometimes. But more often than not, it's metastasis. Metastasis is the process by which a local tumor can escape their local environment and spread into other tissue. It's a very complicated process. So if I still go in, as I am ever, right, 15 hours in, still saying words. And these words are about metastasis, which is the process by which cancer cells escape their local environment. It's very complicated. But if you want to simplify this arrangement, it goes a little something like this. First, the cell loses cell-cell adhesion, so the stickiness it has to its neighbors. Then it goes through a process of intravascularization. This word is so hard. Intravestration, which is invasion of tissue. So it leaves the local environment, it invades the tissue, it circulates into the blood, and then it lands somewhere. Lung, colon, kidney, liver, brain. Bad news. Now you have double cancer. Terrible, right? So the start of metastasis is commonly thought to be represented by the epithelial to mesenchymal transition state, or transition, or EMT. So epithelium, we love these guys, organized, columnar, terminal, well behaved, unproblematic. Transitioning to a more mesenchymal behavior, however, this is what is problematic. Disorganized, multipolar, helps differentiating cells. Important for our development as well, but you don't want this to be unregulated because cancer would love to be more like mesenchyme, because mesenchyme can all do all sorts of fancy things besides their job. So I'll say about triple negative. Oh, okay, yeah, no, I think you got it. Yeah, you got it. Okay. If you want to send me YouTube videos, you can join the Discord, !Discord, and put it in the content share channel. I think whatever video you have is pretty sick, and I would love to show it off stream if that's okay. I know PIs can be kind of territorial, so make sure you get permission first. But not for this stream. I don't have the energy or brain to do that coordination, but maybe one day. Oh my god. So what are the normal roles of the mesenchymal cells? They do a lot of like helping with differentiating cells, and you can kind of imagine that might be kind of useful for a cancer cell. Okay, so here's a neat image I found that I could show because of copyright. You can kind of understand why a cancer cell undergoing the EMT transition would be problematic, right? Epithelial, you have this nice organization, columns, it's great. But then you get one guy starting to get his own ideas. He wants to become a mesenchymal cell, and look, he can just leave the area, right? Shrink, lose the connection, right? These thin horizontal lines. You can think of these as staples, stapling the epithelial together so that they can function as a tissue. But when you start transitioning out of epithelial fate, becoming mesenchyme, you lose those horizontal staples, and then you can squeeze yourself out and just leave. Whoops, just popped out, just like that. That's crazy. So these mesenchymal cells exhibit some stem cell-like qualities, making them cancer-like. So they don't officially get to be called cancer at this point, but they are heading there. Glitches out of bounds, exactly. They clipped out of the epithelial layer is, I think, a wonderful way of putting it. So the epithelial mesenchymal transition is not always a bad thing. This is a developmental program that is hard-coded into your DNA. Your body uses it during development, right? Especially a part of building the neural crest tube. I believe this is a video. I hope this is a video. Please be a video. Can't actually show this. Okay. Why even allow me to embed a video if you won't even let me show it? Here, watch this video. A company of biologists. This beautiful process is mediated by the EMT transition, the epithelial mesenchymal transition. Now, are these mesenchymal cells distinct from canonical MSCs, mesenchymal stem cells? So that's a good question. I don't know the answer. It's a great question. I don't have enough cancer knowledge to give you a good answer there. Can you tell I'm getting tired? But yeah, this beautiful process of coordination of the formation of the neural crest tube is mediated by this program that cancer cells also use. So this itself is not a bad thing. It's a bad thing when cancer is doing it, though. Beautiful video. So I'm a broken record. Once again, cancer has a way of perverting, and I do believe this is perversion, because this is a healthy thing for your body to do as it's developing, but cancer has a way of perverting, corrupting biological processes. And so understanding more about how this EMT transition happens may allow us to limit or delay the onset of metastasis. Again, the goal of cancer treatment isn't to make you cancer free, although if it can happen, then why not do that? The goal really is to get you to die of other reasons before you die of cancer. And so maybe that means delaying metastasis until your life ends for some other reason. So, really understanding this transition to delay metastasis until you die of natural causes is a worthwhile goal. And again, a goal that is funded by the Breast Cancer Research Foundation. So, that's why this is so important. More research emerges at a third state in between epithelial and mesenchymal, and that's this partial state. And this is a headache, right? Labels, biology and labels, right? There's something in between E and M, EMT, and this is unique. So maybe this is why metastasis treatments don't often work all that well. Maybe the treatments that we do have to attack metastasis don't work because they're trying to prevent mesenchymal cells, or they're trying to keep cells being epithelial, but they're not considering that there's a third thing in the middle that might be worth attacking instead. Okay, so if we know how this partial state works, maybe we can attack it. Hey, Comey, welcome back. Okay, so Wnt signaling. Again, I know Wnt is cool. It's one of the goats of developmental biology. Wnt does so much. Okay, molecule Wnt. It guides the developmental process, regulates cell proliferation, regulates cell migration, regulates cell fate, and amongst its many duties, regulates the EMT transition and also stem-like qualities. Wnt and sonic hedgehog signaling are responsible for the dorsolateral development, right? So how the body knows to develop your chest from your back, that's Wnt, baby. Really important. I just find it really difficult. That's all. Worse, that reminds me, what do you call a cell site? A DNA. Come on, that's a good joke. Come on. I'm proud of that one. Okay, so here's the Wnt signaling pathway. This is one of two. There's the canonical Wnt signaling pathway, and then the non-canonical. It's a whole mess, dude. This is really cool. Trust me, you'll learn it when you go to graduate school. But just know that Wnt is a secreted signal that you get from the environment that changes, that binds to the different Wnt receptors, and it triggers a whole bunch of cascades that tell your cell to do certain things. And for our story today, one of those genes that Wnt can activate is FOXC2 and FOXMT1, both factors known to drive cancer aggressiveness and stem-like properties. I'm gonna skip the next couple things. I'm tired, baby. I am so suffering. 9 more hours of this. 8.5, to be fair. Surely we raise the rest of this money by 8.5 hours, right? So we want to know how what role Wnt signal is playing in the story of the EMT transition, and this is their basic model, right? Yeah, I do need the power nap, y'all. If someone has got an extra $1,000 lying around. Oh. So this is the simple model. It starts with the tumor making TCF1 that leads to Wnt signaling. It turns on these programs, FOXC2 and FOXTM1, and that forces the cells into this stemness transition state. So how do we test this? First, let's make sure what actually encourages stemness in a partial EMT state before doing anything too fancy. So we do a little bit of cell sorting to see if there is a real transition state. So this is cool. I'm not going to spend too much time on it. But basically, cell sorting allows us to identify cell populations. In this case, we're going to be taking advantage of receptors on the cell membrane. So you can digest the connection between cells and actually organize them to go through a machine one by one. And through fancy magic, I will not explain, the machine will tell you this cell has this receptor, this cell has that receptor. And then you can tell the general population of the cells that you're working with here. So when we take a look at cancer cells, what do we find? We're looking at two markers. We're looking at CD44, which is the mesenchymal marker. That's on the y-axis, you could say. And then we're looking at CD104, which is the epithelial marker. So if you are mesenchyme, you have a lot of CD44. If you have a lot of CD104, you are an epithelial cell. So chat, which quadrant, and please give me a chance to rest my voice, which quadrant represents the transition cell? What quadrant represents the transition cell? Cell sorting, love it. Flow cytometry. Where are the transition cells here? Why would I donate $1,000 for a nap when I can get 40 jokes? Brutal. Brutal. Okay, so Q3, a lot of Q3s in chat. So it's a little complicated to go through. So I'll say this. Q1 is high in CD44, so high in CD44, but not high in CD104, right? That's this quadrant. That's primarily CD44, so this quadrant is going to be mesenchymal. Q3 is going to be high in CD104 and low in CD44. So this is going to be primarily epithelial. Q4 is going to be low in both markers, so maybe neither. Q2. Q2 is the right answer. Q2 is the quadrant for the transition state. And surprisingly, in breast cancer lines, you can see that, yeah, it makes sense. Most of them are mesenchymal, right? It's cancer. Mesenchyme makes sense. But not a whole lot of them happen to be in the transition state, which is kind of interesting. Misspelled, you wrote CD14. Yeah, I know, forgive me. I was tired of making these lines. I've done nothing but think about breast cancer for the last 3 weeks. Okay, so Q2. You can see a lot more mesenchyme, not a whole lot that are kind of in this transition state that have both the epithelial and mesenchymal marker. And yeah, you could say that maybe you're not surprised because this is a transition state. If you're thinking about chemistry, well, transition states are often very short-lived, so maybe that's not surprising. But being able to do this at all is really, really helpful because now we can isolate, we can purify just those cells that are in the transition state. So we can take our fancy flow cytometer and we can separate cells. Or I don't know if they do this by flow cytometry. They might do a column method of some kind. I didn't read too carefully, but they have a method where they can separate these cancer cells into only mesenchyme, fate, and the transition state. And so they separate these cells and they allow these cells to grow into tumors, and they found something very interesting. They injected these breast cancer cells into a mouse. Some of these breast cancer cells were from the mesenchymal state, and some of those breast cancers were from the transition state. So E/M, that's the transition state, and M is the regular mesenchyme. So, chat, if you had to pick a cancer to be infected by, would you pick E and M, like the transition cancer, or would you pick the mesenchyme cancer? If you had to pick for your health, okay, Ki67 is a marker for a dividing cell. It's basically giving you the marker for rapidly dividing tissue. Which of these would you choose to be infected by? Yeah. Yeah. Yeah, absolutely. I mean, Cairo says the right answer. You'd pick the benign one. Pick a benign one. But if you had to pick between transition state and mesenchyme, look at how nasty that transition state tumor is. by 70 days after injection with the tumor, if you took tumor cells that originated from the mesenchyme, the tumor is bad. Yeah, it's there. But a tumor that grows from the transition state looks to be way worse. Maybe more than double the size, which is crazy. So I'm convinced now. I was skeptical before, but this data makes me convinced, okay, I'm buying what you're selling now. The transition state between E and M, this middle ground, is a real distinct thing that matters. And it's surprising. It's worse than the mesenchyme state. What do you see here, chat? These are breast cancer cells in a dish. What do you notice about their behavior? Do I have anything to drink? Yeah, I should hydrate. I'm sounding a bit hoarse. DNA. What do you see here? Describe to me the data. What is the transition state tumor doing the mesenchyme cancer is not doing? What do you notice is different here? Spread a lot more, maybe. There's a lot more in the E and M state. There's something about the pattern of behavior as well. The pattern of the data. You're all correct. There's something else that's important about what's happening here. It might be a little hard to get, so I'll keep going. So even in a dish, these are organoids. These cells are clumping. In the mesenchyme, they clump too. You have these balls. But the transition cancers are forming masses. And they're forming it at a much higher rate, at least in this picture. Okay. Kairos called me out. Don't trust this immediately, but it's important to use your eyes before you read numbers because you need to make sure that they match up. So they're making organoids. Even in a dish, they're sticking to each other. They're sticky. Remember that a metastatic tumor, if they stick to something else, congratulations, you have tumor 2.0, right? And look at how sticky these transition tumors are. They're clumping. They're clumping. Okay, so what did the data? Oh, I don't have the data. Bummer. I think the data the numbers reflect the increased clumping as well. I think if, it's because I read the paper, you didn't. So if you want to call these equal in terms of their propensity to clump, I think that's totally valid. That's why it's important to have the data. My bad. I think they do actually find that the transition cells do clump at a higher rate. But yeah, I could buy, just from this picture, I would believe that maybe they're the same rate, it's just E and M have more. I could believe that you believe that. To me, it does look like, even though there's a higher density in E and M, I do think that that is meaningful, just from this picture. Of course, I wish I had the actual data here. So that's my bad for not including the numbers. So here are the results so far. Most cells in a line of selected breast cancer are not actually in transition state. However, when purified, injection of this transition state line into mice causes cancer that grow bigger, that show more tumorigenic properties, and show a level of stemness. I maybe exclude that because I was so sick and tired of preparing. This was a lot of work, y'all. I put a lot of hours into this presentation. And I think we can agree from the data that I've shown that this transition state is worth studying. So it's time to actually test this. Okay, so before we can ask, does Wnt convert mesenchymal breast cancer cells into a stem-like state, we must first ask if our cells are... Oh, no, I'm skipping this, dude. It's okay. We don't need to talk about this. I don't want to talk about any of this. Wnt, it stresses me out, and I'm sorry. Okay. Oh, this presentation is so long. Um, okay, well, we'll end here. Okay, so basically, the stuff that I mentioned before, I don't have the brain here 1 a.m. where I am talking for my 15th and a half hour in a row to go through all of that. I'm sorry. But we'll pick it up from here. So just take my word, they did a pretty good job connecting these lines. So the tumor is making TCF1. They do show that it affects Wnt signaling. They do show that Wnt signaling affects FOXC2, FOXTM1. But I think the more interesting part of the story is what the heck does this do to cause stemness? What does this do to cause stemness? So, a known regulator of the EM transition state is TP63, tumor protein 63. And a natural question you might want to ask is, does this thing downstream of Wnt signaling affect this tumor protein? And remember, the transition state is a part of healthy biology. In a healthy cell, TP63's job is to make sure that the skin and teeth develop properly. Some computer work reveals that there are putative, meaning predicted sites, for this thing to bind to a tumor suppressing protein in TP63. But only a certain isoform. This is a detail for nerds, not going to focus on that too much. So does FOXC2 actually control this protein? Well, we do see that TAP63 is high in the transition state, really, really high. That's good. We know from previous literature that this protein regulates this, so their system is set up properly. What's also nice, for the sake of research, is that in breast cancer lines, when this is knocked down, you see a lot less TP63 RNA. The connection is there, but it's not clear how the connection, how direct the connection is. Thanks for hanging out, Kairos. Thank you for sticking by for so long. Okay. I even wrote that I'm tired here. I am genuinely so tired preparing this section that I'm going to skip the finer details and just say, yes, they worked out a lot of this pathway. They did a good job here. Dude, this paper's huge. Just look at all this work they did. Tumor TCF1 does Wnt signaling, which creates a transient burst of phosphoric. So it pings the ERK pathway. It doesn't make ERK go up and it doesn't stay up, but ERK goes up, ERK goes down very, very quickly. It's a transient burst. And this works because Wnt signaling temporarily disables EGFR, and EGFR is something that is always activating phospho-ERK. So they worked out a lot of detail on how this works. It pings phospho-ERK by disabling EGFR temporarily. Crazy. And this transient burst of signal, this ping actually activates this protein called CDK4/6, which then does FOXC2, et cetera, and then that leads to stemness. Oh, this paper is huge. It's so good. It's hard. Aren't we all nerds? Don't you mean bioinformaticians? Yes, I do mean all of you pathway nerds. I'm too tired to deal with your pathway stuff. But now that the details are set, we have an interesting angle of attack. It's time to start rubbing our hands together and start poisoning some cancers. So, remember the target is the EM transition state. Mice were injected with this particular subset of cancer cells that come in the form of this transition state. And then they were treated with a combination therapy targeting ERK and CDK4/6. These drugs were previously shown to be effective for breast cancer treatment. And now what we find. So, blue, DEPI, tells us where the cells are, and that's their DNA. BRDU, synthetic thymine, marks the synthesis of new DNA. This is a proliferation marker, growth. The more green you see, the more it is replicating DNA, the more it's getting ready to divide. And here we have untreated. And then we give it a Wnt signal. Remember, Wnt controls growth. So I'm just going to say right off the bat, vehicle, don't worry about that. That's our control. We just give this cell Wnt. Wnt is a growth signal, makes sense. The signal is green, and green goes a lot higher because Wnt is active. What about when we start poisoning things? What happens to the proliferation signal green when we start hitting it with CDK and ERK inhibitors? What happens to proliferation? What happens for the ability of these cancer cells to divide when you hit them with these poisons? Less DNA synthesis. Yeah. Not a lot of green means there's not a lot of DNA synthesis happening, not a lot of new DNA synthesis happening. And when you combine the drugs, I don't know about you, Chad, I don't see any green in the combination therapy. Working out exactly this pathway, the beautiful work that went into determining how all of this was linked together, now they know they can hit these targets. And when they do, oh, that's awesome. You knock them down completely. They do not divide at all in a dish. In a dish. That's exciting. So, what did we learn here? I was in a mood, apparently, when I was making these slides. Cell biology is complicated. The transition state cells, however, I do find this to be very interesting. It's something that I will keep in mind for future papers that, maybe that have been ignored in previous research. And this paper, again, funded by the Breast Cancer Research Foundation, the very foundation that we are raising money for. Remember, this is for them. They're funding this amazing research that are making the real case for the fact that there's this third population out there, a third population of cells that are maybe really bad. And maybe we should not be focused on mesenchyme or epithelium, but the cells that are in the middle. I think that's pretty compelling. Okay. Talking for 24 hours is pretty hard, let me tell you that. Okay, next paper. I love this paper. This paper is so good. Again, funded by the Breast Cancer Research Foundation. I love this paper. All right, engineering nerds. This one's for you engineers. Tumor microenvironment on a chip. Already, doesn't that tingle the neurons? And single cell analysis reveals synergistic stromal immune crosstalk on breast cancer progression. I love this paper. This is a great paper. Okay, what is this all about? Come on. We have to begin this paper with a bit of a theoretical conversation about research models. So, what even is a research model? A model is a tool to use while studying some sort of phenomenon. And these models are always simplified versions of their human counterparts. Oh, sorry, my voice is going. Human biology is impossibly complicated, and for ethical reasons, you can't always experiment directly on humans, right? And, oh gosh. So, I'm not going to do the thing where I ask you about what are some examples of research models. I'm sure you already know. I'm just going to continue on with the paper. There's something that my PI, when I was a graduate student, used to say all the time, and it's that all models are wrong. And they are. This is something that Reddit loves to talk about. Models are wrong. It's true. To study a system, you have to simplify it. Systems are often too complicated to study in their natural environment, so a model is needed. So the sentiment that all models are wrong is fair. That doesn't mean they're not useful. Thank you so much for the $50 donation, Anonymous. That's awesome. Thank you so much. The $50 incentive? Which one is that? Oh, I love Blender, dude. I love Blender. I love Blender. And that is brought to you by the anonymous donation. Thank you so much. Okay. So, it's a trade-off, right? How there are two types of validity that I am concerned with when regards to constructing a model, and that is face validity, aka how well does the results reflect real life? How well does this apply to humans? And also construct validity. How well do the results measure the phenomenon in question? So, does your model actually measure the thing that you want to measure? And these are two different ideas, slightly different. And it's a trade-off, it's a balancing act. More complicated models have better face validity, simpler models will have more construct validity generally. And so, I like, chat, don't be Redditors, man. There are so many R science posts. You read those comments, and I just get so discouraged about the state of the public understanding of science from people who think that they are smart, right? If you were commenting on an R science post and you talk like you know what you're talking about, I'm going to assume you think you're smart. And if I had $1 for every Redditor who thinks they're smart because they go, this study was done in mice, it's meaningless. If I had $1 for every time, we'd blow past the milestone already. Let me tell you what. So don't be a Redditor. Understand that yes, mice are not humans, but that doesn't mean it's not useful. You just have to assign it a confidence value. So top one here, that's a Reddit take. Bottom tier, this is, in my opinion, the responsible take. The experiment was done in mice and therefore does not capture all aspects of human biology. However, the results are promising and may explain to some degree some aspect of the human experience. More testing is needed. That should be your line. Okay? There's one thing I want you to take away from this stream is don't be a Redditor. So cancer, as you can imagine, pretty complicated. These are two figures from two papers from Frontiers. You don't have to know anything about cancer to know that this is an overwhelming figure, right? Within a tumor, you have T cells, dendritic cells, T regulatory cells, cancer stem cells, which are different, cancer-associated fibroblasts, extracellular matrix, a ton of different types of macrophages. So a tumor isn't one thing. It's a zoo. And that's going to be hard to study in a dish. And the tumor microenvironment is also different. You have all these different immune cells and support cells. They all talk to the tumor. And notice, they don't even include the neuron. The neuron is a relatively new discovery, but a more accurate discovery is maybe snake a neuron in here somehow, right? It's so complicated. So how do you even study something this complicated? Well, we've done a bunch of things where we've injected tumors in mice. We've seen that already. But what if we want to study a very specific aspect of the tumor? Well, as an engineer, you might want to think about breaking down the problem. So, the breast cancer microenvironment is flooded with macrophages and cancer-associated fibroblasts. These are producing a kind of barrier that keeps the tumor where it is, but also is not entirely straightforward because they also help the tumor in a lot of ways. So when we try and develop treatments to attack the macrophage and attack the fibroblast, to attack the tumor, again, the role of these are complicated in the context of a tumor, but they are supportive of the tumor. And when we design treatments, they don't really work. And that's weird. Because in theory, again, the theme of today is you can have the best idea and reality is just maybe different by accident. Maybe you did nothing wrong. Oh, take a sip. Oh, hot water's so good. Maybe it's because tumors are freaking complicated and you can't just study them isolated in a dish. Maybe this architecture, the structure of the tumor is actually important, but growing a tumor organoid is not exactly the easiest thing to manipulate either. Some people can, depending on the question they want to ask, but if you want to investigate how all these cells play with each other, you gotta try something different. So maybe the reason why targeting cancer-associated fibroblasts and the macrophages, maybe it doesn't work because if you purify them and you treat them individually, the complicated reality of tumor biology when they are talking to each other might prevent an effective treatment from actually working in a real animal. So crosstalk may sabotage these research efforts, right? The sum is greater, the whole is greater than the sum of its parts. And so maybe you need to grow a whole tumor, or maybe you can grow just enough of the tumor. That's an interesting idea. But we don't have the tools to build a tumor from the ground up, so how are we going to figure this out? We can't just use tiny little tweezers and manually place different cells in different places and build a tumor that way. We can't 3D print a tumor, not yet. But what can we do? And here I get the joy of introducing you to microfluidics. This one's for you engineers. So in figure A, we have the real breast cancer tumor microenvironment. There's the tumor cells in the middle and a bunch of immune cells and supporting cells around it. And say you are a scientist and you want to study how these cells talk to each other in a realistic way. And you want a high level of control. A high level of control. Well, you can build a microfluidic device that simulates the tumor microenvironment. This is what they're calling the triculture TMEC model, tumor microenvironment model. So in red here, you have layer 1. Here you inject tumor cells. L2, you inject stroma, right, supporting cells. You mimic this. This is messy and complicated. Here in a microfluidic model, you can mimic this by artificially producing these barriers. And then 3 media channels, food. In pink, that's how they get fed. So by fabricating a little microfluidic device, you can actually, to some degree, mimic the tumor microenvironment. At least the interaction between tumors and their supporting cells, the stroma. Very cool, love this. What does this device even look like? How micro is micro anyway? Well, this is the real size of this little device. Pretty micro, size of a penny. So how does this work? You inject tumor into the red region. It stops here because there's a semi-permeable layer, and then you add in stroma cells. They have some contact with the tumor cells, and then you feed them, you give them media, and then you let them go for 3 days. And again, we're studying cancer, so we're interested in metastasis when the tumor leaves their environment. So after 3 days, we should see those tumors punch through that barrier and escape into the stroma. Crazy idea. This is a crazy idea. Think of this. Humans are so clever. Having to teach, being forced to teach engineering at school, like as my previous before Phi job, really gave me an appreciation, because to me, I was like a purist. Clocker wasn't wrong, where I had a superiority complex, where I was like, nature is the ultimate engineer. How could anything be more beautiful than nature? Humans are pretty smart too. I do have to admit that. This is clever. If you want to study how cancer infiltrates and breaks through the basement to infect other regions of the body, constructing a microfluidic device where you can provide the tumor a space to punch through that membrane and study it in high definition is genius. It's genius. So the green region is going to be consisting of cancer-associated fibroblasts as well as tumor-associated macrophages. Those are the principal components of the support cast of these cancers. So here's what this experiment looks like. So you make the thing, you grow some macrophages, make sure they're ready. Then you inject your cancer, you inject your macrophages, you inject your fibroblasts, all into a hydrogel, because we're going in 3D now, right? We're trying to mimic the cancer as well as possible, so we're injecting them into a hydrogel to mimic the 3D environment of the cancer. And then we're going to do a bunch of science. But that's what this is so far. So here's the research question. How do tumor cells, macrophages, and fibroblasts interact to promote tumor invasion? We get to watch this, and it is so cool. So again, to review, in red, tumor cells. In green, the support cast. So we have different conditions: tumor cells only, tumor plus macrophage, tumor plus fibroblast, and then all three cell types. So how does the presence of the supporting cast surrounding the tumor microenvironment, how does that influence cancer's ability to become metastatic? This is cool. So here, on day 0, monoculture, this is cancer only. And you can see it's grown up to that barrier. And then you can see it punches through that barrier in red. They're labeled with M cherry. So pay attention to this. Day 0, day 3. Only cancer. And they punch through that barrier, and this is how far they get. What happens when you add fibroblasts? So day 0, just the tumor cells, and then the supporting cast, and then now look at how far the tumor has invaded. Macrophages, and then all three, tumor, macrophage, and fibroblast. Look at how far these cancers have grown into the surrounding tissue. Can anybody tell me which condition is the cancer the most aggressive? Can you tell? Is it cancer alone? Cancer plus fibroblasts, which is that second column? Cancer plus macrophages, the third column? Or cancer plus all three? Which condition is most severe? Looks like all three. They do all have invasion, but I am telling you, one of these is worse than the others. And here's a hint. The white dashed line indicates the maximum migration distance of the tumor cells. So it's cool to look at the red, but they took the work out of it and just compared the size of the circle drawn by the dotted white lines. Which condition yields the most aggressive tumors here? It's an interesting one. I like this picture a lot. Oh. Gotta hydrate. Oh, it's good. It's good. So Spot says try, and I would agree. Right? It looks like when you have all of the components of the tumor microenvironment that we care about, the macrophages, the fibroblasts, it looks like the cancer is able to invade a lot deeper into the surrounding tissue. So what does the data look like? Here's migration distance, right? If you add fibroblasts, you get deeper invasion into the surrounding tissue. If you add macrophages, you get the same thing. But when you add all three together, there's a big difference. Looks like this is like 450. Looks like this is about 750. It's about, a little less than double the distance. It is much more capable of invasion when you are growing all three of these cell types. That's compelling. And then when you normalize, you actually find that the only condition that matters is actually the try condition when you do some normalization. So as a bonus, only the triple composition affects tumor proliferation too. So it's not just about migration, but for some reason, having all three is the only condition that causes there to be more proliferative cancer cells. And that is the perfect example of why sometimes the whole is greater than the sum of its parts. Remember, cancer plus fibroblast grows the same. Cancer plus macrophages grows about the same. Cancer plus fibroblast plus macrophage, all of a sudden they're cooking up a stew, and that stew's name is cancer proliferation. And I don't know what to make of that. Macrophages and fibroblasts and cancer cells, they're talking to each other. And it's that conversation that makes this more deadly. So, we know that when paired with fibroblasts and macrophages, tumor cells move deeper into the surrounding tissue. But what could explain why tumor cells move deeper? Speed? That's certainly an option. So, does speed matter? We get to watch a video. These are cancer cells in red, and they are growing. And these little colored markers are just little pins that the computer put on them so that you can track their movement. And so when I play this video, does it look like speed is all that different? I mean, this is one sample, right? This is just how the thing is done. The data is here. Thank you so much, Frost, for hanging out. I'm up 4.5 hours after your usual bedtime. Go to bed. I'm the only one that has to stay up, but thank you so much for the support. It's awesome that you were here for so long. Spot says, can we eavesdrop on that conversation? I would love to. That's what the authors of this paper want to do. So looking at the speed of migration between these different culture systems, I don't really think that we can conclude that, we know that the tumor cells move deeper into the stromal tissue, but when we really look at them, they don't really move faster. So chat, I know it's late at night. You might not want to think about it, but some of you might be in time zones where you might be more awake than I am. So tumors move deeper into the stroma when you culture them with fibroblasts and with macrophages. Cool. But they're not faster. So what the hell is going on? What the hell is going on? Anybody have a guess? If it's not speed, why would the cancer cell, or how would the cancer cell move deeper into the stromal tissue if its speed isn't affected? Any ideas? This one's kind of tough, especially if you are like me and have been awake since 7 a.m. this morning. I actually didn't sleep very well because I was so anxious about this stream. So I have an excuse to be tired. What about you, chat? I shouldn't be razzing chat. You're all very kind. I don't want to make you feel bad for hanging out with me. I'm just so tired. This is a wild guess, but maybe polarity. Maybe it's dividing specifically in an outwards away from the tumor direction. Not a wild guess. Not a wild guess at all. I'd call that directionality. And that's exactly what the researchers thought to look at next, because kind of weird. You would think that maybe they were faster, but in this weird case, they're not actually faster. What about directionality? So monoculture, you've seen this video already, and this is the triculture. This is hard to see, so I'll play these a couple of times. And, I don't really love this video, but take a look at the paths these cancer cells took between these two. Remember, the lines are being traced. What would you say is going on in between these two? And again, I don't love these videos, but this is their data, and we're just gonna have to trust that they did the analysis correctly. But this guy up here, this guy moved. He might not have moved very quickly, but he did move in a direction. You can tell that there are tails. Whereas in the monoculture condition, it just looks like Brownian motion. They don't ever really leave their initial spot. They kind of move in circles. Here, in the triculture system, they move within, you can't really say intentionality, but they do move in a direction, right? Like, these guys are in circles. Brownian motion will tend to do that just through random chance. But like green, he might be growing in the wrong direction, but he's determined. The red guy's moving out. Purple guy is moving to the side. Like they are moving further away from their original destination. They do look a bit more directed. So it does look like having stroma, have both macrophages and fibroblasts, helps cancer find their way into the stroma, maybe. Chemotaxis, yeah, maybe. Maybe there's a chemical that the macrophages and the fibroblasts are spitting out that cancer cells are drawn towards because they're not going faster. They just tend to have a bit of direction to them. And you can see that in the actual data. I don't know how they measure directedness. I'm just going to trust that it makes sense. But yeah. And it looks like macrophages contribute a bit more than fibroblasts do to this effect. So what the heck is going on? The researchers don't go directly to what is the stroma spitting out, but rather how is the stroma structured. And so they just take a look at the different structures of the kinds of cancer. So they image them. And I'm just going to say this, but you can kind of see. So I don't actually remember what the difference between these images are, top and bottom. I think it may be different regions. I think this is the interface. This is the barrier between cancer and then the supporting cells up here, I think. And then this might be just the stroma itself, although I could be wrong on that. But the idea here is that in the monoculture, you see that the cells are kind of small. When you start giving them more supporting cells, they grow to be longer. So maybe there's, because the cancer cells get longer, they might be moving in a more polar direction. At least that's a hypothesis, I think. So do a bunch of classification stuff that I'm not going to spend too much time worrying about, but they're noticing and they're being very observational about this, right? They're trying to find out what the differences are between cancer cells that are grown with and without these supporting cells. So they're looking at everything about the cell they can, including their shape. So they break shape down into a couple of different categories, including circularity, aspect ratio, and protrusiveness, which they've illustrated here. Hi, first time to your stream. Thank you so much for tuning in. Hell of a stream you picked. This is not typical of my content. I want to go to sleep so bad. But we are not at the $2,000 milestone yet, and I must continue. But it is only 1 a.m., 1:20 a.m., and oh my god, I'm so tired. 8 more hours. Oh, the idea of stopping sounds so good right now, but I can't. So they're noticing that depending on what the cancer cells were cultured with, fibroblasts or macrophage, they have different shapes as determined by circularity, aspect ratio, and protrusiveness, right? So I'm not entirely sure what to make of that. Smarter people than me might, but it is interesting generally to know. Cancer is actually my biology project. Awesome. Maybe I can gain more insight from my chart. That would be lovely. That would be lovely. So we've got some interesting observations of the effect of the stroma, which is the supporting cells of the tumor, on breast cancer invasion. Thank you so much for the $25 donation, Silvat. Let me check if you redeemed incentive. Okay, so I owe you a joke that I will do at the end of this paper. Okay, but the shape thing is mostly just trivia. Just trivia. Whether or not it comes into play later is a little unknown, but we gotta do something about this cancer, y'all. So it's time to find some targets. And what's our favorite thing to do when we try to find targets? That's right, it's RNA-seq. Let's find out what mRNAs cancer cells are making when they have supporting cells versus when they don't have supporting cells. This time in single cells, as Spot has correctly called, single cell RNA-seq. I fell asleep. Did we solve cancer yet? We're getting closer. So, how are we going to figure out which genes are involved specifically with a 3D cancer's ability to invade the stroma? And they come up with a very clever experiment. They're going to be sequencing both 2D culture and 3D culture. So 2D culture is a flat sheath of cancer cells. You grow them on a glass cover slip or a flask, and they grow in a neat layer. 3D culture, you grow in a hydrogel, where they literally grow in 3 dimensions. So to figure out what genes are turned on in a 3-dimensional environment, when you do have supporting cells and when you don't, figuring out what those unique genes are going to give us an avenue of attack, which is all about, that is the entirety of breast cancer drug design, finding these targets. Okay, RNA-seq allows us to find out which cells are expressing which genes at which levels. Not brain enough to tell you about this, but basically you can do your microfluidic setup, culture your cancer, either have them with supporting cells or without supporting cells. You can extract their DNA, extract their genetic material, and then sequence it. It's fancy stuff. You'll learn this in undergraduate or grad school. And then you can analyze the data. Okay. So, volcano plots. These are data that tell you whether or not a gene is statistically significantly higher or lower in a particular condition. So, we have 2 categories here. Either, what is this? I need to reread this. So, each dot is a different gene. X-axis is fold change from 2D culture in a dish versus 3D culture. So we're looking at what is more pronounced in 3D over 2D. And then Y is significance. It's not magnitude, but it is statistical significance. So in the 3D TMEC high, these are genes that are more highly expressed in 3D culture than in 2D. That's on the right. And then blue, genes that are lower in 3D culture than in 2D. So the volcano plot is important, but you can't really learn a lot from just the volcano plot. This has got to be transformed into something else. So what are we turning this data into? Well, each dot was a gene, and it's really hard to have a human mind comprehend that, so we organized them into pathways. So now each dot is a different pathway, and now you can kind of see what pathways scientists are interested in. So there's TNFA, that's a immune thing, very scientific. Immune system pathways, inflammatory pathways, cytokine pathways, right? So each dot now is a group of dots. And we can start to figure out whether or not there's a valid target to hit here. And when you take a closer look as to these genes, you actually do start finding that there are some pretty interesting targets made available here. And in particular, the scientists really care about this target, KYNU, KYNU. No breaks. I don't even have $5 to donate. Hey, if you're here, you're contributing, right? Your audience participation in the stream is not only keeping me alive right now, because boy, do I want to apoptose, but it juices the numbers, makes this a bit more of an occasion. Next year, maybe it'll be bigger and we can do more and better things. I took a 15-minute break for lunch and a 15-minute break for breakfast. Or dinner or whatever. And I had a couple of breaks where I hung out with my friends and we did some science adjacent content, trivia, stock photo, game, Nobel Prize tier list. So I guess it's more accurate to say I technically did 10 hours of science. That's a bit more true. Yeah, Jack Stat, big fan favorite in the biology world. But these particular researchers are more interested in KYNU. And I don't know anything about this gene, but they love this thing. So let's learn about why they chose to focus on this guy. You can see that it is higher in the triculture, lower in the mono. So that is why it draws their interest. Kainu. This is kainurinase. This gene produces an enzyme that cleaves a metabolic byproduct of tryptophan. And this is a key step in one biosynthesis pathway to make NAD+. That's right, the citric acid cycle strikes once more. Again, I told you, the citric acid cycle deserves an S tier placement because it shows up everywhere all the time. I'm a little behind, but what is 3D and 2D culture? Yeah, that's a great question. 2D culture is when you grow cancers on a dish, like a flat glass thing, 2D, one layer of cells. 3D culture, you grow them in a hydrogel. That hydrogel allows the cancers to grow in 3D instead of 2D. A Thought Emporium collab. I'll say this. I don't want to... My feelings on the Thought Emporium are complicated. So I won't go into further detail. I think Thought Emporium is a great content creator. There are certain aspects of the way the Thought Emporium does content that I do not personally approve of. So while I do acknowledge that he is a good biology YouTuber, I don't want to get your hopes up. It's not happening. There's no drama between us personally. Like, I've never talked to the Thought Emporium, and don't like stir up anything. Like, I don't want to. I've never made a video expose or anything. There are certain aspects of certain videos that I don't like, and please respect my rules and like don't bring it up. Don't Fi is beef with. No, I don't have beef. Okay, there are personal choices that he makes that I don't agree with. That's all. We don't see eye to eye on how we do YouTube, and that's okay. We can have differences. No one's necessarily right, necessarily wrong, but we don't agree, so collab is probably not happening. Okay, with that in mind, back to kainurinase. So, they're interested in this enzyme. It's part of tryptophan biosynthesis pathway. For our interest, this particular pathway is upregulated in immunosuppressive conditions, potentially meaning that when cancer cells are allowed to talk to macrophages and fibroblasts, maybe they suppress immunity. It's pretty interesting. Pretty interesting thought. So this already is a part of the cancer story. Now it's showing up again, weirdly, in this model, this microfluidic model that we have here. It's kind of a wacko theory, right? It seems a little far-fetched. This thing is involved in tryptophan catabolism, but like, why? Like, I'm not a good enough cancer biologist to see the line between tryptophan catabolism and cancer metastasis. It seems so indirect. Like, NAD+ is just needed for regular cellular processes too. So is this just noise? Like, what is this? Why do we care here? Well, this is patient data. And what we see is that the genetic pathways that are involved in catabolism leading to NA production actually do have an impact or are correlated with survival outcomes, right? When you have a lot of these genes turned on, you can actually see that patients do not survive as well in both of these databases. Thank you so much for the donation, Pifby. Thank you so much. Thanks for the hard work, Fi. Here's a bit of ATP to keep you going. Thank you. I could really use a bit of ATP. Thank you so much. So, even though I don't see the connection, the fact that there's a correlation here makes it worth investigating. I think that's fair. It's like barely significant in the metabrick, but technically is true. Okay, this paper does a lot of things that I'm skipping. Okay, so they do a bunch of cool things, including tracing macrophage development through pseudo-time. Okay, very fancy theoretical stuff. Basically comparing macrophages in a 2D environment to the 3D with or without fibroblasts, and seeing which genes turn on in which conditions, right? Fancy things, I'm not going to explain them. They make my brain hurt. Mapping out complex interactions between the different cell types. It's a lot, it's a big part of the paper. How come all these papers have a computational element? What happened to the good old days where we were doing classic mole bio? All this computational stuff makes my head hurt. I admire you if you are capable of doing this kind of content, but I can't. I'm not smart too. Okay, but chirinase came out as a potential target, and as Anna is pointing out, it is a cause, maybe a case of correlation and causation, right? So far, it's come up a lot, and the data is there, it's tempting. And that's why I say here, it is a potential target to hit. We still don't know if hitting this does anything. So we do know why this is tempting, though. This gene is only upregulated in tumor cells in a 3D environment when having contact with macrophages and fibroblasts, and patients who end up having more of this gene end up faring worse than their counterparts. So let's hit this gene. Let's see if hitting this gene prevents tissue invasion. So thankfully, we already have a drug to hit this enzyme. It's called RO618048, and it is a drug that will inhibit the action of this enzyme. So if we can knock down this enzyme's activity to a normal level, could we stop tumor invasion? So the hypothesis here, we attack this enzyme in these tumor cells. And if we attack the tumor cells with this inhibitor, hopefully we can stop metastasis from happening. That's the goal. That's the hope. That's the prayer. So we have our microfluidic device. We're setting it up. Mono, triculture. So this is cancer alone. And in the tri conditions, this is cancer plus stromal supporting cells, the membrane. And then we're going to hit this enzyme with this drug over a different set of concentrations. And we're going to see whether or not we can stop migration from happening. That's the basic gist of things. Now, this is a better online class than any other. I wouldn't compare me to your teachers. Your teachers have a job to do. I'm doing this for fun. I can be fun and entertaining because there's no pop quiz at the end of this, right? Your teacher has to worry about you actually remembering all the things that I'm telling you. And not saying that you're going to forget, but I don't have to worry about if you'll forget. If you were actually my student, I would have you do a lot of quizzes, because if my job were to make sure that you internalized and memorized this, that's different. What I'm doing is keeping you engaged with the field of cancer biology and biology in general. That's a different job than your teacher. We shouldn't be compared. I respect your teachers. So, what's happening here? So, here we go. No drug, okay? Just the cancer. You see that doesn't really invade. You add that tri, and you get a lot of invasion, right? We know this. This is old data, just with a different skin. Now, chat, what do you suppose happens when you add the drug? The drug that supposedly kills this potential metastasis. What do you expect the effect of this drug to be, if everything is correct? Hopefully no difference between mono and tri. Mono same as tri. I agree. If this drug works, this cancer should not be able to invade the tissue. Are you right, chat? Tell me. Are you right? 5 micromolar. Relatively low concentration of this drug. How does that affect migration? Worked somewhat, I would agree. That circle's a lot smaller. 20, and we're getting even smaller. 100, teeny tiny. Awesome. So even at low doses, drugging kainu reduces the increased aggressiveness tumors invade with when they are co-cultured here. So it looks like kainu maybe not a bad target for metastasis. So just to go over the data, mono and tri, we already saw, that's this picture. A lot more aggressive migration in the tri. But when you hit it with a drug, it's all the same from there. Thank you, Anonymous, for the $5 donation. What's your favorite non-science hobby? Thank you for making immunology so engaging. The video about prions is one of my favorites. Thank you so much for the $5 donation. Thank you for being an audience member. My favorite non-science hobby is probably being a dungeon master. I love playing D&D, dude. With a group of good friends, I love storytelling. That's maybe not a surprise. Biology is the greatest story ever told. Physicists might disagree. They would say maybe the story of the universe is better, but me, I'm a humble man. Life on Earth, that's all I care about. And the story of biology is the story of evolution. The story of immunology is the story between, the struggle for existence that Darwin so poetically wrote about in his book. Stories. I love stories. And as a dungeon master, I get to write stories without actually being good at writing. I tried writing a book one time because I love stories that much. I'm a terrible fiction writer. I don't know how to write fiction. But a dungeon master, I could do that. I could play D&D. That's probably my favorite hobby outside of biology. Thank you for the question. Oh, goodness. Okay, so we've reached the end of the paper. And I love this paper. It's great. A new little tool to study the conversation between tumors and their supporting cells. And from that conversation, we eavesdropped, as it was so poetically described earlier on in chat. We eavesdropped on their conversation, and what is that? Oh, cancer cells, when they're listening to macrophages and fibroblasts, they're making an enzyme involved in tryptophan catabolism. Who knows what the heck that's doing? But for some reason, it's important for cancer migration. And when we hit that enzyme with an inhibitor, no more migration, no more metastasis. That's something that would not have been achievable in 2D culture. This 3D tumor on a chip system is amazing. And this is not the only lab that's doing it. We are getting to understand in finer grain detail more and more exactly how these interactions happen. As a former neurobiologist, I'm assuming you've seen the comparisons between neuron organization and the structure of the brain with the organization of galaxies and dark matter filaments in the large scale of the universe. Yeah, I love neurons. Love me under a favorite cell type. Even though I do a lot of immunology on my channel, I'm an immunology hobbyist. My professional background is in neurobiology. Love me a neuron. So, triple negative breast cancer cells are getting signals from both macrophages and fibroblasts to direct them towards invading tissue. This interaction is not merely additive, it's synergistic. They're having a three-way conversation. Kainu emerges as a target to hit, responsible for tryptophan catabolism to NAD, and somehow is bad for cancer patients. Not entirely clear why. But hitting this has been shown in a dish to be pretty promising. And again, funded by the Breast Cancer Research Foundation. I'm very excited to see if this works in a mouse. Favorite cell type is a neuron, not a neutrophil? Okay, you're a human. Are humans your favorite animal? Maybe they are, but my guess is that your favorite animal is not a human. Okay. My God, okay. I'm so tired. I'm gonna take a quick break. I'll be back in a couple minutes as I hydrate, use the restroom, stretch, stand up, make sure I don't get any blood clots in my legs. I will be right back for investigating the relationship between obesity and cancer, something that I feel very strongly about. All right, so don't go anywhere. I will be right back. All right, I'm back. I'm not gonna do coffee. I had a tea in the morning, and we're gonna power through on sheer willpower alone. Because I need to be able to sleep after all of this. If I have a coffee now, I'm not sleeping, and I really want to sleep. If anybody has a spare $1,000, I'll just go to bed, take a quick nap. But if you have a spare $8,000, I'll just end the stream right away. Um, let's talk about the way that fat and cancer interact, because it's important. But I'm going to do it my way. I'm going to do it my way. Might sound different than your doctor, because I think I have a pretty unique perspective here. So, I think this paper was also funded by the Breast Cancer Research Foundation. I don't have the figure up here, but I think it is, if I remember correctly. So, this paper is called "The Non-Steroidal Anti-Inflammatory Drug Sulindac Reverses Obesity-Driven Immunosuppression in Triple-Negative Breast Cancer Progression." This is something that I'm pretty passionate about. Before I decided on neurobiology, I actually was really interested in the biology of fat. So this is personally meaningful for me. So obesity is a known risk factor for cancer. Fat cells are known chemical signalers, which can heighten the risk for cancer. And from the way that modern society is going, it's not going to be a problem that goes away anytime soon. 1 in 8 people in the world are obese. 2 in 5 adults in the USA. And cancer isn't the only health problem obese people face, and I'm sure you don't need to be reminded. Okay? So fat cells do all sorts of crazy things: inflammation, impaired immunity, and they also do a bunch of growth factor hormonal signaling, right? A lot of things that cancers love. But I'm not here to guilt people. I don't want to do that. This is an objective look at the existence of fat and its relationship to cancer. So obesity is a complex, nuanced metabolic condition. And I want to have a little soapbox on this because this is a personally meaningful journey that I've been on before, because I used to be a pretty fat phobic individual. I'll admit that. I believed obesity to be a moral failing. Just eat better, exercise. It's just that simple. I no longer think that way. I fell for the meme of calories in versus calories out. It's a very common refrain. Makes sense. It's thermodynamics. If you eat less than the energy you burn, you will lose weight. And you're right. You're absolutely right. But science isn't that simple. Thermodynamically, that's a solid argument. Yeah. Eat less, move more, you'll lose weight. True. Let's talk about being humans. That's not easy. And different people will have a different propensity for losing weight, and they will have different propensities for metabolic rate, right? Completely ignores the genetics as well. Yeah. I hate this argument, calories in versus calories out, because it is technically correct only from a physics point of view, not from a biological point of view. It's so simple that it's actually kind of useless, because again, I know that excess fat is a known risk factor for health, but I will ban you for moralizing obesity. I feel very strong about this, okay? Because I've grown as a human. So why is calories in versus calories out a bit of a meme? Well, if you look at the formula here, weight loss, yeah, it's true. Calories in versus calorie out, I agree with that. But this ignores the nuance. How easy it is for different people with different physiological, biopsychosocial backgrounds to regulate diet. And at the same time, is calories out the same for everybody? I know the food that you buy, it's standardized for a 2,000 calorie diet, but let me tell you, if I ate a 2,000 calorie diet, I would gain a lot of weight pretty quickly. I don't eat 2,000 calories a day. Just not my metabolism. But if I ate like I were a 2,000 calorie kind of dude, it would not look good on me. You know, it's not appropriate for everybody's biology. So, what's going on here? A combination. Kirkazada has a beautiful video on this, but yeah, weight loss is not just eat less and work out more, because humans are not that simple. It's technically true, but it is not feasible. It is difficult. And I think a part of maturing as like a human being is acknowledging that if something is hard, you just don't do it. It takes a lot of effort to do hard things. And instead of guilting people into doing something that is hard, I think it makes more sense to lower the barrier, make the hard thing a little less hard. I have a lot of sympathy for obese people. I do. Because when I eat a pizza, I'm full, dude. My body says stop. And it might say stop tomorrow too, because I get like sick if I overeat. Some people don't have that. You never really think about that, do you? You know, when you're thinking about obesity, it's complex. There are many reasons why somebody might be, obese. Could be a metabolic problem, genetic problem, a psychological problem. Maybe food is a crutch. Maybe you need it to feel comfort. Everybody's different. But I know that I can maintain a healthy weight just by listening to my body. Not everybody can do that. Not everybody can do that. It's important to keep that in mind. Yeah, whole pizza is not a typical thing. And as I age, I am less capable of doing so. And I am always punished for it. Like my body told me I did something bad. Not everybody's body tells them that, right? That's an important thing to keep in mind when it comes to eating, obesity, etc. So I'm sure my audience already knows, but just for me, keep that in mind. That's my soapbox. So, what are some methods to mitigate obesity-related contributions to breast cancer? And yeah, I think someone in the chat said weight and fat, it's correlated with each other. Not necessarily the same thing. Technically, the correlation is with obesity, having more weight than is required, which doesn't directly connect it to fat, but like probably fat. Probably fat. I don't imagine the same risk factors to be of concern if we're talking about bodybuilders, for example. Although I could be wrong. It could be the weight itself causing pressure and strain on internal organs like the heart that might cause inflammation or something. Like I could imagine it being weight and not fat as well, but I think it's probably fat. So diet and exercise, yeah. Oh yeah, dude. If you are obese and you do diet and exercise and you keep it up and you lose that weight, dude, I am so happy for you. You're a rock star. You have the discipline and the time and the strength, and it comes easier for you. I'm happy for you. Do it the all natural way. That's awesome. Not a lot of people can do it. That is just, I'm just saying, the numbers speak for themselves. So, sustained weight loss for people who are obese is horribly complicated. There are all sorts of complicated psychological ideas that are baked into the process of losing weight. And again, I was fat phobic before. I'm not saying it's healthy to be fat or healthy to carry that extra weight. I'm not. But I'm saying we can maybe treat it differently than being a moral failing. I don't think fat people are lazy. I just think maybe your body's doing something that it could be doing better, as most of our bodies can. Nobody's body is perfect, but in their particular case, it's probably metabolic related. And if you can go on a fitness journey and a diet journey and you can lose the weight without ever having to touch a drug, that's awesome. But let's also take the time to acknowledge that losing weight is hard. And there's all sorts of psychological challenges that prevent people from keeping the weight off. positive things like, losing weight, it's exciting, gives you a sense of purpose and new community opportunities. But also, when you lose that weight, you always have that fear that you will bounce back. And that fear can be kind of anxiety inducing. Loneliness, there's some loneliness too. Frustration when you don't lose fast enough or to your expectation. Criticism from all sorts of different places. And feeling deprived. Food is beautiful. It's lovely. And having to keep a strict diet sucks. So I can't blame people for not losing the weight. I would probably find it difficult too. Even if not obese, having a good diet and exercise is great for the body. Yeah, do it. Everybody should have a healthy weight, exercise and diet. It's better for more than just weight, right? Exercise is pro-inflammatory in all the right ways. Helps your immune system. Helps declog your arteries. All sorts of great non-obesity related things come from diet and exercise. Wonderful things. Diet and exercise, not super good by the numbers. People fail at it more often than they succeed. And I view that as a health issue, not a moral issue. Not that you didn't work hard enough, because it's hard. And I can't blame you for that. Food is great. Was there a trigger for you to lose weight? The trigger for me was puberty. I got lucky. That's why I'm so humbled. I was a fat kid. And about the time when I hit puberty, my mom signed me up for back-to-back swimming lessons. So, as a fat kid, I had to be thrown into the pool, and I would swim for an hour, get out of the pool, and then I would get right back in. And the instructors would look at me like I'm crazy, like, class is over, right? I'm like, oh, no, I'm actually here for simultaneous lessons. And as a kid, this is where I got my sense of entitlement from, is when I was a teen, I was like, well, I was fat, and then I, just swam a bunch, and then I got skinny after that. Now I'm like average, maybe even a little on the side of heading towards overweight. But as an adult, correlation is not causation, right? My mom signed me up for back-to-back swimming lessons right around when puberty hit. So, as an adult, knowing what I do about biology, was it swimming that made me lose weight? To a certain degree, yeah. Is it the reason why I was a skinny high schooler? No. Because I didn't swim every year in high school. I swam for a couple years and then I stopped when I was in high school and I just kept on being skinny. Why? Genetics. Lucky. That's all. Not better than anybody, just lucky. So, move more, eat less. Good for your health. By the numbers, not that effective to lose weight. So the data vary quite a bit, but many studies show that the majority, more than 50% of obese people do not lose weight sustainably. When the weight is lost, it's usually not that much. And you can, depending on your attitude, you could be like, obese people are so lazy, whatever. No, this data to me, when I read this data, it shows that people are trying. Come on, people aren't dumb. The extra weight is not healthy. People know that. They're trying to lose it. But doing it through diet and exercise is really, really difficult. It's really hard to keep off because your body doesn't want to do it. And so there's an interesting dovetailing with cancer here, where extra weight can cause inflammation, which can lead to cancer. A type of drug that you might have in your cabinet right now are the NSAIDs, like ibuprofen. They can block inflammation. So this paper is asking the question, we know that obesity causes inflammation, which can lead to cancer. Can we prevent cancer from getting worse by including an NSAID in our treatment regime? Interesting question, and a cheap one. Ibuprofen is cheap. These are not expensive treatments, and I like this approach. So, we know that diet and exercise, dude, if you can do it, you're awesome. Congratulations. I love that for you. Not everybody can. Not everybody can. Some people go the bariatric surgery route, which is actually a really good option for keeping weight off. It's much more effective than diet and exercise. You get a bariatric surgery to remove the excess weight, and that keeps more people in, you could call it obesity remission, than diet and exercise does. However, again, I am motivated by accessibility. And this just isn't that accessible, right? It's surgery, it's expensive. And the very virtue of being obese makes any surgery more likely to cause complications. So that's not really an option for a lot of people. Not a safe one, at least. So bariatric surgery, maybe not really the route that people want to go down. Not everybody, at least. What else do we have? And as Pepik just brought up, Ozempic. Yeah, GLP-1 agonists, Ozempic. Cheaper than surgery, but not ibuprofen cheap. 200 to 400 USD a month without insurance. But we don't know if GLP-1s actually do anything to cancer. Maybe they do. Hey, I wouldn't be surprised if we learned that GLP-1 agonists also have anti-cancer effects as well. This drug seems like a miracle. And if you're a European bro, okay, get all your American healthcare jokes out now. Yeah, our insurance sucks. Whatever. I'll own that. Healthcare is terrible. Not because of doctors, but because insurance is terrible. I've seen healing cancer with ibuprofen as a meme once. Well, it's important to know that the NSAID that they're using in this isn't actually ibuprofen. I just said ibuprofen because that's the NSAID people are more familiar with. And again, it's not... I think in this presentation, I have a lot, like, go to your freaking doctor, dude. Don't take medical advice from a Nutrafol online. And I'm explicitly telling you that this is a very specific application, right? The hypothesis is that the NSAIDs can help with inflammation that's caused by carrying extra weight. If you are of healthy weight, taking an extra drug might just upset your stomach or cause other bad side effects. This should not be considered preventative care, right? For healthy people. I am not going to read Holiday your comment. We don't say that word. We don't say that word in my universe. I think you know which one. Okay, so weight loss isn't cheap and it ain't easy, and we don't really know what GLP-1s do for cancer, although I'm optimistic. What about NSAIDs? Like ibuprofen, Advil, aspirin. These are non-steroidal anti-inflammatory drugs which might counterbalance the inflammation that is produced by carrying extra weight. Oh, to address your comment, Holly, I have a rule that to keep my content, my channel, everything drama-free, we don't name names. We don't name politicians, we don't name political parties. We talk about science, we stay within science. So, there is a name in your previous comment, and we're just not going to talk about that. So the appeal here, and it's something that I appreciate the Breast Cancer Research Foundation for looking into, is that, a lot of cancer treatments pretty expensive. Ibuprofen pretty cheap, It could be a cheap addition to an anti-cancer therapy strategy designed by your oncologist. Don't just start popping Advil. Advil has consequences for your stomach lining. As Pepik brings up, are there any problems with chronic doses of NSAIDs? Yeah. Your stomach will not be happy. At the very least, there's probably other stuff. Go watch Chubby Emu. A man took 98 tablets of ibuprofen, and this is what happened to their kidney, right? I'm sure you can find the answer on Chubby Emu's channel, an actual real doctor, and not a scientist like me. Okay. So, the middleman. What about ibuprofen as a aspect of an anti-cancer therapy strategy? Not sufficient, but a part of a treatment strategy. That's interesting, I think. So, today's star of the show is Sulindac, which is not ibuprofen, but it works on a similar pathway, or thought to be. We still don't actually know what these NSAIDs do, which is crazy to say in the year 2026, right? How long have we gone not knowing how Advil works? It's crazy. But the hypothesis is that they work by inhibiting COX-1 and COX-2 enzymes, which limit the production of prostaglandin, which limits inflammation, which can allow for some pain relief. Sometimes this drug is even used in a combination therapy for certain patients with a high risk of certain colon cancers. So this is not new to the cancer scene. It's new in breast cancer, but it is not new for colon cancer. So this hypothesis has some juice to it. I think it's worth investigating. Yeah, we don't know how Advil works. Again, we have an idea that it's probably doing something with COX-1 and COX-2. I'm not entirely sure. I don't know why. This is the common, maybe we do know, and this is like a common urban legend, but whenever you look up the mechanism, it's like, this is hypothesized to work in these mechanisms, and I just don't know why they haven't figured it out yet. I mean, it's safe and it works, and it's kind of a waste of money to figure out how it works, unless it's really important for human health. That's the difference between medicine and biology. Biology, we want to know everything. Medicine, we want to know it works. Advil works. Is it safe? Cool. Print it. Go for it. So, Sulindac, definitely worth talking about, I think. Yay, there's my chubby emu link. Don't be a freaking idiot, dude. I'm not a doctor. This is not medical advice. Because this is very tempting, right? The wrong person watches this stream, they're downing ibuprofen to cure their cancer. That's not how this works. This is a basic science paper. Don't take NSAIDs to treat your cancer unless a doctor tells you it's a good idea. A doctor with a degree in medicine. So if Dr. Phil tells you, maybe don't trust him. Every medicine has a trade-off, and your doctor will know if it is worth the risk for you specifically, right? So Advil, I mean, it seems like it's pretty harmless, but again, you take enough of that over a long enough period of time, and it's bad news for you as well. Your oncologist will know the relative risks. And if you are high risk for colon cancer, then your doctor might believe, hey, I think you would probably rather have an ulcer than colon cancer. And you'd probably agree. But for most people, it's like, if you do this, you're gonna get an ulcer, and you're probably gonna get cancer at some point too. It's just not gonna help you. So listen to your freaking doctor, okay? A man took 1,000 ibuprofen tablets in a month. That's crazy. People are nuts, dude. Okay, so NSAIDs in this case are kind of like a hammer looking for a nail. Okay, on paper, they seem like a great tool to prevent cancer, but in practice, their effect is small when tested with cancer patients, and there's significant risk to consider for otherwise healthy individuals, right? You don't want to just be downing NSAIDs for no reason. Again, see the Chubby Emu video. But how would you test if NSAIDs could reduce cancer aggression in a certain individuals, right? You be the scientist. You be the hey Purkinje, you be the scientist here. Okay. So we know in a randomized sample of all cancer patients, NSAIDs are not very useful. But these authors are hypothesizing that maybe we're looking too broadly. Maybe for obese patients, NSAIDs might be a strategy. And how would you try to test that? Hello, Purkinje. Purkinje's here. Purkinje's saying, I want to go to bed. Come to bed. And I'm saying, no. I have to stream for 7 more hours. Oh, she's rubbing the microphone. Double-blind clinical trial with obese patients, since the drugs are already safe and approved over the counter. but not approved for this. And we generally don't want to start with people. So maybe eventually double-blind clinical trial, sure. Right now, probably not. What can we do in the realm of molecular biology instead? Oh, I don't know if you can hear that, but Purkinje is batting the microphone with their tail. Like, shut this microphone off. Come to bed. Sorry. I'm talking about cancer. Test a control group with cancer who takes NSAIDs and one who doesn't. Another control. Yeah, sure. But how? What is the control group going to be? Who's in that control group? How do we test this? In the molecular biology field. What is our go-to move? Our ace in the hole. What do we do here? Are we tired? Are we tired? Use mice. Yeah, use mice, exactly. I could just go on through these You Be the Scientist questions because it is getting late. It is getting late. So, we start off with mice. We inject them with metastatic tumors. And then they're either given the drug or they're not, as Chad has recommended, except instead of people, let's start with mice, just to make sure. Oof. Am I remembering correctly that obesity increases inflammation? That's correct. That is correct. So, these mice, you give them cancer, you split them into two groups. High calorie diet, low calorie diet, you give them obesity. So these are not genetically predisposed mice. These are mice that started as a healthy weight and then acquired obesity, which does matter to a certain degree. So here's the data. Tumor volume on Y. Different colors. Control, regular diet. SUL for Sulindac, that's the NSAID. DIO for diet-induced obesity, so these are the obese mice. Then DIO plus Sulindac. And then EO771, that's the triple negative breast cancer line that they're using here. So, what effect do NSAIDs have? There's a lot to unpack in this figure. Just want you to sit on this for a little bit. I want you to sit on it while I take another drink. My throat is really feeling it at hour 17. What can we conclude about the action of the NSAID Sulindac on the tumor volume in obese mice? What can we say is the case? NSAIDs increased cancer growth, did it? Did NSAIDs increase tumor volume? Maybe the colors are a little hard to see. This purple up here, this is DIO, this is diet-induced obesity, this is just obese mice. And there's a redder one, this is diet-induced obesity plus NSAID. And then the two darker ones are control. Seems to reduce it. Seems to have helped quite a bit, reduced the tumor volume. Yeah. That's surprising. This NSAID, all it does is prevent inflammation. Like what you take for a headache. Only in obese mice did this reduce tumor volume, and it reduced it a lot. Threefold decrease, only in obese mice. The difference is not huge in the control mice, you can see. I wouldn't think too much about that because, yeah, if you are obese, look, obese mice, they get cancer pretty bad. That's a big tumor. It's a big tumor. And when you don't have that extra weight, it's not as bad for you, it's slower. So I don't know if ibuprofen would help, but the difference is not big anyway. So Why bother taking a medicine that could screw up your guts, right? If you take it long enough, right, might not be worth it for you. But yeah, Sulindac alone doesn't appear to have an effect on tumor volume, but it does have an effect only in obese mice. It's worth keeping in mind. That's the goal, right? This is a potentially cheap addition to a plan for treatment. Take this therapy and this NSAID, and maybe we can beat your cancer. What about metastasis? No more pictures of tumors, just the data. Sulindac plus minus, so the gray bar here is without NSAID, and then after, that's plus NSAID. And then here we have the obese mice. Gosh, that's crazy. Yeah, I mean, I don't want to freak anybody out who is obese because I've already talked about on my soapbox. Like, I get that it's hard to lose weight. I wish you well. But look at that diet induced obesity mouse. And again, mice aren't humans. This is an artificial system. But at least in this mouse model, 80% of these mice, after they've been injected with tumors, get lung metastasis. That's brutal. Cancer and obesity. Cancer feeds on that obesity. And when you give them the NSAID, oh, look at that. From 80% to 20. That's a big deal. And in terms of metastasis, it also looks like it might have an effect in the regular weight mice, but that difference is not statistically significant, so it's kind of a shoulder shrug. But it is cool. Right? I'm glad you think so, Drunken. It is cool. Reduced metastasis in both, which is kind of counterintuitive. It's not necessarily counterintuitive, it's just a little contradictory to the last figure, but since the difference isn't significant to begin with, I wouldn't put too much stock here. I also wonder how obese these mice were. I imagine the authors probably would have tried to make them as obese as physically possible without killing them to exaggerate the effects, yes. Probably. I don't know how heavy these mice were. It's probably in the supplemental. And that's a good point. Like, this is not meant to be a one-to-one comparison to human biology because these are extremes. These are extremes because if you want to get data, you kind of have to exaggerate. And that's not a bad thing because it lets you use fewer mice. You make the effect bigger, you don't need as many ends. Fewer mice have to suffer. I think that's okay. Not an easy call from a morality perspective, but hey, that's biology, man. So, it's interesting, isn't it? Again, Sulindac alone doesn't do anything in normal weight mice, but it does a lot in these obese mice. There's a lot. So, here are the effects. In our triple negative breast cancer model in mice, Sulindac has been shown to reduce tumor volume and metastasis in diet-induced obese mice. So what is Sulindac doing? I don't know. Time to return to our old friend gene expression, RNA-seq. What is the mouse making in response to these NSAIDs? I've shown this slide like 8 times today. I'm not going to do it again. Measuring RNA, the intermediate between DNA and protein. And when you cluster this analysis, and again, I am not a computer guy, so I've forgotten a lot about how this analysis works, but if you cluster these, patterns start to emerge. And you start seeing interesting patterns here. How does one attain obese mice? You get regular mice and you feed them a high-calorie diet. A lot of fat, a lot of sugar. And it's a little hard to read, but NES is Dio over condition. So, for example, I'm not even going to deal with this, dude. My brain cannot read a map like this right now. Just trust me that there's something interesting happening, and we'll go over it in the next figure. So, it looked like in that previous figure that one of the pathways that has changed in when you give these obese mice this anti-inflammatory drug is antigen expression. And this is near and dear to my heart. I love this protein. MHC, Major Histocompatibility Complex. I worked on it as a graduate student. So what is antigen presentation? Well, antigen presentation is the cellular program that enables a cell to tell the immune system that it's sick. Most cells make a protein called MHC, and as a part of the lifetime of MHC, it can pick up little bits and pieces of proteins that are acquired either from the environment or within the cell. So you can imagine if a cell is infected with a virus, MHC will display a fragment of that virus on the surface. And if a T cell doesn't recognize that as being belonging to you, you're getting blown up. Why is that relevant for obesity? That's kind of weird. That's why I love this paper so much. It's bizarre. So T cells are evolved to recognize these self versus non-self antigens. And if the T cell runs into this non-self antigen, it'll blow that cell up. Sick. Non-self antigens can also come from bacterial, viral, or fungal infections. They can also come from cancer. Some very clever T cells can kill cancers based off of the different markers that cancer cells spit out. So T cell immunity is a very important part of anti-cancer defense. But in obesity, apparently in these mice, they don't do antigen presentation quite as much. And I don't know why. That part of the picture isn't clear. What other things are different in these obese mice? Well, it looks like the antigen presentation, specifically in macrophages that surround the tumor, is also down, which can be really annoying because macrophages are supposed to be on your side. They're supposed to be showing off to the T cells and B cells and dendritic cells. Hey, look at me, I'm doing a great job. I'm sampling the environment and I'm teaching you whether or not there's a threat in the local environment or not. And their antigen presentation is down too. Bizarre. So, Sulindac might be playing a role in restoring this antigen presentation. Is that true? Well, we'll have to see. So, we know that antigen presentation is messed up in obesity. What about the T cells? So, I don't know how they measure species diversity. I'm not a T cell scientist, but healthy weight mice, that's the dark bubbles, right? Black and this purple. Healthy weight mice make a lot of different T cells. A lot of species diversity. In diet-induced obese mice, however, they don't make a lot of different T cells. The number's all the way down here. And it doesn't really look like, hold on, let me look at this more carefully. Oh, okay. So orange, diet-induced obesity, that's down here. Yellow is up here. Yellow is here. I couldn't see it because I'm so tired, but yellow is up here. So when you give an obese mouse with cancer this NSAID, what happens to their T cell species diversity? My brain just lagged there real bad. It recovers, yeah. And it recovers a lot too. It's on that line, right? It is as if this mouse was a healthy weight. It makes a variety of T cells. Immunity is restored. That's awesome. Other findings in this paper. Diet-induced obesity messes with tumors in all sorts of different ways. Antigen presentation, tumor, and also tumor-associated macrophages, T cell diversity. But they've also done other things that I'm not going to get to. Tumor microenvironment differences in metabolism, oxidative phosphorylation, and hypoxia. Dude, I cannot be bothered to think about metabolism right now. But I'm satisfied with this investigation into NSAIDs. And why is that? Do the NSAIDs reduce obesity? They don't. Just the inflammation. So, to sum, obesity offers unique challenges when it comes to fighting cancer. NSAIDs could provide a selective benefit specifically for obese patients. And again, don't be an idiot, talk to your doctor. Obesity causes issues with metabolism, inflammation, T cell homeostasis, all of which cancer would love. Cancer loves that stuff. NSAIDs like Sulindac could potentially serve as a low-cost therapeutic add-on for obese patients in particular. It doesn't seem to do much of your healthy weight. So, obese tumor-injected mice have a reduced tumor volume and metastasis, and they get back to healthy weight, back to their cancer returns to the levels of healthy weight mice after Sulindac treatment. It's amazing. What is Sulindac doing? We don't know all the details, but we know the end effect. Sulindac restored T cell diversity and helped antigen presentation function again. So again, tumors, tremendously clever. They don't want to tell cancer. They don't want to tell T cells that they're cancer cells. So what do? They rip off their antigen presentation proteins off of the membrane. They hide them. It's good for the cancer. That means the T cell can't fight them. But Sulindac, for some reason, don't know how this works, it's magic, puts that receptor back on the surface, allows the T cell to do its job again. And I love that because I'm all about accessibility, right? A treatment, engineering is only good engineering if humanity is involved. If a treatment is made for only rich people, I don't like it. How many more hours do I have to go? 7? 6.5. 6.5 more hours. If we don't know what Sulindac is doing, how do we know that the anti-inflammatory effects cause the effects in the paper? We don't. That's a hypothesis. We know Sulindac restored T cell diversity. We know it restored antigen presentation. Those things have to do with inflammation. You can connect the dots. But it's kind of a mystery. We know the end point. T cells are back in the game. Antigen presentations are back in the game. And also, cancer is not as severe in obese mice when you give them this drug. It's not direct evidence that it is because antigen presentation and T cell diversity, like they didn't link these mechanisms with better tumor outcomes for these mice, but I think that's probably their next paper. Have you tried with other NSAIDs? Not in this paper, at least. How long do things like this take to get for the research? Is it too inconsistent to give an average? What do you mean by that? I'm just not entirely clear as to what the question actually is. It's also in mice, so you can't really take too much away for humanity. But again, a measured interpretation is, oh, that's how it works in mice. Sure hope it works the same way in humans. But yeah, and again, funded by the lovely charity that we're donating to today, a way for obese patients to deal with cancers that are going to be more aggressive because they are obese, not their fault. Yeah, good paper. I like that. It takes a while for clinical trials to happen. Yeah, this is probably not going to hit a clinical trial for quite a bit. A couple of years, maybe. I think it's probably a bit faster because Sulindac is already approved for other things, so it might not take the entire 10 years, but I'm not a drug pipeline expert either. Okay. Half an hour until my friends join me for what I am calling sad boy hours. Because we're getting to that time We're getting into twilight hours. Where, you know, I already cried on stream, so I don't know. I'm just so tired. Maybe I'll cry again. I don't know. Anything can happen. But this next half an hour, this is just for chatting. Okay, this is just you and me, chat. Let's talk. I'm too tired to worry about if I'm being entertaining, so maybe this is actually a good chatting session. I think I owe somebody a joke too. I think I missed one, so let me think of a joke in this interim period. Uh, what joke can I make? Hmm. I'm so tired. I keep on trying to think of a joke, but the only thing that I can think about is I'm so tired. 17 and a half hours into stream. I am exhausted. 7 more hours to go, though. Oh, okay. VTheorem with the save. Yes, please. Tell me your joke. You thought of one before you went to sleep last night. That's awesome. Save me. I can't think of one. My brain is so toast. Hey, Bradley. Hey, Fi. I never understood how insane microbiology was until I came across your channel. Very happy to hear that. The generational E. coli study is probably my new favorite experiment, so I'm so grateful for this. Thank you. I think that video didn't get the love it deserved. Maybe it's my fault for not marking it correctly, but I love that video. You know, the idea of growing E. coli forever is just so interesting. So I'm glad that you are here joining us today. Want to hear a joke about potassium? Yeah, go ahead. Go ahead. Could chat have a stretch and a walk for me? Yeah, I'll get up and stretch. That's fair. I'll hydrate too. Oh boy. Oh my god. Oh boy. A little bit of low action, chill, calm action. I think it's probably fine. It's skeleton themed and it's gonna take 2 messages. I swear I've walked past that blasted ridge before, but I can't be lost, with my map. I'm a cartilage. Oh my god, these are bone jokes. Insight, and I won't clasp long all up bone in the wilderness. Oh my god, that's so funny. I'm terrible at bones. If I had to tier list parts of the body that I know very little about, I think bones are pretty high. But thank you for that. Thank you for coming in clutch, because I'm not prepared to come up with a joke. Spepik, thank you so much for the donation. Thank you. That's humorous. That's nice. Good job, Chad. That's awesome. What do you call fly with no wings? Oh, sorry, your potassium joke. Okay, yeah, that's a good one. That's classic. That's a banger. What do you call a fly with no wings, Evan? I'm curious. What do you call a fly with no wings? I don't know. A walk. That's pretty good. I like that. Yeah, I could use the jokes. Maybe one reason that video didn't do as well is Veritasium has a full-length video. Yeah, but that's never stopped me before, right? Other big creators have made videos on giant viruses. I think Kyrgyzstan actually had one about giant viruses. I still made mine and it still did well. And I thought with a bit of extra Phi, the neutrophil spice, I would have something different to offer than Veritasium, especially because he made that video a while ago. But maybe I didn't evoke, because what I'm finding is that I think my audience likes it when I evoke the glitchy nature of molecular biology, which I also love too. What's your favorite protein? Might be a bad time to ask this question. You've probably heard 10 times, but it's for charity. Yeah. Oh, I'll answer that. My favorite protein is major histocompatibility complex 1. The protein that does antigen presentation tells the T cell, hey bozo, There's a virus inside of me. I love this protein for a couple of different reasons. One, because of its normal immune function. It's a great protein. It tells the T cell the cell is infected, and the T cell comes and nukes that cell. Love that. It's awesome. It's also very interesting from a genetic perspective because no two people's MHCs are completely alike, because as a species, it is valuable for us to have different MHCs. Because different MHCs can display different antigens, meaning if we want to survive as a species, we can't all have the same immune system. And the genetics of MHC polymorphisms is also really, really interesting. So another interesting dimension there. Next reason why I love MHC. The immune evasion arms race. Story time, chat. Long ago, pathogens and host cells engaged in mortal combat. And so, host cells evolved a system. It's called acquired adaptive immunity. Okay, virus. Okay, bacteria. You outsmarted the macrophage. You outsmarted the neutrophil. But now you're playing with the big boys. T cells and B cells. Specificity. I'm going to evolve as the host a way to sniff you out. And then when I sniff you out, I'm going to delete you. Okay? That's pretty good. It's a pretty good strategy. Works very well. Some viruses evolve a neat trick. They pull MHC out of the membrane. Some of them literally drag that protein out of the membrane. Some viruses stop the production of that protein. All of a sudden, T cells are blind again. They're blind again. Oh, just a normal day in T cell land. No infected cells here. Clever viruses. What does the host do? Anybody know this story? What does the host do? Does the MHC gene use VDJ recombination? No, it's just a ton of alleles. It's an inheritance and mating patterns. So think about this, chat. A virus just evolved the ability to pull MHC out of the membrane to hide from T cells. Does anybody know what the host evolved to counter those viruses? Anybody know? This immune cell doesn't get talked a lot about quite as often. But it is really cool. Thank you so much for the donation, Cleo. Thank you for the donation. Thank you so much. Welcome, Cleo. Thank you. Yeah, so some of you are familiar with the story. Hairday says, kill anything without MHC. It's so funny. Hairday was here when I started this stream. Now you're here again when it is so dark outside. Kill anything without MHC. Yeah, the host evolved a way to do that. How did it do that? Spot says, doesn't the immune system also kill cells that don't present MHC? Yeah. Yeah. So the T cell needs MHC. But there's another cell type called the NK cell, natural killer cells. Natural, such a cool name, natural killer cells. Why natural? Because they don't need antigen presentation to kill. They don't have to cooperate in that space. So when the natural killer cell comes by a cell, sniffing along their surface, if the natural killer cell goes, oh, I don't think there's any MHC on you, that means you've been infected by a virus. And the NK cell will nuke that cell. The arms race. The arms race. Isn't that amazing? The back and forth of biology is incredible. But wait, chat. That's not where this story ends. T cells counter viruses. Viruses counter T cells. NK cells counter viruses. Okay, some viruses have a more amazing evolutionary arms. So certain viruses are pulling them out, but they get countered by NK cells. That sucks. Chat, I want you to think about for a hot second. What else could a virus possibly do to escape the immune system? It tried removing it. It tried stopping the MHC protein from being made. And what did it do? It just encouraged the evolution of the natural killer cell. So what is the virus's next move? What could you do? You already tried removing the protein. And yeah, we have some guesses in chat. Very clever, very clever. Make a decoy. Oh, a decoy, a decoy. How smart is that? So these viruses make trap MHCs, or antigens, that make it look like to the T cell, oh, everything's fine. You know, the T cell comes, they go, they sniff at the infected cell, they go, oh, carry on. You got your MHC, you are producing a peptide that looks like it belongs to the body, carry on. What about the NK cell? The NK cell comes over and it sniffs, it goes, oh, you've got MHC. Okay. You don't appear to be infected by a virus. Carry on. All of this evolutionary battle occurring around one protein, the MHC1 protein. And I love that. But I'm not done yet. Because I am a neurobiologist. The immune system is my hobby. So what does this protein do in the brain? Oof, it's a good question. Not only is this protein a crazy immune front for battle, right? This central piece. You know, I've mentioned antigen presentation in almost every one of my virus videos. If a virus is going to attack you, it has to evolve something to deal with your MHC. It is the gatekeeper for infection. But it's also doing something in the brain. And that's weird. And you know, if you know me, that I like weird. The immune system is not typically thought of to be active in the brain. I mentioned this in the rabies video. You don't want your immune system to be active in the brain because the immune system are full of bad guys. Really can put the hurt on. So what is an immune protein doing in the brain? The brain's not supposed to be infected. There's the blood-brain barrier to try to prevent that. It turns out that MHC in the brain is doing a lot. Memory, learning, synaptic transmission. Okay. Inflammation, that's not surprising, it's an immune protein. But a part of the tool box that your neurons have to adjust its own firing potential comes from an immune protein. And this is why labels don't mean anything in biology. I mean, they do. Labels are useful. But they're not hard categories. Because MHC, it's an immune protein for sure. Fights viruses, yeah. But it's also a neuronal protein, doing totally unrelated things in the brain, controlling the number of connections that your neurons make with one another, determining how loud your neurons talk to each other, determining the growth at certain stages. And that is the mystery that I wanted to solve as a graduate student. My favorite protein. Do you think that's worthy of being my favorite protein? I think so. Is MHC your favorite protein now? I hope it is. I love this protein. I'm going to make a video on it one day. I'm not ready yet. But yeah, that's crazy. Is it kind of like using an umbrella as a water bowl? We don't know. We don't really know. The outside of MHC, we know that's important, right? That's the part that talks to T cells. The inside part, the part that sticks into the cell, for many decades, thought to be useless, not thought to be important at all. Research has shown that that part of MHC that everybody thought was useless, that's the part that neurons care about. Yeah. Not so useless after all. Again, evolution, pretty good at trimming the fat. Not a lot of things are truly useless. Unknown function. Yeah. So thank you for allowing me to soapbox on my favorite protein. I love that protein, man. But that's maybe not like hard to understand. It is the protein I worked on as a graduate student. What's my second favorite protein? It's kind of an interesting question as I kill a bit of time before sad boy hours start. Maybe I should make sure that the permissions here are set up correctly. Sorry, chat. I'll let you stew on MHC for a bit as I work out. Because I know I had permissions issues with Discord before. Gotta try to fix that. How do I do that? Oh, I regret waiting this long to fix Discord. I'm so tired. What's your non-favorite non-protein coding DNA or RNA? RNA. I think anybody who picks DNA over RNA is crazy. RNA does everything. DNA is just a fancy hard drive. I mean, it can do neat things too, but I think you would be hard pressed to find a biologist that prefers thinking about RNA, prefers thinking about DNA over RNA. RNA is just incredible. Oh my god, how do I do this? Server settings, roles, collaborators, permissions, messages, connect to voice chat, speak, video. Okay, great. I think that should be good. Okay. Yeah, that looks good to me. Hopefully my friends can join me now. When they are able to. Thank you so much for stopping by, Iridium. Thank you so much. How long do you think we need until we need phages to fight superbugs? I think phages are a very interesting tool. I'm a little worried because, you know, when you fight fire with fire, things can get kind of suspect because phages are lifelike. And when you use something that is lifelike to fight something that has lifelike things can do all sorts of crazy things. And I don't think you can cram enough genetic information into a phage to code in a self-destruct button like you can do with bacteria. I think it's also time I head off. Thank you so much for the awesome stream. I learned a lot too. Thank you for spending the time with me. I do appreciate it. Yeah. Phages are great though. I'm becoming a little more afraid of them as we go on for potentially unintended effects on the microbiome. But maybe I've been fear-mongered. I can accept that maybe I am too afraid. But I'm not going to tell you to be afraid because I'm not an expert. My attitude is changing anyway. Couldn't you code the phage to need something? these genetic dependence modules, I don't know enough about phage to know how much you can cram into them because they're pretty small. You could maybe. I don't know. You might be right, Hair Day. That's the limit of my knowledge, though, which you found. So I applaud you for that. Oh, oh my god, I'm so scared. Hey, two octonologists woke up. What are we doing? I just finished talking about the connection between obesity and cancer, and potentially uh, some promising findings suggesting that an NSAID, something like ibuprofen, might be an effective part of a cancer therapy treatment for obese patients. And now, because I ended early, I'm just relaxing with chat. I am almost 18 hours into this stream, and we're just kind of vibing until 3 a.m. here, where I don't know if anybody will be awake or anybody is hanging around, but I will, and whoever wants to hop into VC, if you want to keep me company while I take a break from talking about science. And maybe it'll just be me and chat talking. That's totally fine too. That's totally fine. How do you typically make bacteria that self-destruct when you want? That's an excellent question. I will tell you what I remember. There's an interesting paper that I read a couple years ago, and this was trying to engineer a bacteria clean up mercury from the environment. Okay. The bacteria eat the mercury, remove it from the environment, then you remove the bacteria. Seems simple enough. But let's be responsible about this thing. We don't want this bacteria escaping. We don't know what it could do to the environment. But life is about managing risk. So if you want cheap and effective ways to remove a pretty terrible pollutant, instead of giving up on bacteria because they could be too dangerous, let's just try to make them as safe as possible. That's what this paper was trying to do. So how did they do it? They rigged this bacteria up with a clever little genetic circuit. You can think of this as programming. It's simplified, but it works for the analogy for educational purposes. And the circuit, again, simplified. It's more complicated than this, but I'm boiling it down to a central theme, which is if mercury, survive. If no mercury, die. It's a pretty simple circuit. It's a little more complicated to rig the circuit genetically. And, eventually we'll do some engineering streams where I'll tell you how they do it. But, yeah, I hope that makes sense. If mercury exists, happy, alive. If you lack mercury, self-destruct. And conceptually, it really is that simple. How optimistic do you feel about engineering bacteria to clean up pollutants like microplastics? Great question, Bradley. I'm biased. Why? Before Phi the Neutrophil, or currently with Phi the Neutrophil, I used to teach at a wonderful university before I got let go. Brilliant students. One of the things that we had them do in my class was to design a project that required biological engineering to solve. So what are these Gen Z kids really thinking about in terms of what they want to do? Every year I taught this class. Every semester I taught this class. At least 3 or 4 projects trying to engineer microbes to clean up microplastics. So am I an expert in this field? No. But some of those students I taught, brilliant. Much smarter than I am. I don't know how to answer your question, Bradley, because I don't think about this kind of science very often. It's kind of out of my field. But I do believe that some of those students are destined for greatness. I do believe that. I think they will go off to do amazing things. maybe one of them will do what you are saying. And for that, I had to say I'm probably optimistic. Oh my God, thank you so much. Huge donation incoming, $200 from, I always get your name wrong because this is a pretty complicated name, but Tass Reginord Hicks. Sorry. It's been a joy being able to catch the latter half of the stream in the time zone friendly hours. Thanks for pushing through the fatigue. Thank you so much for the $200 donation. We are so close. It's right there, chat. We are so close to reaching the $2,000 milestone goal, double what we raised last year. We can do it. $90 more. We still have so many hours left. We still have so many hours left. There's 6 more hours left, basically. But thank you so much. Oh, I remember you from the profile picture. I drew you as the meme, the bouncer meme. Great profile picture, by the way. It's great. That's awesome, dude. We're so close to that. We're in the home stretch, y'all. Yeah, bioremediation, exactly, too, Octonal. I remember some news about bacteria that could turn a type of plastic into vanillin, I think. I think maybe Nile Red did a video on that too, if I'm remembering correctly. But yeah, bioremediation. The thing about engineering microbes is that it's pretty cheap, which means it's accessible. When we think cheap, we often think, oh, corporate greed. And that's true. Right? The thought of making products cheaper can often come with downsides, but biological engineering to solve these problems, like, would you rather deploy a big net or complicated synthetic chemical barge whose job it is to clean this up, or a couple of yeasty boys, throw them into the environment, have them eat the mercury, pick them up later? I pick the yeasty boys. You can grow those suckers for, I don't know, $20. $20 solution versus, 500,000 from chemistry or civil engineering, or like $100 from biology. There's no competition. Another question: what is the difference between a coronavirus and an influenza virus? Oh, dude, I'm not ready to talk about coronaviruses. I think I'm sorry. Maybe I didn't get to your question before. I saw somebody else had asked, or maybe it was you. I saw somebody asked about COVID. Dude, I'm not doing a COVID video. I'm sorry, not yet. I get a lot of crazies in the comments when I talk about completely uncontroversial things. If I were to touch COVID, yeah, it's not happening. Have I read The Selfish Gene by Richard Dawkins? What do I think of it? I don't read books. I only read papers. I'm a freak. I'm a nasty little guy, and the only thing I do is read papers. I don't like pre-chewed content. So I have no opinion on The Selfish Gene. Yeah. Just outsource your work to bacteria. Classic. Yeah, that's the life of a biological engineer. I'm going to go ahead and move over to the sad boy hours, as it is officially time to start sad boy hours here. And I'm going to join the VC, and yeah. Maybe it'll just be us, chat, as we continue to wax poetic about the state of biology or your studies or anything else you want to talk about. But yeah, I'm just going to hang out here. And if my friends want to come and hang out in this dark hour, I'd be very welcoming of their presence. It would also be hilarious if nobody showed up, because it's it's a pretty big ask. It's pretty late at night. And even in Europe, it's pretty early too. I enjoyed the BRB animations for the stream so far. Yeah, it was a lot of fun to do. It was great. Neutrophil in space. It's sad boy hours. This is how I feel at 3 a.m. This is how I feel at 3 a.m. And I've talked about cancer for 18 hours now. I think how I know I've hit my limit is I, in this moment, I don't want to do this again. Not 24 consecutive hours, because let me tell you, my brain is so slow. My brain is so slow right now. My throat is dying. But I can't say that I'm not so happy that we've almost doubled our stretch goal from last year. Remember, last year the goal was 1,000. It wasn't. It was 500, and 1,000 was the stretch goal. This time, 2,000 is the goal, and we're so close to hitting it. And I think that's meaningful. I'm glad to have been a part of this. Yeah. I talked a little bit about this at the first half of the stream, so if you weren't there for my soapbox then, it's not about. I don't think about charitable contributions as being what can my dollar do? Here's the secret: research is tremendously expensive. But it is about belonging. And I'm proud of what, even if we don't hit that goal, I'm really proud of us. We, even if nobody else cares, you can go to bed and know that you took part in one of the greatest stories that will be told. One day, this beast will be slain. Yep, all the directions go through Tiltify, but they all end up at VTheorem. So if you follow the links, that's how you can donate to them directly. I'm starting my 4th uni semester tomorrow. Suggestions on how to figure out what to do afterwards. Oh, that's great. My so 4th uni semester, that's about sophomore year. So sophomore year, I was still pre-med. I wanted to be a doctor. I wanted to treat patients, use my biology for good. And that's a good time to be in university because you're about halfway, you've gotten your feet wet. You kind of have an idea of what you would like to do, but you haven't committed yet, which is a great time. I think what you should do is talk to people who are working in their fields, because you'll never know what you want to do unless you get some real eyes in front of you. So go and talk to your career advisor. If you're a part of your university, you've probably got one. Because my career advisor saved my life, man, multiple times. My pre-health advisor talked to me when I was a junior, 3rd year of college, he said. And I've told this story a couple of times. So I apologize if you've heard this story before, but new audience here. I don't think I've seen your name around. The pre-health advisor said, okay, Phi, so you are here because you are on the track to go to medical school. Why do you want to be a doctor? I said, fine. I knew this question was coming. I think my answer is good. I love biology and I want to help people with my love of biology. What better way to do that than be a doctor? And he said, why don't you get your PhD instead? You can still do a lot of good with a PhD in biology. And I would be lying if I said that was an obvious answer and I predicted that, but I would be lying. Because it wasn't obvious to me. I was so committed to the idea of doing medicine instead of biology that I never seriously considered going into research instead. And it made me doubt. Because up until my 3rd year of college, I was more concerned if I was smart enough to be a doctor. That was my main concern. I didn't care about anything else. Grades over everything. And I never stopped to consider, because I just assumed if I were smart enough to be a doctor, I should be a doctor. I don't know if I'm smart enough to be a doctor. But when he asked me that question, I had to seriously consider that maybe that life isn't for me. Because the idea of studying biology forever, ooh, that sounds good. That sounds pretty good to me. And it meant something coming from the pre-health advisor. His job is to make the university look good. More pre-meds that get to med school, the better, right? And here he was with a conflict of interest, telling me straight to my face, maybe you shouldn't be a doctor. If it had come from anybody else, I would have brushed them off. But from him, yeah. And I think he was right. Not that I should be a researcher. I tried to get my PhD, didn't work out. I half blame COVID for that. But he is right in saying that I am much happier being who I am now, getting to talk to all of you wonderful people about the science that I love so purely, that the idea of spending my life in the clinic and knowing what I have now, this audience, this stream, the privilege to get to say I was a part of a movement that raised almost $2,000 for breast cancer. It's meaningful. Couldn't give that up. Yeah. Karis, you can just
Hello.
jump. Oh, kind of jump scare. My buddy.
Hello.
Karis, you're a bit loud. Let me correct that. Okay, I think you
Okay,
should be good
okay.
now. Oh my guy,
Fantastic.
first time on the
Is
channel.
it time to be sad?
It's time to be sad. It's time to be sad.
I love that.
It's
I'm
time
really
to be
good at being sad.
time to be contemplative. Do you want to introduce yourself? Because this is the first time
Yes.
that you've been on my channel, I believe.
Right, right. Hello. Hello, everyone. Hello, everyone who watches VTheorem. I am Karasir Fanor, the first and last philosopher VTuber, the last librarian of Alexandria. And I'm sorry I don't have enough gusto today and right now because it's sad boy hours for you, right? It's like for me it's really early in the morning. And I kind of just woke up. Which is in its own way like a little like I resonate with the sad boy like fatigue a little bit. But no, I'm here and I have collaborated with you, Fi, once, but it was over at my channel. I asked you a bunch of questions about stuff that, you know,
They were
about certain
fun.
things and about
Yeah.
and it was really fun and it was also me poking at you and going like, okay, Fi, I know you're a biologist, but what about this other thing that you don't really think about sometimes? We should think about it. And
Yeah.
I'm really good at being super annoying, I guess, is what I'm trying to say.
As is your prerogative as a philosopher, right? So let me do my introduction of you. Okay.
Okay. Yeah.
This is my guy, okay? I love talking to this man. He's very interesting. And I hope you will receive him warmly as well. Because I think science and philosophy have a kind of tension. Put it that way. But I've never felt the tension between us in a negative kind of way, right? Just it's interesting picking apart how a philosopher thinks versus how a scientist thinks, because I don't think you would take offense to say that your thinking is in the realm of the theoretical and my thinking is more real in a experimental sense. But I'm not saying that one is better than the other, but it is concrete versus abstract. And yeah, I've talked a lot with this guy, Karis, behind the scenes about philosophy of content making, which is actually something that I want to talk to you here. I was like, is Karis going to show up? I want to talk to him about content creation so bad.
Yeah, yeah.
But I do want to finish addressing something before the conversation I wanted to finish. And maybe you can have some input too, because it's about deciding what you want to do with your life. But thank you so much for the donation, who redeemed the incentive. What's your favorite cell type, non-white blood cell. I answered this before, so you can have another question if you want, but it's actually never been the white blood cell, despite being a neutrophil. Like, it would be so full of myself as a neutrophil if I said the neutrophil is my favorite cell. You're a human, Garrus. Is your favorite your favorite animal a human?
I know that my favorite human is probably myself.
Okay, that's fair. No, I see that.
My favorite animal?
What's your
I do
favorite
really
animal?
like humans. I think humans are so, like, you know what I think? I think saying humans are your favorite animals is like saying, like, is like picking Mario and Mario Kart.
Yeah.
It's
I
a
mean,
bit
I
like
guess you
that to
could
me.
do
I
it.
don't know how to explain it, but
That's funny. That's funny. I would say it's probably an uncommon pick. You're probably, I mean, maybe you're picking Mario because you're a tryhard for some reason, but it's not a common pick. But my actual favorite cell type, it was never anything in the immune system. It's the neuron. I love the neuron. Big brains. So yeah, you can have an extra one. Favorite ice cream? I love me an alcohol flavored ice cream. Boy, howdy, rum raisin. A good one. Not the fake stuff that tastes like chemicals, but a good rum raisin. And oh my God, Hair Day again. Hair Day contributed $500 earlier. And again, at 3 a.m., Hair Day pushes us over the milestone. Thank you so much. Hair Day says, too bad you didn't get to say that you raised almost 2K anymore. Unrelenting glory. Let's go. Quiet claps because Toxo is sleeping. Thank you so much, Hair Day. Huge. Oh my God, I feel like a weight has been lifted off my shoulder. We did it. That's awesome. 2K.
Gosh, what do we do at 2K? Is there anything you do at 2K?
2K, I will make a original biology song. I will, and this is part of why I like having you on, because you were recently talking about your adventures in music too. And I want to talk about that.
Yeah.
I wonder if Pemba will join us at some point. Yeah. Thank you so much, Hair Day.
Consult if he does, yeah.
Absolute monster right here. Loud claps. Oh, Toxo, are you awake? Am I keeping you up, Toxo? I'm sorry. Am I being too loud? I can be more quiet. I'm sorry. I thought it was being
I can
quiet.
celebrate I was gonna say I could celebrate pop off for you, but it is also quite early for me as well. So I will clap. I don't know if my mic is picking this up.
It sounds like farts.
Okay, well.
Toxo, I'm sorry. If you really need me to be quiet, I can turn down the speech. Unless you're tuning into stream and you're just hanging out with us, because you're in the other room. I can't tell if you're actually sleeping or not, or if I'm being too loud. Yeah,
Let's go.
If you want to sleep, please do so. You don't have to stay up and keep me company while I talk to Kerosene Fedora. Yeah, to address a comment from the viewers, any thoughts about the negative repercussions that come from AI protein folding? Could we be entering a new age? I think AI protein folding is great. Negative repercussions like water usage? Probably. That's probably bad. But in terms of AI and biology, good friends. Awesome stuff. AI and art, garbage. Trash. But I'm generally optimistic about the effect of AI on biology. Now, let me make an exception. Policy given to us by the government directing us to use AI when it is not appropriate. That's different. I woke up naturally for sad boy hours. But then Karis came, so I was not needed. Man, you can stay.
You're still needed.
Want.
I think the more
valid. the
not merrier,
the less
maybe,
sad.
the more the I think collective sadness is great. It's a great vibe to have.
What about parties with bad intentions with this new level of information understanding? Look, there's always going to be bioterrorists. They could do terrible things even before AI, right? AI doesn't make them more dangerous, in my opinion. And, we have government and law enforcement and anti-terrorists departments, right? I would say I'm not too pressed because you for a health researcher, AI is an incredible tool. For someone with nefarious intent, I can't imagine how you could cause harm that you can't do with other methods. Oh, Toxo's here. Toxo brought a chair over. You should probably go to bed. You want an earbud?
Hello, I'm here for sad boy hours.
Toxo's here for sad boy hours.
Hello.
This is
Hello,
Karis
Tox.
for Nor.
Hi, Karis.
Hello, hello.
Yeah,
Let's be sad together.
we're being sad together, but I wanna I wanna talk because Toxo put the earbud back in my ear.
This is
Was it the wrong ear? Sorry, I gave you the wrong earbud. I am so tired. We've been talking a lot about content creation, and you recently had a New Year's resolution stream, and I
I did.
want you to tell me and my chat what your freaking deal is, man. Like, what is your thing, dude? What
What's
is
my
a philosophy
thing?
VTuber? What is your thing? Please pitch yourself, because I feel like I don't even know, but that's kind of why I love it so much, because it is... Okay, hold on. Let me give you an important piece of context as to who this man who is in this void space is. In order to join his Discord, you have to do logic puzzles. Okay, that's the kind of guy this guy is. You have to do a couple of logic puzzles before you step foot into his Discord, which is crazy. Gatekeeping, right? That's the kind of
Well,
character.
it's all free though.
That's the kind of character this Karis Fanor is. I googled all the answers, by the way. I did not do them legitimately.
cheater.
I cheated.
How did you did you Google like the form? Because I've rewritten the answers from like existing puzzles. So I imagine if you looked up the existing puzzles, you could draw like an inference and say, okay, the logic works this way, so I do this. Like, how did you Google it? Because I rewrote them.
I mean, the direct text matching doesn't have to necessarily be a thing when the form of the puzzle exists elsewhere. And also, I did it a while ago, so I don't really remember. But the thing is that what a character, what a lad that makes you do 5 increasingly difficult logic puzzles to even step foot into his Discord. Messed up, honestly. I would curse, but I can't on my stream. But I want to say curse words because that's terrible.
It's messed up. You can say it's
hot.
effed up.
It's hot.
That's maybe the extent we can go for it.
So with that context
But
in mind,
I think
like, what are you about, man? What's your freaking deal, dude?
I am a philosopher VTuber. I started streaming it like a year and a half ago, and I think what I quickly came to and found very difficult to express, yet felt all the same, was that I did not want to make video essays. I did not want to affirm or comfort as the primary objective of my philosophy, because I thought that there's a sort of insidious thing that happens in educational content on YouTube, not by intention of the people who make the education, because those are not the people I have a problem with at all. Really, the deeper issues lie within the structure of the platform, the maximizing of retention, of being able to be presented information that is easy, exactly, and digestible, and just appealing overall. And that, I think, works for a lot of subjects, for a lot of things. Like I learned a lot of history from like epic rap battles of history. There's no like it's not like it's bad because it's educational and it's on YouTube and YouTube is bad because it makes money. That's not really what I'm saying. But for something like philosophy in particular, and there's this guy, he used to do stuff for, what's his name? Michael Burns, I think. He used to do videos for this channel called Wisecrack, which is like a philosophy channel on YouTube. And yeah. I think they shut down recently because like as like a business, like
Can I a little bit of lore?
Yeah.
Should I don't know. I don't think it's traceable to me. I'm going to be very broad with this. I took a gig writing for Wisecrack, actually.
What?
Yeah.
Once upon a time.
Once upon a time, I took a gig writing for Wisecrack. I'm not going to tell you which video.
Right.
But yeah. So I am familiar with the Wisecrack channel.
That's
Yeah.
so wait, that's so cool. Did you write like did you write like a section of it, or did you write like the whole? How did it like work? If you don't mind, like getting
That's a little too
not gonna
much. That might give me away.
Okay,
But
okay. That
I
would give
was
it away.
commissioned. They paid me
That's
money to do a bit of writing for them
Ooh.
before Find the Neutrophil.
Ooh. That is so cool. Anyways, yeah, so this guy, he used to do like Wisecrack. I don't think he didn't own Wisecrack, but he was like a host for a while. His name was like Michael Burns, and he made a video recently that kind of like really just captures everything I feel about philosophy YouTube. Video titles just philosophy YouTube is broken or whatever. And I think like it really summarily captures everything I feel about the matter, and that's so much of, if not all of philosophy that it's done on YouTube, because it is limited by the form in which it appears in.
Dude, you talk like a
The
philosopher.
fact that it's a
man.
video.
You talk like a freaking philosopher. I can.
Thank you.
Let me try. That's not a compliment.
Even better, thank you.
Because I hear what you're saying because I relate. I think a lot about
Yeah.
this in my own content, but can I rephrase it in my own terms, see if you can agree? Because
Yes,
I want to
absolutely.
put your
Yes.
words into words that I find more palatable. And
Yeah,
also, I love the fact that
go.
you can tell me if I'm too much for you, but I feel like we have a kind of rapport where I'm kind of comfortable being a little mean to you.
I love that.
You want philosophy to be practiced, not for philosophy to be taught.
Yes.
How's that? When you make
Yes.
a video essay, the viewer is in a passive relationship with the video. You put it on second monitor, you play League of Legends or whatever. It's a very passive experience. But practicing philosophy is a lot harder. And I relate to that because what are we doing on this stream? It's 24 hours of me talking about breast cancer research, not the headline, not the press release that the university published. I am showing you the data. And I'm not giving you the answer. You are using your brain, you are looking at the fluorescence, you are looking at these numbers, and you are telling me what that data means. Practicing biology, similarly to you, you want people to practice philosophy. So how do you get people to do that?
That's a great way to summarize it. And you know what? That's why I tuned in for bits of the AM session, and I saw the data, and I was there for like one of perhaps several of your be the scientist moments, which I really enjoy. Like I have to say
Thank
that this is why I approached you in the first place, really, is that there's the fact that you do this, because I think it's of course so important to practice the thing, to be involved in the thing. You hit the nail on the head. It is very like philosophy YouTube is very passive, at least the way, because it's YouTube. The way you consume it has to be passive. There's a gap in time and space, and to some extent that is fine for things such as, you know, retention, or not retention, but like for recalling stuff, for memorizing facts. That can
And
be helpful. Like
if
you
I
can
could
learn
add
from a
on to that
textbook.
idea, there's no
Yeah.
two-way interaction, right? I just went on a huge rant 2 hours ago. Feels like graduate school is useful because your professors will dunk on you, and that's an important part of your education. If you watch a video essay, the video essayist can't dunk on you. They can't tell you you're wrong. Getting told you're wrong is so important to learning, and if you aren't getting grilled, right? How much grilling did you get in your philosophy education, by the way?
Oh, let me think.
How many
much
arguments did you
grilling
get into?
I get? So I think in my undergraduate schooling in philosophy, it was less me dunking or being dunked, and more me almost watching warfare in real time between my professor, who was my supervisor at the time, and like everyone else in the department.
That's sick. You were
The professor I had,
outcast.
he was the only continental philosopher, which is to say that he's the only guy who wants to teach like Plato, without kind of reducing it to just, okay, here are the arguments. Logic A, logic B leads to conclusion C, so on and so forth. The premise A and B, and then with a certain bit of logic leads to conclusion C. And he was very against that. He was like, no, I think people should like be suspended in that unknown, and rather than kind of just logically master puzzles, like rather than solve everything like a logic puzzle, he thought that there should be a different approach to philosophy, and that's me grossly summarizing the divide between continental and analytic philosophy. But anyways, basically he disagreed with the whole department on the matter, and what made matters worse was that he was not very willing to um put his head down per se and like pump out the research the same way the other professors were. Now, and without giving away too much, obviously, um, the the school I went to was a very like relatively really high ranked school and that cared a lot about its ranking.
Smarty
And
pants over
so
here.
this refusal to not paper pump meant that he was essentially, dead weight, I guess, in the eyes of the department, and they really wanted to get rid of him. And well, they did. He was my supervisor for my like final undergrad dis, and then the moment I was done with my thing, and I don't think he had another student lined up after that, they fired him. It was interesting because he taught me Socrates, and he taught the story of the hemlock and the being voted out by the people and everything, and then to just
Full of Socrates.
to receive it, yeah, on one hand to receive it verbally through lecture, and then to watch it in real time has left this like lasting impression on me on like how I guess maybe even dramatic philosophy could be.
Yeah, because you make people
But,
do a
yeah.
Discord freaking logic puzzle to you and talk to you. Like you do have a flair for the dramatic. It's crazy.
It is entirely free though. That has to be the emphasis. Is like I don't gatekeep anything monetarily. I gatekeep
Yeah.
it by effort, I think.
Yeah, you make people work for it.
But I think it's good to like kind of submerge yourself in that like that suspension, that like, oh, I don't know, oh, the I'm not sure, and and resisting the desire to immediately have any sort of anxiety in yourself quelled. And I don't mean anxiety in like the medical sense, or like the
Yeah,
yeah
that's why like
neurological sense. Yeah.
like genuinely you are one of my most favorite creators to talk to, because I think you are an actual artist. I didn't say a good artist. Okay, don't get too ahead of yourself.
Not
okay?
yet.
I'm not calling your content good, but
Yeah.
I am saying that the way that you think about content is artful. I can tell it comes directly from your soul, right? It is not cynical. You have a particular experience you want your audience to have, and no one can tell you that you're right or wrong because it comes directly from you, right? It is not market approved or tested, or because I could tell you, like, if I were running your channel, I would do this or that, but I'm not you, and the art that I would make is different than the art that you would make. So I actually really respect that you do these wild and wacky experimental things from my perspective. So it is so interesting to be able to talk to you on certain things like that. Yeah, the Discord thing is crazy. And also like science and philosophy, I mean, there is a philosophy of science, but there's overlap between our fields too. And I think we're only this antagonistic, or I'm mostly I think you've been very nice to me and I've been very mean to you. Behind the scenes we're very nice to each other. But I just find it funny that you exist in a very kind way. Like I think it's very cool, and I appreciate that you are here sharing the VTuber space. Because I'm also in like I've always had a flair for like the weird, right? I've talked about this on stream. Like I picked my PhD on weird, I picked my studies on weird. I don't have conventional taste, and I like to push boundaries. And one of the boundaries that can be pushed are VTubing, right? How many chatting? How many chatting do we need? How many video game do we need? Like those are good. It's not bad if you enjoy them, but for me, there's this fatigue. I need the novelty, and I appreciate that you're doing something kind of weird. And that's pretty sick.
I think, as you say, there's enough video game. If you take a look at my channel right now, you will see that it's a lot of video game. It's a lot of like me appealing to the sort of like, not the standard VTuber affairs, but like the something that's like easy and
Familiar.
comfortable, and yeah, familiar. Because part of what I try to do is I try to like almost disguise myself in a way, like almost nefariously. I'm disguising myself as like I'm just like a normal VTuber. I'm like a VTuber, and then in my channel description is like I'm the I'm a philosopher VTuber. What does that mean? Oh, it just means I talk about philosophy. And you come in with the expectation maybe of like, oh, this guy just has maybe some fun facts in his head, and maybe he just likes playing video games a lot. You click on my stream, and then I will start berating people in chat
I
for
love it.
having
It's so fun. It's so fun. Please subscribe to him and join me in like I think he's a fantastic streamer. It's really funny to be in your chat sometimes. Like really great conversation.
I think the back and forth is what matters. Yeah, it's
Yeah.
more than anything.
Well, because
like,
you
I could give you facts, but,
Socrates guy, right? Like that's what I love about talking to you is that like I'm a trained communicator, but that communication is mostly one way, right? The annoying philosopher debate bros, you have that kind of blood in you. I'm not calling you a debate bro, but you have the like the bloodline.
I was forged in the debate bro lands.
Yeah,
it's
you
true.
were exactly like your flesh is the same flesh that gave rise to debate bros, so you have a flair for that kind of interaction, whereas I'm pretty milquetoast. I think my streams are good, but for a different reason. I have a very like buttoned up professional because that's just like who I am, but I love that on your stream you just fucking get into it, and I love that. So yeah, and yeah, again, as you just said, like you had been trained by someone who trained you in Socrates, and Socrates was the ultimate debate bro, right?
Oh yeah, he's so annoying, wasn't he?
He's so annoying, dude. So annoying. He like died for being annoying, right? The dude's like
yeah.
so annoying he died.
We're like we've had enough of this guy. Let's get him out of here.
Yeah.
Let's make him drink hemlock and everything.
I showed a picture of Nietzsche earlier on stream, and I actually had to pass that to three carrots who were like, am I doing philosophy right? Am I gonna get called the fucking the 3 a.m. curse, my bad.
No.
I made it.
The monetization.
18 and a half hours without cursing. And of course it's during
I'm glad that
hours.
I was there to witness it. I curse a lot on my channel.
Yeah.
Maybe maybe it was my fault, because you're used to, I think, listening to my voice and thinking I should like curse.
But I know it's just so funny because I think I'm a lot looser because it's so just like purely funny to be talking to you. Yeah, and I had to be like on my knees and be like, Mr. Philosopher, is Nietzsche? Can I say this about Nietzsche? And you're very kind. Again, we're very nice to each other behind the scenes.
That wasn't how I read it. That wasn't how I read the message. That was the voice
I want to
in my
be
head.
cool and have a philosopher on the screen because I want to seem smart, and I had to make sure to get that approved by you.
It worked, I think. Like what you sent me, at least. I haven't gone back into the session to check what you've done in actuality, but I think like
there's
what
a
you
sense
sent
of
to me
like
made a lot of sense.
self gatekeeping that I do, where I feel like I haven't earned the right to have a favorite philosopher because I don't really. I've read a little bit of Nietzsche when I had a philosophy phase, enough to be like, I like this guy. But I feel like such a faker when I talk about philosophy because I feel like it's so gatekept, I don't feel right saying that because when you call yourself a philosopher, it's like obnoxious, right? So I just try to stay away from that kind of thing. But I do like Nietzsche a lot. I think he was had interesting ideas, influential ideas for my personal development.
I think he was my favorite for a while, and I had to like jump ship very quickly because I realized early on that like not early on, but I realized in my like post-grad study that like a lot of people knew more Nietzsche than I did. And
then you got
because of the way I was trained, and I was like, ooh, I don't know if this is my guy, because they know him a lot more, or at least the people I sparred with. Really, I would compare it. I would say, okay, are you familiar with Dragon Ball Z?
Of course I am.
I am. I was in my postgrad, and I'll be forever grateful for this. But I was the Krillin of the group, and I had to learn on my feet. Like I didn't realize the pressure that could be kept up by like several other philosophers, not only who knew what they were talking about, but came from completely different backgrounds to mine, that did not have the same teachers, they did not have the same methodologies, that had their own like struggles and their own things, and they were so powerful. It was like I was really like, I was Krillin in the Cell games, like standing there watching them flash by and going like, oh my god, this is philosophy. This is like the duel of the century, you know. This is everything. This is everything I thought I wanted to be. But then I took a closer look at them and also myself, and I realized I don't know if I want to be Goku. I don't know if that's my thing necessarily.
That's funny.
Or really, a lot of them were not. Most of them were not Goku. Most of them were more Vegetas than they were Gokus.
In what, demeanor?
They're very, very powerful, mind you. Yeah, in demeanor and in power level. Like I was Krillin, and a lot of them were like Vegeta.
That's so funny. That's so funny. Hey, you know what Krillin does have that those other Dragon Ball Z characters don't?
What does he have?
Destructo Disc.
That's true.
You know, if you know anything about DBZ, even Frieza, even Cell, they fear the Krillin special. So don't count yourself out too much. Krillin's pretty good.
The Destructo Disc, my one technique. It's never landed yet, but people gotta watch out.
They have to respect the
when it shows up.
Disc. Exactly.
Yep.
I want to invite another collaborator on because I think this conversation is going to be interesting. Penbu, you can come in and chat now if you want.
Hello, Penbu is here yet. Sorry.
Oh, here we go.
Hello.
Hey, Penbu. You want to introduce yourself to my audience? This is also your first time on the channel. I was on your channel earlier last year, but first time.
That's right.
Please introduce yourself.
Good morning. My name is Fenbu, in the programming, puzzle gaming, music making, VTuber analytics. I should also not be awake at this hour.
Oh, very good. But thank you for joining me. I enjoy the company, and I think so context. I collaborated with Penbu. You might not know this, but you know that Blender song that I made for my Plague video. I did that with the help of some friends, not Penbu, but some friends. And I just think it would be so funny to have songs like original songs in my Find the Neutral videos. And so the Blender song was the melody that I made in my head, but I was carried all the way to the end point by my friends. And I felt so bad because like they were doing the work for free. I didn't pay them. They didn't take payment anyway. So I approached Penbu and I said, Penbu, I know you know music, and I would love to get my feet wet when it comes to making music myself. Can you teach me the very basics? And Penbu, you know, incredibly grateful for this. You made like a whole workbook for me, like an interactive workbook where I could play with music in a way that I hadn't been able to do before. So a collaboration I'm very grateful for. And because we hit the $2,000 raised milestone, I will be putting those skills to use at some point this year, trying to actually make, mostly, I'm still going to have help, but to make the step in making my own music. And I wanted to kind of open this sad boy talk talking about like just how we feel about music, because something that Karis that you said in your New Year's resolution stream, which I immediately logged on into, is your attitude, your New Year's resolution with regards to music. Could you talk a little bit more about that? And then we'll hear from Penbu, who has way more experience.
Yep.
The
actual opinion. I think like I wanna do music so badly. I want it so bad. And the worst part is I was not trained for it, and I'm at a stage in my life where I could learn music, but it would like there's a real like opportunity cost to pursuing any sort of new skill. But I don't think it's something that's totally foreign that I've never done, because I listen to music, I enjoy music. I like to think that I can appreciate the composition of like pieces. I like to think that I can appreciate like album structure. I'm not a heretic. I do not go into Spotify and click on like shuffle on like a new artist's release. I will listen to tracks in track order. I will appreciate the way it's been put out. I'll listen to the lyrics. I'm a big fan of hip hop in that regard. I will like, I try to appreciate everything sonically that's going on. I try to like really dig into like alternative or different like beats. I think not beats, but what's the word? Percussions, like I'm not, as you could tell, I do not have the musical vocabulary for this. And like my resolution was like I need to stop like kind of standing outside of it and gazing into it, and I should try to get into it. I should try to walk into music. I am not going to, reinvent the wheel or do anything crazy in it, because that's well beyond my capacities, but I want to at least participate in musicality in a way that's like truly like, I guess, like skillful. And I think Penbu, you would definitely have a lot to say about this as probably the expert here between the three of us. But like I find it so part of it is so daunting, but part of it is so like, at least in my like New Year's resolutions, I think I have to do this now, because if I don't throw myself into the unknown vortex of embarrassment, then I'm never gonna be able to participate in music the way that I like want to. That, and it'll like limit the way I can interact with the world and my vision of stuff and everything. I don't know. What do you think about that?
Yeah, I mean, I think people do see music, or at least making music as something that's pretty intimidating. I know why you did at some point too, um, but it is not that difficult these days to get into, especially with the advent of technology. Um, I'd say there's two routes that you can go about it. One, you can try finding instruments and approach it that way, or you can mess around with a score writer or a DAW, and then try it from that angle. I would say if you were to try, well, as a blanket recommendation, I guess, if you were to try one of those two, I would consider picking the instruments, because then you can actually, physically hear the sounds that you're making and stuff. But the melodica is a good instrument to start off with. It's like a keyboard instrument they blow into. If you've seen the anime Your Lie in April, the main protagonist plays one. Sorry, the female protagonist plays one during one of the early episodes. But yeah, that one's a good one because you get it's very easy to make a sound, and it sounds nice, and it's also very cheap.
So I have a question, and I'm interested to see what your reactions to the idea of music comes from. Is like it's interesting because we do have all sorts of different levels of musicality here, but like Karis, this urge from you to make music came on so strong, and I want to know what it is that you are trying to prove is not the word that I want to use, because that sounds maybe too antagonistic, and I'm not trying to be mean. But like like where does the desire from, come from out of nowhere? Right? I know you appreciate music, but what exactly happens in your brain that makes you want to actually make music? And I'm sure Pembu will have a different answer.
Um, I want music to me, or at least whatever musical pursuit I'm planning in the short term, is solely for content creation. It is solely for my own work as Karasufanor, and it's because I think only being only by doing this personally, or at least having it like I okay, only doing it personally am I really going to be able to fully capture the moment and perhaps the emotion that I feel when I want to do a particular thing. I'm trying really hard not to like explain it, because then it would spoil it a little bit. But I basically think that the musical element of this form of like video, of stream, or whatever, is the last thing I need to fully capture the heartstring. Like it's I need to I have an obsession, and I've only grown more obsessed with it recently, with playing and tugging at the heartstring. And I think music is such a powerful tool for that I'd be remiss to not even try to navigate it with my own grubby fingers. Like I've been using and borrowing music for a lot of the stuff I do, and it's never arbitrary. It's never like, oh, I'm just gonna put music here because it's like um it's some dead air and I just need to keep the audience hooked or whatever. No, like all the time whenever I use music for anything that I do, it's super intentional, down to like the track name, down to the specific like recording of it, and when that particular recording takes place, and uh like what context that track exists in, whether it be an album or even just like a video game soundtrack. And I've always thought that like that's me just borrowing because I don't have the means to capture that by myself. But like the moment I can do that, I feel like I would be so I don't know, really how else to describe it, but I'd be so powerful.
I have my own answer to this, which is different in crucial ways, but also very similar. But I want to hear from Penbu, as someone who has been making music for a while. Like, where does that desire from you come from, as someone who has been making music and playing music?
Yeah, I think for me, I've been musically inclined for a good portion of my life. If I really think about it, it was probably as a kid. But the reason why I still do music now is because it's a way to express myself. Um, it's because so there are a couple of compositions that are on my channel that were direct results of a certain situations and emotions in my life. For example, which one was it? I'm forgetting now. Okay, one example here is looking back, one of my compositions. I don't know if that was necessarily intentional for this, but I turned it into a uh, first anniversary video with a collage or montage of a lot of scenes of my first year as a VTuber, and I think the sentiment that it sounds like certainly carries that. Oh, the reason why I haven't is because I don't. Yeah, right, because I haven't published it yet. There was another piece I was working on, um, has yet to be published, waiting on art, uh, that I wasn't going to do anything with, but then I associated it with my decision to leave my job, and so that carried um, that situation. I think carried the work to completion because I had such strong association to that heavy decision, and so I had to do something with it.
See, that's interesting to me because, and I'm going to try to interpret what you two have said, and it is as I predicted. There's a difference that comes from someone who's very seasoned and like noobs like us. Like, Kaz, would you say you were making music for other people or for yourself?
Oh, I don't know.
Would you be
truthfully, I think
making music if there was nobody listening?
I don't know if I'd be talking if nobody was listening.
Fair enough.
I think like the music, I think to me is like an extension of my like, I guess, to be as pretentious as possible, my art.
My
And you wouldn't understand my
My
art.
art.
But it's yeah, it's an extension of that like self-expression as you say, Penbu, and I think like it to me it's the dimension that like I'm somewhat familiar with, but have never been able to practice like um myself personally. I would say on a level that I'd be happy with, and the desire to like practice, the desire to get into the desire to play the music is the same, I guess, desire that fuels my desire to speak, really, to exist to a very large extent, to do wacky and to pursue the unconventional. That's all like the self-expression bit, and I think the music is it plays into that self-expression, and I think like it probably not to the extent that you command music because you're I would again like probably the seasoned one of the three of us here. The way the noob thought in me is like all it needs to do, the music for me, is it just needs to echo the art that I'm already doing and just add like another layer in it to create that harmony. And it for now it doesn't have to do anything beyond that.
But so
Yeah,
what I sorry, so specifically the question that I posed is, you are creating this feeling that comes from within you. Is it so that you can have a musical representation of those feelings to exist? Or is it for that someone to listen and have the same feelings that you did as you were composing that piece? Is it for you or is it for somebody else?
I think for now it's the latter, but it the music is for the other person, but the art is for me.
Art is for
I
me.
guess.
That's fair. What about Pembu? Because there was some, I don't know if that's how you feel. I have a hypothesis, but like in the same lens, like when you make a piece of art, yes, music, all art is self-expression to a certain degree, but is it more important for you to, oh, I have this feeling, I gotta get it down on paper, I gotta transfer this feeling into a sonic sound, and as soon as I make it, doesn't matter if anybody else listens, I'm happy that I've gotten it out, that I figured out my feelings and I've put it onto paper? Or are you making it for someone else to listen to?
Yeah, I think it obviously depends on the piece. Like, for example, I recently created a BGM track for somebody for commission. That obviously has to be for someone else.
Yeah.
But for my own stuff, primarily it is first and foremost for me, and I have to be satisfied with what I put out. And if people don't like it, that's up to them. If people do like it, then great.
Yeah, that's kind of like I had this. I think that my hypothesis was right, because I think when you have musicality about you, it becomes like a part of your DNA. Like you like live and breathe, and it's natural. Whereas noobs like me and Karis, like this it's new and exciting. And I relate more to Karis in the situation where, like, why did I make the Blender song? And it's because I had a funny joke in my head and a kind of vision I thought would be funny. But I didn't make it to laugh at myself. I made it to share with people, right? I think Toxo's going to bed. Good night, Toxo. Thanks for staying up.
Night Toxo.
I made it so that people would laugh. I had a joke in my head, and I wanted that joke to happen in other people's brains. And similarly for this song, I'll tease the topic. I'm writing a song about cell death. Oh, Toxo's back.
Wrong.
Oh, Toxo's back.
Oh, hello.
Toxo went to get her iPad. Yeah. So like, much like Karis, like this as a noob, the idea of making music is appealing because there are certain sentiments that I cannot evoke in somebody just with my words alone. That there's a limit to what script writing and a video can do. And there's a specific feeling that I want to evoke in an audience member, because I think that feels good, when the feeling that you have, that you want to express, is received in the way that you intend it to be received. There's a kind of connection there. And I think that's what makes art worth doing, right? That's my whole AI art rant, When you make AI art, quote unquote, you're not an artist, you're a producer. You tell the AI, do this. The AI says like this, and you say, no, too many fingers. And you shuffle back and forth, but it's not your art. You're a producer. You're saying yes and you're saying no. And there's no connection there. Whatever the AI made is not your blood, sweat, and tears. It's not the exact emotion you felt. It is not a representation of all the little experiences and motor quirks you might have, or neuron connections that you might exhibit. Your art is your fingerprint. You might not like your art, but it is your fingerprint. And I think, like,
Do you guys? Sorry.
yeah,
continue.
no, go ahead. That's about where that thought was going to end anyway.
Oh, okay. Do you guys? Sorry to barge in. Do you guys want your art to be liked? Because I really desperately want my art to be liked, and I don't think I want everyone to like it. Because I think if everyone likes it, then it's bad. But I want a lot of people to like it, and more importantly, I want people who I like the art of to like my art in some way, even if they don't agree with it. To at least go like, oh, I see what you were trying to do, and I think that was an interesting pursuit. Do you guys have that, or is it just me?
Yeah. I actually talked about this when I got monetized. I was like, I would love to do YouTube full time. I would love to make this my job. You don't know how bad I want this. Enough to stay up 24 hours to push myself to be the content creator that I want to be. I love this more than anything. Do I care if people like my art? I do. But I don't know how many people will like the art. And I think I'm okay if not everybody likes it, because, yeah, I mean, like, I don't care, Karis, if you watch my videos. It's not for everybody, because it is for biology nerds, right? I just don't know how many biology nerds are out there. I hope there are enough of you that like my content to give me a wage. I think it's a little different. Art gets a little polluted when there's financial incentive, but yeah, undoubtedly, I need my art to be liked, and I will. But I need it to be authentic too. If here's the thing, if art that came from my soul sucked, I would like stop.
Yeah,
That's so mean to say. Yeah, if my soul sucked and it's just like, well, this is the best I can do, this is who I am, and if nobody cared, I'd be like, oh, okay, maybe I'd do something else. But I believe in the ability to improve, so it's not even, you know. But where does the improvement start? You can get into a whole thing about that. But what about you, Pembu?
Yeah, I think to an extent I am in the same boat as you because I'm a VTuber who at the moment does not have a full-time job because I quit to do this. Um, but at the same time, yes, I think validation is good. But I think I would still be creating music even if I weren't being validated.
Yeah, and I think that's the difference between us. I would not do creative stuff if I didn't have an audience.
Do you think your art would be sorry? Continue.
No, go ahead. Go ahead.
Pepper, do you think your art would be different if people didn't like, or like, you know, yeah, if you didn't have like any sort of validation? Like, because I feel like if I did my art and nobody cared at all, I would still do it, but I would be angrier. How does that like translate to you?
Yeah. So as I said before, because I make things primarily for myself, as long as I'm okay with it, it doesn't matter what anyone else thinks.
That's
At
confidence, dude.
all?
That's unshakable confidence. I love
Yeah,
that.
that's
Yeah, because me and
that's
Karis,
really
we're like little
commendable.
insecure babies, dude. We're like,
Yeah, we're like, no, people need to
no,
like
you
my
need to
stuff.
like me. Here's the thing: I've trashed projects because I hated them. Like they came from my soul, and I looked at it, and I was like, no one cares. And haven't seen the light of day, and I don't like them. I acknowledge that they were important for my journey as an artist. Like, yeah, it's important that I made videos I didn't like, that nobody ever saw. I didn't even give them the chance, is the thing, because I know it's bad, so I'm not going to show anybody. But it's the best that I could do at the time. I am very motivated by audience sentiment, as terrible as that might be.
Yeah,
I think it's human though, is it not? Sorry,
Yeah.
you were saying something.
Oh no, you can follow your thought first.
Like I just wanted to briefly say that, yeah, isn't that human though? Like it's maybe like a flaw, and it certainly is not to imply that the opposite is inhuman per se. Like I think there is a very admirable and also very likewise human quality to being like secure and being confident and like being knowing what you want out of something. But isn't it also very human to want to be wanted, to be liked, to be appreciated for what you're trying to do, especially when you're like, I guess, you know, putting your soul out there, you're like handing it out to the world, and you would probably want people to go, yeah, I think that's pretty cool. Maybe
But
not everyone,
I
but like
think-
just somebody.
I think what the difference is that the action of making a Fight the Neutrophil video in a vacuum, terrible. If there wasn't an audience for this, I wouldn't do it, right? The thing is, with my art in my YouTube channel, I want the audience to experience the same joy that I feel when I'm studying biology. And I will do what it takes to get that to happen. But dude, I hate editing. I like script writing. That part's the best. Recording, editing, thumbnail, terrible. I hate it all. But it's worth it because I have some validation that, at least for some, this is why comments are important, by the way. Numbers are great, don't get me wrong, but the comments are what's really heartening, because I have a belief that there are real humans on the opposite side of this screen that are experiencing that connection, right? The thing in my brain, the good thing that happened in my brain that I want to happen in your brain, it's happening for some people. And I love that. That's the best feeling in the world. Doesn't matter what the number is. Well, now that I'm doing it full time, numbers do kind of matter. But before, when I was doing this part time, it didn't matter as much. And I think the difference is that something like music, if you just live it, then the action is pleasurable. Whereas making a video it's a kind of a drag. I don't get intrinsic reward. I get extrinsic from the comments and the attention and all that. So I think it is human, but I'm saying that like I envy people who have the relationship they have with their art where it's more natural, flows easier. And I'm wondering what you think about that. Anybody?
100% relate on my part. Like that has to be. It's so important to see a comment. I don't really care for like the viewership or like the likes, but like one comment is like, I don't know. Like if I had to quantify it, a comment is like 500 views or something. Like a single comment is like no longer just a statistic on a screen. It's someone sitting down and going like, "Hey, I really liked this," or "Oh, I really found this part funny," or like, "Oh, I think you could do this next time," and that is like a real person. Hopefully, it's. I don't think I've encountered anything that sounds like it's been written by a bot. Yeah,
I have comments that look like bots.
Really?
Yeah, for
do you
sure.
think they're real people who type like bots?
I get a lot of highs
you'd
and
be
hellos.
Sorry, Pimpu.
I get a lot of highs and hellos in my comments for some reason.
It's just like who is doing this? Maybe they're real people, but like yeah, when you get a weirdo comment, you're like, "Are you a bot?" Like, is this? Because sometimes it's just like nice video. Okay.
Yep.
A bot could have
Nice
written
video. Yeah, but it might have been a real person though, who just has yet to really like express themselves or learned how to express themselves in a way that's not been
I shouldn't be too
grossly
harsh.
like misappropriated by like people who run AI bots right now.
Yeah.
Yeah, I just assume they're all teenagers or something.
Yeah.
Oh man. Yeah. I mean, I get it. One of the actually little behind the scenes for the stream. One of the incentives was going to be, I was gonna like roast terrible comments that have been left on my videos, but I don't want to be like the roast guy. I don't want my content to ever look confrontational. But behind the scenes, I
You
keep
got the
showing
debate bro
screenshots.
in you.
Yeah, I don't want to. I have debate bro in me is the thing that I am also. I'm not that different than you. The difference is I don't want to. I am ashamed of that part of my my being. I don't like that I have debate bro in me. I rejected this aspect of the self. Because it's like not as a bard for biology, you know, I don't want to be the Phi is the call out guy or Phi is mean to his commenters, right? I'm sure like most of the comments are great. Just some of them are like, what planet were you born on? What actual planet were you born on? It wasn't Earth, let me tell you that. I do it. Just this one comment. Just allow me this one. It's sad boy hours. Can I have one for sad boy hours?
Yeah, go for it.
The number of comments that deny the existence of viruses. Full stop. Not COVID. Okay, COVID not existing. That's so yesterday. That's boring. Boring conspiracy. No viruses. No bacteria either. By the way, a couple of these comments. Germ theory, not a thing. I was like, what? What are you talking about? Have you never eaten like 3-day-old leftovers? Like, have you never had a food poisoning incident? Where do you think that poison comes from?
The government.
The government? The government snuck into your home and poisoned your leftover food? Like, what? How do you people put 2 socks on every day? Like, I know, and I have to reel myself back. Conspiracy theories come from a complex milieu of socio-political factors that blah, 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 blah. But sometimes in the comment section, I just want to like scream. No more. That's the only comment you'll get out of me because it's really unprofessional to rag on these comments, but I just needed one for sad boy hours. Germs exist. Holy cow.
Tembu, have you ever had a comment that's made you like just boil with rage or anything similar?
No, I barely get comments, and any comments are either neutral, like the highs and hellos, or they're positive. So
Mm-mm.
No
No one's like, your art sucks, I hate it. Like no one's done that yet.
not yet.
That's
I still have yet to get a hater.
Oh, that'll be a monumentous day. I think I like turned to Toxo when it happened to me and I was like, hey, my first hater, and it was a happy moment. And then after months of bad comments. I'm like, look at this stupid comment. Look at this goddamn
I
stupid
hate
comment.
this guy. I don't know who this is.
I don't know
Probably
this
a
guy,
teenager,
but
but I hate this person.
yeah, I don't comment, I don't delete it or anything. But just like I'm reading these comments. The meme about like YouTubers being like, you know, you can read 100 nice comments, and then that one comment will just, you get one guide, and I'm like, yeah, it happens. I thought I was better than that, but I've been getting one guide a lot recently. Stupid comments. So it does help though. The more good comments I see, like there could be a day where it's just good comments, and then I'll have a good day. And then when there's a bad comment, I'm like, well, god damn it. So good comments are still very important, but man, the power that stupid comments have, I'm like, I am so tilted.
I've had to only on one occasion, thankfully, but hide comments and like delete them because it was like really, really, really bad stuff. I made a stream on the male loneliness epidemic.
I was
explaining
there.
my brief thoughts
I
on it.
was there.
Yeah, and it was like, ooh, the comment section was so bad that I had to do it. And I have a pretty high tolerance for like for BS, frankly. Like I think I have a really I'm willing to hear people out, even if I think they're totally wrong. Because like, you know, the
That's your
debate bro in
job.
me is
You're a
like,
debate
even
bro.
if exactly, like even if they're wrong, I need to preserve their argument so I can take it down. So I can like, you know, not like by deleting it, but by rebuttaling with my own thing. And like I hold very strongly to that. But like so someone like me, I had to go into that VOD and delete comments because it was really they weren't arguments. It was just stuff that was pure hate speech, and I was like, oh my god, no. I know and that's on brand for like sad boy hours, but I do want to kind of twist the question in the opposite direction to hopefully get a little bit of positivity out of this. Have you guys gotten any like really, really nice comments that have truly like touched your heart?
Yeah. I've had a couple that have left a big impact on me that I've like read over and over again. Um it's the idea of like, I had the intention to create a feeling in you. It is the same feeling that I had. And just comments that are always like, you know, this made me feel a certain way. I'm like, that's the point. Biology is beautiful. If you learn something, that's bonus for my content, philosophy, right? I'm not an education channel. If I were an education channel, I'd be giving you quizzes. I'm an entertainment channel. I use what I learned to make things entertaining. And when people fall in love with that kind of biology, when people say, you know, I'm a whatever year medical student and I love your videos, I'm like, oh, I couldn't do what you do, but you know, that if I can make biology go down smoother, because you got to learn a lot of it when you're in med school, you got to learn a lot of it when you're in school for biology and other science related subjects. And like, it makes me really happy to know that there are students out there who are choosing to stay with biology in some form or another, because I'm not going to own it completely, but at least in part due to seeing the passion that I have for it. Because if it's not biology for me, I'm a very passionate person. Could have been anything that I'm really into. The feeling of sharing stuff that you like is so basic and so human. So I do read those comments over and over again. And to address Hareday's comment, I thought the ratio was like 10-1. It is more like 10-1. I was being exaggerating, because I think there's an interesting relationship between the time a video is published and the quality of comments. Like I might have to do I might blame bots. Like legitimately, I might be blaming bots. To the first 72 hours after a video gets published, oh, the praise, the adoration, oh, nom, 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 give it to me, nom, 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 I chew that up. You get a month out and you get real weird stuff in the comment section. You get real weird takes. And that might be that like YouTube bots are like prowling for certain like keywords, like virus. Like I did 2 videos on viruses back to back, and the wildest takes on viruses are there. And I'm like, I don't believe you're a real person. And I'm sad because your comment is dumb. But yeah, and then those are good comments. Helps me remember that there are real people across the aisle here. Need to hydrate. Oh my God.
Hardy says, a guy said that I use like 5 words to scar him for life. I want to be responsible with this power. I think it's so much easier to be mean than it is to be kind. Because if you are kind, generally it's super generic stuff like you are a person, you're valid, or you know, this is good, or I like it. But like, and that is somewhat to me easily replicable. Oh, nice, like the nice video comments. It's like it could very well be sincere. It could very well be sincere, but I wouldn't know because of, unfortunately, the platform that we exist in, and like the existence of bots and the existence of being able to copy and paste and manufacture human interaction, and
It
you know, modify
exists.
it, right? Like it's so much easier to be mean because you can be a bot and be mean. You couldn't be a bot and be kind, because I think the meanness of it, I think that the distinction between like a nice comment and one that is intended to hurt you is like a bot could write a mean comment and it does its job if you get mad at it. If a bot were to write a nice comment and you felt like appreciated or whatever, I would feel like it's not doing the job of reaffirming and validating your existence because it's a bot. But if it's getting you mad, it's doing its job and like, you know, getting you mad. And so because of that, I think in general, it's so much easier to be like horrible to people.
Dude, I love that because I've been talking about this for the last couple days. Actually, the idea, like, and I'm not the first person to. I'm sure you say this all the time as a philosopher. I'm not the first person to think of this, right? It's like so easy to be mean, but I think in my context as a biologist, as a person who had a couple years of academic training under my belt, it goes the same for science as well. You read a paper, it's really easy. If you have a little bit of training, you can point out a lot that certain papers are doing wrong. Because science is hard and you can't have a perfect experiment. My most famous example is there's a paper published by NYU about vaping mice. Okay, a big deal on Reddit. And that paper, not good. I admit that. The paper's not good. So what they did was they got mice, they hotboxed them with vapor from vapes for like 8 hours a day. And lo and behold, when you did that, the mice got cancer. They were trying to show that vaping is not as harmless as you would expect. But when you posted this on Reddit to everybody, they were using mice. They were hotboxing them from 8 hours. This is not how people ingest vape. Yeah. Okay. That's true. It's so easy. It's punching down. What can you learn from this? Well, what you can learn is that there is an upper limit. There is an exposure limit. You can't have free lunch forever with vapes. But no one acknowledges that because it's really hard to actually find out what is good about something. It takes a lot more effort. It's so easy to because you know why? Because I've been thinking about why this is. Like, why is it so easy to be mean and point out when things are bad, then be kind and tell you when things are good? I think it has a lot to do with our, and you know, this is a little bit of evolutionary psychology, which is not we don't stand exactly in this house. But this is sad boy hours. We're philosophizing. This isn't really psychology. This is philosophy. It is what I call psychology with no evidence. Okay. That's one just for you. But if I'm philosophizing, I think it's because we have an immediate reaction to disgust. You know, you look at an AI painting, you see those extra fingers, ew. All of a sudden, everything you think about is extra fingers. And evolutionarily speaking, this makes a lot of sense. Disgust is an important protective reaction meant to protect you from ingesting things that you should not be eating. But this attitude kind of extends beyond just eating weird plants, right? Or eating foods that taste bad. People that act in a way you don't like, disgust, immediate, protective, get away from me. You being around me is bad news bears. But kindness is something that you have to like feel and like learn almost to a certain degree, right? Starts from birth. You are the recipient of love. You are shown how to love. And a child that doesn't grow with love, you know, it's not innate in the way that disgust just triggers instantly. Ew, disgusting, bad, I hate it. You see this a lot with like shows, right? I just watched Has-Been Hotel. I liked it, okay, Twitter. I liked Has-Been Hotel. Can I like it? Okay. Can a guy like a thing?
You know, that means you hate other thing? How could you?
That means I'm problematic or whatever. There are good things about Hasman Hotel. Okay, but if you go online and you search Hasman Hotel video essay on YouTube, you would think it's the worst show in existence, man. It's so easy to say what is wrong about a thing. It's not even intellectually stimulating anymore. Like, I don't watch video essays anymore because, I don't know, it doesn't feel interesting. Yeah, okay. Whatever property isn't perfect. Okay, I probably could have guessed. What's better content is, why is it good? Because, and I think I've been thinking about this point in particular, there's this old argument that you should turn your brain off when you watch whatever or play whatever, and that idea got a lot of pushback in the mid 2010s. Turn your brain off. The reaction was, why would I turn my brain off, right? You're asking me to be more stupid when I'm enjoying media? No. That's not what this is about, okay? Turning your brain off means that you are allowing the art to interact with your emotions directly without the annoying filter that is your thinking mind, okay? Because I would consider myself to be someone who thinks a lot. I don't think I'm smart. Not a lot of these thoughts mean anything, but I do run my stupid brain a lot of the time. And the problem that causes for you, if you're ever in therapy, is you get really good at coming up with stuff to justify your own attitudes, right? This thing on the top of our head can interfere with your heart. And I think that these stupid video essays that are so critical of everything, yeah, I mean, it's attractive content because people will eat that stuff up. But I think life just gets better when you not necessarily turn your brain off, but allow the art to affect you emotionally. And then you think about why you had that emotion. And then you can learn something about yourself. Or why did I feel anger at this? Or why did I feel happy? Okay, that's not turning your brain off. That's allowing the art to hit you directly and then thinking about why that is. And then you actually build something constructive. You learn something about yourself instead of tearing a piece of art down. I'm like super salty. Dude, fandom sucks. I wonder what you think of that.
I think that the turn your brain off sentiment is I think what it should be is it should be turn off your attack mode for a second and try to see what it's trying to do. Because I think it's a lot easier to just attack and go like, oh, and nitpick and say this thing is bad and this thing is bad. And the reason why it has such stock in this attention economy is because people want to feel like they have the real opinion, and that can't be the one that everyone has. So popular thing comes out, the cycle of thing that is dunking on it. It doesn't have to be too popular, really. Just something that you maybe have just heard the name.
It's relevant, yeah.
Cycle of the video essay that comes out dunking on it or trashing a specific aspect of it will do well and be popular because I think it's partially because people are motivated by this desire to want to feel like they're thinking for themselves. They have a real opinion, and usually that just becomes to be critical and very and to perceive popular thing as negative. Like that is all it looks like to them, and it's not because they're actually thinking. It's because they want to wear the guise of thinking. And the way that presents itself oftentimes is just disagreeing with popular thing. And you can disagree with popular thing. And it does take, on some level, if you had no thoughts in your head, you would just agree to everything that's put onto you. But I think this is only maybe like one step in the thinking process, which is to first at least challenge and question the thing that's in front of you. And that challenging and questioning doesn't have to lead to a takedown. But if it doesn't lead to a takedown, it doesn't look like you're thinking. Because if I watched K-pop Demon Hunters and I just said I liked it out of context and I don't explain why, people are gonna think, well, it's because you're like an uncritical person who has no media literacy, blah 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 blah, and you'll never see the end of it because people want to prove that they're smart so badly without actually having to do the more complicated and also less rewarding work of sometimes just going like, yeah, popular thing is good because it was made well.
It's just this is a perfect example of Penbu, are you on TikTok at all? Like, do you?
I'm not. No.
Okay, there's
thankfully
a TikTok that came to my attention. I don't remember what the song is. Toxa, do you remember?
I don't even know what TikTok you're talking about.
Oh, the duet one.
Oh. No, I don't know what the song
Gosh darn it.
is.
Okay, basically there was a duet, beautiful duet. TikTok has a duet function. There's the first singer and she's singing a song. I don't remember what the song is. And then a lady duets with her. And the harmony that she chose was pretty discordant. Like it clashes, right?
I remember this one.
Oh, you do know this one. Okay, great. And the crap that lady got on TikTok is a terrible crime.
It's As the World Caves In.
Yeah, as the world caves in. And I get so mad when I think about this because it's exactly as you said, Karis. It's like kids that like just finished Music Theory 1 or something, like going, oh, her harmony is clashing. Clashing equals bad. Bad equals you suck. Like chill for a sec. Can you chill? Can you like feel the emotion and the effect that discord causes? Not everything has to be a chord that resolves well. I'm a music noob and I know that, right? You strategically can introduce chaos into a composition and it makes it more interesting, right? It is a tool. And I happen to think that sounds pretty good to me. But the desire to flex that you know something more than somebody else, man, that's so annoying. And I really have a distaste for it because I used to be like that too, as an annoying debate bro kid. And I had to take a step back because these are children probably who are crapping on her. It's just children crapping on children. But it doesn't make it any less aggravating because you can see how bright the light is at the other side of the tunnel where, dude, music is better when you just like it. Why would you choose to hate more things? Like, that doesn't make you happier. And this isn't about toxic positivity, right? But if you had a choice to like more music or to not like more music, why would you choose to not like more music? It's so makes me mad.
I think part of it is not just that they choose to dislike it per se, because that could just be held within. That could just be a subjective, like, I didn't like that. I don't like
And
that.
that's
But
fine.
it's
fine.
yeah, but it's the putting it out there. It's the hate comment, right? It's the attack that I think is worthy of the zooming in on, because I think my and a reason I say theory is because I can't test this and I have not personally quite experienced it, perhaps to the same degree. But I think part of it is because it's part of what motivates like hate commenting, not just dislike, but hate commenting is the desire to be felt, the desire to have an impact on the world that exists in everyone and is unmet in most. That like you feel as though you're alienated from everything that you do around you and you have no power and you have no influence on stuff, that you feel as though just that little bit of like, okay, yeah, I'm doing something. My actions have caused something in this world to happen, namely this person being sad or whatever, and I feel so superior about it, is negative and it's toxic, but it is sort of validating because it's like at least the feedback is like, oh, your comment got to them, or your comment is like agreed
Yeah. That's why
to
I
be
won't
like, this critical thing.
That's why I won't ever respond to these hate comments. I don't want to give them what they're looking for, which is the ability to know that they affected change, which is just so sad. Like if you are someone who leaves these comments, I know this is a common refrain, like, oh, I feel bad for these people. But like these people need that clapback, right? They need it. They need it like food and water. and that is so depressing. Because sad makes it feel like, oh, I'm better than you. No. I acknowledge that everybody has this desire, but like whatever happened in your life where you need me to clap back at you right now, I hope things get better. That's so... I'm depressed on your behalf. You might not know you're depressed. I'll be depressed for both of us when you leave me a terrible comment like that. Exactly. It's and because it's also fun to be critical, right? It happened to me in graduate school. The minute you learn, oh, science isn't perfect, I can pick apart everything, it's addicting. And my professor, my advisor, actually be like, stop that. Like, stop. She actually sit me down during my PhD and being like, the point of reading research is not to criticize it. It is to find out what is true. And you are doing a bad job of finding out what is true if the only things that you can think to say are negative. That conversation changed my life, changed how I look at art too. You're right. The point isn't to nitpick. The point is to find out what is good. And for some pieces of art, there's nothing good. That's valid, right? But you have to try. It's harder to try to find the good than it is to find the bad, because the bad sticks out like a sore thumb. The good you have to dig for. The good you have to dig for. So, interesting comment. Criticism is required to prevent media and genres from getting overwhelmed with slop. I mean, hypothetically, I think that's true. I mean, reception is important. Hollywood sees their numbers go down. That's bad. Do we need media critics to do that? That's kind of the idea that I've been doubting more and more.
I think we need media critics and media critics, and all of them need to
it's all turtles.
have some the philosopher. I think like the most important thing is, like, to me at least, for every valid criticism, like, I'll play a video game on my channel and I'll like critique it all. I'll say I didn't like these parts, but I will only do that if I have a proposition for what could improve it, not by turning it into something that I would make, but trying to turn it to something, to more of the thing that I think the creator wants to make. And that is monumentally more difficult than just saying this is inconsistent, this is bad, because it requires you to stand in their shoes and think, okay, I want to make this piece, I want to make this game about these characters, or I want to explore these themes. Like, if I were to do that, how should I go about it? And if I, as Karasufino, disagree with the artist here, and I say I didn't like this, it's not because it's not to my tastes, it's usually because I think I see what your vision is, and I want to propose this alternative in achieving that vision that I think would be just as sincere, but maybe avoid some of the pitfalls that come from this. A quick example would be me playing Detroit Become Human recently, and I love the game to bits. I absolutely adore the game, but there's like stuff that's really wrong with it that, like, it deserves my intense criticism. But my intense criticism is followed by like okay, here's how we fix it. Here's what I think improves it. Here's how we do it, without sacrificing the whole like thesis of the game and the experiences and the emotional weight and juggling all of that. And only in doing so does I feel like criticism, that criticism feel like not valid maybe, but does criticism truly like matter, is because like I need to show that I just want this thing to improve. I'm not trying to take it down.
As a person who minored in rhetoric, my angle of trying to, be more cognizant of this kind of thing is that I think you can fix criticism by just rewording things a little bit, right? Nothing is good, nothing is bad. Shut up. Don't say those words. I mean, not speaking to you, Karis, but in general, I think those are bad in a scientific sense. Those are what I would call, as a biologist, imprecise. I just hit you with the imprecise, okay? Get used to that, hearing that a lot in your biology education. Ooh. When you start writing your own science, you're going to get burned over coals for using words that don't have intrinsic meaning. Good and bad, terrible words. Not well defined. Shouldn't use them. Good and bad, those are words for Karasufino when he describes morality and ethics. Okay? From a criticism perspective, maybe you wouldn't even use those words, honestly. Like,
it's a little dicey there, but yeah, continue. Your point stands.
Instead of good and bad, we should reframe art criticism in effective and ineffective. This is a much better rhetoric, in my opinion. Why? Because the artist had a goal. I want to make you feel sad in this part of this video game. It either worked or it didn't. Was it good? Was it bad? Who cares? That doesn't mean anything, okay? Did it make you sad as the audience, intended audience? Did it make you sad? If yes, it was effective. If not, it was ineffective. That's a much cleaner way of describing criticism. And then you take out the bullying, okay? The lady doing that harmony, was her intention to make a harmony that causes some anxiety? Because the song is about the end of the world. Her intention was probably to create a harmony that does provoke a little bit of that anxiety. I would say that's fantastically effective. Now, just because it's effective at the original goal doesn't mean it is suitable for everybody's taste. So that's why there are two dimensions to this. Effective, not effective, like and dislike. Do not use good or bad, because that makes you into a tastemaker, which is outsourcing your own creative ability to somebody else. I think tastemakers, bad. I'm using the word bad, right? when you are looking towards these YouTube video essayists, you're outsourcing your own ability to think. And in an age of AI, you need to retain that ability. You know, listen to the music. Don't listen to what other people are saying. Maybe if you want a recommendation, that's totally fine. Like, hey, this video is good. I think you'll like it. That's a recommendation. That's different. However, if you see, oh, Hazbin Hotel sucks, and then you just assume Hazbin Hotel sucks, you've just outsourced your thinking. Terrible. So instead, when you talk about art, and I know YouTube will never do this because the thumbnail Hazbin Hotel is ineffective, that doesn't get clicks. Or I did not like Hazbin Hotel, that doesn't get clicks. Hazbin Hotel is bad. Now you're starting to provoke emotion, right? You are starting to grab at people's desire to crap on the show. You are trying to form a group identity as people who hate the show, right? I know people won't do it, but if my audience, just a little language change can really make a big difference in the way you think of things. Effective, ineffective, like, dislike, no good, no bad. No good, no bad.
And Boo, what do you think? What are your thoughts on like musical critique, right? In regards to the TikTok, but also maybe just in general.
Yeah. Well, first, I guess the point that Fi had made, I think I can stand behind that because I am pretty guilty of using not ineffective words, imprecise words, but mostly because I don't want to form a complete thought. Sometimes. But yeah, I think in terms of music critique can be useful as a way of one, I guess, exposure to things you may not have seen, but also to reframe things in ways that you may not have thought of. So Adam Neely, for example, did a video essay or some sort of review or something of that TikTok, which
That's
is why I
the
know
one I watched. Adam Neely, the goat.
Yeah. And one, I hadn't heard of it before that, and two, it exposed me to like different ways of seeing why, like the thought process behind it or whatever. But I think, as I was been touched before, I think if you take that and then not process it and think for yourself, then that's when it becomes, to use the word, bad. Because I think it's one thing to take opinions and opinions and whatnot from others and take that as input for your own critique, or I guess critique would be an okay word to use there. And then another thing to just take that wholesale and own it as your own.
Yeah, the dogpiling, the excuse to cause misery to another human. It's easy. If you I know a thing or two about music theory, crap on this poor lady. It's terrible. Yeah. Oh man, how are y'all doing in terms of the time of day for you all?
I am just about to go. I think I have to leave.
Oh, great! It was nice
in a few
chatting with
moments.
you.
So yeah, thank you so much for having these sad boy hours. I love being sad. I'm really good at being sad. I'm really good at being a boy. I don't know which one I'm better at the moment. I'm still figuring it out.
Competitively ranked boy, competitive MMR boy.
But yeah, thank you so much, Fi. It was lovely to talk to you. It was,
I'm glad you could be on the channel.
Lovely speaking with you as well, Penbu.
That's everything.
We should talk more. I really am interested in like the, I guess, musical discussion really is where my headspace is at right now. And Toxo, if you're still here, if she's still there. But yeah, thank you so much, guys. I'm gonna go now. You guys can now talk about me.
You want to plug your channel or anything before you go?
Oh yeah.
Where people can find you and your Discord, your Discord from hell.
You can find me on YouTube. My Discord from hell. I think. Hold on. Let me just type.
Yeah.
Hello, it's me. So you could just click on my name, I think, and that just brings you to my channel. And if you're on my channel, you should be able to find my Discord on my channel links, I think.
Well, people can't actually click on YouTube unless you're moderator.
Wait, really? No.
Yeah, but you can
Never
copy
Or
his
if they have
app.
hyper chat enabled,
You can
or
copy his
install
app
that.
Hyperchat.
and then search it in
Okay.
the YouTube search bar. It should be okay.
You could do that. I'm gonna link my channel there too. I think that's what the link is.
Yeah, one of the nice things I think about the handles. I mean, I don't like it in general, but at least you can copy and paste the handle and plug it into your URL.
Mm. But yeah, that's gonna be it for me. If you want philosophy content, don't come to me because I don't serve content, or I try not to. Well, I'm doing a bit of I'm doing a bit of a subversion of that.
Thank you,
I'm actually
Toxel.
working. It's given me like a an existential crisis that I think everyone will see really soon. But I'm working on like content. I'm working on like a trolley problem video, and the more I dig into it, the more I'm thinking about like the hellscape that is philosophy on YouTube at the moment, or at least the way I think philosophy should be done. And it's a whole can of worms. But anyways, if you want content, you'll get probably 1 or 2 pieces of content from me, and then the rest of it is just me being me. No, I'm not. I wouldn't debate you. Look, I wouldn't debate people, but I will maybe make fun of you if you have a bad opinion.
All right, looking
Maybe. No
forward to that
promises,
trolley
no
problem
guarantees.
video. Definitely looking forward to that. I'm sure it'll be pretty spicy.
Yes, but that's everything I have to plug, I guess. Thank you so much for having me.
Thanks
Please
for
enjoy the
stopping
rest of
by,
the conversation and the
Karis.
stream. Good luck on the 24-hour stream. I haven't watched everything, but from what I have seen, oh my god, that's a lot of stuff you're doing.
Oh yeah,
And it's really cool.
Yeah.
So good luck on that.
4.5
Thank you.
more hours.
You could do it. You could do it. You could do it.
Thank you for the
Good luck. Good luck. Good
encouragement.
luck. Bye.
Bye.
Yeah, as for me, I should still be good until the end of the segments.
All right, cool.
I
appreciate the
cheated
company.
and took a nap before this.
You know, I stayed up recently all night to watch the League of Legends tournament, the worlds. I can't even remember, but yeah, for there I also cheated and took a nap to watch it because League of Legends Worlds was happening and it was overnight in my time zone, and I like nodded off. I'm like pretty good because talking is keeping me active, but boy, this is hard. Okay, 12 hours I thought was very doable. 24, I don't know if I do this again. I'm having a rough time. I almost use the word bad time, but I'm not having a bad time. I'm talking to my friends. I'm talking about cancer. I'm talking about biology. But it is hard.
Yeah, it's certainly a big jump between 12 and 24.
Yeah,
I've done
some
an
would say
11-hour
double.
stream where I had recorded an entire cover by myself. Well, the first 3 of which were wasted because I didn't use a metronome. I had to start from scratch.
Oh my god. Dude, I hate that, man. Remaking content is crazy. Like I that is the most fun. Like when I have to re-record a part of a script, I like it's not even fun, right? The first time doing something, then redoing something. I think Sideray showed me a video from Matt Pack talking about the creative process on YouTube and being like, there's a chart that you can graph your happiness or your satisfaction with your art as a function of project completion. So you start your project, you're all happy, it's a great idea, you start working on it, you're a genius, and then as you start putting work into it, you have to redo things. It's not as good as you thought. It doesn't actually work, and then it blows, and then you feel really bad. But then you start working at it, and then you start feeling pretty good again. And by the time you publish the video, you're mostly okay. Otherwise, you wouldn't have published it. And that video by Matt Pat, I think really, I don't know if that's universal. Like when you compose or when you make music, is it like how deliberate are you with music design? Does it just flow out of you and then you listen to it and you go, oh yeah, that sounded good? Or is there like when you make music, is there a sound in your head and then is it when you make God, sorry, I'm again 19 hours in. When you make music, is it primarily reactive? You let it flow out of you and then you refine it? Or is it prescriptive, meaning it comes from an idea you had in your brain? I find that to be pretty interesting.
Yeah, I think it's a mixture of both. And I think it also does depend on the piece. For my workflow, I tend to come with an idea that was inspired by some sort of improv
Mm-hmm.
most of the time. So that part, I guess, is reactionary. And then I'll use theory to build on that. And so that part is a little bit more intentional. But it goes back and forth in that whatever I hear coming out of that theory, maybe I'll have more ideas or whatnot. But on the opposite side, uh, my latest piece, One Step Back, Two Steps Forward, released just a couple weeks ago. Um, that one I took a bit more of a methodical approach where the piano would do an 8-bar melody and repeat that, and then I had the lead guitar also do an 8-bar melody and repeat that, but shifted so that so the piano and the guitar are never playing the same two things at the same time. And so I built the first half around that idea, and then see where that took me.
Meaningful.
And then eventually I reacted to that and felt that idea was starting to get stale, and so I made a transition and then turned it into something a little bit more unified. I don't know if unified is the word I would say, but I let go of that restriction.
Some musicians in chat contributing too. I find music to be so interesting, man. Like the art of music to me is so foreign, and maybe that's why I'm obsessed with it currently, like Karis is, because it is so direct, I think, in a way that feels magical, because the rules of music are meant to be broken, but you kind of have to like know how music is built, and that requires some skill that you have to develop. And I think the iterative process is kind of interesting because you can have it flow out of you, and other musicians in chat are, I think validating that idea. Sometimes the noise just has to come out, and then you use your, I mean, that's so pure for an art, right? As I was saying, you feel it first, and then you can refine it with that thinking head of yours. But it's important that it captures some element of some emotion that you have. Otherwise, what are you doing? You're just engineering tunes for the top 40 or whatever, right? Built in a lab to appeal to mass numbers of people. But real art, as opposed to craft, has to come from a feeling that you have, and that is something that I think is true.
Yeah, the way I like to say it is that music is discovered, not created. And so
that's cool.
I think theory is a tool that you have to discover music, but it's certainly not required to, I guess. So there's a lot of tools at your disposal, theory being one of them. And then your emotions, your insights are another tool to help you see some of the gems that might be lying in there.
I'm so jealous, man.
Yeah, Apps is here.
Jealous.
Somebody from my community here is for me, I hear in my head sound sometimes sensation playing it as well as the texture of what I feel sound is. And accidents happen, like pressing the wrong note inspires a whole new line of thinking. And so that I think embodies a lot of, or like one aspect of music is discovery, because I get this sometimes too, where I had meant to do something else, but then I pressed the wrong note or entered the wrong key or whatever. I got something different, but this sounds interesting and I wanted to explore it.
You can tell me if I'm off base here, but I've noticed the correlation, the thinking scientist that I am. I think the more experience that an artist has in their given field, the more overall optimistic and positive they are with regards to art that other people make. And the reason why I thought of that as you were talking is that, like, yeah, I mean, like the TikTok kids. Like, okay, you learned what discordant harmony is. Clap on the back. But I think when you do something for a long time, and this is hypothesis because I would consider myself to be a very young artist. I'm not young, but I only started experimenting with making art in a very serious capacity when this channel started. I had been making art for a while, but all bad and sporadic. Fi is my first very, very serious, put my whole heart and soul into it project. And I find that as I do more of it, the less critical I am of other creators. And I wonder it's because you have these insights, making music for so long, that you begin to actually enjoy more music more. Like you've learned to love more. Would you say that that is true?
Yeah, I think there's certainly an aspect of that. Um, for the way I see it, I think is that as you become more experienced, you know what the stumbling blocks are for people, and you realize that people start from that. Like people are always at some point in their art. I mean, I don't want to say career, but art progress or whatever. Um, and you started there too at some point too. And so you know what they're going through. And even if for somebody who's further ahead than you, then you realize as you have more experience that you have things that you can learn from them, right?
Yeah.
And so you
Sorry, I
have
just
the
realized
empathy
that
for the people who are. Sorry, go ahead.
yeah, so sorry. I would just say I forgot that I don't know how long the pin was gone for, but the pin for the donation link is up again in case anybody wanted to donate for the cancer charity. Sorry, that's all I wanted to point out.
That's all right.
Thank you, Toxic, for
Um,
noticing.
so yeah, you have the empathy, or you develop the empathy for the people who are uh who are I'm blanking out on words, who are behind you in progress, and then you have the excitement to learn from the people who are ahead of you. Now, of course, there are some negative emotions that could get in the way of that, but um, I would like to think that as you gain more experience, you get less and less of those, or at least you learn to deal with those negative emotions.
And I think there's also maybe an aspect of like, as you were saying, like you, because you have traveled this journey quite a long time, you can tell and you can give more grace to artists that have less experience than you because they're walking the same path and you've walked in those shoes before. Do you get a sense that like for those less experienced artists that you give them more grace because you're like, I can see what you're cooking, like I can imagine what it is that you are actually cooking, and I'm excited for what you are cooking. So it might not be polished, but you can kind of tell that it could be really good. Do you feel that way?
Yeah, no, I think that resonates with me a lot because as somebody who is less experienced, you have a vision that you might not be able to execute because you don't have the tools to do so. And so I think if you have the opportunity to work with somebody who is more experienced, right, the two of you can bring that vision to life more effectively than any single one of you could.
Yeah. No, I do find that very meaningful. I just wish that like this faux intellectualism that Karis was talking about, it's just I don't know, they're young. They're young and inexperienced. Can't really fault them for too much. But it's just frustrating because like the art is so much better when you like it.
Yeah, I think it ties a little bit back into that. Now, I know this has been somewhat debunked by other people, but I think there is still a little bit of truth to it. The Dunning-Kruger curve or effect, where
Mm.
um, I think in people who are very critical about things are at the peak because they feel like they know a lot of things, but they don't see what's ahead to learn.
Yeah, I get that. Yeah, graduate school was a big humbling moment for me, right? Because I say now that I don't feel like I'm that smart. Before graduate school, I was more positive. Now I think I'm more realistic. That humility is important though, and I think humility makes your life happier too. Because I think humility it protects you in a kind of way. Because if you think you're hot stuff and then one day you get proven you're not hot stuff, that's gonna hurt. But if you already don't really think maybe you think you're warm stuff. I think that's everybody's ideal situation. You're warm stuff. I'm warm stuff. If I'm shown that I'm a little cooler than I am warm, that's fine. But if you go into every situation thinking you're hot stuff, that's asking for trouble. That's not really wisdom. That's obvious. But I think the difference is that people don't realize when they're getting the feedback of intellectualism from being critical, they are losing sight of the fact that the world is bigger than criticism. I know we've been on that topic for a while, but I just Toxo can confirm. I've been thinking a lot about that kind of idea.
Yeah.
Yeah. It is interesting, I think. Or maybe not. I was planning on making a members only video on this because members only videos is where I put long-winded rants like this because, I feel weirdly enough like, Has Been Hotel, I've been talking about the show a lot. Vizzy Pop, the creator, was a YouTuber. Like this series debuted on YouTube. And I think I feel a kinship in the sense of like, everybody is very quick to point out the things that you do wrong. And those voices can be very loud. But there's also a lot of wonderful things. I'm not an art critique like YouTuber, so I'll probably never make a dedicated Has Been Hotel video. But just to say, like, here's my recommendation. Give it a chance. There's a lot of cursing in season 1. I found it to be annoying. Notice how I quantified it. I didn't say it was bad or good. The cursing in season 1, I did not particularly care for it. But if you can get past it, it's pretty good, I think. Pretty good. Yeah, we got a comment saying that if you have enough unwarranted confidence, your ego can survive even the most objectively humiliating disasters. Sigma emoji. Yeah, but imagine having to live your life like that.
I think you can have confidence and still have humility.
Yeah. That's just called accuracy, right?
Yeah.
That's just having self-awareness, which is harder than you would think, right? Because I think nobody believes that they don't have self-awareness by definition. I don't know if I have self-awareness. I hope I do. That's about all I can guarantee. There's a chance I look back to these 24 hours and I really regret it, especially these sad boy hours. I feel like the stuff I'm saying is interesting. I cannot guarantee that it is interesting. I'm so tired.
I mean, you got a whole chat here listening to what you have to say, so it is at some baseline interesting.
Maybe Chad is insane too. That's also possible. In science, you consider all alternate hypotheses, and maybe Chad is just as crazy as I am right now.
Perhaps.
Yeah. As Tyler and all said, yeah, you watched Has Been Hotel. They tried really hard to be edgy season 1, and then they realized cursing is only funny when you earn a curse, right? Earned curses are very funny. Season 1, a lot of unearned curses, I think. But overall, love the show. Oh my god. The sun. I mean, it's wintertime, so the sun's not coming up, but we're about.
The sun is nowhere.
The summer nowhere near. It feels like it should be coming up. I am so tired, and Perkinji is like, "Why haven't you gone to bed?" And I'm like, "Because I'm raising money for cancer charity."
If we want to talk sad to end out the last of this segment, then I will talk about how I have recently, over the last month, become Perkinji's least favorite
No,
and or organism in this household. Perkinji used to sleep with me every night, and it was beautiful, and it was my happy place to be with her in a nice warm cuddle. But even today, I cheated my way through this, and I went to bed. And you know where Perkinji was this whole time? In Phi's jacket, which he keeps on the floor for some reason, but she was just cuddled up in Phi's jacket in the office behind him streaming while I was, in this nice cozy area, but she didn't want anything to do with me.
No, she's just concerned for me. She's wondering if I've lost the ability to sleep. She was making sure I was okay.
And it's That's my contribution to these sad boy times. We can all mourn the loss of mine and Perkinji's relationship.
No, she still loves you.
Thank you,
she
everybody.
still loves you. This is my TED Talk. No, she still loves
This is my TED Talk.
Just go through
My
a phase.
cat hates me.
No, she can't hate you. She's a good girl. She love you. Okay. I think that's the sad boy segment. I think we're good. Thank you, Penboo, for coming on. Thank you, Toxo, for staying up even though you didn't have to. I appreciate the company. Penboo, you have anything you want to plug?
Sure, yeah. Just to reintroduce myself for anyone who doesn't know me, my name is Penry, the programming, puzzle gaming, and music making VTuber on Linux. There are a couple events coming up. I think I'm participating in the Dup Cup this weekend, a Tetris tournament. And also be releasing a couple music projects in the next couple weeks. So if you're interested in my music, you can go check that out.
Oh, let me grab your YouTube channel so I can plug you.
Much appreciated.
Okay, nice. Okay, so if you're interested in music, and all you do a lot of other things, man. But if you're interested in this conversation that Penboo and I have had, you know, why don't you go give him a follow on this channel, you know? Okay. Thank you so much, Mari, for the donation. And they ask favorite movie from the last 3 years. You don't have to stay with this, Penboo, but yeah, but once again, thank you. I'm so out of it. Thank you again for coming. You can just hang out if you want to hear what my favorite movie in the last 3 years has been. I don't remember, Toxo. What movies have we watched?
We don't watch movies.
We don't really watch movies.
Unfortunate.
I thought Inside Out 2 was cool. I thought the art behind the decisions, because you know I'm a neuro person. There's a lot of design Easter eggs in Inside Out 2 that I resonate with, and I like the Inside Out franchise. There's probably a better answer than that, but that's the first one that came to mind. As you can tell, I'm very invested in marrying your brain with your emotions and not keeping them separate. So that movie I enjoyed.
Yeah, I think I've seen the first one, but not the second one yet.
Yeah, they're a nice set of movies, I'll say. Okay, say goodbye to Penboo. Penboo, say bye. Bye, Penboo.
Bye. Good luck
Bye-bye.
on the rest of the stream, and looking forward to that song.
Thank you so much. Thank you for getting me started on my journey there. Peace.
All right, bye.
Oh, so Apit says, my mom had breast cancer years ago and is still with us today. Thank goodness. Thankful to the nurses and doctors that help her to recovery. Yeah. Breast cancer affects so many people. The most common cancer diagnosed in women. But it is a battle that we are winning. Not won yet. Don't take your foot off the pedal yet. But of the cancers that exist, breast cancer is one of the more you can do something about this kind of cancers. And I'm very happy that your mom is still with you, because that's all we want with our loved ones, right? More time with them. And, you know, part of this charity stream is exactly that. How do we get it so that people with breast cancer, men and women, get to have a longer, healthier life, to spend more time with the people they love? That's what this stream is about. And we are reaching the end of sad boy hours. Says, as happy boy hours. Let's just get started, dude. Let's just get into it, man. I don't care, dude. We're running on fumes and a dream. Do I want any food? Yeah, can you give me a protein bar, Toxo? Thank you. Just something to sit in my stomach as I head into the last leg of the journey. We got a couple more papers. It's 5 a.m. We've got 4 more papers to get through. Okay? We're back to the charity. Okay. How are we doing out there, by the way? How are we doing? Checking in with all of you. You know, it's not easy listening to me yap this long. I cannot wait for that apoptosis. I am so ready to... I'm not going to say it on stream, but you know. We are so back. Yeah, surprise, man. We're so back. Enough of the fun time I'm having with friends. Now the fun time I'm having discussing cancer research. Okay, we're back. Okay. Crazy paper, y'all. Crazy paper. Just read this title and tell me this isn't crazy. Tumor priming by ultrasound mechanogenetics for CAR T cell therapy. It's a lot of words. Thank you for the protein bar. Okay, good night, Toxo. All right, I gotta eat on mic. Hold on, let me take a quick couple bites. I've been awake for a very long time. I need a bit of food. Okay. Okay. The tumor priming by ultrasound situation is insane. Thank you. Thank you for the Charlie meme. What the hell is this paper? This paper by this lab, you can tell a lot of effort went into this. A lot of people are involved in this project. Claims to be able to use the power of ultrasound to weaken tumor cells to the point where they can be cleaned up by CAR T cell therapy. Ultrasound. This sounds like gobbledygook. This sounds like chakras. This sounds like chi. This sounds like fake science. But it's real. And, if you want to believe in chi or chakra, that's up to you. But I think that's more of a spiritual concept than it is a science data-based backing, right? You can believe in chi. That's totally fine. In the lab, maybe not. This sounds like, is it a pun? Yeah, no. I don't intend on anything, Trenton. I don't intend on anything anymore. I am so tired. But this paper is claiming to be able to use ultrasound technology, the same technology you use to get a look at that fetus of yours before it is born. Sound waves that bounce off and give you an image. How could that possibly be used to fight breast cancer? Let's find out together. Okay, it's happy boy hours time. Okay, shot of energy. By the way, I've had one tea the entire day. I am this far in without a single coffee. I estimate I've had maybe 50 milligrams of caffeine at 7 a.m. Most of this has been sheer excitement and passion for this project versus chemical substances. And yes, I am bragging. Let's get into this. This paper is bonkers. Okay? That is the sentiment. This paper is absolutely nutso. Why is that? What is a CAR T cell? This was a question in the Octo Lady trivia stream. It stands for chimeric antigen receptor T cells. What do these mean? These are patient-derived, genetically engineered cells that are delivered back to the patient. So we're going to take your T cells and we're going to genetically engineer them so that they can fight cancer, and then we're going to put them back into you. Basically, plug and play T cells. Because the cancers in your body have likely evolved to hide from the T cells that you have. But what if you could supercharge those T cells? That's what CAR T cell therapy is all about. Supercharging your natural immune system. The problem, or rather, this slide is telling you more about CAR T cell therapy. These are an absolute revolution in cancer therapy. These things are so cool. And they showed up in my previous stream on glioblastoma as well. You're going to be seeing a lot of CAR T cell stuff, man. Getting to build your own cancer killer is incredible. But there's a problem. CAR T cell therapy is expensive. Let's play a game. For a patient, how expensive do you think a course of CAR T cell therapy would set them back by in US dollars? Just guess. This is a miracle. This therapy is amazing. It's also way too much money. In the US, yeah. Let's use US dollars. Yeah. Is this the price of Red Bulls? I'm too tired to remember what those are. All right. The correct answer is somewhere here already. I don't know what the figure is for the per treatment. Because Surprise Fancy gave me your answer in each time. I don't actually know how long the therapies go for. But the amount that you would have to pay for a full course of this is about $300,000 to $400,000. It is not cheap. And it is good. This is some good stuff. Okay. So the thing is, if we're going to have an incredibly expensive treatment, it better work. And it does. It does. It works really well. But they're not perfect because, again, cancer is unfair and it will evolve to escape these guys too, or it is possible. Right? Again, CAR T cells, incredible revolution in cancer technology. Not invincible. How can we help those CAR cells? Because again, if you're going to be dropping $300,000 for this, you better live forever, dude. And it's just too expensive for it to not work completely. That's what this paper is trying to do. Let's try using ultrasound and weaken cancer so that CAR T cells can jump on them. And it sounds like made up, it sounds fake. But big if true, ultrasound, relatively cheap. If you're having a baby, you know, not as bad as a cancer treatment. And the thing is, if it is possible that these scientists can show that ultrasound can play a role in sensitizing cancer cells so that the big expensive CAR T cell therapy can go further, that's a big W. That's a big W. And until I read this paper, I didn't realize this isn't actually that wackadoodle. This is not that crazy. Ultrasound has been used to precisely change the temperature of an area in the body. How late is it? It is 5 a.m. for me. I've been streaming for 20 hours. 20 hours now. Oh, thank you, Penbu. I totally missed your donation. I got too wrapped up. Penbu says, doing my part, have a good rest later. 5. Thank you, Penbu, for the donation. 5 a.m. True. So, ultrasound has been used in the context of developing fancy treatments because ultrasound has a good propensity for penetration. And it can communicate because raising the temperature of a certain area is a signal. And we know ultrasound can penetrate in your body. We use ultrasound to find that baby of yours, right? So we know that the waves can travel deep into your body, making it categorically better than putting your hand into a water bath or something, right? If you want to heat the inside of your body, it's going to take a lot of energy if you want to go from out to in, but waves can penetrate. You don't have to heat up the whole outside of your hand before the inner layers get heated as well. So we know ultrasound has some neat properties that can be taken advantage of. Again, I said this earlier in a different stream. Don't be a freaking idiot. Don't go burning your body. That's not what this is about. Okay, temperature in this case is a very specific ideal. It is a way to communicate between cells. And so you can manipulate cells with ultrasound because you can get in there into the body and do all sorts of crazy things. But it's not as simple as, oh, it get hotter, you're going to kill the cancer. Not exactly true. So not only can ultrasound enable this precise control of temperature, recent discoveries have also revealed that cancer cells are especially sensitive to mechanical perturbation. So getting jostled by ultrasound, we already know that cancer cells don't appreciate getting jimmied like that. So you have to think, could ultrasound force Could ultrasound force cancer cells to change their behavior, priming them for destruction? This is an interesting thought. We know that on its own, ultrasound cannot deal with cancer on its own, but maybe in combination with other things, now we're talking. Is it integrin / cadherins? I'm gonna be real wacky. My brain is so gone. I'm on autopilot. I did the prep before and I'm just vibing with whatever's showing up on screen. I don't remember this paper. No. The white cat ear, someone paid me Cider paid me $100 to put them on, and no one has paid me to take them off, so the cat ears will stay. I am learning my slides in real time, exactly. This is live, baby. The very definition of live. So, yeah. Maybe ultrasound alone, not good enough, but with CAR T cells, maybe we're cooking there. It was myelin? Ah, gotcha. So, how does a cancer react to ultrasound? What is the mechanism by which ultrasound is converted into something that can potentially affect a cell? We get to talk about a very cool membrane channel, piezo-1. Here's a picture. Side view, top view, left to right. This is a membrane channel. Ah, I wish Clockwork were here. This thing's cool. This membrane channel can sense membrane tension, meaning it can be switched open with ultrasound. So it detects how tightly that membrane is pulled. Meaning a calcium signal can ping with ultrasound, because as soon as this channel opens, it lets in extracellular calcium. And what did I say earlier in stream? Calcium is a cell signal. It turns on a whole bunch of things in the cell. And so from a theoretical perspective, if you attack a cancer cell with an ultrasonic wave, you could potentially force this membrane open and get calcium to flood into the cell. And an engineer looks at that and goes, ooh, if we can turn the cancer signaling on, then we can take advantage of this to deal with cancer. And there's going to be something very interesting later on to show you how they took advantage of these. So there are many cancers that upregulate this kind of mechanosensitive receptor. My hypothesis is because cancer cells are doing a lot of moving, that maybe they need a lot of detectors for tension. But because they evolved to need these mechanosensitive receptors, now we have another target to attack them by. Cancer is smart, but we're also pretty smart. But to do that, I want to look at piezo-1 a bit more, because it is fancy Legos. This would make for a genuinely great Clockwork video. And I'm sad that he's not here with me now, because I'm not a talented enough structural biologist to explain it to you. But these are some pictures that I managed to grab. So, what are we looking at here? This is the membrane in blue, and then the protein is here. This is the channel, and it's closed in this configuration. But when the membrane is stretched, you can imagine this like curved shape. When it's stretched, it actually will shove open this channel and cause this plug to open up and out. You can kind of see that here. The cup down here, look at this cup. This is when membrane tension is low, right? It's a floppy membrane. It's slack, we'll say. And then you tighten that membrane. You know, you ever play with those like little cup toys? You flip them and they build all that tension and then they pop. I don't know what they're called, but kids love them. Kind of what's happening here, right? A floppy membrane is going to have a little bit of slack, and when the membrane is slack, the channel is closed. When the membrane is pulled taut, you flatten, it goes up and out just like those little toys. You flatten that membrane and the protein itself will conform to that shape, push this plug up and out, open the calcium channel. That's sick. I love that. That's structural biology, baby. That's a Clockwork video. But that's just a bit of trivia, right? A little bit of form equals function. Proteins have a certain form. You modify their Lego bits and they do something else. Very cool. So what is the mechanism? Hypothesized mechanism. Okay, so we have this piezo mechanosensitive receptor. We know that when the membrane tightens, it lets calcium in. So we know that if we blast it with an ultrasonic wave, the cell will tighten, and calcium will flood in. What can we do with that? What can we do with that? Seems unrelated to fighting cancer. Well, the clever scientists here have an angle. On its own, ultrasound won't do much to combat tumors, besides potentially opening up some cancer channels, right? It's not entirely clear how this is connected to anything. But what if we combined ultrasound with gene therapy to manipulate cancer cells into being more vulnerable to destruction? This is a Trojan horse technique. What if we deliver a gene therapy to the tumor directly and force the tumor to take up a kill switch? Right, a kill switch, a self-destruct button, and then activate that self-destruct button with ultrasound. And then only the cells that are targeted with the ultrasound will become sensitized. They will become weakened. And then you can add on your expensive CAR T cell therapy on top of that. Does this strategy make sense to you, chat? The wonderful thing about ultrasound is that it can force calcium into the cell. Alone, who cares? I don't care. But what if you genetically hooked up calcium to a cellular response of some kind? Maybe one that makes it weaker to CAR T cells. Now we're cooking. Now we're cooking. It's not the ultrasound, particularly. The ultrasound opens the membrane, brings in the calcium. That calcium is going to signal an artificial gene that you stuff into that cancer cell, and that artificial gene is going to make them weaker to CAR T cells. I hope that strategy makes sense. I hope anything I'm saying makes sense at this point. So, you cooking them cells? Yeah, actually, that's actually not a bad way to think about it. The ultrasound does kind of cook these cells in a little bit, as you'll see in future figures here. So, two treatments at the same time. We're going to Trojan horse something into the cancer, and then that's going to weaken them when it's activated by ultrasound, and the CAR T cells will take care of the rest. Is that adenine receptor thing you alluded to earlier? Not quite. I mean, CAR T cells are that, right? Those are T cells that you take, you genetically modify them to fight cancer. But this paper's innovation is hooking up ultrasound to a genetic manipulation in the cancer itself. Complicated. I'll explain it again a bit later. But I want to just point out that this isn't that crazy of an idea. Prostate cancer. This one's for the fellas. They are known to be mechanosensitive. If you jiggle and jaggle them, they will respond to this ultrasound. And white is an indication of calcium signal. You can stain these cells with the calcium dye. It makes them light up when there's calcium inside. On the left, you have no ultrasound, and on the right, you have ultrasound. You can tell. Cancers, at least prostate cancer, can be very reactive to ultrasound. But it's just calcium. Doesn't mean anything until we give the gene therapy. This doesn't matter. This just tells you the calcium response stuff, if you're curious, because this is electrophysiology. So this window describes when the ultrasound is turned on. You can see this is like the calcium light, the intensity, this color. And the second you turn the ultrasound on, you get a big spike in calcium, and then it drops off, a transient spike. That's characteristic of a calcium signal. I don't know how cell response index is measured. How do we know it's calcium? When you remove the calcium response genes, it decreases. Right, so it isn't some other thing that's making calcium. or some other thing that's making the signal, it's calcium. So that's how we know, because in this experiment we knocked the relevant genes out and the response was lower. Fair enough. So all of this data to just show you, okay, the calcium responsiveness of prostate cancer is real. And this kind of reaction to ultrasound also works in 3D culture too. So this is in cells, right? Cancer cells on a dish. This is 3D, like an organoid. And it's the same thing. So you turn that ultrasound on, you get a big cancer spike that drops off, a big calcium spike that drops off. You can see that in the color here, right? 0, calcium signal is low. You turn that ultrasound on, there's a pulse, it gets really high, and then it peters off. Calcium signaling. Interestingly, this ultrasound sensitivity was preserved across different cancers. So we already knew from before that prostate cancer was sensitive. But they also tested 2 interesting cancers, right? They tested last year's cancer, which is human glioblastoma. That's the last marathon stream I did. And also breast cancer, MDA-MB231, triple negative breast cancer. So all of these cancers are potentially vulnerable. Like I imagine like a cybersecurity firm, right? The battle between hacker and a hacker and cybersecurity expert. You're always patching up vulnerabilities that hackers can exploit every time you patch a system. And it looks like these cancers all have this exploit, this security vulnerability. They're all responsive to ultrasound. They're not weak to it. They don't die when you expose them to ultrasound. But it can force them to do something that they might not want to do, and that's to take in calcium. So when done correctly, ultrasound doesn't kill and doesn't heat up tissue, which is an issue with older applications of ultrasound. So you can deliver a super precise measured signal to a precise depth in the body. So say you have a tumor in the breast. You can calibrate the ultrasound to only affect a specific layer of the breast tissue. That's how precise you can make it. FUS, focused ultrasound. So what do we have here? We are showing that like ultrasound before, they were playing in the past with the idea of like cooking cells, as Surprise Pants had said, because ultrasound can mess with things physically. But when you use focused ultrasound, you can actually see that you don't get heat conduction. It's not a danger to the neighboring tissue. You're not going to burn anything. But you still get a calcium signal, right? So FUS focused ultrasound. PTX is an apoptotic activator. This is their proxy for cell death. So, cells, they live, right? They have a certain level of cell death that is normal. You hit them with that ultrasound, and they're still fine. They don't really do anything. When you activate apoptosis, then they start dying. So this shows that the use of this technology is relatively safe for healthy tissue. Same thing here. Temperature without ultrasound, it's pretty cool. Or rather, it's normal. This is delta temperature, the temperature change. With ultrasound, it stays the same. And then when you manually heat it up, well, duh. If you cook the cells, they heat up. That's essentially what this control is. So not that surprising, I think. So this makes ultrasound a particularly appealing signal because it's not apparently harmful to healthy normal tissue. So let's start messing with ultrasound. So it's a bit of a neat parlor trick, but what can we do with this? This is the system. And I want to dwell on this for a little bit. Ultrasound, they're using to attack the cancer cell, but we know that ultrasound alone doesn't do anything, doesn't kill the cell, but it forces them to take up calcium. And the power of this design is that they're going to genetically hook this up to some kind of payload, right? I'm using cybersecurity terms, like a virus has a payload. I guess not just computer viruses, but I've been reading cybersecurity stuff, so that's what's in my head. Like when you get malware installed, right? There's a payload, something that it does. So what is it going to do? It's not. This is pretty intense. Okay. I don't think I have the brain for it right now, but I'm gonna try. I'm gonna try my best. Okay. The researchers are trying to sneak in a Trojan horse into these tumor cells. Okay, because ultrasound is only 1/3 of this idea. The ultrasound opens the gate for the signal. It is the trigger. But what is actually being triggered? They're putting in a payload that looks a little something like this. This is going to activate NFAT, it's calcium signal, and it's going to come into the nucleus. That's the natural effect of calcium. It's going to suck this transcription factor into the nucleus. It's going to bind a promoter and make a payload. The genetic engineering aspect of this is that this segment of DNA is the thing that they are trying to sneak into the tumor cells. So they're going to give this payload a promoter that matches NFAT. What is that actual payload? Don't worry about this. I don't want to talk about it anyway. The payload is going to be, should I talk about it? No, I don't want to talk about it. I don't want to talk about it. I'm too tired. I don't want to talk about it. Thank you so much, Anonymous, for 1639. Thank you for donating. Forgot to fill out my question for the reward. Go ahead and you can go ahead. I trust you. Go ahead and ask your question. Go ahead and ask your question. Are you playing Doom on cancer cells? Kind of. They're Doom because they're gonna die. Because humans are clever. So yeah, they're trying to rig in a genetic payload, but they have to do some clever tricks with genetic engineering, including delivering an AND gate. Okay? This is for you engineering nerds. You electrical engineering nerds. An AND gate. This system that they genetically engineered, when there is no calcium around, the system doesn't work. Nothing is made. When calcium is around, the genetically engineered construct comes in and hangs out in the nucleus, but it doesn't bind. When doxycycline, another trigger, comes in, it helps this thing that you genetically engineered attach to your gene of interest and actually make your thing. So there are two triggers, ultrasound and doxycycline. You need very careful control if you're going to assassinate this cancer. Okay, you want a very quick activation that is not messy at all. And to do that, you need an AND gate that just gives you a little more control over the Trojan horse that we are snaking in here. Okay, but we talked forever about the AND gate, the engineering. What is the actual payload? Like, why are we doing this? Okay, why are we doing this? What is the gene of interest? Let's not talk about this. Okay, this is okay. Well, we should. This is how the AND gate works because I'm sure engineers find this fascinating. The gene of interest that we're testing this system with is mCherry. mCherry is a simple red fluorescent protein. So this is the strategy. We genetically engineer the cancer cells to make the payload stuff. The payload is only activated when calcium and doxycycline are both present. This gives us extremely precise control. But before we get into the killing part of this, we have to talk about whether or not this circuit works. So instead of a gene that does anything interesting, we have mCherry, which just glows red. So don't worry about the payload for now. Let's just check if the circuit works. This AND gate, doxycycline plus calcium. So dox stands for doxycycline. ATP, they are using as a proxy for calcium because when you dump ATP in, that triggers calcium intake. So this is their way of shoving the cell manually full of calcium. So when there's no treatment, the I see this as, I don't remember. This is just regular light, put it that way. Contrast microscopy, it just shows you the shape of the cells. And then mCherry down here, the red signal. So, chat. can you tell me, and please lock in chat. I need a little break so I can drink my water. But looking at this data, does the AND gate work? What's the AND gate work? Does the AND gate work? Surprise Pants says, what is that sussy red doing in the docs only part? That's interesting. Chat, what do you think? What do you think? Look with your own eyes. How well does this AND gate work? Is this a perfect AND gate like you have in computer science? What does it look like to you? Yeah, kind of works, but not completely. Docs activates. Yeah, I'd say so too. Surprise Pants, you're right on the money. Doxycycline, it looks like the system is leaky. Guess what? It's biology. Biology is leaky. So, that's just reality, right? But your eyes are not lying to you. That is sussy. Ideally, that red wouldn't be there if the AND gate were working properly. But in biology, sometimes you just gotta go, good enough. Good enough. And then doxycycline plus ATP, the signal is a lot brighter. So AND is working asterisk. Working all right. Not perfect, but working all right. So the red output in the AND condition means that the circuit works. There's a bit of doxy here, and you're right to point that out. It means it's leaky. The AND gate is leaky. So we forced calcium uptake with ATP. That's the generally accepted way to do it, supposedly. But we care about ultrasound. Can we trigger this AND gate with ultrasound? So same deal. Payload is just M-cherry. They're just making sure the circuit works. And M-cherry, they false colored, I guess this is what's called as a false color. The machine thinks this is white, and then the machine will color it red for our human eyes. This, they didn't bother to false color for some reason. Does this mean it will activate in healthy cells? Only the cells that take up this genetic payload, right? The whole idea is we're going to sneak this into a cancer cell. And the way that we'll do that will come later. But you're right to think about what implications this might have on healthy tissue. That's a good question. I'll say they have an answer for you, Alec. They have an answer for you. So I'm just going to go through this part. It's the same experiment, except this time we're using ultrasound and not ATP. And it looks like, yeah, doxycycline alone, we get the same result. It's a little leaky. You see some M-cherry fluorescence here. But doxycycline plus ultrasound, it's a lot more. So it looks a lot like this, just in a slightly different system. This looks like an organoid. So this is 2D culture, flat surface. Cancer grows on a flat surface. This is 3D culture where it forms balls, but the light pattern is the same, right? Doxycycline is a little leaky here. Doxycycline plus ATP is working. Here it's the same thing. Doxycycline, it's a bit leaky. Doxycycline plus ultrasound is a lot better. So it works. But only in a dish. What about in an actual mouse tumor? So ultrasound alone seems to activate M-cherry expression, right? So this is a heat map. The redder you are, the brighter that activation is. So in a real mouse, the injected tumor, ultrasound, no ultrasound, you get some basal level of calcium, right? Because cells are doing calcium stuff all the time. There's never going to be zero calcium. 6 hours, 12 hours, and 24 hours of ultrasound. You can see, yeah, ultrasound is forcing calcium activity in this region of the mouse. And it's a lot bigger when you add in that doxycycline. Very cool. So the signal works, right? We can get the cells of the tumor to actually take in calcium. Actually, I don't know if they address your question, Alec. I think I maybe removed it from the presentation because I was like, I'm so tired. They have a method that is able to sneak in this Trojan horse preferentially to cancer cells, and I don't remember what they do there, but healthy versus non-healthy. We also saw earlier in the presentation that just ultrasound does nothing to healthy cells. Okay, so finally, we know that the AND gate works, kind of. and it works in mice. How are we going to get this to actually make a treatment? What is the payload? The payload isn't anything toxic on its own. It's not poison. It's a membrane protein called CD19. And why might that be? This is half the treatment. This is half the story. This payload that we have so carefully designed a genetic circuit to express is a humble membrane protein, but it is a target that T cells can use to attack the cancer. Here's the thing. I've mentioned it before. Cancer, incredibly clever. Okay? It knows that T cells are hunting it. It doesn't know. Okay, yeah. Things in biology, they don't know anything. Okay, yeah. Figuratively, they know they're being hunted by T cells. So they pull their T cell receptor antigens outside. They pull them into the cell surface so the T cells can't kill them. They cloak themselves by removing their weak points. The whole reason for this genetic circuit is to force the cancer to hold a big red sign that says T cells. Oh, T cells, come over here. Wolf whistle, whatever you want to do. And then the CAR T cells that have been genetically engineered will be targeted towards that CD19. It is as if you force the cancer cells to hold a red towel and you put the bull into the arena. It's like you stapled the red tower to the matador's hands, right? You just unleashed the T cell, the genetically engineered T cell who was bred, evolved, engineered to kill anything with CD19 on it. You just made the cancer cell make that CD19. That's the theory here. Yeah, kick me sign. That's true. You forced all that stuff. It doesn't matter if you understood the circuit. That's mostly for engineering nerds. But the thing I want you to appreciate is this is a treatment that involves gene therapy of the cancer cell to force it to make a big kick me sign so that when you put in your genetically engineered T cells, they will kick that cancer. It's awesome. What a cool approach. Why is this so complicated, dude? Like 3-4 papers ago, it was just take aspirin. Why are we doing this? This is so complicated. So step 1, gene delivery. Okay, inject the whatever it is, adenovirus, whatever type infection. I don't remember what they use here. But do the whole thing of gene therapy and trick the cancer into making your genetic payload. Activate that payload with ultrasound, and then inject your own genetically engineered CAR T cells back into yourself, and then hopefully the CAR T cells will learn. So why go with this crazy complicated design? This is a kill shot. Okay. So far, a lot of these therapies are about delaying the inevitable, right? A lot of these treatments have been, hopefully you die of old age before you die of cancer, and that's fine. This design is out for blood. This design wants cancer dead. Because here's what the intent behind this design is. For most treatments, you're never going to get rid of all the cancer, okay? Cancer is really tough to get rid of. This is different. This is an attempt to train CAR T cells to kill cancer cells by forcing them to make this kick me sign. And CAR T cells, as they gain experience, as they fight, just like a video game, they're going to learn to fight this cancer. And the hope here, the reason why this is a kill shot is that, CAR T cell stuff has been done before, but they're not perfect. Cancers can come back. This is hoping that this concentrated school of CAR T cell, of cancer kick me signs is like that's not the word. It's like a boot camp, right? You got all these cancer cells with kick me signs all in one place, You inject the CAR T cells, and they train on them. They go, oh, CAR T, CD19 tumor cell, pleased to meet you, die. And what's fun about this approach is that even if your cancer didn't get the gene therapy, there's a chance that these really well-trained CAR T cells will kill them anyway because they're that thirsty for blood. This is personal, exactly surprise pants. Like, this treatment feels like deliberate revenge. This is an angry method of drug design. Because you can see in figure 3, killing beyond, going beyond. Not just affecting the gene therapy cells, but the cancer cells, any of the nearby cancer cells. Like, this is personal. This CAR T cell is coming for the cancer's family, not just the cancer itself. Do you understand why that's a kill shot? Plenty of cancers reoccur. But the hope here is that these CAR T cells will learn enough in this boot camp to go for it, to go for the kill shot, to make sure that every last cancer cell is destroyed. This is a cure. It's a possible cure. Like, I don't know if it'll work or not. I'm not a cancer biologist. I don't know how likely it is that this translates into the clinic, but I can appreciate what they were going for. Wouldn't the cancer family be the healthy cells? It could be the ones that are less cancer-like but still threatening to become cancer, and hopefully this training doesn't result in the massive death of regular cells, but that's always a possibility. That's why I'm like, this is a really cool approach. I don't know if it works. I don't have an opinion on that. I hope it does, because it's cool. What did the CAR T cells scream when they kill them? That's up for you to decide, man. Thank you so much for the donation, Duncan Murphy. What's your favorite way we found a novel biological agent or mechanism? Oh, good question. God, that's an interesting one. Nobel Prize for decoding the protein code. If you go back to watch my segment with Clockwork like 6 hours ago, at the beginning of the stream, we put collectively in triple S tier the Nobel Peace Prize of, I think, 1968, where a bunch of geniuses decoded the connection between the DNA code and protein. Beautiful paper. The most beautiful piece of literature I've ever read in my entire life. So the mechanism in that sense is translation, how tRNAs work. So beautiful. Can't explain it, but I was left with a feeling of awe and like a genuine emotion. I likened it to when mathematicians look at the Euler equation, Euler's identity. E and pi and I and all these constants of the universe all lining up in a convenient way is interesting. There's a symmetry, a beauty to it. I feel that same way about the tRNA Nobel Prize. Sick. Okay, so this is a kill shot. This is incredibly ambitious for a cancer treatment. But it is complicated. We have to acknowledge this, right? I've been talking on my high horse. Accessibility. I really value it. This is not accessible. Dude, half the treatment is CAR T cells, which already comes with a huge price tag, and the other half is genetic therapy, gene therapy. That's not cheap either, man. But I think of this more like a fashion runway, right? The stuff that you see in a fashion show, no one's going to wear that on the street. That's not practical. But it can show designers what's possible in the design space of fashion. So the way I look at this is not like, oh, that's going to happen to me one day, because if it does, you're a millionaire. You can afford it. But it is a proof of principle. Can this idea work? And if so, can we make it cheaper? But first we have to see if it actually does work. So we have our mice here. Mice injected with breast cancer cells. Then you give them the gene therapy on day 5. That's what day 5 is, lenti injection. And then you give them the CAR T cell therapy on day 10. And then on days 12, 14, and 16, you hit them with doxycycline and ultrasound. See how this is so complicated. This is not an elegant design at all. But it is cool. Thank you so much for hanging out, Duncan. Thank you. And chat, take a look at this data. Give me a little bit of time to hydrate. Do you think this crazy strategy worked? Controls the tumor-injected mice with all the genetic engineering, but none of the activation. FUS is ultrasound. Dox is doxycycline, the second AND gate. Dox plus ultrasound, well, you know what that means. Y-axis is normalized tumor growth, normalized to control. What happens when the entire system is assembled? Is this crazy strategy something that might be working? How good is this? How good is this, chat? The numbers don't lie. Money is great. Money is great. Let me tell you what. Yeah. I look at that and I see that the scientists behind this are probably pretty happy. Now, I said this was a kill shot. Is this a kill shot? No. Day 17, day 21, you still get a tumor. It's tiny though. And that's heartening. Do you see why at the beginning of the stream I said we are going to cure this thing? It's going to happen. Maybe in our lifetime. And I think it's a good time to buy in to become a part of the machine that kills this thing. Because you can't help but be inspired by that figure, man. This is complicated and it's not perfect. Again, they still had tumors. Like, I thought this thing was supposed to be a kill shot. But I'll take really, really good too. Maybe not perfect, but I'll take really good as well. That's inspiring. That's progress. It's not in humans. Let's not get too ahead of ourselves, but it could be one day. And you heard it here first on the Find the Neutrophil channel. Oh, actually, we've actually gotten to the point where let me go ahead and do what I'm supposed to be doing here and raising the I can't even think anymore. Sorry, give me a second. I realized that the stretch goal is 5,000. So we hit our goal of 2,000. I'm very happy. The stretch goal is technically 5,000. Give me a second to update that. I don't know how to update it. I don't know how to update it. What the heck? This was so easy yesterday. Why can't I update this? Raising the timer. Okay. Incentives, milestones. Okay, now we can activate the $5,000 stretch goal. I'm already very happy with 2K, but why not see how far we can push this thing, huh? It'd be a shame not to have our sights set up higher. Okay, back into the science. Where were we? Where were we before I got greedy with the donations? I gotta bring up everything. Yeah, I mean, it's wonderful, right? The Mad Lads did it. This is huge, a huge difference in tumor growth. So, to some, CAR T cells, genetically engineered T cells that hunt for antigens designed by humans, often ones that are commonly found on cancers. But CAR T cells don't usually finish the job. More often times, cancers can escape their wrath by pulling the antigen off the surface, or sometimes T cells can just miss. To solve this issue, the authors invented a gene therapy that is activated by the combination of ultrasound and antibiotics to force tumors to make a big old kick me sign. The CAR T cells are charged up to the max, learning and fighting against these tumors, making this antigen. So again, this is hoping that the gene therapy might not have to work on every cancer cell, but it works enough of them. Hopefully, the CAR T cells will learn to fight them. And if everything goes well, this could result in the complete deletion of a tumor, mechanistically speaking. I think that would be pretty incredible. Didn't see it this time, but for a proof of principle, it's pretty rad. In other words, the CAR T cells would eat no crumbs. I'm really proud of that one. CAR T cells would eat no crumbs. We don't want it to leave no crumbs. Crumbs are bad. I love this paper. I think it's really cool. Any questions? We have a bit of time before heading into the next one. Any questions on any of this? CAR T cells cartoonishly licking their plate. Yeah. They are. Great end comment. Thank you. I'm proud of that one. I am. OMG, CAR T cell slay. So true, bestie. So true. I didn't want to do so many CAR T cell papers, but I figured I would do at least one because they are very important. What a hopeful paper it is. Right, we started this stream, I think on paper 4 or something, when I started crying because I was like, oh, it's like I feel like I got away free because I'm no longer doing bench science, and when frustrating results are happening, I feel like that it should have been me. But this one is pretty hopeful. They had a crazy idea and it works in a mouse. I hope it works in a human. What about the ultrasound makes the CAR T cell therapy so much more effective? It is because they combined the ultrasound with gene therapy. They snuck a genetic payload into the code of the cancer cells. The ultrasound activated that payload and caused them to make a big kick me sign. That's why. It's not just ultrasound. Ultrasound itself doesn't do much. Ultrasound in combination with clever genetic engineering, then we're talking. Fi, I don't think anyone has asked you in a while, but how are you doing? I'm about as well as you can expect. 3 more hours. But let me tell you, in an abstract sense, I'm happy. I'm pretty happy. I'm happy that despite being 6 a.m. where I am, there's still 45 of you here. That's more than a classroom. Some of you have been here for a long time. And that's impressive. Because I don't consider myself to be a very entertaining streamer. I'm not particularly funny or high energy, especially not 21 hours into a stream. But I am very heartened by the fact that there are people who are sticking with me. It means a lot. And I am optimistic for humanity because people like you exist. It's nice. It's interesting that the AND gate wasn't complete and also not symmetric. Yeah, that's true. I mean, you can design all sorts of things on paper. In biology, you just kind of have to accept that biology is weird Legos that bump into one another, and that can sometimes be leaky. So this is just a concept. There wouldn't be a way for an ultra-rich person to ask scientists to treat their cancer this way right now. Oh, I'm sure you can have someone do this through illegal channels. Would it be smart? No. This is a mouse thing right now. But, I mean, if we're talking like Jeff Bezos, I'm sure Jeff Bezos can pay somebody to try this. That's why accessibility is so important to me. Because you can bet your rear behind that any of the really rich guys we know of ever get cancer, they're probably getting CAR T cell stuff. They can afford it. This stuff is highly experimental, though, in this paper. I've had a good time so far. I'm glad. I'm having a good time, too. I am physically not feeling very well. I might not be able to talk Sunday or Monday, but that's okay. After the stream, I'm writing. My next video is going to be about the birth of new genes, kind of an offshoot of the E. coli video I did a while back. So I'm not going to be needing my voice for recording anyway. But I can tell my vocal cords are stressed. This topic is so fascinating. It's great. Yeah, this is entertaining. I'm glad. I'm glad that there's an audience for this. It might not be Mr. Beast levels, but the fact that I have a community that appreciates this science is meaningful. And I enjoy it. I just made it to your latest video and saw the thing about the live stream. I'm obsessed. You've made it just in time. I'm in the last 3 hours of a 24-hour marathon. And I'm so tired. I'm so tired. Despite being in high school and not understanding a lot of English terminology, it is really fun. Thank you for saying that. I think one of the most valuable parts of this community is the international aspect of it. Like, when I look at my analytics, I'm like, I am amazed at how many of you come from countries where English is not the first language spoken, because English is a very difficult language to begin with, and then having to learn science in English. Ugh. Respect. Respect you all. A while ago, I binge watched your videos, and then I watched that stream announcement, thought it already passed, and wait, is this a new video? And I kept it in mind, and here I am. Well, I'm glad you found your way to me. Sorry that you couldn't see me when I was more alive. But yeah, I am very happy, and we're still getting through this, you know? I timed these out pretty well. That was just about an hour worth of content. But it's time to go to the next one. We are heading into the 22nd hour of streaming. I cannot wait for apoptosis. Yeah, I have some concerns. Gene therapies can be expensive. Certain gene therapies can be more than $1 million per dose. It's not cheap. CAR T cell therapies are a couple thousand, 100,000 USD per treatment course. And there's just so many moving pieces. This is not an elegant treatment. I think this is very cool. I don't know how well this translates to something that regular humans can ever have outside of a research setting. I think of it like a fashion runway, right? You look at those fashion models going down the steps, they're wearing meat dresses and whatever. Corpse of Big Bird. And that's not meant to be worn on the street. That's not real fashion. It is art for the designer. And I think this is art for the designer. I don't see this being a real treatment one day. $1 million per dose, man. For you and me, unless that YouTube buddy comes in and I'm Mr. Beast, I'm not getting this. Can't afford it. So that's why I love that a lot of the BCRF research has come in the form of treatment plans that are more accessible to the average human, which I really value. Oh my god, this paper. Oh, this is a banger too. Why did I save so many of these awesome papers for the very end where I'm too tired to actually talk about them? Why did I do this to myself? Okay. I need to eat a little more of this protein bar and drink some water. These titles have words I've never seen before. This was pretty simple. Yeah, I don't discount that you might not have seen them, but these are pretty easy ideas to understand behind this title. Protein bar. Hyper Kenji. Yeah, this was published recently. I wanted to show you the cutting edge of research. Yeah, Grandpa Fi in the background is adorable. That's the Soy Jack meme. That's Soy L or Soy R. I don't know which one that is. The Soy Jack meme. I don't know if that makes it less cute for you. Yeah, that is Soy Jack 5. That's the pogging and the air, the arm pointing. So good. I need to hydrate, my god. This paper's insane. What is it about? Okay. Enhancing the in vitro anti-cancer efficacy of Pax. I don't know how to pronounce this, dude. Paclitaxel against triple negative breast cancer via an exopolysaccharide from a haloalkophilic archaeal string. Let me break this down for you. We have a drug. It kind of works against triple negative breast cancer, the worst kind of breast cancer. How do we make it better? Why don't we take a sugar from archaea and see what that does? Archaea being the third domain of life, or the second, depending on who you ask. You know those little buggers that grow in hot springs that don't want anything to do with the human body because they're chilling in hot springs and salt lakes and stuff like that? Well, what if we used one of their sugars and saw whether or not it could weaken cancer to the point where the drug that we do have works even better? Wild. This is a wild idea. How do people even think of this stuff? It's crazy. So, what do we got going on here? What are archaea or archaea? Archaea, they are a domain of life, either parallel to eukarya or encompassing eukarya, depending on whom you ask. And they are typically known for minding their own goddamn business, hanging out in saltwater lakes, hot springs, whatever. The vast majority of archaea appear not to interact with human biology at all, although more recent research is revealing that archaea might play a role in certain dental diseases, and I made a video about them. The reason why the archaea for that video is square is because, you know, square bacteria don't really exist. The square shape is kind of unique to this branch of life. In the bottom right, we have Haloquadrum, an actual square-shaped microbe. Isn't that fun? Thank you so much for the donation, Anonymous. Meso tardigrada. Yeah, tardigrades. We love extremophiles on this channel. Oh boy, my throat. Really feeling it. So, we don't really think about archaea when it comes to medicine. And why would we? These guys are just chilling in hot tubs, dude. They don't want anything to do with human bodies. That's why it's a bit crazy that we can make medicine out of them, potentially. I thought the square cell was a bit of a meme. No, it's a real reference. Holoquadrum. Salt archaea. Actually square. Yeah, so we even think of this stuff, man. This is crazy. Apparently, researchers have been experimenting with these exopolysaccharides. So these, to break that long word down for you, it's basically sugars that the archaea spit out exo for out, polysaccharides for sugar. And apparently people have been really interested in these due to their interesting physical and biochemical properties. And this kind of makes sense. These guys live a wackadoodle life. An archaea that has to survive being boiled, you best believe that they're going to be making stuff that is weird and wacky. Their sugars, their exopolysaccharides, they have a lot of interesting biochemical effects. Like they have interesting gelling properties, which makes sense because to survive boiling, you probably have to do quite a fair bit to not die. Have a nice night. Thank you. It's morning here. So while material scientists have been interested in exopolysaccharides from archaea, what the heck does cancer biology have to do with any of this? Research from the last decade has shown that maybe this could be a thing, and we'll see why that might be. So here's one of these polysaccharides. Is this Taxol? I don't remember what this molecule is, so sorry, I'm so tired. But here's the research question. Could an archaeal, a sugar from this branch of life that we don't even think about anymore, could these sugars be actually a useful part of an anti-cancer therapy regime? That's how I would draw a bike. Yeah, true. So we're particularly interested at sensitizing tumors to existing drugs, like this chemotherapy drug. Oh, the short name is Taxol. Great, I know Taxol. And again, to remind everybody, chemotherapy sucks. You know, losing your hair, feeling weak, nauseous, can't keep down your food, no appetite. Chemotherapy is poison. That's not to say that you shouldn't take chemotherapy, but chemotherapy is literally poison that poisons the cancer worse than it poisons you, but it is poison. Unfortunately, for triple negative breast cancer, you don't really have another option. I'm sorry. Sucks. But if your cancer does not respond to hormones, you might have to take chemotherapy instead. And just like the CAR T cell thing with money, if you are going to have to suffer through chemotherapy, you better make sure it works. You better make sure it works. Sadly experienced chemo firsthand. I'm so sorry. Happy it exists. Yeah, that's what I'm saying. I don't mean to scare anybody when chemotherapy comes up. Life isn't fair. But it can save your life. But it is going to make you remember how it did it. It's bad. But it could give you more years. That's valuable. Again, chemotherapy is poison. A lot of them target mitosis. Cancers typically proliferate more aggressively than normal cells. So if you target mitosis, and cancer cells love doing mitosis, we're just kind of hoping that the cancer dies before you do. It's a lot to ask from your body. So an important improvement we could make to the lives of triple negative breast cancer patients is asking this question. What if there was another compound you could take that let you get away with less chemotherapy? It's not a replacement. Chemotherapy is good. But what if we could get away with less chemotherapy? Sure, you would be happy, right? Sure, Trenton would be happy if you didn't have to take the same dose of that poison. That's what a lot of people are trying to figure out here. So that's the goal. This wacko molecule from this wacko microbe, could it lead to happier patients? We'll see. I find this so fascinating. You know, when we work with bacteria as a molecular biologist, you really oftentimes just buy it from a freezer somewhere. Archaea are pretty rare, and you kind of have to go fishing. And this is so cool. They had to go to the salterns, which are these coastal regions that salt accumulates on the northwestern coast of Egypt, and actually do wet biology, field biology. They had to grab the media, you know, the water from the salterns, and try to grow them, which I think is fascinating. So this is a little bit behind the scenes. A lot of bacteria are very happy to eat whatever. They're not picky. E. coli famously not picky. The most used model organism on planet Earth. Not picky. You give E. coli a little bit of salt, a little bit of sugar, a little bit of protein. E. coli is happy. 4 degrees, 42 degrees, doesn't matter for E. coli. E. coli is chilling, dude. Archaea are whiny, temperamental little things. And if you are isolating an archaea from a salt mine, then you would have to know, you should expect that they're going to be a little particular how they want to live. And you can imagine that salty archaea, not the only things that grow in salter, and so they had to isolate, try and purify archaea, which is hard to do. I had childhood non-Hodgkin's lymphoma, and one of the chemotherapies I was given was named cytozan. It was created after research on mustard gas after World War II. This was in 1994. Oh. Well, I'm glad you're still here with us. Seems like it worked. I wonder how your experience with chemotherapy happened, though. Because, you don't have to share. It's painful. Thank you so much for joining membership, Audrey. Thank you so much. But yeah, they're fishing for archaea. It's so funny to me because the bacteria, you just kind of you chuck biomolecules at them. They're happy. They're chill. How do you purify archaea? You kind of can't. Being able to purify archaea means you know what they want to grow, and that's still very not clear to scientists today. But they can do the best they can. So they took these samples from Egypt, and these salterns, and they grew them in a special archaea-friendly media called Horikoshi media, and a lot of salt, NaCl, table salt, to ensure that only microbes that love table salt could survive. So even though you might not be able to get one species, you could do a pretty good job eliminating a lot just by dumping a bunch of salt in this medium. And then this medium was optimized for halophilic archaea with 4 molar NaCl, which is crazy. It's a lot of salt. I need to point out how crazy this medium is. All right, to grow these archaea, it's 4 molar. For those of you who haven't taken chemistry in a while, I will put that into context for you. 5 to 10 millimolar. That's the salinity of E. coli cytosol, the insides of E. coli, which is a similar molarity from mammalian cytosol. The broth that E. coli grows in, about 100 millimolar sodium chloride. Human blood, 100 millimolar. And when you run the numbers, archaea grow at 400 times the salt concentration than our blood. 400 times as concentrated. That's crazy. Are they considered extremophiles? Yes. The tardigrade, not an extremophile. Extremotolerant. The tardigrade does not enjoy being frozen. It does not enjoy being burned. It does not enjoy being thrown into the vacuum of space. The tardigrade wants to be on a leaf and chill. That's why they're extremotolerant. These archaea are true extremophiles. They can't live without their salt. Need a lot of it. So, they did a bunch of fancy molecular biology things that I'm not going to talk about. That eventually they isolate a particular fraction, a sample, with a high level of biological activity. This fraction, this sample they collected, they lucked out and it made a ton of exopolysaccharide. Using a fancy technique called 16S RNA sequencing, they found that in this highly productive fraction, where this bar is, using this sequencing technology, they can find that the majority of this fraction is made up of a single species, Natrialba chahanoensis, a halophile. Okay, we've got our archaea. We've got our sugar producing guy over here. Long story short of my experience with chemo, when I was 5, oh, I'm so sorry, you were so young. When receiving it, since they did not have much other options, I vividly remember the sensation, like your own body was dying. Yeah. And you were 5. And this is why, folks, the Breast Cancer Research Foundation would like to move beyond chemotherapy, if possible. Happy you're still here with us. It clearly worked, but bad, you know? A 5-year-old kid feeling like he is dying. Not the best medicine. So finding a way for that 5-year-old kid to maybe take less of that medicine that makes you feel like you're dying, it's an important mission. Thank you for the super chat, Otter Lee. Hey Fi, keep doing such a good job. I work in pharmacy as a healthcare assistant, and the research of you raising money will help so many people. Thank you for the encouragement. Thank you so much. And thank you for hanging out with me, Otter Lee. So we've got our system running. We have our archaea. It's going to be making the fancy sugars we want them to make. Very cool. There's a lot of cultural optimization here that I don't want to go over. It's mostly pretty boring. But they're basically tinkering with the recipe. You know, they're tinkering with the broth, the ingredients to the soup, as it were. And yeah, I'm not going to go over this. I can't even use my brain to interpret this. I think, oh, I can. Right, they were testing different sugars, arabinose, fructose, you know, carbon sources, and, you know, nitrogen sources, as you can see on the right here. They wanted to get the tastiest food. Make this archaea the happiest. Get them to really make a ton of this sugar. Oh god, I can't, dude. There's a lot of chemistry in the paper too. I just ignored. This is FTIR, FLARE, transform, infrared spectroscopy. This basically tells you roughly what the chemical looks like. GCMS, similar. These are both complementary techniques that tell you about the chemistry of the thing that you're actually making, this exopolysaccharide. Because notice, I've just been calling it exopolysaccharide. That's because it is a very vague term. They're just spitting out slop, and it's not all one thing. And look at this graph, true. But you can characterize it to see kind of what the slop looks like, to get a better idea of what it is these cells are actually making. So they take their collection of archaea sugars and they throw them on top of cancer cells. So I've done you a favor and I've labeled these. So IC50, you know, shout out to Arlie, working in a pharmacy. Probably encountered this measure before, the IC50. This is the inhibitory concentration, aka how much drug do you need to halt 50% of the growth? Gives you a rough measurement of how effective your drug is. And we have 2 different measurements in this crosshatch pattern. This is the IC50. So how much do you need to stop this cancer from growing? And the SI is the selectivity index. Don't worry about what this is, just if it's above 1, relatively generally considered safe. So chat, PBMCs, that's just regular blood. Look how much drug you need to kill regular blood. And MDA, MB231, we've seen this line a lot. Compare these two bars, regular blood, breast cancer bar. What can you say about how much drug you need to inhibit the growth of regular blood versus breast cancer, triple negative breast cancer? What do you say, chat? What does it look like? As I eat my protein bar. Come on, chat. You can do this one. I believe in you. How much drug do you need to stop regular blood from growing? How much drug do you need to stop breast cancer from growing? You can do this. You got this. All right. Let me do the math. Yeah. Ponder it for a bit. This is exciting stuff. Yeah. With some number crunchers in chat. You need a third of the drug. It's pretty good. I'm pretty happy with that. It's kind of amazing to me. It does appear that for whatever reason, these cancers are sensitive to the exopolysaccharides. That's pretty interesting to me. Let's continue. The thing is that these numbers mean very little to me. I'm not a drug designer, but they seem kind of high. Milligram per mil. It seems like you need a lot of this polysaccharide, but I don't know because I don't know the molecular weight. And without knowing the molecular weight, I can't work out a molarity. But even so, even though there's this relationship where you need a lot less to inhibit the growth of cancer, objectively, it's a lot of mass. To me, it feels like a lot. That's my personal vibe. You sound tired. Ooh, it's a good reason for that. We're so close. Does halt 50% mean stop multiplication of 50% of cells or make the total difference born dead to 50% of the original value? It's a good question. And it's to halt 50% of maximum growth. So imagine you grew them and you were chilling, you didn't add any drug. You call that 100%. Now you add the drug. And then the amount of drug that you needed to get that growth to be halved, that's the IC50 in this case. This is maybe a minor point here. Oh my god. Does the EPS arrest the cell cycle? So it's a fancy graph. Don't worry about it too much. Just M1, M2, M3. It's different stages of mitosis. M1, M2, and M3. Early, mid, and late mitosis. So like, okay, the polysaccharide, it's doing something on its own. What is it doing? Maybe it's doing something fancy with the cell cycle. But have a look at these charts and take a look at how wide these lines are. The progression through the cell cycle. kind of seems the same to me. So does the exopolysaccharide from the archaea actually arrest the cell cycle? I don't really know. M1, M2, M3, they kind of look the same. I'm not a flow expert though. And even if there are differences, the researchers admit that they're not statistically significant. So the polysaccharide's doing something. You might need a lot of it to do something, but it is doing something. Just not entirely clear what it's doing. Is the EPS itself the one's killing cell, or is the EPS plus chemotherapy? I believe this particular graph is just EPS, just the polysaccharide, I believe. I could be wrong, but I'm pretty sure it's just the polysaccharide. Because I think, is it? Yeah. That one was just polysaccharide. Now, you know, by eye, just like looking at the IC50 for the EPS, it's not that exciting. Doesn't really have a strong effect on the cell cycle. But, you know, I think originally, if I were to put myself in their shoes, they were maybe hoping that this polysaccharide was going to do big numbers, but then when they tried it, it kind of sucked, and it does. Like, this difference is meaningful. It is selective. That's true, but as Trenton said, this is a lot. Like, I didn't know until, Trenton, you did the math. That's a lot, man. It felt like a lot from lab experience, but when you do the math, yeah. The polysaccharide itself, not a great drug. So I think instead of giving up, they thought, okay, we had a good idea. We thought. Because in other papers, you've seen that this sugar does, or you know, archaeal sugars could cause problems for cancer, but it just wasn't that impressive this time around. So I think the idea for combination therapy came out because this result was a little disappointing. So Drunken, you're kind of right. Like, is it the combination there? It's not. I think they're saying honestly, like, hey, we thought this polysaccharide was going to do crazy things, and it didn't. But what if, you know, this is trying to salvage a little bummer moment. So I assume, and they could be wrong, but I assume this is why they, instead of looking at just the polysaccharide, went, what if we like combined it with something and made it better? And I admire that. That's picking yourself up off the floor after you've been knocked down by cancer. And I'm glad they did, because the next stuff is pretty interesting. We don't need to talk about how bad chemotherapy sucks, you get it. So, there's this model in drug development. This is something that Audrey might also be aware of. The Shu-Talele analysis for drug synergism. I have never seen this before. It's my first time. And I might be saying Shu-Talele completely wrong too. But this is a special numerical test that can enable you to detect synergy. So we've been saying a couple of times, sometimes the whole is more than the sum of its parts. This is a mathematical manipulation that you can do to find out whether or not that is true. I don't really know how it works, but I assume that it does. And on the y-axis, we have fraction of cell population affected. And on the x-axis, we have dose. So you should expect that the higher the dose, the higher fraction of the cell population is affected in terms of cancer. So EPS, that is the polysaccharide alone. PXL is the existing chemotherapy. And then we have the EPS PXL. This is interesting. It's a little tricky to read this if you have zero chemistry background. So Hairday, EPS looks ineffective. Why do you say that? I agree with you, but why? What are you looking at to make that determination here? It's a good insight. Because, you know, to me, to my eye, I'm looking at this as a naive observer, and I'm noticing, hey, look, the blue line goes up. EPS is the best. Right? Wow. 100% of the population affected. Looks pretty good. Why does that mean that EPS looks ineffective? Which is correct. Why am I saying that EPS, even though the curve goes all the way up to 1, doesn't look too good of a treatment? I'll say this. Oh, that's also a bummer. I don't think they did a point at the end. How are they drawing this curve? It's kind of crazy. But yeah, chat, you're getting it. You need a high dose for the effect. The curve is all the way to the right. More effective treatments will be curved to the left. Because when the curve is right shifted, right, let me show you the mouse cursor. The more you move to the right, the higher the dose. And then you draw the line up. That's your fraction affected. And for the blue curve is generally to the right of everything else, making it like not the best. You can see in the green line that you hit that 50% maybe a bit sooner, depending. It's a little hard to tell, honestly. But this rises very quick. At a low dose, you get a pretty big effect. Like it peters off like over here, just like at like, I don't know, 5 or something. You draw a line straight up here. Oh, look at that. You've gotten 40% of cell population affected for free, basically, for almost nothing. So it's not about the height of the curves, but about its position. The further right your curve is, the more drug you need for the same cellular response. The more left shifted you are, the less dose you need for the same cellular response. And again, you can see here, 40% of the cells affected by the combination therapy, you get it almost for free. It's almost nothing. So I think the drug combo looks pretty good. Shifts leftward, meaning you need a lot less drug for equivalent inhibitory effect. So by eye, it looks like these could be synergistic. Maybe. Not confirmed yet. Math. Combination index. I don't know how this math works. Just trust me that it does. If the number is greater than 1, it is antagonistic at high doses. If it is lower than 1, it is synergistic at low doses. So they do some combinations like high sugar, high chemotherapy, lower sugar, keep the chemotherapy the same. You know, they mix and match high and low combinations. And what they get out of that is a combination index and actually one more thing, dose reduction index. So the way you read a dose reduction curve is that if your dose reduction DRI is greater than 1, that means the combination has reduced the need for that drug for the same effect. So for 48% of the inhibitory effect, the DRI is huge, which is great. That means for the same drug effect, you can lower the dose a lot more using this calculation. And as you can see, as you increase the concentrations, that synergy starts to decrease. Right? The dose reduction gets closer to 1. But at low doses, at like 48% effect, this is a rock star. Okay, not only can you reduce, because here's the chemotherapy one, this column. At high doses, this is pretty good, 1.3. And then between 53 and 45, it's like, you know. But at 48% effective ability, you can lower the chemotherapy dose by a good amount, potentially. The data seem to suggest. But of course, this is just in vitro work. It's just cells in a dish. Does this work in a real human? It's a little unclear. To do that, we get to talk about the wound healing assay. This is something that happened a lot last year, but weirdly is something that we are just getting to. This is a proxy for studying cancer. So what does cancer like to do? It likes to grow and it likes to move. And when you have a wound from a cut or something, cells that are more cancerous are going to move faster to do more wound healing. So even though wound healing sounds like a nice thing, in this particular case, you don't want there to be a lot of wound healing because that means those cells are more like cancer. I have to wonder what the drugs are doing. For the chemotherapy, it's usually attacking mitosis. For the membrane sugar, who the hell knows? It's crazy. So you have a dish of confluent cells. This is a jargony word to just mean cancer cells cover every inch of this dish. You take a glass rod and you just scratch the surface. Remove those cells. And then you track over time how quickly those cancer cells close that gap. It's not perfect, but wound healing is a pretty reliable metric for cancerous growth, so I enjoy it here. So, this is the control condition at 0 time. What does that mean? Oh, my brain. Oh, okay, this is no drug. Tell me, chat. No drug. You do this wound scratch, right? You can see there's this dead space. This is where the glass rod was scratched along the surface, taking with it all the tumor cells. What happens after 48 hours when you don't add a drug? No drugs. Come on, chat, you gotta help me. You gotta help me. I'm dying out here, man. I'm dying out here, man. My YouTube studio is crashed. Oh, there we go. I'm back. What happens to cancer cells after you wound them? Yeah, what happens? Helicornix says it spreads. And more specifically, can we be more specific besides it spreads? A little more specific. It bridged the gap in almost a day, definitely by 48 hours. Yeah. You're right, Alicornic. It did spread. But in the context of wound healing, what we really want to know if cancer moved in to fill that gap. And in 24 hours, oh yeah. If you don't add a drug, that wound is getting fixed. Okay. 48 hours. Now we're testing the drugs. Chemotherapy alone. It's the first one. Archaea sugar in 2. And then the combination therapy. Using your eyes, chat. Which of these looks like to be the best cancer treatment studied using wound healing? This is 48 hours after injury. Remember what this assay looked like, right? At 0, you scratch it, and by 48 hours, regular cancer cells that are untreated will bridge that gap, heal the wound. What about when we treat with drugs? PXL, that's chemotherapy. EPS, that's Archaea sugar. And then the combination. Did this work? What does this look like to you? Faraday, when you say the best, what do you mean by that? What does best mean to you? Be a little more precise. PXL looks the least healed. Interesting. Interesting. Otterly says the wound is healed. The cells reinvade the area within 48 hours. To the point you need treatment for more than 48 hours. The combination try to halt the reinvasion of the breast. Yeah. After we do this simple assay, there are other things to consider for sure. Drunken says the combo is more effective. The EBS ones don't look very stretched. This has to do with attachment to ECM, right? Yeah, in a way. It is connected. ECM is the stuff that tumors like to crawl on. So there's a bit of disagreement in chat. Let's see. I mean, you could be right. To my eye, maybe I'm seeing what I want to see, but I think the combination is got the best. And you wouldn't be blamed, right? These bounding boxes, I think this is not a great example of their images, but I think I'd say so. To me, this looks a lot less healed than PXL for sure, because just the chemotherapy alone, like I see quite a bit of cells that are hanging out here. EPS, I do see quite a bit here. And PXL, there are cells here, but they also don't look very healthy. Here you have these flat, smooth cells. Here these cells are round. They look like they're dying. It looks like this wound healing is not going great here, actually. ECM not showing and staining. This is not stained. This is actually just looking at the cells under a microscope with light. Hello, am I still awake? Barely. So here's the data. Oh. I don't have the data. It's just trust my word. Like, it looks a lot less healed from my eye. It's so hard to find the words, but the images are so hard to interpret that the subjectivity without some form of quantitative data makes it probably more difficult to interpret. Yeah, I think I was lazy and I didn't include the actual quantification here because I find the pictures to be more important for content. But yeah, probably should have had the data here, and they found the data do reflect what I am saying is true here. But I wouldn't blame you if you thought otherwise, because yeah, these pictures are not obviously different. That's fair. What's going on? So someone had a question before. So what are these drugs doing? Well, we know PXL is a chemotherapy drug. We have a good idea that's probably doing some anti-mitosis stuff. What about the combination? That's kind of interesting. Because now we're also talking about this archaeal sugar. And so we looked at some markers for cancer stuff. MDA is the oxidative stress indicator. MMP9, metastatic protein, CAPS3, apoptotic factor. And what we find is that there's kind of an additive effect for metastatic protein, right? Chemotherapy drug inhibits metastatic protein. That makes sense. Makes sense that chemotherapy drug would inhibit metastatic protein. Also, the archaeal polysaccharide does the same thing. And when you combine them, the effect is even greater. It's interesting. So metastasis is down and apoptosis is up, right? Because as you can see in panel C, the combination chemotherapy PXL and the archaeal exopolysaccharide EPS combined, you do see more apoptotic factor, more cell death, which in our case is very good because we want that cancer to die. To some, I think it's cool as heck that scientists are experimenting with archaeal products. And I'm gonna be honest, as much as I talked about how cool this paper is, what really gets my gander at the end of the day is how much of this polysaccharide they needed to use. This doesn't seem practical for a drug design perspective. But at the very least, it opened their eyes to potential new angles of attack, oxidative stress, apoptotic factors, etc. I think it's definitely interesting to keep in mind. I'm not that excited over the magnitude of these differences as I was in some other papers, but I do think it's kind of cool. I enjoyed this one. And there was some evidence that by using an archaeal polysaccharide that you might be able to get away with a bit less chemo, which is a good thing. The further we can move away from chemo reliance, the better. It is going to be an unfortunate mandatory part of people's lives. But the idea of combination therapy might allow them to live a better life. I need a short break. Oh, I need a short break. We're heading into the last two papers, and I need to use the restroom. I will be right back. Enjoy the BRB screen. All right, we're back. We're back in business, baby. Ah, I'm getting close to apoptosis. That's right. That's right. Oh. I mean, doctors do this all the time. Continuity of care, right? There are some doctors that work 24-hour, even 48-hour shifts. It's not that impossible for a human being to do. But I do like sleep a lot. Oh, okay. We are in the final stretch. That does not make it healthy, trust me in this. I believe. I don't do this frequently, and I probably not going to do it again next year. 12 hours is a good challenge. 24 is madness. Madness. Oh, okay. Let's finish strong. I wanted to include a paper that could be done by a lab that doesn't necessarily have all the ingredients to do some fancy molecular biology, because I think it's important to acknowledge that you can do some pretty cool science just with a computer and a GPU. So, what are we doing here? We are doing another different approach. So we've seen a lot of different strategies when it comes to drug design, and now we're going to go with a computational approach. So this is one that goes out to my computer nerds out there, okay? The design, molecular docking, and molecular dynamics of thiourea iron 3 metal complexes as nude T5 inhibitors of breast cancer treatment. Removing all the fancy speak behind that, basically we are talking about simulations. Playing fancy Legos on the computer. Can we find a chemical that binds to a cancer protein and stops it from growing? Yeah, next year for sure, 12 hours. I'm not doing 24 again. My body can't take this. So, why did I pick this paper is because I think structural biologists are wizards. They're wizards for sure. They are imbued with deep, arcane, magical energies, and I am not privy to any of their ability. But I think it's a matter of personal growth that I try anyway. Right? Love pushing back, love learning, seeing what I can do, etc., etc. So, let's take a look at what the structural biologists are doing, why don't we? The computational biologists, I should say. Oh, past 5. You're so smart. I'm going to guess by this point that my brain is probably running on Windows 95 or something. This is going to be a brief conceptual overview. Structural biologists are wizards, so I wasn't going to understand a lot of it to begin with. So let's go. Let's go. Let's take a look at what the computer nerds are doing. So new T5. Tempting target for breast cancer. Conveniently for us, elevated in breast cancer tissue. Again, a lot of research is dedicated to finding ways in which breast cancer, or cancer in general, operates differently than regular healthy cells. There's an added bonus. This isn't just coincidence. Sometimes you get a protein that is just by chance higher in a cancer cell. It's not really doing anything interesting. But this is a pretty interesting target. Remember a couple of hours back when I talked about the EMT and MET transition? The epithelial to mesenchymal transition. There's a reverse transition too, mesenchyme to epithelial transition. This enables metastasis. And we've heard multiple times the stream, metastasis, bad, bad, no good, very bad, don't like it. We also know this protein is maybe also involved in angiogenesis, which is the growth of new blood vessels for the tumor. So not only is this a convenient target because it is way up in breast cancer tissue compared to the surrounding healthy tissue, but also logically it just kind of makes sense, right? This is not a random protein doing something random, like, I don't know. Can't even think of a random protein. Lysosomal digestion, right? It's got a known direct relationship with the function of cancer, namely the EMT and MET transition and angiogenesis. Seems like a pretty tasty target. So, a class of drug that people have been playing with called iron complexes. It's a class of molecules that are being investigated for their anti-cancer properties. And these are pretty mysterious and flexible. The mechanisms by which these drugs can work can vary pretty widely. Sometimes they can form bonds that interfere with enzymatic activity. Sometimes they can just yoink out an important ion from an enzyme. Disable that enzyme because you've ruined it. You've stolen the ion out from the center. This paper seeks to use computational simulations to try and play fancy Legos until we find a iron chelating drug target that fits very neatly inside of this protein to block it from its function. Remember, this protein, shown in orange and green, it's a no-no protein in the context of cancer. It's probably cancer, helping cancer do cancer things, and we would really like to find a chelating drug that blocks it. And the authors chose 10 compounds. You can see them here. So the center of this molecule is always going to be the same. It's going to be this iron ion in the middle, and then some double bonds. I'm not a chemist. But then you have these R groups. These are variable attachments. So this core is always the same, and then we're going to add some wings to it. And then so, you know, this is wing 1, wing 2, wing 3, etc., etc. 10 of these different structures, just to try them out. And instead of wasting a lot of time in the lab, these clever little guys figured they could do it on a computer first. Eventually, of course, this should happen in real life, but this is a good start. There's already a well-characterized binder to new T5. Here it is. I'm pointing to it with arrows here. It's a little hard to see. The color scheme isn't exactly great. These 10 compounds were chosen to test certain hypotheses surrounding new T5's internal structure based off this known compound. So we know a little bit about what can fit in here. And those 10 compounds that we just saw are going to take lessons from this compound and design them after that. Cool. So, taking a closer look at the protein, you see here this yellow pocket. This is the substrate binding site. This is where we want that compound to fit in. And molecular simulations are trying to get the designed compounds to fit neatly into this yellow circle. Admirable. Makes sense to me. Cram something in here, find the right jigsaw puzzle, and maybe you got a cancer drug on your hands. Oh, Drunken Master, I need to caution you. AlphaFold, very powerful tool. AlphaFold is predictive. Okay? No matter how good that AI is, it is a guess. Which means you need to be careful. It's good, don't get me wrong. It's pretty dang good, but it is a prediction. If you want real structures, you need to go to the Protein Data Bank, PDB. And I know PewDiePie advocated for it. It's useful, it's nice. But you still need the real people to do the work. PDB, Protein Data Bank, those are the real structures. AlphaFold is a guess. It's a good one, but it is a guess. And as an experimentalist, I gotta nip that one in the bud as I seize it. Online, some people hate any AI in biology or any science. Personally, whatever Phi is currently talking about could be much faster researched alongside humans. This is a computational approach based off of rational design. Humans are still pretty good at that. And see, look, not that complicated. If we were talking about designing bigger compounds, sure, get AI to do it. This, not that hard. There's no reason to believe that AI could do a better job for something like this than a human could. AI is useful in biology, don't get me wrong. This particular project, you don't really need AI to do this particular question. So, I don't know how any of these simulations work, but apparently they can predict binding affinity and inhibition constants. Very useful terms for biochemistry folks. Which means you can kind of guess which of these structures are going to work better than others. And you get a whole bunch of other predictions as well. What amino acids are important? So our ideal compounds will have a very low binding affinity and inhibition constant for good drug candidates, right? Low binding, sorry. This is confusing because it's biochemistry. You want this number to be small, which indicates that it is better at binding. I know there's a lot of negatives in biology. We see compound 2 minus 8.77. What this is saying is that when this compound binds to the drug target, it releases a lot of energy, meaning that it is stable. The energy of the reactants is higher than the products. So energetically, thermodynamically, this binding is more stable if the number is a greater negative, right? The further away you get from 0 in the negative direction, the more energy that binding releases, which can mean that the interaction is more stable. The inhibition constant is the inhibition constant is the amount of drug you need to inhibit predicted half of the binding sites. This is all prediction, of course. So you want a low number. And so which candidates stick out to you? Compound 2, minus 8.77 for that binding affinity, and a pretty low inhibition constant. Remember, looking at that column, some of those are millimolar and some of them are nanomolar. Whole order of magnitude different. So the researchers narrowed down their 10 compounds, narrowed it down to 4, based on the thermodynamics here. Reasonable narrowing down. Why did I do this? Oh, it's because, yeah, 2 of the compounds that they had chosen, 6 and 7. Hold your applause. Or applaud, I don't care. Yep, they chose product 6 and 7 and 2 others. This is something cool. Ramachandran plots. So, in biochemistry, when you are talking about the linkages between the components of a protein, amino acids, just get the basic idea, because I don't fully understand Ramachandran plots myself, but I do get the basic gist. Phi and psi, side array phi, these are terms that describe rotations around certain bonds within a protein. And not all of those bond rotation angles are mathematically possible. So a Ramachandran plot tells you which combination of phi and psi angles are allowed, favored, and disfavored. And each dot is one amino acid in the protein chain after it is bound to your drug of interest. So what we're looking for here is a reality check. A Ramachandran plot tells us, whoopsie doodle, your drug accidentally made the protein illegal. That's no good. I want to be on the right side of the law. So each compound was simulated with protein, and the protein was analyzed for legality via bond formation. You want your dots, so every dot is an amino acid. You want all those to be in these comfortable red islands. And if all of those dots are in the comfortable red islands, then you have a probable structure. Means you didn't break any biochemical laws. Good to check. Compounds 2 and 10 have an illegal amino acid. Yeah, Hair Day, you spotted it. Looks like they have a bond that we don't like. All the way in the yellow area. These are amino acids that the authors highlighted. I'm calling them illegal, but these are disfavored amino acids, meaning the quality of the simulation was less than ideal. You shouldn't have illegal combinations of phi and psi angles. Meaning, again, this is all happening on the computer. It just means that you can trust the 2 and 10 data a little less, because it just doesn't work. The math spat out a configuration that should not be possible. Something's wrong there. It's not the only metric though. There's also something called root mean square deviation, RMSD, that allows us to measure conformational stability, how stable the structure is. Another thing that you can simulate. A low RMSD indicates that the protein ligand structure is stable. And a non-ligand looks like it changes a lot over 100 nanoseconds of simulation. And if we take a look at these different compounds, 2, 6, 7, 9, 10, and then a random thing that doesn't fit at all, we can kind of see that, if the non-ligand, that brown graph, that's the sign that the drug doesn't bind at all. So I would say that 7 doesn't look very good. Because 7, that yellow graph, looks a lot like that brown graph. And that brown graph is telling us what it looks like when a drug is not binding. But 2 in red, 6 in green, and 10 in this kind of blue, teal. That looks pretty good. They got a low root mean square deviation. Looks pretty stable. And that's pretty neat. So what do these compounds look like? We can see that they have images, simulated images. These are not real crystal structures. But you can kind of see how using a computer model, you can kind of guesstimate how this drug might interact with the internal workings of a protein. And that's also really cool, right? I mean, they might not be right, but you can generate hypotheses. You started with 10 compounds, now we're down to 3. You know what's a lot easier to test than 10 compounds? 3 compounds. So, computer simulation will never replace experimentation, but you can bet your bottom dollar that it can make it easier to test, because you just don't have to test everything. Computers are supposed to make our lives easier. And one way in which we can make our lives easier is by ruling out things that don't make mathematical sense. Maybe you miss something. Maybe one of those simulations was not totally good enough and you missed something. There's always that risk. There's always that risk. But if you've crossed your T's and dotted your I's, this should save you a lot of work. Just running a simple simulation. Cut down 10 candidates to test, now we're down to 3. The dynamic simulations also show that compound 6, 7, and 10 all stay within the active site of the target protein, T5. So when you simulate these proteins, they don't wander off, they don't do anything funny and walk along the protein or anything weird like that. They actually stay pretty stably within the active region of the enzyme. This is good. This is showing that from the way that the computer understands biochemistry and structural biology, that this should be, in theory, a good inhibitor. Good drug to potentially attack cancer with. That's cool. I like that. This one's a bit of a shorter one. So here's the summary. Molecular dynamics simulations offer a pretty cheap and effective way to narrow down hypotheses. Even though this work took place all on a computer, these researchers may have narrowed down the pool of drug candidates in this class without having to sacrifice a single mouse. There is value in that. Computer simulations are an efficient, cost-saving method to generate hypotheses, and can be done by many labs across the globe, not just ones with fancy research budgets. Using molecular dynamics, several good candidates for further testing have been identified. And yeah, as Audley pointed out, with anything with a simulation, you just kind of have to be ready for your thing to be crap. But it might not be. And again, it's cheap. It's a cheap way to try to make your work more efficient. Could miss something, it's definitely true. But I am, again, all about accessibility, and I happen to know, don't ask me how I know, stuff like antibodies are really expensive. Biology is not cheap. But if there's something that you can do to help make your workflow more efficient, then why not take that? Right now, this lab, you just have to spend a little bit of GPU power, and now they don't have to synthesize 10 different compounds and test this protein. Now they can try to get away with testing 3. And yeah, they might find garbage. But they did make their research more efficient. Saved a bit of money. And I have to acknowledge my privilege that I've been fortunate enough to be a part of so many universities with nice research budgets. But shout out to the hard work that less than rich universities are doing too. Don't want to forget you in this battle. And again, US universities, powerhouses. Higher education in the US is one of the things that as an American we can actually be proud of, because our schools are awesome. Not all countries have schools like the ones that we have in the US, and they gotta be able to do science too. So yeah, obviously test it, but also value the fact that computers can make things a bit more efficient. My uni actually ran student projects on the computational models for drug design, if that helps put into perspective how accessible it is. Yeah. You don't need fancy stuff to run some simulations to do this kind of work, and I figured I wanted to shout that out. Okay. Perkinji's gonna riot. I need to feed her. It's breakfast time for Perkinji. It just shows you how long this stream has gone. 22 hours in. So, enjoy the be right back screen. We might be heading into the final paper? Question mark? This is a short paper though. I truly don't remember, but be right back. I am going to feed my cats breakfast because now the sun is actually coming up. So I will see you soon. All right. All right. Where are we? Getting up and feeding my cat was nice. Getting up and stretching to hydrate a little bit. I'm back. I've made it. Give me a moment to hydrate. In the last stretch of the stream. It's an hour and a half more. Okay, let me hydrate a bit. All right. How are y'all doing? How are y'all doing? Thank you so much for the donation, Surprise Pants. Let's go, almost. Thank you so much. Oh man, what a day. What a day. All right, what do we have left? How many papers are even left? I've lost track of my existence. I'm dissociating. It talked about molecular dynamics. It talked about obesity. What slide number are we on right now? Oh. Okay. Slide 447 out of 474. This is the last paper, chat. Let's just take that in for a moment. This is the last paper that I have to do. I'm excited. Because I think I saved the best for last. I think I saved the best for last. This is cool paper. This is a pretty cool paper. Okay, let's talk about pretty cool paper. And then we'll chat. I'll say a couple of parting words. Maybe a little bit of reflection. Because it does look like we'll be done with time to spare. So I do intend on ending at 9. Try my best to keep my promises. Okay. Last paper. Oh, and I love this paper too. Synergistic. I love the word synergy. Immunomodulatory effects of naringenin. I don't know how to say any of these biology words. Naringenin, whatever. And photothermal therapy in triple negative breast cancer. This is another wackadoodle approach. But I actually really like this wackadoodle approach. I think this approach is pretty interesting. And I'm going to tell you why. So I feel like for the last couple of entries, we've talked a lot about combination therapies, you know? We've tried a lot. We've tried a lot to cure cancer. But a lot of the time, those often come up a bit short. The idea of combination therapy makes it so that, well, it's okay that your drug sucked. Cancer is terrible. But you know what's better than one drug? 2. So, it's a pretty interesting principle. Because you can start doing wacky and wild things with combinations. So what are we doing here? There are 2 components to this pitch for a drug against triple negative breast cancer. And the first component is a photothermal therapy. Sounds fake. Light heat therapy with biomineralized nanoparticles. Again, if you didn't know any science, this would all sound like mumbo jumbo, something somebody cooked up and is trying to sell you. The second component is a flavonoid, naturally occurring, just a natural product. Don't know why they picked this one, but they did. And these two things that you wouldn't necessarily think of as being cancer treatments, they are trying to convince us that somehow they are going to form something very special. And they are going to be something that kills cancer. And I think that's awesome. Because I've been on a nanoparticle kick the last couple of years studying cancer biology. So this is really special to me. But I never had a formal training in nanoparticles. I've been kind of picking it up as I go. And there's something very surprising about this approach to me that I'll get into. I've done this part a couple of times, but again, chemotherapy sucks. Let's try to do better. But again, it's unfortunately one of the only available biological vulnerabilities that triple negative breast cancer actually has. Again, triple negative breast cancer being the type of cancer that is the worst to get, or one of the worst to get. Why? Because you can't actually do all that much. It's probably going to be chemotherapy. If you have a hormone receptive cancer, then you can use hormone blockers. And that works quite well. Because in hormone receptive cancers, as I've explained 10 hours ago, hormone receptive cells are responsive to hormone therapy, which aren't as extreme as chemotherapy. But again, for triple negative breast cancer, this 10 to 15% of the population, it's probably chemo. We don't like that here. So, what do we do about that? Chemo is going to be hard to replace. Okay, chemo is pretty good. Has helped a lot of people live fulfilling lives. Long, good lives. So they could spend time with the people that they love. But boy, does it suck. It's poison. It just poisons cancer worse than it poisons you. So of course, whatever treatment is going to attempt to replace chemotherapy is probably going to be something wackadoodle and out of the box, kind of like this. This strategy is weird, but I love it for it. So this strategy involves taking a mediocre compound, shown here, and enhancing its ability to fight cancer with another strategy, photothermal stimulation. Jesse, it's time to cook. Specifically, cook the cancer. That's awesome. You're still alive. I went to sleep and woke up again. I did promise 24 hours, didn't I? And, you know, for some reason, nobody's donated $8,000 to get me to take a nap immediately. Or to go to bed immediately. Wow. Hour 22.5. I'm so glad I found this stream from Octopus Lady. She's the best, dude. She's the best. Though I missed her part. It'll be on the VOD. Thank you for hanging out with me here. Oh. So, mediocre compound on its own, but its name is not chemotherapy, so I'm happy. And then we're going to combine this with something called photothermal simulation. What's that all about? What are we talking about here? So, photothermal therapy using light to increase the temperature of cancer cells. Burn them. Pretty straightforward. But the practice of photothermal therapy is not straightforward. Don't be an idiot. You can't just burn your cancer at home. It's not how that works. You need to specifically target the correct tissue and not damage the surrounding tissue, because guess what? If you unnecessarily damage your body in the process of trying to cure your cancer, you might even make it so that cancer has an easier time to spread. So please see a doctor. My goodness. So while photothermal therapy could kill cancer, it's also going to do stuff to your regular cells too. It's going to cook them. So how do we Trojan horse just the cancer cells? Right? Because photons, they don't care if you're from cancer or not. This is a physics treatment. It doesn't care if you're making protein X or protein Y. This is physics now. So scientists have to be really clever here, right? You want to use photothermal therapy, but you need a way for it to only kill cancer cells. So, I saw the channel was live yesterday. I did not get the notification it was 12 hours. I definitely know I want to watch the next 14 hours. Let's go. That'd be crazy if you managed to work your way through. I think the VOD is so boring. There's a lot of pauses because my brain is so tired. But Brendan asks, is there something that only cancer cells absorb or absorb more readily than other cells? That's an interesting idea, isn't it? This is why basic research into cancer biology is so important. We have to find out all the nifty things we can exploit to kill this thing. And you would be right, Trenton. There is. Cancer cells are known to absorb the protein albumin at a much higher rate than healthy cells. Why? I don't know. For some reason, cancer cells just gobble this stuff up. They love eating up albumin. And regular cells don't really care. And that's interesting. Again, every little difference we can find. You might not find it important, but that's why the engineer is impressive. The engineer looks at cancer cells and it says, hey, you like albumin? Cancer cell says yes. Engineer says, well, I'm going to give you albumin. And I'm going to do that with a nanoparticle. So if you want to talk about Trojan horsing just the cancer cells, albumin's a good choice. Regular cells don't care for it. Cancer cells, they eat that up, no crumbs. But how do we do it? Just because you know albumin is something that the cell wants, doesn't necessarily allow it to translate into therapy. Here's where biomineralization into a nanoparticle comes in. We can transform albumin into a delivery system for something else. And what might that be? In this case, it's kind of itself. Sometimes nanoparticles are used to deliver other things. But in this case, the albumin nanoparticle itself is the thing that is going to allow us to cook only cancer cells. You're still way more engaging than like a quarter of my professors. Thank you, but I'm really wondering, I am literally falling asleep, so your professors need to get with it. How do you synthesize a nanoparticle? This is fascinating. It's shockingly simple. You can buy bovine serum albumin from a lab store supply store. I'm not saying you can, but if you're a researcher, you can. And you mix it with potassium permanganate, very common chemical reagent. If you're a chemist, you know this compound. And just by stirring these together, you'll find that the bovine serum albumin will biomineralize. The protein forms these repeated structures, these particles, that conjugate with the MnO2. I'm just going to call it MnO2. And these nanoparticles have a very useful property. Even though I think they're really weird, because, you know, when I was learning about nanoparticle stuff, it was gold and silver because they were inert and could be used to deliver drugs across the membrane. Like, those are really, really hot. I've never heard of a protein nanoparticle before. That's kind of interesting. Here are some pictures of the formation. Not really a lot to look at here, but if you're curious, this is what they look like under a microscope. Yeah, that is easy, right? You just stir 2 ingredients. Come on. And then you wash them. Bang, nanoparticles. It's cool. And there's a unique chemical property of this particular nanoparticle. It gets hot. Really hot. A heat map. This is an image, an infrared image of some kind, or false colored somehow, that tells you the temperature of a nanoparticle. On the right here we have saline, just salt, nothing there. You shine an infrared laser, and you can kind of see, yeah, the saline heats up a little bit, right? This area of blue, it's a little lighter than the surrounding area, indicating that we went from 20 degrees to a little warmer than 20 degrees in the saline condition, sure. And we have NGR, which is a peptide anti-cancer compound. This is just some other thing, and look, doesn't absorb heat very well at all. You load in the nanoparticle, and look what happens. Blazing red hot, 60 Celsius in the center of this nanoparticle. 60 Celsius. Burning. That's hot. That's really hot. It's white hot in that nanoparticle. And we are going to use this to cook. We're going to cook these cancer cells. Burn them. Pyro. Team Fortress 2. We're going to melt them. Burn in heck. So, let's look at the behavior of these nanoparticles. You turn the laser on. About 5 minutes. It reaches a peak temperature of 60 degrees Celsius. Hot. And then over the next 7-8 minutes, looks like. Quick math in my head. Remember, I haven't slept all day. A steady drop off. It gets hot pretty quick and it drops off. Meaning if we snuck these into cancer cells, which can, by the way, because these are made of albumin and cancer cells love eating up albumin, they will willingly take up this Trojan horse. We can burn them. That's awesome. That's sick. So when you use this, does it work? Triple negative breast cancer cell lines. A. This is a known anti-cancer agent. Increasing in concentration. What do you see? Cell viability. Normalized to 100% in the control. Yeah. This is a known cancer agent, NGR. You use a cancer drug, it kills the cancer. It's pretty straightforward. What about our nanoparticle? Nanoparticle alone. You just give the cancer cell the nanoparticle. Well, it looks like the nanoparticle alone isn't as good as the cancer drug at killing cancer. You have to give the cancer a lot of this nanoparticle to actually kill it. Are you surprised? Did that surprise you? Does that make any sense? You're just talking about burning and stuff. Does it surprise you that just giving the nanoparticle to the cancer cell is maybe not that effective? Some questions about denaturation. Proteins are all different. Some proteins denature at very low temperatures, others denature at very high temperatures. Couldn't tell you what most proteins denature at. It's kind of a difficult question to answer. most proteins are denatured at 95 with detergent, like you would do for a Western blot. But yeah, would it surprise you? You know, I was just talking such a big game about this nanoparticle. This nanoparticle's awesome. And you give it to the cancer cell, doesn't really care. Only starts caring at huge concentrations, 500 micromolar, 1,000 micromolar. That's a lot of nanoparticle. Wacky says, did they do the heat process after applying the nanoparticle? No, just the nanoparticle. It's a little cute trick. Good job, Wacky. It's not surprising. It's just a nanoparticle. Not that surprising. You need to actually turn on the laser to kill anything. Okay, so we know the particle itself, not that dangerous to cancer. But what about when we turn the laser on? What do we get there? Now what do we see? Control is control. Just the triple negative breast cancer cell. The nanoparticle is a nanoparticle, MnO2 at BSA. NRG is a real, actual cancer drug. PTT, this is a nanoparticle plus near IR light. This is when you actually turn the laser on. And then PTT slash NRG, known combination. Look at that. Isn't that kind of interesting? So again, nanoparticle alone. Your kind of crappy drug, NRG, doesn't work too good. You can see cell viability 100%. And then when you add this compound, it's 80%. It's not that good. Not that good. PTT, that's nanoparticle plus laser. All right. 70% maybe. When you combine the therapy, now look at what we've got. Quite a bit of a reduction. There's an argument for there being synergy here. Can't really confirm without some tests, but it does look like the sum, you know, the whole is more than the sum of its parts here. It's kind of cool. It is really interesting. I mean, this is cool, right? To do this in a dish and just see if the cancer cells die or not, but cancer is more than just survival. The cancer is also proliferation. And you measure that in a different way. And that's with the wound healing assay. It's a proxy for cancerous growth potential. And when you do the full combination therapy, nanoparticle plus laser plus your crappy drug, what happens in wound healing? Again, if you don't know how to read this, triple negative breast cancer cells were plated on a glass dish, allowed to grow until they were full. And they took a glass rod and scraped it across the surface, just like that. Scraped it right across. And there's a dead zone in between these two red lines. The top, we have day 1. The first day that you do this, and you see that there's a dead zone between these red lines. This set of breast cancer cells is not going to get any treatment. This set of breast cancer cells is going to get combination crappy drug and nanoparticle plus light. So what can we say about wound healing in these two situations? The bounding box is this dead zone, and the red lines, they border the empty area that resulted when you dragged that rod across the glass plate. Trent says, so basically the growth has been severely disrupted or halted. Yeah. Cancer cells love growing. All right, take a look at this control. This is cancer. A lot of cells up here, a lot of cells down here. You scrape across that glass plate, remove the breast cancer cells in the middle. 72 hours later, it's like nothing ever happened. Cancer cells are growing, happy. They've closed the wound because they are cancer-like. right? They are dividing a lot and moving a lot and seeking to form. Okay? But when you add this combination therapy, infrared light plus your nanoparticle plus crappy drug, instead of healing, it mostly stays the same. Right? This is complete closure. This cancer healed beautifully. Nothing was stopping it from healing. And in this cell line, something was stopping it from healing. It's your drugs. Your crappy NRG drug on its own is pretty crappy. The nanoparticle alone is pretty crappy, but combined, yeah, they're doing something. They're doing something. It still grew somewhat, yeah. It tried to close the wound, but it's a lot slower than the control. That's exciting. So, we can't just live in a fantasy world where things that we do in dishes automatically work for people. Gotta test this in an animal, man. We gotta. So how do you do that? You take a mouse, you inject in your nanoparticle, a cancerous mouse, no, cancer mouse. You inject in your nanoparticle, you give them your crappy drug, you give them the infrared light to attack that nanoparticle, heat it up and cook it. Yeah, and then you inject the crappy drug for 3 days, and then you check the tumors, right? So basically taking what we did to the plates and doing it in an animal, in a mouse. This is a heat map of the injected tumor site. And again, we have here a heat map based on temperature. So, what conclusions can you draw from this? What does this look like to you? First, inspect it with your eyes, not with your mind. Just think about the shapes and the colors and the differences between the different columns. And if you do that, coming up with an intelligent answer will be a lot easier, trust me. The graph before looks so ominous. What do they mean by sacrifice? It means sacrifice. Lab animals don't live a good life. How do you think they got these tumors to look at under an infrared camera? That's why it's important to not use mice all willy-nilly. You have to be very careful. Yeah, it got a lot hotter. Remember, this is the nanoparticle, MnO2 at BSI. And then when you add the drug, it doesn't seem to make a difference to my eye. But if you compare saline, which is salt water, to the nanoparticle plus laser, this gets hotter. That's cooking, dude. That's cooking. So it looks like the photothermal laser doesn't really change the temperature of tissue without the nanoparticle, right? It's a little spicy here. All right, the green, that's about a little under 30 degrees. But remember, like, these tissue typically, 37 is body temperature. These are actually a little cold, weirdly enough. But the point is that they don't heat up nearly as hot unless there is the nanoparticle there, which is great because you don't want to be burning yourself trying to get rid of your cancer. This is convenient. So we know that the heating element works, right? Infrared lasers working, the nanoparticle is getting hot. How does this actually affect the tumors that are growing in these mice? Here's your warning, pictures of tumors. Had a couple of these warnings, but if you're squeamish, you might want to look away. Okay? Okay. tumors on the screen. First row, saline, no treatment, just cancer. NRG is our kind of crappy cancer drug. Okay, observe that column. PTT is the nanoparticle plus heat. Look at that column, cool. And the combo, PTT and NRG. Take a look at those tumors. What can you conclude here? Again, first look with your eyes, not with your mind. But what could you say about the effect of this combination therapy on tumor volume? Comparing the saline control, which is just salt. It's not expected to do much to tumors. Comparing that to the PTT NRG line, combination therapy. Combination nanoparticle and laser blast, and kind of mediocre drug. Combo seems to do great. The tumor turns into a ruler. That's very funny. True. The tumors are smaller or have shrunk. Yeah. Tumor growth was lessened. I buy it. Do I have the data here? I don't remember. Okay, I chose not to include the numbers, but you're right. Combo therapy seems to be working, at least in tumor volume terms. What's happening? Why is the tumor smaller? Could be for a couple of reasons. Could be that the tumor never grew. It's possible. Could be that the tumor died. It's also possible. Or it could be the case that the tumor never grew. These are three distinct processes. Did the tumor die? And is that why it's smaller? If the tumor died, then we should see a lot of evidence of apoptosis proteins, panic proteins, self-destruction proteins. But we don't. So what other reason could there be? Besides death of the tumor. Oh man, I'm like so out of it. Oh, closing my eyes for just a little bit is so nice. Thank you for the donation, Diamond. Once again, great stream. The commitment is crazy. Make sure to take a rest a lot when it ends. Oh, you won't be able to stop me from taking a rest here. Hey, Clockwork. Clockwork. Do you know a protein called piezo-1? I forgot that I wanted to show you a cool protein. You might already be aware of it. Piezo-1. You know that? You know that protein? You familiar? I wonder if the stream is going to conclude with you just sleeping and the stream just going onward. It might. It might. I am in the process of losing my marbles. Make sure to hydrate. I will. I'll take a little bit of a hydration break. Okay, so tumor size. Lower in the combination therapy. Why might a tumor be smaller? Could have died. It could have failed to grow. There's some third reason I don't remember. Crap. Could have died, could have failed to grow. There was a third one I said, and I like completely forgot. This is a real sign of my cognitive decline. Yeah, piezo-1 is one of those mechanosensors. When are we gonna get the piezo-1 clockwork video, dude? The sensory of the memory, because it showed up in a cancer stream. Showed up in a paper here. I was like, I took a look because I didn't know about piezo-1 before. I took one good look at that protein. I was like, that's a clockwork protein if there is ever one. It was cool. That's all I'm saying. But yeah, I don't remember what the third hypothesis was, but a tumor might be small because it's dying, or a tumor might be small because it's not growing. And we can figure this out with a neat tool called Ki-67. This is a marker that, when you detect it, marks the process of proliferation. So the more red signal you find, the more cells are growing or dividing. It's a little different than cell density, which we were guessing with before. The amount of red on these slides tells you the degree by which the cells are actively replicating. So, chat. With that in mind, is the tumor smaller because it is dying or because it is not growing? What do you think? Oh my god, another 6-7. Jesus. Jesus, I've gotten 67 twice by other people on my own stream. That's devastating. Devastating. Any thoughts, chat? Because I know I have one. I cannot wait to apoptose. But I believe in you. You know, help me push through this one last hour of streaming, y'all. The red denotes cell division. What does it look like in the combination therapy? Did the tumor start similar before the treatment? Yeah, got the same injection. It should be the same when they started. So what is this telling you? Skylar says it's not proliferating as much in the combo. Yeah. So again, tumor might be different. Two hypotheses. Maybe the combination therapy is killing the big tumor, shrinking it. Or maybe the tumor never got to get big in the first place. And this Ki-67 proliferation test it gives us a little hint that there's not a lot of proliferation going on. So it's possible that these tumors never grew. And that's interesting. And what else you wouldn't like to grow? Tumors. So, whatever this combination therapy is doing, it looks like it's having an effect. And of course, unless you totally arrest all these cells, there's going to be some baseline level of cell growth anyway. What's important is that it looks lower than doing nothing. I think that's interesting. It's a little hard to tell the difference between the speckle, but it looks a little sparser. It does. I think you trust your eyes on that one. But good job, Chad. So, whatever is happening, this combination therapy is working through proliferation. Cells don't want to divide anymore. They're trying to answer why. I'm too tired. Past Phi knows that I was gonna be cooked at this stage. But they were gonna do fancy computer stuff, immunological network analyses to reveal that this kind of crappy drug that they're making is maybe working on immune cells, including our guys, the T cells, activating cytotoxic and helper T cells. And so in blue, you see the marker of nuclei. In yeah, this is just that we're not going to worry too much about that. What we should be seeing here is that better immune. I don't even understand what I wrote here. Better immune evasion. I think it means worse immune evasion. Because when you have the saline control, wow, I'm still awake enough to figure out I was wrong. That's kind of cool, actually. CD3, CD8, CD4, these are markers for different types of T cells. And in regular cancer, you don't find too many of them. But when you hit them with the wombo combo, the nanoparticle, the infrared light, and your crappy drug, something interesting happens. A lot more CD3s activating T cells. a lot more cytotoxic T cells, a lot more helper T cells. It looks like the double whammy of nanoparticle, infrared light, and NGR look to be making a big impact on the immune presence in the cell. So I don't really know what to make of the last thing. What did we learn last time? What was the last thing? Yes, this thing. Proliferation thing. They just kind of like say that and then they move on. Something more interesting like the immune system. So we're going to look at some more tumors. 3, 2, 1. Not only are there changes in tumor volume, but also in metastasis. We're looking at the lung now. Histology, which means I don't know how to read this. My guess is that we are looking for the size of the tissue that exists in these slices. And you can kind of tell that in regular breast cancer, tissue is very dense. Very dense. A lot of cells, probably because they're proliferating. When you hit patients with triple negative breast cancer, wham bam, thank you, ma'am. Or is this in mice? I don't really remember. It's probably for mice, let's be real. They're tumors to the left. I said they were humans. Oh my god, I'm wrong. This is mouse stuff. When you do this experiment in mice, you actually see that the tumors that have had this wombo combo treatment develop a lot slower. They're a lot smaller than the tumors in the untreated version. Next. There's one more hour left, chat. Let's go. One last hour to donate to charity. I'm not a histopathology guy. I don't know how to read this data. But the authors agree that the safety profile of their combo treatment is pretty good compared to the gross morphology of their assessed organs. Are relatively normal. Hey, Purkinje. Welcome back, Purkinje. Hello. Can y'all hear her? She's so loud. Very talkative. Oh, bumped the mic too. Hello, what can I do for you? Would you like for me to do your litter box? Can I wait till later, please? Purkinje, I'm begging you. I'm so tired. Looks like a bunch of squinty orbs. Squishy orbs, yeah, me too. I see. That can't really tell, dude. It's all the same to me. It looks like the liver, whatever this gap is closed in the treatment, but they don't appear to like mention that. And I know histology slides can be kind of wacky. So you really have to have an experience with histology to like tell what's happening here. I'm not even gonna try. The authors say that the tissue looks largely normal. I don't really think the liver looks the same, but maybe there's a lot of variance. Maybe this can be normal, maybe that can be normal too. I don't really know. I'm not a histology guy. I guess just the tissue looks like how tissue should look. Yeah, that's what they're saying. And I don't know how they should look. I'm not a doctor. So I just nod my head and I say, okay, whatever you say, bro. What's more important to me is function. So some basic kidney tests. Useful. Making sure we're not poisoning the kidney. So the amount of creatinine, the amount of urea, ALT, all metrics of kidney function. Yeah. NS is all across the board, not significant. I buy that. Doesn't really look like the kidneys are all that different. It's not the only organ. Maybe something else is messed up. The kidneys look fine. That's chill. So just some. I'm sorry I didn't give this paper the energy that it deserves, because this is a genuinely very cool paper. Combining a nanoparticle strategy with photothermal therapy, with a traditional chemical treatment to inhibit cancer growth for a relatively cheap price, right? That CAR T cell paper I covered a few papers back. That one was crazy. That one was gene therapy in combo with CAR T cells. You're paying up over $1 million for that kind of treatment. This one's a lot cheaper. A little bit of bovine serum albumin. Not vegan. Sorry. But a little bit of that goes a long way. A little bit of potassium permanganate. A little bit of labor, and you got yourself some nanoparticles that might, if you expose them to any kind of tissue, might be important. Oh my god, I'm just making words up. None of that made sense. Sorry. My brain just went into autopilot. Fine, you gotta think. Gotta pull it together, man. My brain was just, oh, that was great. That's a crazy feeling. It's a crazy feeling to not be in control of the words, because my lips were just moving. I was just yapping there. That's not at all what I meant to say. This study's cool because the nanoparticle works to some degree. The tumors love taking it up. You don't even have to trick them. You don't have to package the albumin nanoparticles in any kind of fancy way. You just put them near the cancer cells, and the cancer cells are like, oh, cool, nanoparticle. You eat that crap up. But the clever scientists snuck a manganese ion inside. Something that responds to photothermal stimulation. Nanoparticle that heats up at a much higher and faster and more deadly rate than a typical tissue would, typical healthy tissue would. And you've Trojan horsed a sleeper cell into your body. Because when you turn that infrared radiation on, those beads are going to heat up to 60 degrees Celsius, which is going to burn through the cancer's insides. All while hopefully leaving the rest of your cells intact. Which is a very different experience to chemotherapy, where the drug itself will hurt you. Will hurt your cells. But again, sometimes these wackadoodle plans, like, oh, I'm gonna make a nanoparticle and then I'm gonna do whatever, add a random flavonoid. I don't know anything about this flavonoid. I don't think they wrote a lot about this flavonoid in the paper. But the idea that you could do that in combination and have a huge loss in tumor volume in a mouse and a huge loss of metastasis, I think. Did they do I don't even remember. Point is, it's better. Combination is better. And that's cool as hell. Does it like take up a single albumin at a time, or is it a lot at once? I think it's like a continuous flow. Yeah, you get in contact with albumin, you eat it up. So however your cells are exposed to albumin, I think depends on the rate. It's a complicated question, though. That's it. Oh, I want to emphasize, yeah, the last paper, expensive. CAR T cells, gene therapy, more than $1 million. PSA nanoparticles, it's not cheap, but it's also not backbreakingly expensive either. And again, accessibility is a really big priority of mine in the lab space. So, I love that the king is what am I saying? I'm just speaking actual, the words that I was gonna say. I'm glad the king is still using a fork and plate to eat his breakfast. It's just words. It's just words coming out. Doesn't make any sense. But yeah. Thank you for the $50 donation, Tyler Null. You're doing great. An hour to go. Thank you so much. We're at the end point here. The final paper, the conclusions. I just need to finish this thought, but I keep on drifting off and talking in my sleep, essentially, because I have no idea what I wanted to say there. The point I want to end on, it's so hard to just end this because I'm fading so fast. The point I want to end on here is that good treatments are human focused, humanity based. Okay. Good engineering should empower humanity, not just the top 0.1%. So I like this. PSA nanoparticle, cheap. Flavonoid, cheap. CAR T cell, expensive. Gene therapy, expensive. So I like this paper a lot because, you know, it might be a good idea to combine two kind of crappy individual therapies. And if it helps somebody with triple negative breast cancer avoid chemotherapy, I'm all for it. I'm all for it. That's awesome. You're saying because if it's one albumin at a time and the nanoparticle is like a ball of multiple albumin, does it need to reform into the particle in the cell to be very warm? It kind of like I don't know for a fact. It's a good question. How I imagine it happening is you have a bag of water and you fill it up with these semi-buoyant balls. They don't all float to the top. They all distribute evenly within the water balloon. And then you throw a wave at it, and then those balls will heat up and they'll heat the water up around them. Right? It's not the ball heating up that matters, the nanoparticle itself. It's that the nanoparticle heats up, which will cause the cancer cell to heat up, ultimately, at the end of the day. At least now I learned why sleep is important. It is. It is very, very important. And I am in severe need of some right now. That was so wild. I really wonder where my brain is going. I was like, my brain thought it was really doing something there. Like, they're gonna say something about a knife and a plate. I don't even know if I said that out loud. Did you hear that, chat? Did you hear me talking about a plate and a fork and a guy? Because that's what I remember, but I don't know if that was reality. The mind is doing crazy things right now. Okay. Oh my god, I said yes, the king uses a fork and plates out of the blue. Yeah, that's what I thought I said. That's what I thought I said. That's so funny. Where was bro going with that? I have no idea where my mind was wandering there. It's a good thing the king uses a fork and plate. Maybe it was something like, you know, something about cancer equality. Like, you know, the rich, poor, cancer affects everybody. Doesn't matter. And it's like we all put on our pants one leg at a time. The king still uses a fork and a knife and a plate. Maybe, I don't know, it's my best guess. That's crazy. I think it's more like the albumin is the carrier. It is, yeah. I've been referring to them as the same thing, but you're right. The albumin is the carrier. It's the manganese ion that is heating up. It's just that the thing that weakens the cancer is the entire thing being heated up, supposedly, and not that the nanoparticle is getting hot. It's that the nanoparticle getting hot makes just the cancer cells very hot. Senescence is setting in. That's a beautiful way of putting it. I feel senescence coming on. Apoptosis is just 45 minutes away. But okay. We're done with the last paper. To some. We can wrap this freaking stream up, dude. That's it. That's all the freaking research, y'all. Oh gosh. Oh boy. What a time. Do I even have a functioning hippocampus to do a retrospective on this? I guess I have to. I've got 45 minutes more of content. We can make this a retrospective. I'll say first what I'm feeling. I typically like to do this before any kind of annoying processing of my thoughts can interfere with my emotions. I would like to first describe the emotions that I am feeling. Good. Not physically. Definitely not physically. I feel terrible. But I feel happy and accomplished. I love pushing my boundaries. I've found out that, yeah, my boundary is 12 hours, dude. 12 hours, that's hard enough. Yeah, last year when I did 12 hours, I'm like, that was pretty easy. But man, dude, if I didn't have sad boy hours at 3 a.m., I would be dead. I would be the fork eating with a I would be the fork eating the king and the knife. Like, it's nice to have structure. Like, the schedule worked out beautifully. I felt like it kept a really fun tempo and gave you all something interesting to think about in between talking about some pretty hard research. And I knew that 3 a.m. was going to be tough to get through. And I'm happy to have some friends who stuck around for that. And my lovely wife, Toxo, kept me company and refilled my hot water. I'm very grateful for that. And I'm proud of what we've accomplished here. Because let's be real, did my streaming audience double between last year and this year? Not really. I didn't grow by that much, streaming audience wise. YouTube, it was a good year. December was a good month. 2 good videos out. Got me back on the map, and I can't wait to get back to making videos. I felt like I could reasonably double the goal because I think with another year of you getting to know me as a creator, I felt that we could connect on that fact that you would trust me with your donation dollars for a great cause, and that I would make it worth your time to hang out with me here. It's a measure of confidence, maybe overconfidence, for sure. But I think in certain ways, you kind of have to be that, right? Scared money don't make money. And if I don't believe in myself, how is anybody else going to believe in myself, right? So doubling the goal, even though my channel hasn't doubled in size, was lofty, was ambitious. And it was doubling a stretch goal, by the way. A real goal for this stream would have been $1,000 based off of calculations. Because I was comparing myself to much bigger content creators and seeing what they could raise. And what you showed me is that you are every bit as... Oh, I'm gonna cry again. What you've shown... Little pansy, dude. I don't have any shame crying on stream. I don't... Just doesn't make for good content. There's a lot of pauses as I'm trying to collect my thoughts and words. You've shown me that my audience is just as passionate as I am. That's important. I feel like I'm a bit much sometimes. I did 24 hours of a live stream. I'm talking about cancer. Because I'm built different. It's not a compliment to myself. I'm a little sick and twisted in the head. This is supposed to be nice words about myself, but I want to express very sincerely that throughout my life, I think I have driven certain people away because it's hard to keep up with someone as crazy as I am. Right? How do you live around my energy? It's hard. I've had a lot of collaborations fall throughout the years of being a content creator, not just on YouTube, before too. Because I think I can be a little much, and I think that's okay. The gamble that I am constantly making, trying to make YouTube a career, is are there enough weirdos like me out there? Probably. A lot of you have stayed up a long time, I've noticed. I've seen your names come in and out of the stream. A lot of you have been here for a long time and coming in and out morning to night, going to bed and coming. You should sleep. I committed to staying up. And that's awesome. And again, the fact that people will stay up with me to review what some would consider to be the most boring thing imaginable, primary scientific data. Y'all are good folks. Y'all are good folks. Maybe that sickness and twistedness is what makes me enjoy your content. I love nerdy stuff. I love some weirdness, and I really like your content. It's fun to watch. Thank you. And if you stuck around for the sad boy talk, I'm always on this idea. Art is a pure representation of your soul. It is vulnerability. But people respond to art because they can identify with it. The audience reflects the content creator. The content creator reflects the audience. I bet all of you in chat are passionate people. And I guess I'm doing a rant because Brady just donated another 100. I'm going on here, the very sincere middle of this conclusion. And Brady, you're gonna make me rant about Blender? Thank you, Brady, for the $100 donation. Thank you so much for the support. Blender. Okay, Blender was not only a useful content creation tool for me personally, after having the bravery to try. Blender, I gotta sincerely thank you for helping me find a bit of community in Clockwork and Brady. And I'm sure I will be meeting more biology content creators who are using this tool in the future. Because I think a crucial aspect on, crucial determinant of friendship is camaraderie. And I think that there is a bond that you can form with people who know what it's like to be working with Blender. So Blender, maybe it's not about the molecules, it's not about the molecules that you render. It's about the friends you made along the way. Blender, thank you for the friends we made along the way. Again, thank you, Brady, for the generous donation. It's awesome. Yeah. I'm not that much into biology. I'm here because of you primarily. Thank you. Okay? This insanity means something to people, and I'm glad for it. I'm lucky. In the cruel environment that is the YouTube algorithm, not everybody's insanity is rewarded quite as nicely as mine is. And I think about sustainability as a content creator a lot. Because there is a reward pathway, a feedback system. You always gotta up the ante. But I'm not gonna up the ante on this, dude. There's no way I'm doing another 24 hours. I couldn't survive it. Once a year, this feels too much. Doing a 12-hour thing per year, not that bad. But you know what the problem is? I want to do this again. Not the 24-hour research, but I had so much fun today. The stock photo with VTheorem, that was so much fun getting to hang out with my VTuber friends. It's so rare we actually all have the time to hang out casually, not making content. And that game was a lot of fun, so much fun. I want to hang out with Octopus Lady more. 2 hours of marine and molecular biology trivia. It is nice to be in the same environment, chatting with a content creator that I respect a lot, who is not only so extremely passionate about their own discipline. I'd say Octopus Lady is just like me. She's also a freak in that regard. It's like game recognizes game, right? I'm like, oh, you're a sick, twisted weirdo. And I'm a sick, twisted weirdo. And you're all in chat here who have stayed up with me. You're all sick and twisted weirdos. And game recognizes game. And, you know, it didn't matter whether I won or lost trivia. That's not what it's about. It's about, I respect you as a content creator. And I would like to share something that I love with you. And I would be happy to share something that you love with me. It's a very nice thing to have done. And the Clockwork Nobel Prize tier list was so much fun, dude. So much fun. Because there aren't that many people that I know where we can point at each other and go, Barbara McClintock in triple S tier? Yeah. Triple S tier? I'm lucky. And I couldn't do this in graduate school because of complicated psychological factors. I loved biology back then just as much now. But the difference is, I don't have to worry about the bench. I don't have to worry about not being a good biologist anymore. Because I was not a great benchtop scientist. It is only in the seat that I hold right now that I can benefit and profit from all of these niceties. Thank you so much for the donation, Evergreen. I just found this channel from the Octopus Lady. What you're doing is amazing. Thank you. Can't wait to watch all your videos. Thank you so much. That means a lot. Yeah. Like, I couldn't nerd out with my fellow graduate students about biology. It's such a try-hard thing to do, right? You're all in the same class. It's a little weird to treat biology like content because biology is work when you're a graduate student. And as a content creator, I don't feel that way. Biology is fun. I just get to have fun. That's cool. The work is making the videos. But I love doing that work too. Don't get me wrong. So, like, I want to find a way to do another 24-hour stream where I don't actually have to be up for 24 hours. Because all of these little games in between and getting to talk about some awesome research and getting to raise all this money, I curse my mortal body. I curse this 24-hour lifespan neutrophil. I curse the fact that I will evaporate at 9 a.m. sharp because of the limitations I have. But I'm glad I did it. It's cool. Found you from Clockwork somehow. Yeah. It's a very cool interconnected community that we've built. And it's only going to get stronger, I think. Yeah, 24 hours is 24 hours. You know, I'm a neutrophil looking out at the twilight hours of my cell cycle. And I'm enjoying the peace that comes with that finality. I personally learned to love math by rendering fractals on my calculator in my free time. I definitely understand that not being in school gives you the liberty to appreciate things. Yeah. It's just the anxiety that you get paired with when you are having to measure up to a certain metric of success. That makes it a lot harder to enjoy it. Because, like, the thing is, with being a benchtop scientist, I loved biology. But the painful reality to acknowledge that maybe I'm not like the best scientist. Does that mean I don't love biology enough? If I love biology enough, maybe I'd be a good scientist. Right? You get in your head about all this. So you try to put some limiters on how much you will allow yourself to fall in love with the subject, which is sad. And a part of the reflection that I'm doing. This is the real sad boy hours as I'm delirious from sleep deprivation. Like, don't expect this kind of sappy stuff from me at all times, right? This is Cancer Stream exclusive. And if you've been watching me last year, you know that too. I don't do that much sappy stuff on stream because I'm not in the mindset to. But if you need your quota of emotionally resonant fi, you get it in these Cancer Streams. That's a Cancer Stream exclusive thing. So, loving biology is not proof of my worth anymore, because it makes me better at my job now. And I cannot wait to celebrate the day when I make a full paycheck off of this job. I made half a paycheck. That's huge. We got to half a paycheck. It's awesome. I don't take that for granted. Work a bit harder, maybe next year that'll become a full paycheck. And we'll all celebrate. I'll probably be emotionally resonant fi the minute I can pay my half of rent. That's gonna be an epic stream. Dude, you're still streaming? Hey, 24 hours is 24 hours. That's what I promised. Not drunk, something more evaporative. Feels that way. I'm fading fast, dude. I love watching your channel because biology is both challenging to understand and to tell about. Yeah. Yeah. That's a perfect consolidation of how I feel about the subject. Not easy. But things that are worth doing in life are not typically easy. Sometimes you need to sweat a little bit. I'm a fan of difficult video games. Celeste. Lovely game. Hard. Very difficult. But there's something about being forced to put in a lot of effort that makes it enjoyable. And that's kind of what Karis... Oh, to shout out Sad Boy RC, that was a lot of fun too. I love talking philosophy and stuff, which I don't get to do for content very often. But the idea behind this style of content that goes against everything the algorithm says you should be is, I have a prediction. There's a lot of doomerism going on because of AI generated content. And I think a lot of people are worried that this is going to tear everybody's brain into mush. That humans are always going to take the path of least resistance. We're going to brain rot ourselves out of thinking critically about anything. And I have to say, looking at things through an emotionally detached lens, do I believe that? A little bit. Yeah. I think the capitalist powers that be have a vested interest in making sure that you are not thinking too carefully about anything, so they can shovel slop into your mouths. I mean, this is not an argument for moral superiority, by the way. I love me some slop. I love me some slop. Hit me up with some slop. Second monitor content. Love that stuff. But man cannot subsist on slop alone. It's not good for you. A little bit. Why not? But you cannot build a healthy mind and a healthy body solely on slop. And I think what makes me optimistic is I think we were evolved to find challenge. You know, we're explorers. We crossed oceans. We wanted to know what was out there as Homo sapiens. And I think what makes me optimistic is that I think what we will see is yeah, AI will be brain rotting us, but then I think there will come a wave of anxiety in the culture because it doesn't feel good to be slopified. Your brain is an energetic organ and needs to be exercised. And while slopification can lower the energy demand of your brain, I think there's something deep within us that craves to be challenged, that needs the challenge. And I think, and I might be naively optimistic here, you can feel free to disagree with me, but I think there is a future for content like me and Clockwork and Octopus Lady and other really in-depth content creators that are not producing slop. Because I think as the amount of slop grows, the white noise in people's brains, the volume is going to get so loud that I think content that challenges you in a targeted way maybe we'll cut through that white noise. I don't know. Maybe I'm optimistic because I have to be. If I didn't think I had a future in YouTube, I wouldn't be doing it. But, I wonder what you think of that. Are you an AI content doomer? Brain rot doomer? Do you kind of get where I'm coming from? Sometimes your brain just feels restless, Not my brain. My brain wants to die right now. But if you don't use it, the miracle organ on top of your shoulders, for enough time, I get anxious. Like a vacation. I think for me, the ideal length for vacation, 4 days. 4 days. Off work for 4 days, I'm happy. Day 5, my brain starts to crave challenge. And I'm not a smart man, right? I'm not here solving math or anything, but it wants to do something that feels healthy, like the brain equivalent of eating broccoli, just something. Gaming, to a certain degree, can be enriching, right? I think that there is room to argue that high skill games that require a lot of introspection and learning and practice and strategy can provide that challenge. But after 4 days, I want to work on Phi again, And I can't be the only one. If you have too much brain rot, that's why, vacations are kind of tricky because, if they're too long, I go crazy. I go nuts. But then after you go nuts for a while, your brain starts to mellow out. Like summer vacation, the first few weeks of summer vacation would be so tough for me because my brain would be like, I am still in work mode. But then after a while, you kind of chill out. Different phases. But then after you chill out, with enough chilling out, I think that anxiety comes back. I need to explore. I need to learn something. And I think that urge to learn will come. And I think if the algorithm be willing, right? Oh, magic algorithm. Hopefully they will find their way to people who make content like we do. Hard stuff. Yeah. That's what I think about that. Slop is not that rewarding to consume. We had human-made slop, like some Netflix content or Yellow Press for a long time. That's true. Still like it though. I think Let It Cake, Is It Cake is like entertainment slop. It's fun. I like it. It's good. But you cannot live on Is It Cake alone, right? That cannot be your entire diet, I think. And I think if people are forced to eat junk food content because it is what is recommended to them, I think I have to believe that people will look for stuff that's better. In ancient Greece, they thought that books caused brain rot. That's hilarious. More recently, I thought that it would be television. Now they think it's AI. I'm sure it'll be fine. Yeah, yeah. Now, don't get me wrong. As a teacher, I did notice the attention span of Gen Z. Leaves something to be desired. So I think history can trend certain ways. I don't think my generation was perfect either. But I do, like, I'm in the middle. I'm not a total optimist. I'm not a total doomer. I think we'll be fine is ultimately true. But I think at the same time, I think we will be worse off is also true. Maybe not at the end, because I think there will be a natural equilibrium. Also, science channels that are using lying to children aren't slop, but just straight up lying is slop. Yeah. I mean, yeah, don't lie. It's pretty straightforward, I think. I do love me some German brain rot sometimes. That stuff is surreal. I can imagine. Yeah, brain rot isn't bad. I enjoy some brain rot. I don't think it's bad. It just can't be your entire media diet. You gotta eat your vegetables too. I think we're vegetable salesmen here. The Fight the Nutrifil channel. What am I even talking about anymore? Just vamping. Watching the sun rise. The final 17 minutes of stream. Coming a long way. Yeah. How do you feel about this stream? So you asked for my reflections. And to summarize them, because I ranted along about them, but if the concise SparkNotes version, I want the feeling of this again. The mental celebration of getting to hang out with my content creator friends and making some fun content. I just don't want the physicality of staying up for 24 hours, y'all. That part sucks. And I'm glad to be in this position, but like from your perspective, like what did this make you feel? That's always my like what it made you think about is an interesting question, but the thing that I am more curious about as a creator myself is how this content makes you feel. Maybe it doesn't make you feel much at all. Maybe it's mostly cognitive. That's fine too. I'd be interested in that. Because the absence of feeling is itself something that is interesting. But yeah, as a creator, I want to know how you feel. Your emotions. Objective survive, exactly. Vegetables that I play. Oh, Clockwork, you missed it. I was nodding off at the end of the paper and I just said words. I was like, the king, I'm glad the king is using a fork and a knife on his plate. Or something like that. My brain thought it was cooking something, but it's just eroding. What was your favorite paper? I really liked the triple negative breast cancer and the neurons one. Oh, that one's a good one. That one's a good one. I think I have to agree. It's probably my favorite paper of the bunch. The idea that neurons and cancer are in a mutually symbiotic relationship with each other, that cancers can corrupt neurons whose duty is to the sperm and the egg. The neuron doesn't owe cancer anything, but because cancer cells are these shapeshifters, they can become stem-like. Hey, what do you want me to be? You want me to be a neuron supporter cells? I can do that because as a cancer, I am stem-like. I'm a chameleon. And getting to actually talk about the ways in which cancer can reprogram neurons and how neurons can reprogram cancers. Oh, I mean, it's neurons. My favorite cell type. It's gotta be the neuron paper. That was definitely my favorite. I only joined like half an hour ago, but I like your content, so I decided to tune in the moment I saw you were live. Thanks for hanging out here. I personally feel mentally stimulated by your content. It feels like unraveling a picture, puzzle piece by piece. I really like that feeling. Yeah. I think it's very human. And working for your food is human, in a way. I think that's nice. I think it's so fun. I meant to rest today, and that's so hard for me. But now I just keep listening to it for hours, and it's holding me from going back to work. I'm sorry about that. And I'm also happy and glad that you are here. I don't know. I haven't started up 24 hours a little, but I feel glad I was able to see the end. Thank you for being here, Hero. I definitely noticed that you were here. Temptation is strong to redeem a biology joke right now. Oh my god. You would do this to me, my own wife. My own wife would redeem a biology joke one in the 11th hour, in the literal last 13 minutes of stream. My own wife would do this to me. The stream is ending. It is ending soon. Good job, you did well. Thank you. Yeah, you're right. It's not the 11th hour. It's the 24th hour. That's crazy. The 24th hour. I'm glad to be having it come to an end. Yeah. Same cognitive stimulation as well, but also the feeling of wonder as well. Yeah, wonder. You can only get that from people who have that wonder themselves. Because, like, AI cannot wander, right? Even the perfect version of AI. Because AI is a conglomerate. AI is an assimilation of every human, which makes it, by definition, not unique. But a soul, a human soul, neutrophil soul, that's unique. Not going to be for everybody. But an AI, that's for everybody, which means it's actually for nobody. So wonder, I think, can only come from a connection that you can have. Because you can identify what makes something wonderful. So I thank you for that compliment. It's meaningful. It's a nice comment. Your content made me feel hopeful for my own studies. I was too scared to study biology because I don't think I'm super smart. But hearing that you feel that way too and still managed is encouraging. Yeah. Oh, Tyler redeemed a joke. I'll give it my best shot. Let me talk to chat first. Yeah, I don't think I'm super smart. I think I work really hard. And if you're prepared to work very hard, then who can stop you? But I think I'm also very reflective. Because I identify smart as people, some people just don't have to work to accomplish anything. They just know everything. They hear a lecturer say something, they understand it immediately. They don't have to chew their food. Their brains chew the food for them. I don't know. This metaphor is getting lost. I had to do quite a bit of chewing. My memory's not very good. I'm also very slow. Slowest reader in my graduate class. But I do think a lot. And a lot of that thinking is inefficient. Could be doing it better. But, something I learned in therapy was admiring or accepting that aspect of myself. I'm obsessed. And I think obsession can be a tool. If you're willing to throw your brain at a problem endlessly, you're going to crack it. Now, do I feel jealous of people that don't have to do that? Oh, I do. But for biology, I think if you are willing to accept that you might have to work harder than other people to get where you want to go, and you keep that humility, I think good things will happen. This is cozy. Thank you. This is a great stream. Thank you, Icky. Oh, you caught all the claps. That's awesome. Let's go. Personally, this was very inspiring. As someone who's supposed to be locked into in for a physiology test, but has struggled with it, this got me back into the mindset that this stuff is very cool, and I love it. And I love biology. Also, just the effort you're putting into this, it shows, and I should do similarly for my test. Thank you very much. Yes, slay. I did not do as well in physiology as I wanted to because, hey, I'm right there with you. Physio is hard. The list of facts, that's my weakness. I love stories. You can tell I'm a storyteller. It's what my channel is about. But in physiology, you just kind of have to know, hey, know all the bones. That's the hard work that I'm referring to. Biology is a lot of that. You can't talk about the fun stuff until you learn everything about the mitochondria. Just not possible. T-Gun, I got you. Thank you, Tyler. I was martially obligated to not redeem. So cruel. Start to understand the complexity of a cell and try to imagine it. It feels like you're watching a megacity at work. Ah, no, it's the coolest thing. It's the coolest. All right. Try to think of a last joke here. Last joke of the stream, probably, unless yeah, you're a biologist, name every bone. So true. Now that's a question you should ask your doctors. That's more appropriate. A joke. What do I end this on? I'm cooking. Despite this late hour, I am cooking. Okay. What do you call a very happy and grateful cell who has discovered a treasure chest? What do you call a very happy and grateful cell who has discovered a treasure chest full of gold? You could say that cell has a new trove filled. I've got a new trove filled. Did that make any sense? I have a new trove filled because I'm a neutrophil. The treasure chest full of gold. I've been awake for the last 24 hours. New trove filled. A DNA. I still got one out though. I still banged one out. Yeah, why are you married to me again? That's crazy. That's crazy work, bro. That's insane. You should consider your life choices. That DNA is so good, dude. I don't even think that's bad. The content. Life is content, content is life. Dude, I'm not even ashamed of the DNA one. That's probably my best joke of the day. Or Noah's shark is probably the second best joke. Like, what do you call the fish that's eaten two of every animal? Noah's shark for octopus lady. I'm proud of that one. Okay, I'm proud of that one. I'm proud of that one. Lip-id. Again, that one was maybe the low point. That one's maybe the worst. Lip-id, I gotta admit, that was pretty bad. I prefer the urchin one. That one got me. Oh, yeah. Yeah, that one was pretty good. That one was pretty good. The joke redeems, I think, were a good idea. I had fun. Although when I got like 3 of them in a row, that's when I was really stressing. Okay, so how much money did we raise today? Wow. 222. Oh my god, I can't even read the freaking numbers, dude. I sound desiccated. I am desiccated. Community. Immune cell members. We raised $2,249.61. The goal was $2,000. Double last year. Thank you all for making this charity stream such a success. I've got a lot of work to do on all these ideas, these milestones that I'll have to carry through, but I am glad that we did so. Now, what I'm not looking forward to is the Blender stream that I'm going to have to do. But yeah, T-minus just about 2 minutes. 2 minutes. 2 minutes, and maybe we'll tab over. Maybe we'll tab over to this. And maybe in these last 2 minutes, I will fade out of existence. Just ever so slightly as we reach the end. Apoptosis in 2 minutes, yeah. What about an R stream with Cy and Mylan? Hey, I want to do another charity stream next year. It's just maybe not going to be 24 hours. But I'm sure that'll be just as bad as a Learning Blender stream. But, you know, I'm open to suggestions from the community, right? Help me plan this next one, because this one was so much fun. And I want to know what you want from me. What do you think would be fun? I think the joke is really fun. $25 for a joke. I think that was sick. Yeah. 1 minute left. 1 minute left. And I get to apoptose. Counting down. The longest stream of my life. Disappearing into the ether. Capsaicin activating. Chromatin condensing. Nuclear envelope dissolving. Everything going dark. Membranes flooding. The natural cycle ends. Good night.